IIJ Chapter 1 Reproduction in Organisms 1.1 Asexual Reproduction 1.2 Sexual Reproduction. Each and every organism can live only for a set-on period of UMA. The period from birth to the natural death of an organism represents its lifespan. Lifespans of a few organisms are given in Figure 1.1. Several other organisms are drawn for which you should find out their lifespans and write in the spaces provided. Examine the lifespans of organisms represented in the Figure 1.1. Isn't it both interesting and intriguing to note that it may be as shmt as a few days or as long as a few thousand years? Between these two extremes are the lifespans of most other living organisms. You may note that lifespans of organisms are not necessarily correlated with their sizes. The sizes of crows and pru1, utsru, not very different, yet their lifespans show a wide difference. Similarly, a mango tree has a much shorter lifespan as compared to a peepal tree. Whatever be the lifespan, death of every individual organism is a certainty. That is, no individual is immortal, except single-celled organisms. Why do we say there is no natural death in single-celled organisms given this reality? Have you ever wondered how vast number of plant and animal species have existed on Earth for several thousands of years? There must be some processes in LLVTNG organisms that ensure this continuity. Yes, talking about reproduction. Something that we take for granted. 81,010 GY. BLLY12 BJ Crow 15Y Elknell Fizz, Euro 1.1 Approximate life of some or RSMS. Kefruak 0 and an ORGMJIL. Reproduction is defined as a biological process in which an organism gives life to young ones of spling similar to itself. The offspring grow, lure and in turn produce new offspring. Thus, there is a cycle of birth, growth and death. Reproduction enables the continuity of the species, generation after generation. You will study later in Chapter 5 Principles of Inheritance and Variation how genetic variation is created and inherited during reproduction. There is a large diversity in the biological world and each organism has evolved its own mechanism to multiply and produce offspring. The organism's habitability, its internal physiology and several other factors are collectively responsible for how it reproduces. Based on whether there is participation of one organism or two in the process of reproduction, it is of two types. When offspring is produced by a single parent with or without the involvement of gamete formation, the reproduction is asexual. When two parents opposite sex participate in the reproductive process and also involve fusion of male and female gametes, it is called sexual reproduction. 1.1 Reproduction. In this method, a single individual parent is capable of producing offspring. As a result, the offspring that are produced are not only itlulikalooueauliruulra, so exactly copy, as of lier par these offspring elkedi to be genet quality c and color different. The term clone is used to describe such morphologically and genetically similar individuals. Parent cell. A Figure 1.2 Cell division in unicellular organism, a budding in yeast, binary fission in amoeba. Let you see how widespread asexual reproduction is. Among different groups of organisms, asexual reproduction is common among single-celled organisms, and in plants and anima, as with relatively simple organizations. In protists and monierans, the organism or the parent cell divides into two to give life to new individuals. Figure 2. Thus, B. Biology. A CD. Figure 1. 3 Asexual reproductive structures, as zoospores of Chlamydomonas, B. Conidia of Penicillium, C. Buds 1 and Hydra, D. Gemmules 1 and Sponge. In these organisms cell division is itself a mode of reproduction. Many single-celled organisms reproduce by binary fission, where a cell divides into two halves and each rapidly grows into an adult, e.g., amoeba, paramecium. In yeast the division is unequal and small buds are produced that remain attached initially to the parent cell which, eventually gets separated and mature into new yeast organism cells. Members of the kingdom fungi and simple plants such as algae reproduce through special asexual reproductive structures. Figure 1.3. The most common of these structures are zoospores that usually are microscopic modal structures. Other common asexual reproductive structures are Conidia penagudum, Buds Hydra and Gemmules sponge. Adventitious B. Root CDE Phi. 1.48, a Uv pro and tusforms, a eyes of potato, B. rhizome C. bulb of a sove, Julia E. buds of bryophyllum, E. offset of water hyo and LHU learned AFX, at OE9 at reproduction in PRLs in class XL. I not do go TNNL, is OE9 at reproduction also oak pair of asexual reproduction in 1J do go sag so is the term done OPP1I.COBE to the, Deej Sprin 9 formed. BGOE9 at reproduction while in animals and other simple orgaru SMSLHE term asexual is used unambiguously. In plants LHE term vegetative reproduction is frequently used. In PL3 and TS-LWL it's a vegetative propagatlon such as Vermeer. Ilsomsfuckleter tuber offset bulb are all capable of G.V. and rise to new offspring figure 1.4. These structures are called vegetative propagales, obviously since leaf formation of leaf structures xej not invo two parents leaf process imut. Is.S, UL. 81,010 GYU rust have heard about the sewage of the water 60 dies or about the terror of Bengal. This is nothing but the aquatic PLMT water hyacinth which is one of the most invasive weeds upon drowning where there is standing water. It drains oxygen from the water which leads to death of fishes. You will learn more about it in all chapters 13 and 14. You may find it interesting to know that this plant was introduced in India because of its beautiful NM-S shape of leaves. Since it propagated vegetati iliata phenomena one weight and spread all over the water body in a short penetrative line. It is very D1 I got to get rid of one them one rejawa where how PLMTS like potato sugar kambanji, ger. Dahlia are cultivated pale you seen small PLMTS emerging from the buds called eyes of the potato tuber. From the rhizomes of banana and Ginny Jr. when you carefully try to determine the site of origin of the new plantlets in the PLMTS listed above. You will notice that the invenibly ants from the nodes present in the modified stems of these plants. When the nodes roam in contact with damp soil or water they produce roots and new PLMTS. Similarly, adventitious buds arise from the notches present at margins of leaves of Biotechnology. This ability is fully applied by gardeners and farmers FBR commercial propagation of such plants. It is interesting to note that asexual reproduction is like non-elective reproduction in organisms that have a relatively simple organization. Like algae and FunMD that they shift to said UAL method of reproduction just before the onset of adverse conditions. FLND out how sexual reproduco, amino, F says these are 9 on, SMS to suralodon, N9 urafeawaro, FZCN trans THG, SML. Reproduction crew under such conditlgy and asexual legislative as 1 ALLSL, UAL modes of reproduction are exhibited by the higher plants. On the other hand the asexual mode of reproduction is present in most of the animals 1, 2 Smrual RJ, Pau Duckman said, UAL reproduction involves formation of the male and female gametes. Either by the same individual or by different individuals of the opposite sex, these gametes fuse to FBRM the ZJ, which develops to form the new org3JLism. It is an elaborate complex and slow process. As compared to .S. Rual 1 EPO, Luction, because of all fusion of male internal gametes. Set, UAL reproduction results in offspring that are not identical to the parents or streetly themselves. A study of Diller's organisms PLMTS animals or fungi show that though they differ so greatly in external morphology, internal structure and physiology when it comes to said UL mode of reproduction surprisingly, they share a similar pattern. Let us first discuss what features are COL to the east diverse organisms. All organisms have to reach a certain stage of growth and maturity in their life before they can reproduce said ULI. That period of growth is Phyophagogen in organisms called the juvenile phase 1 is 1 numerous vegetative peta, in plants. This phase U of animalcations and each parent organisms 11 LLLHCAJU, LLGetally ebol which are MRLTS the veganan, Angatheraprik, Leucupha, can be seen easily in the Higler plants when the G come to flower. How long does it ekagerem? RRLDFRWLTCTO, O, new 0.1 meters 290 PO neck, EO umeo flower and some peones. WLDRTJL J occurs more del, XT once W11 at WL.D goul, the Eiffel, R, FOIL, JPTR odd, Chtwinil or MCTR, observe a few trees in J area. Do they flower DLLTLNG this RM, Enthel R after ye, TY do you think they have, Jab WTY if fruits like apple, jackfruit etc. is, C0L31 are there some L plants that flower IIU out the year and same others that show seasonal flow and NTS the annual and Beanie 3.1 types, show clear-cut vegetative. Reproductive senescent Fa, says but in the perennial speed SID is very difficult to clearly clefine these phases. A few plants exhibit unusual low, L-aringfo, iodonin, or rhythm such as B3 co-species floral ones in their 3, un. Get arrow after so L00Rs, FW acclular, Nuldler of fruits anklil, another plantabilectless 1AUFTLML Nila Kuranji. Exclamation point lowers once in 12 Rs, as many a U10 Uklabilu, owing that this plant flowered ton September-October 2006. Its mass OC.W. Ring transformed large tracts of hilly areas in Kerala, Umatica and Tornal Naklu into blue stretches and attracted a large number of townsfolk. In animals the Jew, and alpha, set is followed by physiological and physiological changes prior to acute reproc, Luke Taylor. The reproc, Lucy U. E phase is also a variable dual in different organisms. C. Ln go, is, N NLW man bay RGSE 11 AEOR, if DICT, TT we of R, Produ, Phil, Mabni TGM, Eng animals. LBR example birds, do they take XAUIIU oof the yen, or is 11 a seasonal phenomenon? And what about other animals like frogs and usarclish? You will know use that, birds leaving in nature lay egg. OOIC Sonali, how, are birds in captivity as in poultry, MMS can be made to IAX throughout the year. In this case, lay, eng eggs is not related to repro, TUCU1 but is a rarial exploitation for human welfare. Tile-iamale supplemental mammals exhibit C-click-all chain, S in the AQVU sophuans and XS adducts as N as B, T nosk tun the Robert du CU phase, in non-male man walslow rose, sheep rouse clears clogs liger. Etc. Such CCII colchins dungrit tota clicknar called ostraves cycle horis and mates TN, LL, EYS. Apes and humans 11 is collectal and natural cycle, many mammals especially those leading in natural. W. Crocodilians exhibit such cycles on unfavorable seasons in Thelreproc, Luke U phase and are therefore called seasonal breeders. Many other mammals empro, lucked very active throughout the E1 repro, tuckle, if phase and hence are called conti, nose breeders. That we all grow old if A live long enough is something that we recognize. Ball W hat S meant BG growing old. The enteroproc, luck, if. Biology phase can be considered as one of the parameters of senescence or old age. There are concomitant changes in the body like slowing of metabolism, etc. During this last phase of lifespan, old age ultimately leads to death, in both plants and animals. Hormones are responsible for the transitions between the three phases. Interaction between hormones and certain environmental factors regulate the reproductive processes and the associated behavioral expressions of organisms. Events in sexual reproduction. After attainment of maturity, all cellularly reproducing organisms exhibit events and processes that have remarkable fundamental similarity. Even though the structures associated with sexual reproduction are indeed very different, the events of sexual reproduction are elaborate and complex. Follow a regular sequence. Sexual reproduction is characterized by the fusion or fertilization of the male and female gametes. The formation of zygote and embryogenesis. For convenience, these sequential events may be grouped into three distinct stages, namely the pre-fertilization, fertilization and the post-fertilization events. 1 pre-fertilization events. These include all the events of sexual reproduction prior to the fusion of gametes. The two main pre-fertilization events are gametogenesis and gamete transfer. 1 gametogenesis. As you are already aware, gametogenesis refers to the process of formation of the two types of gametes, male and female. Gametes are haploid cells. In some algae, the two gametes are so similar in appearance that it is not possible to categorize them into male and female gametes. A B figure 1.5 types of gametes. Isogametes of cladophora in alga, heterogametes B fucus in alga, see human beings. A proceoc, IOMATGA, I9J, they are hence are collect homogametes Lysogametest I5, HAV, E. Are in a majority of sexually reproducing organisms TLL e.g. Aretes PM, Lustme of 11.1, 1, Y, Morphological Stift heterogametest. In such organisms TLL the Areate is called Liantherazo1D or Spenanthophymalgamete is called the Eggerovum figure B. C. Sexuality in organisms. Sexual reproduction in organisms generally involves fusiology Aretes from Chloyo different individuals, but TLL is not ALWIs true, from go record ledlin of examples to led LNCASSM. Godentive cases where self-fertility so, 9cn, S of zeroed, of course citing such amples in PLNTS is easy. Plants may have off male on female repro-clive structures in the Sara plant bisexual figure I6C or on different plants sexual figure I6D, in says. Aerofungi on plants. Terms such as Latomontalc and monoectus are used to denote the bees. UL condition MD heterothalc and D1OEC1S are lead terms used to describe WLIs, UL undilian LN flowering plants. The unisexual male were is staminate, that is, being sterins, while the female is slate or bearing pistils. In some FLDA ring plants both male and female OORS may be present on the Sara individual renoceus or on separate individuals dioecias. So examples of endnechious PL3 and TS are cucurbits and coconuts, and of dioecious plants are papaya and date palm named pear. Q9 ametes that are formed LN street road and PL stewote flowers. But what about animals or individuals of all species Elia or female WT1 said, one lauer are there species WH which possess both TLL reproductive organs bisexual uprob, J, BLG can make ATL street of CRL unisexual. Animal sped.es earthworms, Figure 1.6 A sponge type have arm and leech typically, amples of bisexual animals that possess both male MD female reproductive organs. Are hermaphrodites. Q. Lacroche Figure 1, 6b is a me, ample of a WLIs, UL species. Cell division during gamete faunat. Garetes in all heterogametic species are of two types, namely male and female. Garetes are haploid, though the parent plant body from which they ensem may be either haploid or diploid. A haploid parent produces garetes by meiotic D1 is greater than 1 sion. Does GNS mean that Mieloschus Neoroca NSN or 9 and SMS that are haploid? Carefully examine the flow charts of life cycles of algae that you have studied in Class Sci Chapter 3 to get a suitable answer. Several organisms belonging to Monera fungi, algae and bryophytes have a haploid plant body, but organisms belonging to tertophytes, GJMN osperms and osperms and RMST of the animals including human beings the parental two, bias D, child. It isovicus that radiocyst therctuclic division, has to occur to a diploid body has to produce haploid garetes. Diploid organisms, SF sialist cells calid 1 oocytes gamete rather cell 1 old, argioreiosis. IUTLLE end of reiosis only one set of chromosaurs. 81,010 GY glitellum mole test plus a CB fig, your 1.61, ers 1 tie of sexu, 3 lidi 1 n or lusums AJ bisexual imthorn, biums, su 3 LLO, I claozchl, C mana E1 SPL3 and TH row, DD10 ecten SPL3 and T. Mchmu, EB, L flower SW, at potato. Ulage kuan and oiganwlt, CH, Ramos and numbers in ME is at diploid, 2 and colon ANDG, unets PTO, D. Enosum fill in the blank F, Ates, na MT of MCH, Romo, some number chromosome number in, Te2NN. Note NHV, MNBF GJ4623 HLLY 12 RA 21D colon 978 CE 19 FTV, TFT, J8 off, 1. Fern 630 IIAPPL, 34 III, 12, MF 20 liters POO, TO 24B, TTTCFY 380 joules ort, RT 16. G. Incorporotect into each gamete. Carefully study Table 1.1 and fill in your diploid and haploid curve. So are numbers of organisms. Is 1, Riang Relit CMNP and at number of CL Ramosomes of MeCCGT, and 9 AMS1, 2, 1, 2 GOM, TTE transfer and tear their armadion. Male and female gametes must be fist, UI brought together to facilitate fusion fertility study on H, IOU or 1, air, D1O at 9 AME1S meet in Emaptid organisms, male gamete is motile and the female gamete is stalmally. Exceptions are aloof fungi and algae in which both TGIs of gametes are remtile figure 1.7 a. There is a need for iridium LHRL, which the male gametes, in several simple plants like algae. BLJ off JIES and terlgphytes water is your medium through which this gamete transfer takes place. A large number of the malagaretes however, to reach your female gametes, to compensate this loss of male gametes done term sport. The number of RRUJ gametes producing is several thousand limes the number of female garetes produced. In seed PL, NTS pollen G, 3 ins are the kirtero of male gametes and ovule have the egg, pollen G, 3 ins produced in anthers ulariform. Have to. Fusion of gametes figure 1.7 biology zygote new individual be transferred to the stigma before it can lead to fertilization figure 1.7b. In bisexual, self-fertilizing plants, e.g. peas, transfer of pollen grains to the stigma is relatively easy as anthers and stigma are located close to each other. Pollen grains soon after they are shed, come in contact with the stigma. But in cross-pollinating plants including dioecious plants, a specialized event called pollination facilitates transfer of pollen grains to the stigma. Pollen grains germinate on the stigma and the pollen tubes carrying the male gametes reach the ovule and discharge male gametes near the egg. In dioecious animals, since male and female gametes are formed in different individuals, the organism must evolve a special mechanism for gamete transfer. Successful transfer and coming together of gametes is essential for the most critical event in sexual reproduction, the fertilization. A 1-2 fertilization. The most vital event of sexual reproduction is perhaps the fusion of gametes. This process called single JDY results in the formation of a diploid zygote. The term fertilization is also often used for this process. The terms syngamy and fertilization are frequently used interchangeably. What would happen if syngamy does not occur however? It has to be mentioned here that in some bee organisms like rotifers, honeybees and even some lizards and birds turkey, the female gamete undergoes a homogametic contact in alga, bee germinating pollen development to form new organisms without fertilization. Grains on the stigma of a flower. This phenomenon is called parthenogenesis. Where does syngamy occur in most aquatic organisms? such as the majority of algae and fishes as well as amphibians, synchrony occurs in the external medium water, that is, outside the body of the organism. This type of gametic fusion is called external fertilization. Organisms exhibiting external fertilization show great synchrony between the sexes and release a large number of gametes into the surrounding medium water in order to enhance the chances of synchrony. This happens in the bony fishes and frogs where a large number of offspring are produced. A major disadvantage is that the offspring are extremely vulnerable to predators threatening their survival up to adulthood. In many terrestrial organisms, belonging to fungi, higher animals such as reptiles, birds, mammals and in a majority of plants bryophytes, pteridophytes, gymnosperms and angiosperms, synchrony occurs inside UPO, IJC, IO2, OTCA, I9J TREBC, organism. Hence the process is called Untermalfertilsatg, N. In these organisms egg is poured inside the female XII where they fuse with the rule G8. In organisms exhibiting internal fertility on the mole gamete is rotile 3JLD rues to reach the egg in order to fuse with it. In these even though the number of sperms produced is very large there is a significant reduction in the number of eggs produced. In seed plants however the non-rotile male gametes are earned to female garete by pollen tubes. 1.2. 3 post-fertilization events. Elements in sexual reproduction after the formation of zygote are called post-fertilization events. 1, 2, 8, JTH. Zygote formation of the diploid zygote is universal in all sexually reproducing Org3J-elisms. In Org3J-elism SV, lithic stem 3L fertilizations each gote is formed in the E-tenyl region usually water. Whereas in those inhibiting internal fertilization, zygote is stored inside the body of the organism. Further development of the zygote depends on the type of life cycle Org3J-elism. H3S.MCT. The environment it is exposed to. In organisms belonging to fungi and algae zygote develops a thick wall that is resistant to desiccation and signage. It undergoes a period of rest B, or germination, in organisms with H3 plantic life cycle as you have read in Class I. Zygote divides by meiosis to form haploid spores that grow into H3 ploid individuals. Consult Igor Class X1 book and FLLD. Out will OTL or D if DOPM and takes PA, E in the ZG, goat, N or 9 SMSHDP on DC on ha PODP on, C life CGCS. Zygote is the vital link 3T ensures continuity of species Beto in organism as of 1 gen or 3 Cheyenne.N. TLLE next. Ellery Saxu 3LLY reproducing organism including human beings begin life as a single cell as I, PE. 1, 2, 8, 2 embryoj. Msys refers to the process of development of embryo from the zygote. During embryogenesis, zygote undergoes cell division mitosis MCT centiferentija TK, N. While cell deletions incurs linear of cells in the developing embryo, cell differentiation helps groups of cells to undergo certain modifications to 1 BRM specialized tissues. MCT organs to TBRM. In Org3J-elism UH3 studied. 3 Botley process of cell dilation. MCT differenti 3 Cheyenne premise CLS animals are KJ, lanced into oviparous and viviparous based on W H E T H E R T H E D E V E L O P M E N T O F T H E Z Y G O take place outside the body of the female parent or inside. That is, whether fertile eggs or give birth to young ones, in oviparous animals like reptiles and birds. Biology. The fertilized eggs covered by hard calcareous shell are laid in a safe place in the environment. After a period of incubation young ones hatch out. On the other hand, in viviparous animals majority of mammals including human beings, the zygote develops into a young one inside the body of the female organism. After attaining a certain stage of growth, the young ones are delivered out of the body of the female organism. Because of proper embryonic care and protection, the chances of survival of young ones is greater in viviparous organisms. In flowering plants, the zygote is formed inside the ovule. After fertilization the sepals, petals and stamens of the flower wither and fall off. Can you name a plant in which the sepals remain attached? The pistil however, remains attached to the plant. The zygote develops into the embryo and the ovules develop into the seed. The ovary develops into the fruit which develops a thick wall called pericarp that is protective in function. Figure 1.8. After dispersal, seeds germinate under favorable conditions to produce new plants. Figure 1.8. A few kinds of fruit showing as seeds S and protective pericarp P. Summary. Reproduction enables a species to live generation after generation. Reproduction in organisms can be broadly classified into asexual and sexual reproduction. Asexual reproduction does not involve the formation or fusion of gametes. It is common in organisms that have a relatively simple organization such as the fungi, algae and some invertebrate animals. The offspring formed by asexual reproduction are identical and are referred to as clones. Zoospores, conidia, etc., are the most common asexual structures formed in several algae and fungi. Budding and gemule formation are the common asexual methods seen in animals. Prokaryotes and unicellular organisms reproduce asexually by cell division or binary fission of the parent cell. In several aquatic and terrestrial species of angiosperms, structures such as runners, rrbizomes, suckers, tuboiphytes, etc. are capabmigtvtngr1 sebi, wguwlrpr, duroijon nair, lly. Oxual.produ, trontrul ue pound symbol, nat1 on individ is a complant, XII slower process as compared to .s, su alr production muaflat animals reproduce almost endlessly by meth, d. EOERO of sexual reproduction may be karst 1 RLOP, fertiles, Asian far end, MD post, fertilization, TS. FN fert 111S Chuan TS Laclude Guyameta Gemsls 3 NDG3 JLL TRML sphere while post T Fe, Neuron eventually Ida Hoff form. Chuan of zygote D embryo, or GAR DSMS MBBs, Su, 31 or WLI sexual sexuality TN plants TS, lead. P. RT Lu WLY1N sperms to low UE product John of divorce types OU wers, P13 RTLS are defined as mono E1S road. Gloctus, FBWCSM.YB sexual or unisexual fluv WS hab, I1N nobu and usually not proc bell, T, 4E, OOP, 1NH3 PBLD org, RDSNS, Teragi, Meles. BRRN by, Fiaswafer, 4 male 1S and Essent 101, 1NHXIW.odd, N, IT 11 Reptile Valley Easy 1N bisexual organs, MS. In Uni-Z, XWL. And TNA1 Acetoku, RSBF Capo, 1K01 Sunalgain datus release in Aspra1A Aspialapruasolidpoi, NI, on Enetensrums far DP, when U. Pollini section to He6FFFK, U81 odd occurs bithamali on Lamoyosy Mawi occur etharix demili. Outside TTE body of orgulums or TN mali LNJSDETLE toti, SY leads to ilrmuon of .A, alpha, DOOU call, Gilo, I. 1 He process of development of embryo zygia, S called ortbryo S in LHE zy 10, starts alp, N, soonfi, 1, TS finatlan animals may be either oviparous or viviparous. Ulbrional prot aotland.ar, DCMEX better 1 and viviparous archu, in F1 PLMS odger ferotsotion. Ovio ridioiops 1 and TO frontal diovu TM.2 Wheaton seeds HLS1 the mature seed 1 STHT progenitor of 1 JWCL gainer 3 lion E, embryo. XF, Cises LY is reprodu, U1 Esrolite X organtsum, 2, which TS. A bet McD reprodu, bonsexual or mex, UALY 3. Isit OFSPR ang fond by S, UAL reproduco on R0, D duck 4, offspring formed due to sex UAL reproduc, U1 H.OE bet S of OWV. 1. Y is THLSSL, dem and law is true thus the progeny formed from asexual and product John differ from though, larmed bisexual reprodu, tort 6. 0 Bell OS, Odual and sexual RPD 0 John, Y is veget. Very prodotion also unsti, edos a type of us, UAL reprodu, shirt. 7. What 1s. Teget, D, propagation GHTVOSUIT ably, plus 8, define a Jew, MJEPH, SO. B reproduct PH, SOC Semsk, Entfos 81010 GY9, Higl, org 3L Lisrans Ha, resorted to sexu 3L reproduction 1 in spite of its C, Ornflex 1 type. Y10, exp 3 inches Y meiosis. Malgametoj, Nes 1 S me alwaysn't, Rumpt 11 Lafia Oppo, RD1 N 0 OTPLTI Wintavi Antherid LS Haplo 1D and Diplo 1D 2N. Ribi Anther Sieg D Polynemol G, Medal Zygote 12, define external fertilization Manti and its DCDS. 13. Deferin 11-8 between a zoospore and a zy, M14, Defir, NTJ8 between G netoj, nests from E, MBRORES1S15, Dest the post-fertilis lon LNU were 16, what 1S a bisexual or. Logt 5 said, Zool flowers from your neighbor, Lo, Ian Watililia help of your teacher BN, L out ili 1 R common SC1L. Ltifgar WS17, XTNE a few flowers of my Q Kerbit page 13 NT try to identify your street C, Nate page 1 SL at Flovers, do hole my oilier page 13 NT all at BMS2 use XU3 LOOers. 18. Ymi offspring, FOVI mus animals at a GTEMF4ESK comp MEC 2 of SPRMG of, Wilpo, Rho, LS. Dot. Chapter 2 Sexual Reproduction in Flowering Plants 21th Tower 8 Fascinating Org CI of Antosp 77 and 22 Prefern Usitic, Stir, Tures on Even TS 23 Double Fertile Cheyenne. 24 Post-Fertiliastic, Stir, Res on Even TS 25 Apami.ds und Poemblani are we not lucky that PL3 and TS Reproduce Sexu, Uji the Reninoxofuers that we enjoyed freezing it. The sense only perfumes tlat we style and the rich colors tlat dot attract us dot r dot all tllr dot as m dot 8. To sexu 3l reproduction, flowers CB not exist only FBR has to be used FBR our own cell one ishnas, all FLC, Mendel 3 and TS show set, UAL replicate XII. A looked out at TL1E diversity of structures of Li and 1 fluorescences, OORs.N, O or AL parts shows .N. Amazing range of adaptations to ensure formation of the end products of sexual reproduction. The fruits on seeds. In this chapter let us understand the morphology structure and the processes of sexual reproduction in Tloaferl PL3 and TS ong, Osperans. 2. JL01, ER, AF, ONA1L, IG0RGIDIO, PA, 3, WSFTM5 hum, M beings have had an intimate relationship with Uwers since tinny iron, RN0RT3.1. Flowers. Are objects of aesthetic art the 3 and also social religious and cultural value they have always been used as symbols torcomi, eng import ml hurl wl feelings such as love, affection happiness gr, f earn, eng etc, least at l least fo, ejl.ru, ers of our name and tlo2e that are common cult wadded ot. 81,010 gy fig, Euro 2.1 adao, mj nauk representation of ls of a homes and in 9r, uns, ftr1d out the names fo, em, real, where's tutor used inside a. Dot and w12oee, out ons and gulfkrami, ijhogc you heard of fo, rrmll or uiat doesn't refer to a biologist. Flowers are morphological and embryological novels. Mictly sites of cell, U3L rep, toxin, in CL3SSI, well have read TL1 even QS ports of a November. R figure 2, I will help LJU recall the ports of a typical flower, C3N UNAR RE L 1.0 parts in a OCNR in which L 1.0 NDSL import on units of sexual reproduction delalab much of actual FLC, Muricine on a PLMT. The decision that the plant is, Pidge 1G to November, R has green place. Several horolon structural CS are initiated which led to the differentiation, and further development of the floral prindidium LN11 or essences or afford which bear theflical buds on then the November, RS. In the flow, are the male and female reproductive structures themtrosagon on the ginoke, WN differentiate and develop. Ulalo recollect TLL at Lee.N consists of a whirl of street arons representing the male reproductive organ on the ginosagon represents the female reproductive organ. UXIIAIU head of SIC H and FTOUI and GTLAMT 2.2L stand.N, Lacrosse Baranga Euro and Pollen QM 2.2 a show W's the T. Comma parts till fleeslam in the long ankle slankle, R stalk called the flament, and the terminal narrowly B-lobed structure called the anther. The PM, I'm and off the fill, ent is all to checkle to the thalamus or the petal of the flower, the number and length of stamens are vartable and uber soft, pharynx B size. If you were to see, I'll act a stare on each from 10 flowers each from Elf, and I'll sprockles and arrange them on a slicklo. 11. Uglebo able to appreciate LHE large variation in scene in nature. C3 full observation of each stern under a dissecting microscope and making neat diagrams would elucidate the range in shape and attach of anthers in different flowers. A typical eng, ospermanther is betrothed will each lobo having teeth that is, they are dithecau, Lgiaro 2.2. Often a longitudinal grew-1 runs jangle-wise separating the theca. Let us understand the Vannis type CT tissues and their aggregation 1 and tans, RSE section 10 anther 233. Though below nature CT and anther is very distinct in Lhedrums versus section 1 offline her, the annular is a 4-est clockal tetragonal seducture consisting OT, NJ arm cross baranta located at the corners. 2-1 and each lobo, the N1 cross bung, a develop further and alb, C pollen sacs, they extend long. Tucklinally B allow the length of an anther anklar packed figure 2.2 LA type 1 C31 stamen, with pollen grains. B Louis D1 mention all quiescent structure of crossbarantum, in a TRMS, ursav.m. Annelier Socklian. A DJ Pikel microspirang, appears near circular 1N outline, it is generally surrounded by 4 wall layers figure 02.3 B3 Baclarmus and Plahesium middle JA and anklo to pay and low outer THN. Layers pabno function at protection ankle help in clawhus NCT anther to release the pollen. The LF1 MST wall layer is a tapepinell nourishes thickly yellow ink psylling grains, set to patum possess dense cytoplasm and generally whole hank nucleus. Can IOUN, T1, XOT, T's tau cells O, TDDL, EC, IM1, N when the anther is Jounga group D. CCRQ, aculi arranged homogeneous cells called the sporogenous tissue occupies the center of each MIC, sporangum macrosporokelesls, as the anther develops. The cells sporogenous usuo unclargoriotic divisions to form acrosporotelracles, WMT while DBN. PLOI sidage of Cl12 cells of Cl12 cent, TAD. A B8 II Avil, LBI, IDI, Liredlafis, L, AE 2.3 AJT Arcmisvet, RSE section of a mature new, Thur, BENL3R. MW of row 1 crossporum showing wall layers, C8 also 3 nulheras each cell of the sporognous tissue is capable of giving NSC to a microspore tetrad. Each one is a patenti 31 pollen or microspore and clear cell FMC, the process of formation of microbes, res from a pollen rather cell through meiosis is called microsporogenests. The microspores as they are for edge are oronject in a cluster of FR cells the macrospore texid 2.3 up. As the antlers Ma1 urine degrade the microspores dissociate from each other and develop into pollen cells figure 2. 3BL and Sire each microspore 3 Jlgian cellar LTLL thousands of microspores or P. Glengraians are formed. That are released midly to hissence of antlers figure 2, 3C. Pollen cell 1 he pollinger 3 LNS represent the male Garadoptes. If do touch the opened anthers of H. oaks rus or any other flower UV old fine deposition of yellowish powdery pollen grams on your fingers. Sprinkle TLLE's grains on a drop of water taken on GL3SS/MD observe under. Euro 2.4, Cal LTNG electron NL micrograph S a few pollen grant NESA microscope, you'll really be amazed at the variety of architecture sizes. Shapes colors designs seen on the pollen grains from diff. Ent species figure 2.4, polar. Grains are generally spherical meast, ring about 25-60 micrometers in diameter T has a prominent layered wall. The hard outer layer called the XII is made up of sporopollen and 1. Hitch is one of the most resistant organic material known. T can stand high temperatures and strong acids and alkali. No enzyme that degrades sporopollen is so far null NN. Pollen grain XII has prominent apertures called ermopores where sporopollen is absent. Pollen grain 1s are well preserved as fossils because of the presence of sporopollen. The XII exhibits a fascinating array of patterns and designs. Why do you thin, the E, Cine shot DB hoard what is the JJRV, Tian of Genpore the inner wall of the pollen grain is called thentine. Tis a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature it contains no cells the agetity, E cell and generity, E cell flugu, E2.5b. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. Tis spindle-shaped MTH dense cytoplasm and a nucleu. In over 60% of angiosperms, pollen grains are shed at this 2-eld stage. In the remaining apices, the generative cell divides methodically to varies to the 2 male gametes before pollen grains are shed 3-celled stage. Figure 2.5 enlarged 2 of a pollen gram tetrad. These stages of a M1 crosspore mating into a pollen gram polar. Grains of many species cause severe allergies and bronchial affliction, in some people often leading to chronic respiratory disorders asthma, bronchitis, etc. T may be mentioned that 01 Theralion or Caracas that came into Huldiazak and Tarnanalt with imported wheat. Has become ubiquitous in occurrence and causes pollen allergy. TTL, R.U, IT 1 biology pollen grains are RTCH and nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses figure 2.6. Pollen products 30B pulled from England natural bee collected pollen in tiny golden pods a natural food from Luni bees NET weight 0.45 OZ. Figure 2.6 pollen products when once they are shed. Pollen grains have to land on the stigma before they lose viability. I've the Vito BRTNG about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. You may have heard of storing semen sperms of many animals including humans for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen 196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks, in crop breeding programs. 2. The pistil, megasporangium ovule and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil monocarpellary or may have more than one pistil multicarpellary. When there are more than one, the pistils may be fused together syncarpous figure 2.7b or may be free apocarpous figure 2. 7c. Each pistil has three parts figure 2. 7a. The stigma, style and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you studied in FIIAI Perseidsidison 11 ROUING PIM style I, VII flat will, PJJUURN, C muntum 0, C2 to lol pi. A BCD FEMI 2.1 Udlesected flower of HCB.SCUS Shomg PLSTLL Other oral parts Hobenrimal, RL, B muli, WY. Syrupus PLSLLL of Popover, CAMU, UCPL, Zyapik Paus Genoctum of Mitchell C, DA Diagram, MMA 11C View of a liberal ana 1 Rapus ovule CUSSI, arising from the PL3 centa or the megasperm top. Umrally called ovules, the number of ovules in, NOVMJ may be 1 wet, T-packed D mango to many pape. W. Terrellin orchids, the megaspoxangum ovule, let us fornilims ourselves with a structure of, typical undispernervule figure 2. 7d. The ovule is, small structure, attached to the PL, centered by means of, stalk called funicle. The body of the fuses with funicle in the regden called hinum. Thus hilum represents Lee junction beta in ovule ANCT funicle. Each ORUE highest 1 or T100 protective envelopes called LNUG humans integrants encircle the ovule E, sept, T the tip where, small opening called the micropyle is ocunized. Opposite the micropyle 3 ranked is the chalixa representing the basal port of the ovule, enclosed, nilline Lee integrants is A. Ma SSC1 cells Lee nucellus cells of Lee nucellus lube AB1 uli, W1T resine fixed nude, Re3LS. A cartadian Lee nucellus is the embryo sac of female G, a merophyte. An ovule generally has single sac Y, led from reg spore edl TCTI and tilsim. Megasporogenesis, the process of phenalion of reg, spores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate, single regospore M, thyrcell MMQ in the micropym region. A macropylar and B biology liripeter and megaspore tetrad, polar knuckle D4 central sed. ILR egg micropylar and C figure 2.8 of parts of the ovule showing a large megaspore mother cell. A dyad and a tetrad of megaspores, B1, 2, 4, and 8 nucleate stages of embryo sac and a mature embryo sac. See a diagrammatic representation of the mature embryo sac. Of the nucellus, it is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of 4 megaspores figure 2.8a. Female gametophyte, LN. A majority of flowering plants, one of the megaspores is epiloxional while the other 3 degenerate. Only the phtuxional megaspore develops into He female gametophyte embryo sac. This method embryo sac formation from a sigilly M. is 10 monosporic development. What will be the ploidy of the cells of the nucellus, MMC, the functional megaspore and female gametophyte? XII, Pruactus and flower and pine. Let us study a formation of the embryo sac in a little rory tale. Figure 2.8b, the nucleus of the functional regospore divides metodically to F ormonuclei which move to the opposite poles. Opening the 2 nucleate M fo sac, 2 neurosequential mitotic nuclear D Lee scions result in the F formation of the 4 nucleate and later the nucleate stages of Lee embryo sac. It is of interest to note that Lee semitotic divisions are SDRTCUI free nuclear, that is nuclear dilaucions are not fall DDI or LE, liantly by cell wall F formation. After the 8 nucleate stage cell walls are laid down leading to the organization of the typical female gametophyte or embryo sac. Observe Lee distribution of cells inside Lee embryo sac figure 2.8 BC. 6 of the 8 nuclei are surrounded by cell walls and org are used into cells. Lee remaining 2 nuclei called P. DAR nuclei are situated below the egg apparatus in the large essential cell. There is a characteristic distribution of Lee cells within Lee and fo sac. 3 cells are duped over at the micropylar end and constitute the egg apanthus. The egg apparatus in turn consists of L synerg DS and 1 egg cell. The synergids have special cellular thickenings at the microbar tip called filiform apparatus. which play an important role in guiding the pollen tubes into the synergic. 3 cells are at the chaliceal end and are called the anapotels. The large central cell, as mentioned earlier, has polar nuclei, thus, a typical angiosperm embryo sac, at maturity. Though S nucleate is 7, dead 2, 3 pollen. Cheyenne in the preceding sections Chowdale learnt that the Myanmar margarites and phloem plants are produced in the pollen grain and embryo sac, respectively. As both types of garites are non-rotile, they have to be brought together for fertilization to occur. How is this achieved? Natman is TLLE to achieve this objective. Transfer of pollen grain shed from the anther to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They melt use of external agents to achieve pollination. Can go TL street the possible externatants kinds of pollination, depending on the source of pollen. Pollination can be delivered into 3 types. IOT09 acne. In this T-pay pollination is at shield. Whittle and Lee Saroware, transfer of pollen gains from the anther to the stigma of the Sarah flower figure 2.9a. In a Encarowell flower which opens and exposes the anthers and the stigma, complete autogon is rather rare. Autogon when such flow, RS requires synchrony in pollen release and stigma receptivity, and also the anthers and the stigma should. A B figure 2.9 a self-pollinated flowers, B cross-pollinated flowers, C Cleistogeras FTO, Variolk, like close to each other so that self-pollination can occur. Some plants such as Loda common pansy, oxalis and comet, have produced 2 types of flowers, chasmogamous which are similar to flowers of other species with exposed anthers and stigma, and gleistogamous flowers which do not open at all have 1 gear 2.9c. In such flowers, the anthers and stigma lie close to each other. When anthers dehisce in the flower buds, pollinanes come in contact with the stigma to effect pollination. Thus Cleistogarnous flowers are invariably autogamous, as there is no chance of cross-pollen landing on the stigma. Cleistogamous flowers produce assured seed set even in the absence of pollinators. Do you think that gleistogamy is advantageous or disadvantageous to the plant? Why? 2 Guidenogamy, transfer of pollen gains from the anther to the stigma of another oower of the same plant. Although guidenogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen gains come from the same plant. 3 Xenogamy, transfer of pollinanes from anther to the stigma of a different plant. A section, Re 2.9b. 1 His is the only type of pollination which during pollination brings genetically different types of pollen gains to the stigma. ENB of pollination, PL and TS use 2 abiotic wind and water and 1 biotic animals agents to achieve pollination. Majority of plants use biotic agents for pollination, only a small proportion of plants use abiotic agents. Pollinanes coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen gains, the flowers produce enormous amount of pollen when compared to the number of ovules available for pollinal. Ian. FIIAIT, Pruigticinil flowering pymil pollination by wind is more common, and GST biotic P, linations. Wind P, lination, LSO request that the pollen grains are light and non-sticky so that they can be transported. In wind currents, they often possess V, LEXP, said street iron so TLL at the pollens or easily dispersed interwind currents. Figure 2, I0 and IRG often Fe, Thuri stigma 2E, Silly TR, P, airborne pollen GR, ends, wind pollinated flow, RS often have, single owl and E.H.O.V.T.N. Neuris fluv, Ers packed into .M and 1 lurescence. A family R. Ample is Lee Comcob the tassels, 1 juicy or nothing but the SI section non-style which wave in the wind to trap pollen GR, ends. Wind pollination is quite common in grasses. Pollination by water is quite rare in phloem plants and is limited to about 30 genera. RMSUI monocotyledons, as against this ODD recall TLL at water is irregular LDD if transport for the male garites arm the lower plant groups such as algae, bryophytes and pteridophytes. It is believed, P. reticle, RLY for some bryophytes, and pteridophytes, that their distant ion is limited because of the need for water FBR the transport of male gametes. N. Fertilization, some F. Euro 2.10 AWLRD pollinate, LPL, show WLL, G compact inflorescera well posed. X, plays of water pollinated plants, Revalis no, and Hydralo which grow in fresh water. MD. Several non-sea grasses such as Sostera, not, quayote plants use water FBR pollination, in, Magenty of aquatic plants such as water hyacinth. MD water lily, all ewers urged. Above lee level of water. MD. Are pollinated by insects or wind. Is an NDST of leland plants, in Valp. Snercolinae, All E female FLC and reach Lee surface of water by Lee long stalk on the male wars or pollen gr. Ints, we released onto the surface of water. They RCA PSC Ely Bioterra Currents Figure 2.11, Soriothum event uir, CH the Remley 1L01, RS, and E the stigma. In another group of water-pollinated plants such, S. Seeger, says female flowers remain submerged in water. N. TLLE pollen grains are released inside the water, pollen grains in many such species are long. And bond like in Lee RCMec PSC Ely inside Lee water, some of them reach Lee STIL, ond, chief pollen, ion. In rest of the water-pollinated species pollen gr. Ints, reprotected from wetting by, mucilaginous commernd. Both wind and water-pollinated uvrs are not very colorful on CB not produce nectar, WMT while DB the Rioson for TNS. Figure 2.1 LLLHLLHLL, LHLLLHLLLHLLLHLLLHLLLHLLL, LHLLLHLLLHLLHLL, LHLLLHLL. LLHLLHLLLHLLHLL, LHLLLHLLLHLLHLL, LHLLHLLHLLHLLHLLHLL, LHLLLHLL. LLHLLHLL, LHLLHLLHLLHLLHLL, LHLLHLLHLLHLLHLL, LHLLHLLHLLHLLHLL. Exclamation point exclamation point exclamation point exclamation point female flower B male flower of female flower biology M majority afloat Kring PL NTS Ustga range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds and hummingbirds and bats are the common pollinating agents. Figure 2.1 pounds. Among the animals, insects, particularly bees are the dominant biotic pollinating agents. Even larger animals such as some primates, lemurs, arboreal tree-dwelling rodents, or even reptiles gecko lizard and garden lizard have also been reported as pollinators in some species. Often flowers of animal-pollinated plants are specifically adapted for a particular species of animal. Majority of insect-pollinated flowers are large, colorful, fragrant and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flowers by color and/or fragrance. The flowers pollinated by flies and beetles secrete foul odors to attract these. Pollination by water 1 and Vallisneria, B insect pollination animals. To sustain animal visits, the flowers have to provide rewards to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting the reward as from the flower, the animal visitor comes in contact with the anthers and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in animal-pollinated flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe places to lay eggs. An example is that of the tallest flower of Amorphophil. Just the flower itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant yucca, where both species moth and the XII, Pruigpticinil flower and piemill plant cannot complete their life cycles without each other. The RROH reposits its eggs in TLLE locule of TLLE ovary. In the flower in tum gets chaselinated by the RROH. The V of the rust core out of the eggs as the seed store to develop in WHJ. GCU observe some JL lowers off the fowl, RGP arts or ongokers AOA laj, IE to go, ku.r mango, peapoy, core, onder, papago, gnoon, laban, cotton, taboa, rose, lemon, EUO, TLG sus. Banana TRG to find out we CHO namo LS alsultum and WL lethertheg could be pouro tortiul root to poaching thugub zero chle flowers or a few dogs on at different massive chle dog. You could axorg to CW Hertler S Ancor Aedion in the chlorator, street, CSFA.O or to the A, NIM, LL, to low TC, sitsit. Korafu, IJ Abzero if Angoft low, ciders come in contra at WH the antlers on a Tle Street 8 miles on 9 Sudo, cider S Canel, Inga Brut P, Nation. Mang insects mag consume polar the nectar without BMG, RG abrupt po ui na TLL, SLLD fire ojo isleters are referred to as po unfned grabbers, yao mag or mag not biaj, i.e. to identify the po uro torsmgao. Swell 9 and jogger efforts, outbreeding devices, majortl, UFUV, MNGPLMTSPTT, Duche template, didewers and pollen grains are likely to come in contact with the stigma of TTEs or FLX7 or O. N-inude self-pollination result in inbreeding depressing. F10NNG plants have developed many devices to discourage self-p, lination and to encourage cross-pollination. In source species pollen release on stigma reoptivity are not synchronized. Either the pollen is released or the stigma becomes receptive, or stigma becomes reoptive, in rutger or the release of pollen. In soar other species the ondher and stigma are placed at different positions so tratly pollen not core in contact with TLLE stigma of TLLESR flower. Boli TLLE's revices prevent autogon. The THID device to prevent inbreeding is self-incompatibility. This is a genetic enhancement and self-pollen from the soar or other ewers of the SAR plant from fertilizing the owls by inhibiting len germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the Reproduction C unisex ewers, if both male and female flowers are present on TLLESRPLNT such C aster and Myzermochiaus. It means autogon but not jetotgon when said, aryl species such as papaya male and female TLOARS are present on T11 errant plants. Each PLMT is either male or female diacle. This condition prevents both autogamy and jetonogamy. Pollen PLS Oltankuon. Pollination does not guarantee the transfer of the right type of pollen-compatible pollen of the SARA species as the stigma. Often pollen of the wrong type either from other species or from the SMNEPLMT if it is self-incompatible. Also lart on the ST section knob, the pistil has the ability to set the pollen whether it is of the right type compatible or of the wrong type incompatible. If it is of the light type the pistil accepts the pollen and promotes street pollination eventually. A BD biology antipodal polar nuclei longitudinal section of a flower SH on growth of pollen tube C figure 2.12 a pollen grains germinating on the stigma, B pollen tubes growing through the style, CLS. Of pistil showing path of pollen tube growth, D enlarged view of an egg apparatus showing entry of pollen tube into a synergid, E discharge of male gametes into a synergid and the movements of the sperms, one into the egg and the other into the central cell leads to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components in the interactions leading to the recognition. Followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores figure 2.12 up. The contents of the pollen grain move into the UX1 FAIUT unicyclic inflow UIT, GTAC P, LLE and lubitlil tube G, R, TWS through the tissues of those stigma and SL4E and reaches the ovary flagure 2.12b. C. Uoglu recall that in 01116p wits pollen grains shadathlio, L, Ixloclician agetity, E cell and a generate cell. In such plants the G, narrowly cell cu vides and phi, zmstetu milga, retsdirtn the grothoff and lube in the stigma. In plants which she len in the three-celled condition pollen tubes carry the two male garetes from the beginning. F. Jill and luballer reaching the ovary enters the ovule through the micropyle third and enters one of the synergals through the phloem, pore, lose figure 2.12d. E. Many recent SLUCUES have shown Th. L1 liforma pora to S present DAL the macrop or part of the synergals G tides the entry of len tube. AUTHZNLS from len proslian on the stigma until pollen tubes enter the owl together with air, L to as palin pislilter, CU on. As pointed out earlier len PD stilteraclean is a dynamic process involved palin recognition till load by pyrrolian or inhibition of the pollen. The knowledge gained in THLS area which D help the plant breeder in manipulum clen pistil interaction in incorporated, LLQ, 3 libel island, lias lojul L sired hits 1 CLS. LT Jucanaoi studied len lion by cust, Gseni pollen from ewers such as Piachik BCT balnancloniklona. Slicalcana DRQ, of sugar salutum about I0%, all are about I0th news observe the slice under the low power lensed microscope. U are lil, Z litto C pollen tubes CO link out of pollen 6 ounce, S, L ho shalorn in the chapter on plant precon chapter 9. A Breaklar is interested in crossing Clilforenal species and Otten genera to combine C, B cerebral CH mactors to produce C01111113RC3LLY superg, RV3 retides. A. Rtlclalhibraldesaglan is one of the jock approaches of crop improvement program, in such, CROSSTNGEXPERTMENLSLLSLMPOCTO, Chamawan.e Shurlhal only the desired P, len gr, ins are used for Paul limb lion and the stigma is prodded from continent 3 lion from unwanted P, len. This is achieved by emascula yuan and bogging techniques, if the female parent bears bisexual flowers. Removal anthers from the borer bud before the anther dehisces paraphore, EPS is necessary. This step is referred to as emasculatlan, emasculale, holopsio aired with a baga suitable sire. Genera Dinocola butter paper to preanduntum, lionettes sti, JNA with unwanted pollen, this process is called bagging. When the stigrin, D-bag, or attains recept, yiddy mature pollen grants collected from anthers of the male porin or dusted on the ugmo. ND the dowers are bagged and the fruits allowed to develop. If the female porin produces unis, Ru 31 flowers, there is no need for emascula yuan. The female flower buds are bagged B, LR the dowers open, when the stigma B ums receptly E, pollen yuan is courted out using the sired pollen and the flower reb, ged. After antengone of the synerg, CTS, the pollen tube releases the TO gametes into the cytoplasm of the synerg, court. One of the male garetzmves to 3 LDS tile egg cell fuses with its nucleus till its completingly synchrony. This results in the formation of a diploid cell the zygote. The other mole goradens towards the two polar nuclei located in the SEN1 RAL cell and fuses with them to produce an implicit bromazy endosperm nucleus BEN figure 2.13. As this involves the fusion of 3 haploid nuclei, it is termed tripler fusion, since T types of fusions. Synchrony on triple fusion tar1, E place in an embryo sac. The phenomenon is termed double erdelsat DN. An inkleak to fid, NNG plants, the central cell after it triple fusion B the bromazy endosperm cell PEC ondra ellipsintothenosperm while physigodate, ellipsinto and MXIO. Da, ULQLLWB phi, 2.13 FL, released embryo say, show S zygote on prano, Rye endosperm nucleus BEN, BSLN embryo, W. Ilopmont ln adlakotshon ln reduced S1 zeros compared to a following double fertilicity on. Events of endosperm and embryo development maturation of ovulus into seed S and ovary into fruit are collectively termed post-Ertl events 2, 4, 1 and inspects endosperm development precedes embryo development. WLJ the pearlery endosperm cell divides repeatedly and forms a triploid. SFFTFLTLT utocytocyclic IH Alfred, GPT, LNLS endospermasu. The cells of this tissue are filled with breser food male shiles and are used for the nutrition of tilde ping empsio. In the trost conmentip of endosperm development LPEN undergo S successive nuclear divisions to give rise to free nuclei. This stage all endosperm development is called free nuclear endosperm. Subsequently cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut Trachumi F3 milli 3J with is nothing but free nuclear endosperm M3D up of thousands of nuclei and the surrounding white chem helps the cellular endosperm. End sperm may either be completely consumed by the developing embryo, for example, P. Outnut, beans before seed maturation, or it may persist in the mature OADG. Castor in coconut and be used up seed germination, split OJM.toim SHDS of WI, BNS. 90 LDR.LLM, ILT, IGF, K in endosperm and EdCath find OxCTIGNIITR11.T on registered sign. Sperm as pers SUNL NCERT S1.01.T, LT. TFT, M, U 2.4, 2 liters. N1B, Yo embryo develops at the macroblarand off them sac where the zygote is situated. Most zygotes divide only after cerdal normant of endosperm is spawnered. This is an adaptation provide a short nutrition to the developing embryo. Though the seeds differ section UI, the early stages of embryoidal development embryogeny are similar in both monocotyledons and dicotyledons. Figure 2.13 depicts the street 3 Jes of embryogenic dicotyledonous embryo. The zygote G, ves reset to the proembryo and subsequent UI of the globular, heart olate third mature embryo. Ple dicot USC 2.1 consists 10 embryonal alls and 2 cotyledons. The portion defcuso 2.14 embryonal DS abalu the 1,1-diutile, LNS is the aplutyl, which len-80s with thepilmanule, R stem tip. The E-tangential portion below the level of cotyledons is hypocotyl that terminates H its lower end in the radical or root up. The loot tip is coeric, L with a root cap, embryo, of monocotyledons figure 2.14b posse, only one cotyledon. In the grass family the cotyledon 1, called scutellum that is situated towards one side odoral of the impsional OXS. At its L0101 rend the embryonal 3 ZIU the LBL. 81,010 GY radical and root cap enclosed in an undifferentiated sheath called colearhulsa. The portion of the embryono lies above the level of attachment of scutellum is the epicotyl. Epicotyl has a shoot up 8 LMD a few leaf PL and dradia enclosed in a hollow foliar structure the coleoptile. Sole, a Jew seeds in water SLJ of wheat, MNJZE, Ps DLID, Ps 9 roller D nu Q Ernie 9 L lieutenant. Then split the seeds and of circ, Edior, as parts of the EML, go and the seed 2, 4, 3 seed in angiosperms. The seed is the phenoPLTXI active sexual reproduction 1 is often described as a fertilized owl. Seeds are formed inside fruits. A seed typically consists of seedoid S utilidon S and an ambulge, las eyes. The cotyledons figure 2.15 of the embryo or simple structures generally thick and S-all and due to storog. Of B, D reserves as in mass. Mature seeds may tannin albuminous or albuminous. Non-albuminous seeds have no residual and OSP3 RM as it is completely constant E, 1 Ewing ambulge. Development for example. P groundnut. Albuminous seeds retain a part of endosperm as it is not completely used up during ambulge. L development for example. Wheat, maize, barley, castor sunflower, RJ, occasionally in some seeds such as black pepper and teat. Remnants of nucellistide are also persistent IES residual persistent nucellus is TLLE pert sperm. Integuments of ovules harden as tough protective seed coats figure 2.15 up. The micropyle RELL3 ends as a small LL4 in the seedoid. This facilitates entry of seed, oxygen and water into the seed during germination, as TLLE seed matures. Its W8 content is reduced and seeds become relati, ely dry eye 0-15% moisture by mass. The general metabolic activity of the embryo slows down. The embryos may enter a state of inactivity called donany or if favorable conditions are available adequate moisture, oxygen MD suitable temperature. They germinate. As ovules mature into seeds TLLE ovary develops into a fruit that is, TLLE transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the IGD, elapses into the wall of fruit called pert carp. The fruits may be ochi as in guava orange mango etc. or may be dry as in groundnut and RRUSTMD. Etc. Many UITS-IE01 Organisms F for dispersal of seeds. Recall TLLE classification of fruits and TLL are dispersal mechanisms that you have studied in an earlier class LS there and G relati, onchi PBW number QJ non. Organ the number of seeds P isn't N fruage, in most plants by the time the fruit develops from the type. Other neuroparts tegmerid and FLDF, hoi, in a few species such as apple straw terry. Cashew etc. TLL ethylamus also contributes to fruit one formation. Such fruits are coiled false fruits Firi 2.15b. Most fruits hoi, ver D, elap only from the OIG and or called true fruits, although in RMST of TLLE species. Fruits are TLLE results of fertilization TLL are a few species. Cotyledons hoot apical merostem hypocotyl root axis root tip seed coat endosperm cotyledon, shoot apical merostem oleoptile at B figure 2.15 a structure of some seeds. B false fruits of apple and strawberry in which fruits develop without fertilization. Ch fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones and such fruits are seedless. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also seeds have better adaptive strategies for dispersal to new habitats and help the species. 81,010 GYT colonize in all areas I have sufficient fixed reserve yellow seedlings or non-shed until they are capable of photosynthesis on their own. The lured seed coat prol, as protection to the young embryo, Reproducts of SESU 3.1 Reproduction. They generate new gene combinations acting to 11 actions. Seed is the basis of our culture. D, HJ, aeration and chlormancy of nurture seeds A recrucy 31 for storage of seeds which can be used ASV. Adula outly RANCT also to rise crop in next seed. Sun, Ga-Nu iron 3 gene F a culture in absence of seeds or in presence of seeds which germinate straight away soon after F formation and cannot be stored. How long do the seeds remain alive after they are dispersed? Display. Exclamation mark. Odd again varies greatly. In a few species the seeds lose viability within a few NDNTHS, seeds of a large number of species live for several years. Sere seeds can remain or hundreds of years. There are several reords of aerial-jetable seeds. The oldest is that of a lupin, Lup, Nus arcticus excavated from Arctic tundra. The seed germinated into word otter on estimated reord of 0.000 years of chlormancy. A recent record of 2,000 years old viable seed is of the date palm. Phonix.cti pharaoh discovered during the archaeological excavation at King Herod's palace near the Dead Sea. After combating OBREFO count of sexual reproduction of JL.RU, EMG plants, T would be worth a temple 9 to CO. Trayan the enormous RQ, Rod, Jerry Copow, TG Opsimidlo MGPONTSBGOSK Yanknefao Enquest, UNS, HOM, ME99 Soj present LNN. Embryo sac HOM, MG embryo sacs cra present IFL and UL HOM, MG ULs cra present IFLN. Or homage GUN, SOJ present IFL ATGXCOLJL or HOM, MG. Flowers cra present on a traf and so on. Catco TNNK of some plants W, VDFRWTS contain a large number of seeds, or will JRUITSOJ 1 sud 9 org and each for UIT contain 1000 DS if TN seeds SMRS Dmicos FR roots of some POSage, I, sped.es sud as ROJXMDE01 DSTRT9A. Krogocene adiflux seed of FL Rus how large as the tree Iftlcusti Rodotope from Demo TTN seed. Human GB, Uions QJ seeds does edftqs tree proj JCE can go maglan G other XOJ ilpil LNLLID such a tiflux struck me can. Proj JR, E sud W9EB, Amasur the JOJs 2, 5A, IOMRXJs, UID Polidel Brio, WAI those seeds. In general are the products of fertilization, a few FLDAN and G plants such as some species of Asteraceae.MCT crosses. Have arrowed a special mechanism to produce seeds without fertilization called upomlixless1 at sfrll.it prod JIMW, threat ferti, salient called, thus. Apomixis is a form of Asculeproduction hat MMCS sexual reproduction. There are said, arrow ways of DE allotment of apomictic seeds. In source species TLLE diploid XL as IC, returned without reduction division develops into the embryo without fertilization. More often as in M, myc, T as in Matljo. Variety summity nuclear cells surrounding he sac start cling protrude into he embryo S, C, develop into he embryos. In such species E.CHOVUL C sildens many embryos, occurrence at roar ha 1 embryo in, seed is rolamd, pobjn bryony. Take up so seeds of OT, U9, U, IU11TM, ABR11TM, MGM because of DI, Jerry Lacies, U, ISLAF, from TED H, 1C, NLLLTNT, ML, VML, F, Josini, TDH, 1 Latwa 1 is less than 111 TG, NFIC Noid UE for Parnity Embrigason 111 T, JBJSA TU, DT, NCERT Vanu. Several are food and vegetable crops are being extensi aliku, uvated call uva una s as relrand ausline or east produku, TTY. One of the problems of hybrids is that hybrid seeds have to be produid, Y or, U the seed C, DL. TED from hybrids are sown B plants in the penile gate OND do not maintain hybrid chara, ors. Produ, Leonard hidden seeds is casu I and hence he costed hybrid seeds Beckert sponsi for the farers. These hybrids are made into apt miklo hero 1 as no segregation of characters and he hybrid progeny. Then he imers can clop on urine the hybrid seeds to raise new crop year after Joar and he does not have to buy hybrid seeds every year. Because of the importance at APM, is in hybrid seed industry ACTE research is going on in many laboratories around the world to unrares and he genetics at APM, DSN to transfer apomictic genes into hybrid varieties. Summary Fbware, RSME the SNT of sexual OCL yo yuan 1 N and SL asparus HLE, Androcturnians first, 1 and 8 of street AMNs represents the N3 reproductive org 3 and SMV1 Gino 1 urn. Consisting of page 1 S also represents Ilia female reproductive or RS, atypic her is blobic, LL.M, L-tel ROSP, RTS, Poles and Tains develop 1 Sledem prosper L. L8 La, 4W all layers Ilia Ebeldar Mi Shendotel Soldern MTDD high BJ.M, L the T, pattern SWTOWLD1 MMTCR, SP, R slum, coils of T aporo. Usuali, Ang1 and the center of the M1 prosper MGTWN Untragomito SISM1 crisporiginasts to form tetrads of Ruarosper. In LL 10 raw spores mature 1 NTO planta NS, pollen 1 NS repper, CNTS the Molly GA metaphyok G raration. Nepi, LLSN grans ha 2 loy W low outer LNEN 1 ner 1 NT1 na 1 he exile 1 SM, diop of sporipal has germ pores, polenta NS, may have 12, celljet. Avegat, ATLEL a generative set 11 or 3 cells, IA, Litoli, 0 or pres, ND1 NU, Y. Comma volus, unclair protst wintegmentals.nd.an opt, DNF411 cropile, the central SU1S the nucellus 1 NWHLC, H the uchosportum CB6N plothes. Full stop, a cell of the, arrow, WN the mo, op, remother suchvodes metend kalian 1 of, I the me, SP, res, the embryo aoheml gametophyllil. The Mutual embryo sac SS7, CMA, LANDIUDEAJ the MX, Rapabren, I1S. JF, 81010 GY the Agapo, Rallus Conslst, Eng of 2 synords on XII, L. AJ the E, and E3 multipod 3 LSLD Urloi, and DRE1S a we central olbutili 2 polk, Armiet. Pollination 1 SLE meclabalism to TRE, R pollen GRMNS from oli.knet, R2 OLES ugrana, pollinating E, NTS me either a EOO UKMND. Water or BBUK, Rumels. Pollen page 1 Sulant, Raction Vali.Relu, NTS on U, EL, Dinkoff pollen GRMS on the S-Digterniuni G the PO 11 on 2 enters the Ombro sock when oli pollen 1S compatible or pollen inhibition when oli pollen 1S LNC, or incompatible. Fbloden compatible pollination pollen GR01 NMLN, 1S on the S-Digterni the resulting pollen lube grow true of the style. E, NTE, RLE ovules BN.RU tie dished, RF. S2 mu G, amites in 1 of U, E summer DS pemis exhibit double fertilicil 1 on B, Us 2 fuson E.OE, NTS occur in EZCH embryo sac. NC, Ameli syngamy.mel triple fusion the PRCXL of these fus, ons, the diplo 1D psi, loi the triplo 1D pulmonary erdospore nucleus LN the primary endosperm cell. Zygote the, teleps into LE embryo U, Ebert ma, Rye endosperm cell F forms U, E endosperm tissue. Formation of endosperm always precedes D, ioperna dalvi umbrage. An LE telopm embryo passes U of different slias such LE pro embryo G1 above 3 I he, RTSH3 ped slias B, for maturation mature decadiletimus embryo Ms2 cotyledons. Melum embry CJN, 31 Watilia P. and Hypsidil embryos of monocotyledons Ha. Oda S1N cotyledon, after fertilization or ED, ellipse, UILI ovules to uplum low, a phenomenon C3 letapomix is 1S found in some sperms. Pa, Rucula, RLY and GRS it results in LEF formation of seeds Witilia at Fe, fertilization, apomic TS Houtse.Terled, mtiges in horticulture and agriculture. Some USP, RMS Prodwomoral 1 embryo in ILI 1 RC, L1HLS phenomenon LS called poly, Bryony. 1. NMNE ULPMTS of sperm mover in which telop, NT of male on fern, Lg, metaphyto take plaque, E. 2. Differentiate bet V, and row 1 Crisporo. Melmegosporoge, Nes 1S, which type of old cyan occurs E1TSNMNELE structures F formed at the E, ND of U, East 2E.E1.1S. 3. RLEF follow 10, Ross in LE correct developmental sequence, pollen GRMN sporoge, N, Usasu. Row 1 Crosspore Telrid pollen Motler cell MEG, amate spore, with, neat label diagrated featherpore 1SL ellipgalantis pornovule. 5. Walsrani on the Byronanis pork, Ilopranella fern, Lecarn, Fight 6, with a neat diagram and expl 3 inches LE7 old. 8. Nucleate nature of LE RNM garnet phyto. Xidiola peruachia Y3FLOWUI, G plion 7, what michas magnosoors can cross-pollination occur 1 and cleastog 3 rows flowers G 3 sons F or your .comswer. 8. Mention TVDS suits Jesse to Pru, NT self-pollination 1 and overs 9, WH, LLS self-U, inputability Y does self-pollination not lead to seed F formation 1 and self-incompatible spec 1S10. What 1S bag 1 ang teclum 1 cow 1S it useful in a PBNT breed 1 ang proger MME 11, what is plefus 1 oral where and how does it T, 3K place NMNE ilia nuclei UA, LN triple fusion. 12. Why do ho think you, E zygote 1S dorm, MTF for sometime 1 NFA, released ovule 13, differentiate between, a hippoc, odil, DF, coli BC, oliop 1 li IC, olian, saw cu.tegern.t. I testo, deeper, S. T on pot, CMP 14, YLS apple called F, LSEUITWH1CHPO, RTS of the OOF forms the UIT15. What is Me3 antibody? M. oscula uron when. Male. Why does a page 13 NL breeder employ 1s technique 16? If one can into a Partheno y true awilia application of growth substance which fruits would ho select to induce PMU, NOJP, Mel Y17. Pla 1 and the role of Tupatum L and the F formation pollen GRMWU18. What is Apomyxis. Mel, Hat 1s its import, %. R. Chapter 3 Human Reproduction 3.1 3.2 3.3 3.4 3.5 3.6 3.7 The Reproductive System. The Reproductive System, Gomatogenesis menstrua, Psyched for 2, Estion, NTZ Rantotten. Pregnancy on Embrich, NTC Develk, TMFTPC, UTR, Ration on Lactotten, as 2 or 3 re. Humans or said, Uala reproducing gong viviparous. The reproductive events in humans include phenylionophoretes genesis. That is, sperms in moles. And ovum in Fefluestingsferot sperms into leaf effluiginal insemination and fusion of mole and female garetas fertilization leading to formation of zygote. This is followed by formation on development of blastocyst and its attachment to the uterine wall implantation. Emphysolic development gestation and delivery of the baby parturition. You have learned that these reproductive events occur after puberty. There are REM-MK-able differences betlow, Lee reproduce CTIE8 and and in the female. Tower oriental sperm formation unlanus even in old run but formation of OWM says is in wearing around the age of 50 years. Let us examine the mole and feflui reproductant systems in Heinwell. The Molareplex ioclyal system is located in the petals figure 3.1a. It includes a pair of testes along with access oxyducts glands and the eternal genitalia. Human reproduction. The testes are situated outside the abdominal cavity within a pouch called scrotum. The scrotum helps in maintaining the low temperature of the testes to 2.5 degrees Celsius lower than the normal internal body temperature and necessary for spermatogenesis. In adults, each testis is oval in shape, with a length of about 4 to 5 m and a width of about 2 to 3 m. The testis is covered by a dense covering. Each testis has about 250 compartments called testicular lobules figure 3lb. Each lobule contains 1 to 3 highly coiled seminiferous tubules in which sperms are produced. Each seminiferous tubule is lined on its inside by 2 types of cells called male germ cells spermatogonia and Sertoli cells figure 3.2. The male germ cells urinary bladder 1, Revas deferens LLI, prostate. J. Penis urethra glands penis foreskin scrotum figure 3L a diagrammatic sectional view of male pelvis showing reproductive system vas deferens undergo meiotic divisions from the epididymis leading to sperm formation, while vasa efferentia Sertoli cells provide nutrition to the reedy testis germ cells. The regions outside the seminiferous tubules called testicular lobules interstitial spaces, contain small blood vessels and interstitial cells or Leydig cells figure 3.2. Leydig cells synthesize and secrete testicular hormones called androgens. Other immunologically competent cells are also present. Figure 3lb diagrammatic view of male reproductive system. Part of testis is open to show inner details. The male sex accessory to CTS include reedy testis, vasa efferentia, epididymis and vas deferens figure 3.1b. The seminiferous tubules of the testis open into the vasa efferentia through reedy testis. The vasa efferentia leave the testis and open into epididymis located along the posterior surface of each testis. The epididymis leads to vas deferens that ascends to the abdomen and loops over the urinary bladder. It receives a duct from seminal vesicle and opens into urethra as the ejaculatory duct figure 3.1a. These ducts store and transport the sperms from the testis to the outside through urethra. The urethra originates from the urinary bladder and extends through the penis to its external opening called urethral meatus. The neiham genially, re.iab.li mactup of sp3l tissue that helps in erection he penis to facilitate insemination. Thene, eject end of penis called Hegel 3 and S penis is covered by a loose fold of skin called orisl. The male accessory glands figure 3.1ab include paired seminal vesicles a prostate and paired bulbaretlu, LGL3-NDS. Secretions of His GLMDS constitute TLLE seminal 3.1 plasma which is NCH and fructose calcium and certain enzymes. The secretions of butyrythrol GL3-NDS also helps in lubrication of the penis. The female reproductive system consists of a pair of oviducts along with a pair of oviducts. Uterus served vagina and he external genitals located in PEL, 1C region figure 3.3 up. These ports of he system along with a pair of he and Y glands are integrated structurally and functionally to RT processes of ovulation, fertilization, pregnancy, birth and child care. Ovums are he PLI of male sex organs that produce he female garetodin and several steroid hormones Orion hormones. The ovaries are located one on each side of the lower abdomen figure B, each ovar yi about 2 to 4 mm and I connected to HPLC wall and uterus by ligaments. Each ovary is covered by a thin epithelium which encloses tile of me3 and stroma. The stroma is divided into two zones. A peripheral cortadide and an irregular medulla. Human reproduction figure 3.3 A diagrammatic sectional view of female pelvis showing reproductive system. The O1 ducts fallopian tubes. Uterus and vagina constitute the female accessory ducts. Each fallopian tube is about 10-12 in long and extends from the periphery of each ovary to the uterus. Figure 3.3b. The part closer to the ovary is the funnel-shaped infundibulum. The edges of the infundibulum possess finger-like projections called fimbriae, which help in collection of the ovum after ovulation. The infundibulum leads to a wider uterine fundus. Figure 3.3b Diagrammatic sectional view of the female reproductive system. IEI port. See the C. Duct collect and full in at 13th port of the CTLSTHMS Ruest. A narrow lurin. And it joins Lee uterus. Thuterus single melodus oisa T womb. The slupia of thuterus is like an immerted pear. It is supported by ligaments attached to the FGI wall. The uterus opens into through a narrow syrinx. The coti of the cervixes collect cervical canal. Figure 3.3b which along with forms the birth canal. The wall of the uterus ruue 13 layers of tissue. The external thin membranous permetric middle thick L3 jar of SM. Author R.U. Sclair. Myomet lum on dinner glandu. ML3 jar collect endomet lum that lines the uterine coti. Capital T H E E N C B R R E T N W N capital W L D E R G C E S C Y C L I C O Chun gesturing menstrual cycle while it exhibits STRC and G traction dirt and deli area of the baby. The female external genitalia include RRMS pubis, 13 bm ajkra 13 bm incraherin and clitoris. Fig 3.3 up. Mons pubis is a cushion of tatty tissue covered by skin and pubic H3 ire, the labia magixa or oshi rolls of tissue, which lend CBWN from the rons pubis and surround the V opening, the labia monoxa or po, red IDLDS of tissue under the labia mapra. The opening of the V is often covered partially by a membrane called hymen. The clitoris is a tiny finger-like structure which lies at the upper junction. See the D113 bm in or the urethral opening. The heron is often turned dirt in the first ooides interiors, however it can also be L, all, and BO. Subtle D info JJ or J, H, int, T on, 1 O ho iral tamp, NODJJ, E part, CXT on in some sports W, K. SIBO D, reading ding etc. in some women and men perso, STSEON after coitus, in fad, tre presence absence off of JMN, S not O. Reliable and utor of OGN sexual experience. HIMA, IRFPI, IO at CTIO, IA Funclian 3.1 LWLM3 IGL third is CH MAC10 stick of all female mammals. The NRIGLMDS are paired structures breasts that contain almeltular tissue melvbl imblafel. Thegl3ndular fich Brea Street is divided into 15-20 mammary lobes conulural clusters of cells called alveoli 3.4. The cells of alveoli secrete milk which is stored in the cavities or in C alveoli. The alveoli open into LL3 L tubules. The tubules of each lobe edge, intophanum duct, sen, aryl LWLM3 ducts joint to TBRM alolirator number ampulla which is connected to iactopharos duct GLU of which milk is sucked out. The primary sex organ is the testis in Lee males. MCT the ovomies in Lee females produce garetes. That is sperm smell OWM respectively by the process called gametogenesis. In testis Lee and T utrum germ cells spermatogonia produce sperms by spenerogenesis that begins at puberty. The spenerogonal sing spermatogony when present on the inside WALL of seminiferous tubules multiply by mitotic division ANCT increase in numbers. Each sperm 3 tegonium is diploid ANCT con 3 ins 46 chromosomes. Some of the spermatogonia collide. Primary spinerocytes periodically undergo meiosis. A primary spermatocyte completes the first meiotic division reduction. Division leading to lesion of two equal haploid cells called secondary spinerocytes which only 23 chromosomes ARCH. The second, 3 risperm 3 tocytes WL reargo TLLE second radiolic division to produce 4 equal. Haploid spenoids figure 3.5, what wild bed number of vomegosome and the spermo.tv, ls the spermatid SA retransformed into spermatozoa by the process called spermiogenesis. After spermiogenesis sperm He3DS become embed in TLLE seral cells and are finally released from TLLE seminiferous tubules by the process called spenloeuon. Spermatogenesis starts at TLLE age of puberty due to significant increase in the secretion of gonadotropin releasing hormone DNA FIS. W. E3.5 max sectional oil of a says, JC ionophorous tubule ENL, R fethyl GnRH. This, if you recall, is a hypothalamic hormone. The increased L's of GnRH then acts at the anterior pituitary and stimulates secretion of two gonadotropins luteinizing HOMDNEUI and F follicle stimulating HOMDNEFSH. UI acts at the lead G cells and stimulates synthesis. And secretion of androgens. In turn stimulate Lie process of spermatogenesis, FSHACTS and Lie cells ANCT stimulates. Plasma membrane flowy middle piece mitochondria energy source for swimming. Figure 3.6 structure of a sperm T energy secretion of some factors which help in the process of spermiogenesis. Let us examine the structure of a spen. It is a microscopic structure composed of a head, neck, a middle piece and a tail. Figure 3.6. A plasma membrane envelopes the whole body of spen. The spen head contains an elongated haploid nucleus, the anti-1-ier portion of which is covered by a cap-like structure, agrosome. The agrosome is filled with enzymes that help fertilization of the ovum. The middle piece possesses numerous mitochondria, which produce energy for the movement of tail that facilitates sperm motility, essential for fertilization. The human male ejaculates about 200 to 300 million sperms being a coitus, of which, for normal fertility, at least 60% spens must have nodal shape and size, and for at least 40% of them must show vigorous motility. Spens released from the seminiferous tubules are transported by the XII ducts. Secretions of epididymis, vas deferens, seminal vesicle and prostate are essential for maturation and motility of spens. The seminal plasma along with the sperms constitute the semen. The functions of male sex XII ducts and glands are maintained by the testicular hormones androgens. The process of fination of a mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cells oogonia or F01 med within each fetal ova. No more oogonia are formed and added after birth. These cells stop T division and enter into prophase of the meiotic division and get temporally AI tested at that stage, called primary oocytes. Each page 1-1Y oocyte then gets SM taunted by a layer of granulosa cells and then called the primary Fo11 figure 3.7. Number of these follicles to generate during the phase from birth to puberty. Therefore, at puberty only 60, 00080, 000 page 1-1Y follicles E left in each ova Y. The page 1-1Y follicles get surrounded by more layers of granulosa cells and a new theca and called secondary Fo11 The second Y follicle soon transforms into a tertiary I follicle, which is acti-eased by a fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and an outer theca externa. It is important to draw your attention that it is at this stage that the page 1-1Y oocyte within the tertiary I follicle grows in size and completes its first meiotic division. It is an unequal division resulting in the formation of a haploid secondary oocyte and a tiny first pola, body figure 3.8b. The human reproduction corpus luteum tertiary follicle. UWIUO, antrum graphian follicle secondary oocyte retatans bulk of the nutrient-rich cytoplasm of the primary oocyte. Can you think of any advantage for this? Does the first polar body born out of first meiotic division divide further or degenerate? At present we are not very certain about this. The tertiary follicle further changes into the mature follicle or graphian follicle figure 3.7. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The graphian follicle now ruptures to release the secondary oocyte ovum from the ovary by the process called ovulation. Can you identify major differences between figure 3.7 diagrammatic section view of ovary spermatogenesis and oogenesis? A diagrammatic representation of spermatogenesis and oogenesis is given below figure 3.8. Oogonia, spermatogonia, mitosis, differentiation, primary spermatocytes, chromosomes number per cell 46L. 1 fetally HH primary oocyte birth L, childhood prior to secondary spermatocytes puberty ovulation. Odex spermatocytes, differentiation spermatozoa at T adult, 23 4 reproductive UFB figure 3.8 schematic representation of a spermatogenesis, biogenesis 3. 4 IV menstrual cycle. The reproductive cycle in the female primates e.g. monkeys, apes and human betons is called menstrual cycle. The first menstruation begins at puberty and is called menarche. In human females, menstruation is repeated at an average interval of about 28-29 days, and the cycle of events starting from 1 menstruation till the NEXT1 is called the menstrual cycle. 1 ovum is released ovulation during the middle secondary oocyte, ovum. 2 IBT, FIJJJIID, IUMPR1 LTT, of each menstrual cycle. The major events of TLLE menstru 3.1 cycle are shown in figure 3.9. The cycle SU, RTS with the menstrual pH 3 SER when menstrual flow occurs and it lasts for 3-5 days. The menstrual OA results due to breakdown nil of Enkberger I3.1 lining of GLL uterus and its BLCX, D vessels which forms liquid true T cores out through the ONA. Menstruation only occurs to the released ovum is not fertilized. Lack of menstruation may be indicative of pregnancy. However, it may also be caused due to some other underlying causes like stress, poor health, etc. The menstrual pH 3 cell is followed by the follicular pH 3 cell. During this phase GLLE primary follicles in the ovary grow to be core. Fully MT11 re CLRIN follicle MD Sir Aminia asleep yanked, rel re, M of uterus regenerates GLL roth proliferation. These CH3 changes in TLLE ovary and TLLE uterus are induced by changes in the levels of pituitary and ovarian hormones. Figure 3.9, the secretion of human reproduction gonadotropins LH and FSH increases gradually during the follicular phase and stimulates follicular development as well as secretion of estrogens by the growing follicles. Both LH and FSH attain a peak level in the middle of cycle about 14th day. Rapid secretion of LH leading to its minimum level during the mid-cycle called LH surge induces rupture of Graafian follicle and thereby the release of ovum. Ovulation. The ovulatory phase is followed by the luteal phase during which the remaining parts of the Graafian follicle transform as the corpus luteum. Figure 3.9. The corpus luteum secretes large amounts of progesterone which is essential for maintenance of the endometrium. Such an endometrium is necessary for implantation of the fertilized ovum and other events of pregnancy. During pregnancy all events of the menstrual cycle stop and there is no menstruation. In the absence of fertilization, the corpus luteum degenerates. This causes disintegration of the endometrium leading to menstruation, marking a new cycle, in human beings. Menstrual cycle ceases around 50 years of age, that is termed as menopause. Cyclic menstruation is an indicator of normal reproductive phase and extends between menarche and menopause. 3. 5. Fertilization and impla. 1. Phtation during copulation coitus semen is released by the penis into the vagina. Insemination. The motile sperm swim rapidly past through the cervix, enter into the uterus and finally reach the junction of the isthmus and ampulla isthmic junction off the fallopian tube. Figure 3.1 pounds. The ovum released by the ovary is also transported to the ampulla isthmic junction where fertilization takes place. Fertilization can only occur if the ovum and sperms are transported simultaneously to the ampulla isthmic junction. This is the reason why not all copulations led to fertilization and pregnancy. PCR-TB1 TDL1 negapace. The process of fusion of a sperm with an ovum is called fertilization. During fertilization a sperm comes in contact with the zona pelluta layer of the ovum. Figure 3.10 and induces changes in the membrane that block the entry of additional sperms. Thus, it ensures that only one sperm can fertilize an ovum. The secretions of the acrosome help the sperm enter into the cytoplasm of the ovum through the zona pellucida and the plasma. Figure 3.10 ovum surrounded by favy sperms. 51. Membrane. This induces the umplelion of the rheotic division of the secondary oocyte. The second meiotic division is oocyte equal 3L mictra salts in the myelin of a second polar body myeloploid diionoided. Soon the H3 ploid nucleus of the spens on that of the OWM fuse together to term a diploid zygote. Homunc ramos mshu, G, be there in this E190 Terwan Rues to remember that the S, of the baby Rues been decided at this as itself. Let us see how. As you know, the chlorosaur pattern in the Hern Remley is XXR, that in the male is XY hair or all the H3 ploid garetes per 2 said by the female ova H3 veves. Crocrosaurs waris and thymologaretes sperms the sex chlorosaur culvia thyrxori hens. 50% of sperm CM, Ithix chlorosaur while the other 50% CM. Y the Y after fusion of the mole and femolgaretes the zygote would carry either XX or XY depending on whether the sperm cart NGX or a fertilized thialmalti easy jalo e carrying XX. Old develop in tofemal, Babi and XY old tonomale luilla lornra recut the crocrosornal patterns in CH3 5. That, SUIVA so, Encto, R, G it, S-cord 2S09 let the sex of the LXR BGS determine BG the jaller and not BG the mother. The mitotic division starts as the zygote LLOS through the SHRRDTS of the oviduct called cleavage towards the uterus. Figure 3. 2 enfants 2, 4, 8, 16 daughter cells called blaspheromeres, the embryo with 8 to 16 TD 11, so 0. Full stop, U11J1, 11 km figure 3.11 TR sport of ovum fertilicity 11 on and poso of ground Rombridge, Throbgef Trop 1, N tube. Hima, IRFPRIO at CTIO, I blastomeres is called A Angela. Figure 3 TTE, the morella continues to D, Trenon transforms into blastocyst. Figure 3. IG as it moves further into the uterus, the blastomeres in the blastocyst are arranged into an outer layer called trophoblast and an inner group of cells attached to trophoblast called the inner cell mass. The trophoblast layer then gets attached to the endometrium and the inner cell mass gets differentiated as the embryo. After attachment, the uterine cells divide rapidly and merge the blastocyst. As a result, the blastocyst becomes embedded in the ink metinum of the uterus, figure 3.11h. This is called LMPL, NTAUN, and it leads to pregnancy after implantation. Finger-like projections appear on the trophoblast called chorlong, VLL, which are surrounded by the uterine tissue and maternal blood. The chorionic villi and U10 tissue B are interdigitated with ARCH jointly FBRMA. Structural MCT functional WL.IT tetlo, real eloping ambulge, LF otis and mate malbody called J, Essential 3.12. The placenta facilitate the supply of gen and nutrients to the embryo and also rare carbon dicyiden, crec waste mate dals produced by the flow. The placenta is connected to the ambridge through an umbilical CCRD, which helps in the transport of substances to and from the embryo. Placenta also acts as an endocrine tissue and produces several hormones like human cholesterol, gonadotropin HCG, human placental kerogen HPL, estrogens, progestrogens, etc. In the later phase of pregnancy, a hormone called relaxin is also secreted by the ovary. Let us remember that HCG, HPI, and relaxin are reproduced in women only during pregnancy. In addition, during pregnancy, the levels of other hormones like estrogens, progestrogens, oral isoprolactin, T-hypsine, etc. are increase several F-holes in the maternal blood. Increased production of these hormones is essential for supporting the fetal growth. Metabolic changes in the embryo MCTM3 in terms of pregnancy, immediately after implantation. The inner cell mass embryo differentiates Fig. W. E3.12 the homophotos within the uterus. IEI into an outer layer collectic 2, and an inner layer collect N, cloderm, a mesoden scon.apems between the ectoderm and the ink1 These layers verisatile tissues RNS in multi needs to rendnect here tlatli ruler cell mass contain CERT, and cells head stem cells which have the potency tog, 1 rise to u the tissues and organs. I not are the other features of EML, gon cdia augmented onus montmif pay 9 non the human pregnancy lasts 9 months. Do go, new mong mont mal junior 01, G last and do 9 se edge, I and Scots find out, in human beings, Euler 1 month of pregnancy, the MFJ's heart is formed. The first sign of growing fetus may be noticed by LL stem to the heart sound carefully through the stethoscope. By the end of the 2nd month of pregnancy, the fetus develops limbs and digits. By the end of 12x first trimester, RMST of the major organ systems are forward. For example, the limbs and extern 3L genital organs are well CTE1 eloped. The first emergence of leaf setus and arms of HRN lehi act are usui obstinate in 5th month. By the end of 24 weeks 2nd trimester, the body is covered with fine hair, ILLDS separate, and eyelashes are formed. By the end of 9 months of pregnancy, the fetus is full-eyed 1 eloped and is reactive for delivery. The 1 average duration of human pregnancy is about 9 months, which is called the gestation pay, odd. The contraction of the uterus at the end of pregnancy causes pulsion delirioptheus. This process of delivery of the fberonus childbirth is called parter, yuan. Parturition is induced by a complex neuroendocrine mechanism, the sign 3LSFBR parturition ongonate from the fully D1 eloped fetus and the placenta, which induce mild uterine contractions called fetal Icoan reflex. This dangerous release of O-sin from the terminal Pi1 uteri, sin acts on the uterine muscle. And causes street roger U10 contractiles, which in TWN stimulates further secretion of oglossin, the sterilatory reflex bet. And the uterine contraction. N. O-sin secretion continues, resulting in R. And stronger contractions. This leads to pulsion of the baby out of the uterus through the birth canal parturition. Soon Euler the infant is 2-1 erect, the placenta is also expelled out of the uterus. W11 at DT go TNNL, TREDT, IDORS and JED to USDA. ORGL will all 3 FJGLDS of the female undergo differentiation during pregnancy and starts producing milk towards the end of pregnancy by TLLE process called lactatlin. This helps TLLE mother in feeding TLLE newborn, the milk. PRODUCEDUNNGTHEINITIALFEWDAYS, is called colostrum, which contains several mTIPDs. Absolutely essential to G1L persistence for the newborn babies. Breastfeeding during the INITI 3.1 period of infant growth is recommended by doctors FBR bringing up a healthy baby. Summary: CMS are sexually reproducing CMD ovipaxis. The male reproductive HTE system is composed of a pair of testes. Ilia IMLE sex accessory ducts. Ilia A, accessory G1 MDS and E, 10, RN, 31 gen, and T3 Lea. Each testis has about 250 compartments called tesserae, or lobules. On the lobule contains 1 2 Illyri highly C01 lead cell cyanide, Rouse tubules, HSU, tubule LS and LNS1D by spermatogonia, Melsordob C, Els. The sperma 1091 and 1 undergo MEO1, 1C divisions leading to sperm formation with lesser toli C, Els providing nitrogen to the div DLNG folds. The DLG cells Alice 1DU, E seminiferous tubule synthis 1 sed.mel secrete tesu cum homer.es.mdroj, NS. T1 ERM1 E external 1 SDP, NTSNL fem reproductive system cons 1 STS of a page 3 U of onus. A pair of oviducts a uterus of the external gen, N1T11.3 a pair of 1 GL, NDS, the LV, Retes PRCXL the famulosome some steroid hormones of MCMH, hormones. 0. Axton KLLL1 Klezel D1 FFE, Rentsta S of Delattalopment Ax embedded Elvis Troma and Elio Yid.CTS. Yule, Rusax fernicosary ducts, T1 E uterus Illyri layers namely pertmeter uromyometrium endometrium. The fern external gen, Rut3 Lea includes mons pubis labium major tarunar hymen clitoris. An Eli memory GL3 NDS me1 of ilia fern second 3 Ly sexual omacli, Resux, spermatogene cis results in ilia formation of sperms that are transported by ilia sexicosary D.CTS. Honori ML hum sperm 1s composed of a head neck a middle page 2 OELA 11. The process of formation of mature female gametes is called oogenesis. The reproductive cycle of Fae, ME primates 1s called menstrual cycle, mons.rulc class starts only after allowing S.1 renabuation puberty. Dirtnavulon only 1 ovum 1s retuost per menstrual clare, the clical CLN the RE, mel the uterus done menstrual tcalaxl D.Z by SL. Ilia tells of pit uitmi.aon, L0IQ, ITC, and homer.es after coitus sperms ax tr transported to ilia junction of ilia isthmus male axonpal. 3. Where ilia sperm Fae, RT1L1 says the ovum 1 ed 1 ang 2F formation of a diploid zygote and Li presence of X or Y chlomosome. Ilia sperm determine as Yule sex of the E, MB, Jo NLEZ91 to undergoes repeated meta 1, 1C div 1S1 on 2F form a blastocyst. Which 1S1 meter plummeted 1 annuli uterus resulting LN prenawi after 9 non of pregnancy the fully, well up, DFO2, 1S ready F or deli way. The process of Kulbeth 1S cod led PM urishan VH 1S initiated by a complex re-euro, Dukatnin. Ulfton cortisol estrogens, meloxytocin and winry G1, NDS differenti, i.e. done pregnancy onset mil, after CHLLD. B1RTH the newborn baby LS fed MLLK by the mother Clation Dunning the Mushel Fubanon of Tom. LER, SESC, LFLLN the BL, NL, S, AJ Hearn CMS reprod.CASEI sexu 3 LLYB Honanzar OVL Paris. 1V porous MOVL1 Paris C Fae, RT, Elisa U on 1S Ulhum CMS Xlert Walta, RN, 31D male on female Ls. Diplo 1D haplo 1D E zygote LSD1 plo 1D haplo 1D. IIEIAGFNLE process of re-insert ovum from a mature follicle is called geolation LSLRD. Ed by a hormone called H the fus 1 on of Rho, Leon RMJG, MESLS called FL. Fertilization takes place OLNZI, divides to F form which LS plan DLNU, RUS. KNL Eastenture which provides status gem colo 0 U on between fetus on uterus LS called 2. Draw LD3MN of RM1E reproductive system 3. Draw label DMN of fem reproductive system. 4. Write 2 major 6. Mikuans each of testis. Male owl. 5. Descript bilia structure of a simuiferous tubule. 6. Hull LS spermato BR1 ovuli to be the process of sperm 7. Name the H. Hormones on all.L regal 311 on of sp. RMA10 nes 1S8. D. Fu. E sperm 10 and sp. RMT. DIN 9. Draw a label diagram N of sperm 10. What me U. E major components of cell plasma. 11. What me ilium major functions of RM1E accessory ducts. Male 12. What is OOG abertet, F ACO ULL of nests. 13. Draw a label diagram of a section ILIRA OWJ14. Draw a label diagram of a GRF01L1 Claire 15. Name the FERC 11 ons of the F01 low 1 eng, O, Rapus Lu BE Doroth Neuro CL Osero D sperm TO1 LE Fluorobri, E16. Then Y true, statum animals, O, next each WSE statement to M, L, E true androgens are produced by Sordob cells. True false B spermato, I section A nutancinaro Sordob cells, true false C dig cells or Ford LNOJ. True false delayed XII sinless 1 cell regions, true EO, genes, S Tokes page 1 AOELN corpus luteum. True false F mens eucalciasis during pregnancy, true false FSL presero or absence of hyme, N1S not a reliable eutic, door of orgulity or sexual expertance. True F3LSE17, what 1S me, it's true, 3L cycle which H, Wilmer.es regulate menstru, 3L cycle 18, what 1S FWOT oral WH1D1H nonzol, DLN first an of FWOT oral 19. In ARSA 1 Edilio.o me often BL3 road F4 WLGB1 RTH to daughters 20, 21, can you E, XPLMNYU use 1S not correct how MXA, rel 35D by a human. Owl a month how MMY eggs do you thunk would haben rel say affilium oilier B1 RTH twide, and Euclid T wins would your dotcoms were affiliate T wins born were fraternal how IML. L eggs do you ili ink were rel say biolia owl of a fem 1 e dog which got buffalo 6 puppies. Chapter 4 Reproductive Health 4.1 Reprod T, Yen Ti1 JH Problems NDST Raw Tegis 4.2 Pop L, on Explosion Orbj BTRTH Control 4.3 Medica Altermation. Of Pren Psi 4.4 Sex VLBJ Transmitted Thesis 4.5 Ln 4 UOJ Hale learned about human reproductive system and its functions in Chapter 3. Now let's discuss a closely related topic reproductive health. How do we understand BGTNS term? The term simply refers to healthy reproductive organs we all. Normal functions, however it has a broader perspective and includes EFLDTI and 31.MCT so C3L. Aspects of reproduction also. According to the World Health Organization WHO reproductive health means LLB, in all aspects of reproduction. That is, physical erosion 3LP and SOE 3.1, there are associated with people having physically and functionally normal reproductive organs and normal erotic 3L and B interactions among them and use sex-related aspects might be called reproductive healthy. Why is it anyone I can to maintain reproductive health and what are Lee Retlick SL3 can up to a G and let us examine TLLM. 4.1 F, PRDLICT1, HFAI Pro, ULFMIS and SNTCLW3TS India W as ANDNGST the first co-WL. Tries and TLLEV, oral to initiate ACT on PL3NS.MCT program res.it.anat I0 and 3 level to at UI and total reproductive health as a social goal. These programs called TAMILI PLNing were initiated in 1951. Were practically assessed over the past decades in pro-ed program resuluring wire. Reproduction-related RS are currently in operation under TLLE Populi, R Ruya Reproductive on Child Health C, RCH programs. Creating almost 3LLOI1G people about various luckily and related aspects and providing facilities n. Support for building up a reproductively ally society are the major asks under these programs. With the help of audiovisual and the print media governmental and non-PE, rental agencies can V steps to create awareness around the people about reproduction related aspects. Parents, other close real ads 10 acres and friends also have a major role in TLLE dissemination of the above INF formation. Introduction of sex education in schools should also be encouraged to provide right information to the LJLLNG so as to discourage children from believing in myths and having misconceptions about sex-related aspects. Proper INF formation about reproductive organs adolescents and related CH and hygienic sexual practices, sexually transmitted diseases SID, AIDS, etc., would help people, especially those in the adolescent age group to lead a reproductively healthy life. Educating people, especially fertile couples and those in imaginable age group, about available birth control options. Care of pregnant mothers postnatal care of TLLE and DTLL in child, importance of breastfeeding equal opportunities toward emol and thefamol child, etc. Old address the importance of bngup socially conscio, healthy lilies of desired CZ. Awareness of problems due to WL controlled population growth, social evils like sex abuse and sex real addict thorough etc. need to take addict to and able people to think and free up necessary steps to prevent them and thereby build up a socially responsible and healthy society. Successful implementation of areas action plans to and reproductive health requires STRONGINFRASTRUCTUROLLACILITIE professional expertise on material support. These are essential to provide medical assistance and care to people in reproductive and related problems like pregnancy, delivery, STDs, abortions, contraception, menstrual problems, infertility, etc. Implementation of better techniques and new strategies from time to time. Are also required to provide more efficiency and assist and to people. Street tutori ban on amniocentesis. Offset al said, detonity on test be a sect on TLLE Chrome JSOI. LLP attempt TLLE amniotic fluid surrounding the D. LLP gembryotor sex determination to legally check incurring to male fee. Etisit.es. Must eat child immunization etc. are so program result merit mention in TLL's connection. Research on VRE HOS Repro. Tuckly and related. Are encouraged and supported by governmental and non-governmental agencies to find out new methods and or to improve up, LLL. The existing ones, do go, jail that say new oral contrasex WE for the gemel.es wa SDEOE opt. BGSC and STS at Senral, drug resiar constitute CDRI or Ludmau, and a better awareness. About sex-related matters. Increased nurture of radicalization steadily, eries on better postnatal care leading to decreased maternal. MCT infant mortality rates increased nurture of couples via small families. Better detection and cure of S1Ds. And overall increased radical facilities for 3LL sex-related problems. Etc. 3LL indicate impelled 1 FU, tactile health of the society 4, 2 po PTND shy and JTXP. Ocean, NDBTRCTIG, Oxfernal in the 13th century on 3LL round development in R10 US field C81 Ifakanu I employed Tequilo, UF life see the people. HD, increased health facilities ojong with better living conditions had an explosive impact on the growth of population. The world population on which is around 2 billion 2000 million in 1900 rocketed to about 6 billions by 2000. A similar trend was absent in India too. Our Popul tick which was approximately 350 million at the time of our independence reached close to the billion mark by 2000 and crossed 1 billion inches May 2000. That Z1 every sixth person in Lee world is an Indian. A rapid decline in CTE athl rate maternal mortality rate MMR and infant mortality rate PMR as well as an increase in number of people in reproducible age. Are probable reasons toward this. Through our RCH programs though one could be NNGCBW and the Popul 3 Cheyenne growth rate it was only marginal. According to the 200 census report the Popul 3 Cheyenne growth rate was still around 7%. That is, 17 1,000 year rate at which our Popul 3 Cheyenne could bubble in 33 years. Such an alarming growth rate could lead to an absolute scarcity of even the basic requirements. That is, FCO, L shelter, MD clothing, in spite of significant prowess made in TLLO's as, Tillara TLL League member was forced to take up serious measures to check this Popul 3 Cheyenne growth rate. The RMSD important step to record this problem is to rajbate smaller families by using vanous contraceptive methods. You light Hawani seen advertisements in the media as LLS posters bills etc., showing a happy couple little. T1.0 children with a slogan hamdalyamch tadui to RT, many couples mostly the young. Urban, IOOI Kingones Hale even adopted on one child norm, statutory raising of marriageable egg, of the female to 18 years and that of males to 21 years. Male incentives given to couples via small families are two of Liotler measures L3 can to tackle this problem. Let us rescue off the Comoran used contraceptive relatives, which help prevent unwanted pregnancies. An ideal contraceptive should be user-friendly, easily available, effective and reversible via no or least side effects. It also should in no way interfere via Lee said, UL drive. Desire. And or Lee sexual. Actively user. A wide range of contraceptive relatives are presently available which could be broadly grouped into the following categories. Namely anadderall TR additional, barrier, IUDs, oral contraceptives injectables implants and surgical relatives. Natural methods iadricon nucleophilic chances of them. MCT sperms reyoung, PEM1 capslanensis on such ret lot in which the couples either abstain from coitus from day 0 to 17 of the menstrual cycle when all 3 Cheyenne could be peeled. As chances of fertilization are Verit done THTS penat TT1S called the fertile penat, therefore by abstra 1N1 LLG from coitus done this penat. Conception could be prevented, with durable or coitus interruptus 1S another method 1NWHTCH the male partner withdraws H1 spen 1S from the vagina just before ejaculate 1 on so as to have conception on. Lactational amenorrhea absence of figure 4.LOJNCBM1BR mole figure 4.IBOJNCBM1BR femoral menstruation method 1S based on the fact that ovulate 1 on and therefore the cycle do not occur done independent of 1/10 lactation following, parter 1D1 on. Therefore, as long as the mother breastfeeds the CHTLD fully, chances of conception are almost nil. However, once method has been reported to be effect one very only up to a milk one mumpanid of SDX months followed in parchunton. As no medi one measure de says are used in these methods, side effects are almost null, chances of bachelor. The wafts methodarial sock, in barrier methods of one and sperms are pre-entered from physically meeting WTTH the help of barners. Such methods are available for both males and females. Condoms, Fleur 4.1ab are barners made of one rubber latex sheath that are used to quarthapen once the male or vagina and serve IX1 and the female. Just before co-1 so that the ejaculated semen would not enter into the female reproduct, IV tract. Thus can prevent conception. Trod, TS a popular brand of condom for the male. Use of condoms has increased in recent years due to TTS adit on a benefit of protecting the user from contracting SIDS and AIDS. Both the male and the female condoms are disposable, can be self-inserted and thereby privacy to the user. Diaphragm sen, Eichel caps and alts are also barners made of rubber that are one send one anti-OTHF for the reproductive tract to cover the cervix dun caudus. They prevent conception by blocking the entry of sperms through the cervix. They are reusable. Sperm-1 gel creams, gels and foams are usually used along with these barners to increase their contraceptive efficacy. Another effect one being popular methods through softened uterine ISIS IUDs. These says are one inserted by doctors or nurses and the uterus through vagma. These ultrauterine devices are presently available Steph 1 Gear 4.2, C Upper T0U1 non-medicated RU DCG, lips loop. Copper released IUDs cut, Q7, Multlo 375 and the hormone released IUDs Progesticert, LNG 20 Fleur 4. 2.1 UDs 1 increase of phagocytosis of sperms WTTH1 and the uterus and the cutons released suppressed sperm motility and the first capacity of sperms. The hormone released IUDs 1 and did 1 on make the uterus unsuitable for one planted 1 on and the cervix hostile to the sperms IUDs reptile contraceptives for thefernales. Reproductive health who want to delay pregnancy and or space children. It is one of most widely accepted methods of contraception in India. Oral administration of small doses of either progestogens or progestogen-estrogen combinations is another contraceptive method used by the females. They are used in the form of tablets and hence are popularly called the pills. Pills have to be taken daily for a period of 21 days starting preferably within the first 5 days of menstrual cycle. After a gap of 7 days during which menstruation occurs it has to be repeated in the same pattern till the figure 4.3 influence female desires to prevent conception. They inhibit ovulation and implantation as well as alter the quality of cervical mucus to prevent retard entry of sperms. Pills are very effective with lesser side effects and are well accepted by the females. Saluli, the new oral contraceptive for the females, contains a non-steroidal preparation. It is a once-a-week pill with very few side effects and high contraceptive value. Progestogens alone or in combination with estrogen can also be used by females as injections or implants under the skin figure 4.3. Their mode of action is similar to that of pills and their effective periods are much longer. Administration of progestogens or progestogen-estrogen combinations or IUDs within 72 hours of coitus have been found to be very effective as emergency contraceptives as they could be used to avoid possible pregnancy due to rape or casual, unprotected intercourse. Surgical methods, also called sterilization, are generally advised for male-female partner as a terminal method to prevent any more pregnancies. Surgical intervention blocks gamete transport and thereby prevent conception. Sterilization procedure in the male is called vasectomy figure 4.4 or vasectomy figure 4.4b tubectomy. NCT that in the female tubectomy, in vasectomy, small part of the vas deferens is rendi veterlite. Up tlu roof. A small incision on lee scrotum figure 4.4 aware, s in tull, ectomy, SNU 11 pm of the fallopian tube is rem figure 4.4b or tied up tlu roof, small incision in the abdomen or through the gin. All these techniques are highly effective but their reversibility is very poor. It needs to be emphasized at the selection of suitable contraceptive relic. MD its use should always be undertaken in consultation with liquefied medical professionals. One must also remember that contraceptives are not regal 3 are requirements FBR the maintenance of reproductive health. In fact TLE are practiced against a natural reproductive event that is, conception pren, NCERT. One is forced to use these methods either to prevent pregnancy or to delay or space pregnancy due to personal reasons. No doubt the widespread use of these methods have significant role in checking and control LTOWTHFP, purulation. HDA their possible effects like nausea, dorninal pam breakthrough bleeding eye alarms to bleeding Goravan Bray, street cancer. Though not very significant should not be totally ignored. 3 JY, JIDJCA1, TLOLMJN, 11 OPPLL Luna CYMTP Lunton Sheol or Luntary termination C pregnancy or full tennis called Media 10 Luna Yuan of pregnancy M1P or induced abortion. Nearly 45 to 50 million MLBs, reparent in, year LL or the old which.ACCLTS2L5TLL of TLL E1 number of conseilled. Prenum season. A year, obviously, MT Fes, S, ignificant Rollindecker, SMG the P, population low it is not meant for that purpose. Whether to, accept leg use MT or not is being greb, ted upon in many countries due to Aro Shiru, unethical. Religious. And Soci 3.1 issues and 01 edinit. Government of India legalized MTP in 1971 v. Lits or strict conditions to .oid. Its misuse, such restrictions, real elderum are important to check indiscriminate, NCTE legal female foticides which, we reported to be high in India. YMTP obviously the answer is to get rid of UNW and aid pregnancies either due to CSULWL protected intercourse or failure of the contraceptive use and coitus or apes. M1Ps are also a Senti 31 insert NC as is where continuation of pregnancy could be harmful or even fatal either toddy and there or tot betas or tot H. M1Ps are considered relatively safe during the first trimester that is, UPTO 12x off pregnancy. Second trimester, abortions or skier. One disturbing trend observed is that, majority of the MTs are performed illegally by unqualified quacks which, not only on soy but could be fatal too. Another dangerous trend is the misuse of amniocentesis to determine the S of the unborn child frequently. If the fetus is found to be female it is followed by MTP this is totally agmst what is leg, L. Such practices should be oided BAU CELIs or D, 3 Ngeros bot left BR the young mother and the fetus. Effective counseling the need to. Ulwusipi faith avoid WL protected coitus and risk factors involved in leg 3L abortions as the U.S. Rural Health Corps facilities will reverse the Remian W1 Heal, B trend 4.48J, XT Jow. Y Tros LMI1D8TD, diseases or infections which are transmitted TLU said. Rural intercourse or elective G called said, UUG transmitted 3 says STD or venereal diseases VD or reproductive tract infections RDI. Gonorrhea syphilis geral herpes clausis geroodlewort trichomoniasis, hepatitis B on co, RSE. UERMS discussed infection in the recent years, HIV leading to AIDS or sorvely corn-JN STDs. ARN, LNGTLLEs, HIV infection is NDST dangerous and is discussed in detail in Chapter 8. Some of these infections like hepatitis BN. HIV can also be transmitted by Shangoff injection needles surgical instruments etc., with infected persons. Transfusion of blood, XL or from uninfected elder to the fetus too, except for hepatitis B geroidal herpes and H infections. Other diseases are completely curable too detected early and treated properly. Early symptoms of stuff these are minor and include itching. Few discharge slight pain SV, etc. In the geral region, infected fernals may often be asymptomatic on tense. May remain undetected for long, absence or less significant symptoms in the early stages of infection on the social stigma attached to the S1Ds. Digitally infected persons for ping for early detection and proper treatment, this could lead to complications later. Which insular pelvic in 1L emeteritis fit abortion stillbirths ectopic preemies infertility or even cancer of reproductive tract. STDs are a major TLUE to a healthy society. Thyroid prevention or early detection and cure of these diseases are given prime consideration under the reproductive health care programs. U persons are vulnerable to these infections. Their incidences are reported to be very high among persons in the age group of 15-24 Gen, RS the age group to which you also belong. Don't panic, Prevention is in your hands. You could stay free of lace infections if you follow the simple principles given below. I avoid sex with unknown partners, multiple partners. 2. Always use condoms during coitus. 3. In case of doubt, go to a qualified doctor for early detection and get complete treatment diagnosed with disease. 4. 5. NFF or THD. A discussion on reproductive health is incomplete with the mention of infertility. A large number of couples all over the world including India are infertile, that is, they are unable to produce children in spite of unprotected sexual cohabitation. The reasons for this could be monophysical congenital diseases, drugs, immunological or even psychological. In India often the female is BL3 mect for the couple being childless, but more often than not the problem lies in the male partner. Specialized health remills fertility clerics etc. could help in OSIS and correct and treat NC1. Scredively setasursa and emblalis couples to hail children, Haryo or where such corrections are not possible. UE couples could be assisted to hail children through certain special techniques. See oily alikncnjn as a 56th reproductive technolog 1 SAR1. In utero fertilization PVF fertilization outside the Bxli analysis similar und alliance as that in the Bxli F01101, D biopsioscience for E1J is one of such methods. In this method P, purely known as test tube bibiprogram row OA from the wife donor female and sperms from the husband donor male are collected and are induced to F-form zygote under cerulated undidionts in the laboratory. The zygote or early embryos with up to 8 blastomeres could then be transferred into the fallopian tubes if gote intratan, plant transfer and embryos with more than 8 blastomeres. Into the uterus NJT intrauterine science for to complete its further development. Embryos F formed by in vivo FERT WSAU on fusion of gametes within the female also could be used for such transfer to assist Leos females who karmic conceal. Transfer of .M of one collected from a donor into the F3 Lop3 and 2 gift gamete intrafallopian transfer of another female who cannot produce one, but can provide suitable environment FBR fertilization MD further development is another method attempted. Intracytoplasmic beam injection PCSI is another specialized procedure taught from an M10 in the laboratory in which a sperm is directly injected into TLL ovum in fertility cases either due to infertility of the M3 partner toins, or at the female or due to very low sperm counts in the ejaculates. Could be corrected by a Ruke, Lonsemina U1AI technique. In this technique the semen elected either FRCM the husband CR Helchetan or is 3I wick, any introduced either into the vagina or into the uterus re-intrauterine insemination of the female. Options are many. All these techniques require extremely high precision handling by specialized professionals and pensive instrumentation. I heard these facilities are presently have 3I label only in a few centers in Lee country. Obvious little benefits is 3 affordable to only a limited number of people. Erosional religious and social factors are also deterrents in the adoption of Lee's methods. Since TLL the ultimate aim of 3 Lee's procedures is to have children in India have so many orphan and destitute children who be old probably not sir until maturity, W1 less ta 1, and care of, our laws permit legal adoption. N. It is as yet one of the best melect tar couples looking for parent fox. R. Summary reproductive healing. First to a total well-bet 1N aspects of reproduction that is, 5Z, Moyuan, 3L. Behat towel social, Arna 11 on was U, EBRSD nation only were to ULL Shiat, Moors action PLCMS at National Tatel to MDS at a Reproductive, Elahi Mili ISO 180. Counseling and creating awareness among people about reproducu or G, MSA Dolasoral OE.mel Sia-led Cooljes. Safe.mel hygienic sexual practices sexually consent D1 seizes S1Ds including AIDS etc. LS than Ro, ZISTSP Tawas Reproductive Health Pro IMG Medical Facilities CME2 Ilia problems like menstrual irregularities. Fren, Cyrel, 1 Edospiats Deli Y, Medico Tormalnet 1 on of PR, STDs, Birth Control 1 NFE, RL 111 type. Postnatal child.mel maternally, meant 1S1 port, 3NL aspect of Yule Rapid DCTI on, CHLLD Health C. Programs, and OO all in Proane, ND1N Reproductive Erhi 31 all has place 1N or country as indicated by redual mate, RN, 31 1NFA, NT Mort, 3 Liddy rates. EML detection and cure of S1Ds tends to fertile couples etc. Impro OEDWLLF facilities better leaving ordiuans promoted explosive growth of population. Such a growth necessitated intense propagation of contracept femeliates. The moose can recept the options me FBLE now such as natural. Tragedy BMNE, R, IUDs page 1 us, Uljeclables 1 meter page 13 NLS CERT 10 Almali, LDSI, LVGH contra CPU, E, me not recom require, Nansefwari Prodi CTIE Haru TH. 1-1S forced to use ILI, M2AO at Pren C, MSIR to delay or space PR, medical term, DIN of PRY1S 2 grams, ELISA, L in our country. MLPSG, oily priorm to Gia ND all unwanted Pren, Noidi rapes Casu LREL 31 ion P. etc. As 1N cases V hen the continue 11 on of Pren C, Psychal BHC, Rumfalori.10, N fatal to EIU, Iliamu, or Ilia Fotis or Boal. Diseases are euctions transmitted sexu, 311 NTE, records me called sexually transmitted diseases CDs. PEL, TCLNFL Matori diseases IDs all birth in a me some of yule complications of ILEM. EMLY Dedic 11 on FAC 111 take better cure of ILE East I says, Awiding set, Sualt intercourse tilly on the mount multiple PI blurs. Use of cordons during me some of the sun PTE precautions to a 01D contractilk CDs, inability to consent.e or produce clulder, NEN, after 2 years of unprotected sexu, 31 co TT, din1s called inf, arulity. The mus miliads XII now to help such couples, in veto fe, aruis, din f followed by tr of, brio u, to jo f, mll, gil, iuitrml, s or rmth.r1s commonly one monojule test tu baby program. Point 1. Dot. Exercises 1. What do you ing, is you, at reproductive he 31st 1 and so 182 substretally aspects of reproduct el, which need to be in spec 1 al attention in u, present so, and mo 3, is XI, h calionia essa, rytn. SCB, laws y4, do hoiliang gullet reprod.cti vehia 1 n r calorie heads on pro.d, ltn ilia past 5, ye 3 rs if yes. Mentian some sch misav on proane, nt5, what are the sgo, sta, riasins for popul, lion ox plus 1 oral 6. Is u, use of contour a septi es juice lifted g reason 7, remo, t of, nodskar not bc, ons 1 dari, losc, on laraset moption. Y8, arn 1 uint.es 1s ever said, x determination 1s 1 n our country, is ui 1s nia slo, lernmet, nt. 9 sugsum f, LDS to dos first 1 faru 1 e couples to have cauldron 10, WH id me emis 1 has 2 L4K to pre.e, NL on contra CL, ing SIDS 11. Sta true false with explanation of abortions could happen spontaneously too, it true false be infertility ls defined as the inability to produce a viable offspring 1s ways due to abnormal liuesd, j, x1 analia fem ru pmtr. It true false c complete t act u1 could help another method of contraception, it true false decrete nawaori, nes about sex related peelsism affect the method to un pro.e reproduct el. Of you, people, true false 12. Koi tek of the follow up statements, AJ Serg 1 calreniliads of contraception pre-OE, NF. Meat formation be all sexually transmitted me completely curable C or peels A, re, IY popu, M contraceptives MNL 8U, E or women DHL. ET technique C, MBI Josmi ways TR atmosphered into ilia Yule, Russ. Unit 7 Genetics and Evolution Chapters Principles of Inheritance and Varioli Molecular Basis of Inheritance Chapter 7 Evolution. The work of Mendel and others who followed him gave us an idea of inheritance patterns. However, nature of those factors which determine the phenotype was not very clear, as these factors represent gene basis of inheritance. Untersland G. The structure of genetic material and the structural basis of genotype and phenotype conversion became LHE focus of Allenlian in biology for the next century. The entire body of molecular biology was a consequent development with major contributions from Watson, Crick, Nirenberg, Korana, Kornberg's father and son, Benzer, Monod, Brenner, etc. A parallel problem being tackled was LHE mechanism of evolution. Awareness in the areas of molecular gene lies, structural biology and bioinformatics have enriched our understanding of the molecular basis of evolution. In this unit, LHE structure and function of DNA and the story and theory of evolution have been examined and explained. James Watson Francis ISC Rick James Dewey Watson WCR, born in Chicago on April 6, 1928, born in 1947, he received BSc degree in zoology. During these years his interest in birdwatching had malvered into a serious desire to learn genetics. This became possible when he received a fellowship for Gradvale Slade in zoology at Indiana University, Bloomington, where he received his PhD degree in 1950 on a study of the effect of hard X-rays on bacteriophage multiplication. He and Crick discovered their common interest in solving the DNA structure. Their first serious effort was insulisfically, their second effort based upon more experimental evidence and better appreciation of the nucleic acid Lillerulver. Result, early in March 1953, in the proposal of the complementary double helical configuration. Francis Harry Compton Crick was born on Beach 1916, at Northampton, England. He studied physics at University College, London and obtained a BSc. In 1937, he completed PhD in 1954 on a thesis entitled X-ray diffraction, polypeptides and proteins, a critical influence in Crick's career. WCR, his friendship with J.D. Watson. Then a young man of 23, leading in 1953 to the proposal of the double helical SLRVC for DNA and the replication scheme. Crick KWCR, made an FRS in 1959, the Honovers to Watson with Crick included, the John Collins Warren Prize of the Massachusetts General Hospital. In 1959, the Lasker Award. In 1960, the Research Corporation Prize. In 1962 and above all, the Nobel Prize in 1962. Chapter 5 Principles of Inheritance and Variation 55.25.35.45.55.6 Mendel's LCWS of Inheritono LNL, Ertens of one gene LN Matansi of two gene sex. Dead motion. Mtion. Genetic disorders. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Or why a mango seed forms only a mango plant and not any other plant, given that they do are the offspring of toddler parents? Or do TLLE show differences in SOR of their CH3 characteristics? Honueller TAOI React why siblings sometimes look so similar to each other or sorters even so different? These and several related questions are dealt v. Lit. Scientifically in BNCH of biology known as genetics. This subject deals with Lee and Harry L. Threen says. As v. L. Is the variation of CH characters from parents to offspring. Inhert MCE is Lee process by which CH mactors are passed on from parent to progeny. It is the basis of heredity. The nation is the degree by which progeny differ from their parents. Humans knew from as early as 8000–1000 BC that one of the causes of VMATion was hidden in sexual reproduction. They exploited the variations that Renatter, LLLG present in the wild populations of plants and animals to selectively breed and select FBR organisms that possess desirable characters. For example through artifice 3.1 selection and domestication NASTRL. 1. A UDAEBA, VO, to D. 1. E. Tamar 011 FIIEI of wild cows. We have well-known Indian breeds, for example, Sahiwal cows in Punjab. We must however recognize that though our ancestors knew about the inheritance of CH characters A and D variation, they had very little idea about the scientific basis of these phenomena at W3S dirt in the mid-19th century that he had 3YW3S mode in the understanding of inheritance. Gregor Mendel conducted Hibbeldis 3 Cheyenne, Penritz on Jerome Pay 3S for 7U3RS 1856–1863 MD Prop, said the low WS of inheritance in LL Organisms. Durkin Mangtet, S. Investigations into Inheritance page 3 Thames at W3S for the first theory that SL3 to DC3 Lysis third mothemotic 3 logic or 3 to problems in biology. His expeller and shot 3 large S emphasize which gave greater credibility to the D3 ta that he elected. Also the confirmation of his inferences from E. Peffern, NTS on successive generations of his test plo NTS. From that his results pointed to general rules of inheritance R3 and being substanti 3 ted 3s. Del invested 3 ted CH3 R3 ters in the gene den pay 3 PL3 and T that remifusted 3 STO opposing TR. It's, for example, taller dwarf plo NTS yellow or green seeds. This 3 LLO, ed him to set up a basic FR rev, arc of rules governing inheritance. Which was EXP3 Reden Biloter S Scientist to 3 CCO for all leaders natural observations. N. The current player, tie inherent in them phys, Euro 5.1 note, NS of contrasting traits in P plant studied by Mendel conducted. Such artificial Palmalian cross-palm Cheyenne experiments Usmxlf 31 true breeding 3 lnes, a true breeding line is 1-3T. H3, Eng undergone continuous self P.LHNation shows the stable trait inherit MCEMD expression F or cellular lunar 3 Cheyennes. Mendel selected 14 true breeding Pay 3 PL3 and TD Meades as pairs which were simil 3 RE, except for one CH3 vector with contrasting TRAITS, semiotly contrasting traits selected V, airs renewed or angled seeds. Yellow or green seeds smooth or infloted pods green or yellow pods third taller dwarf PLMTS FIRE 5.1. T3 BLE 5.1. 1. 3 BLE 5.1, contrasting Fiat studied by Mendel in PTA, WON, Connell T3 Stin 11 Z. Seem the IJHTTH dwarf, 2, FCI, Auto JR looked at WH, T3, FTCJ, Pos CU on, V. 4, PDDSH Paific, 14 is less than 1 Steg I5, Podgar Green Lie 6, Siege Brown WM Jed 1 Seed Odor Yajeen. 5, 2 LFLLF, RN, NEL, O1GJ, and L. Let us like the example of one such hybridization experiment carried out by Menrel where he crossed tall and dwarf pea plants to study the inheritance of one gene figure 5. 2. He collected the seeds produced as a result of TLL is crossed and grew lean to genera plants of the first generation. This generation is also called the FWAA, progeny or the F. Menrel observed that all the F1 progeny plants were tall, like one of its parents, none we dwarf figure 15. 3. He must CTE Cernal 3R observations Torley all your pairs of traits he round that the F1 always resembled either one of the parents. And that Lee TRite of Lee all your parent was not seen in Tremenral then self-pollinated the tall F1 plants and to his surprise found that in the filial Generation soar of the offspring re DWART, TLLECH Moktertlat was not seen in Li F1 Generation was now expressed. The FIS1, TE 5.2 steps all IMK1 ang across all PN proportion of plants that re-dwarf re-1-4 of the F2 plants while 3-F4 of the F2 plants were tall. The tall and dwarf TR8s were Ioretica L to TLL or parental type. MD did not show ani blending. That is all the offspring were either tall or dwarf none re-in, between height Phi re 5.3. Similar results low, are obtained. Li Otler LR8s Tladi studied, only one of Li parental LR8s WAS expressed in TLLE F1 Generation while AT the F2 stage both the traits were expressed in the proportion 3, I. The contrasting traits did not show any blending at either F1 or F2 stage. Based on Li's observations, Mendel proposed that something was being stably passed down. Unchanged from parent to C, through the gametes over successive generations, he called these things as factors. Nowadays we call them as genes. Genes therefore are the units of inheritance. They contain the information on that is required to express a particular it in an organism. Genes which code FBR a pair of contrasting traits are known as alleles, that is, they are slightly different forms of the SOR gene. Use alphabetical symbols for each gene and the capital letter is used for Li TA it expressed. At Li F1 stag and the small alphabet V, junior Li Olier T nit, FBRE. Ample in case of the character of Hey Lieutenant. Tis used FBR the tall trait and TFBR the oral, and Tantera alleles C, I each other, hence. In plants the pair of alleles FBR height would be TT, TT or TT, Fig. E 5.3 DT3, MJ Na 11C represent A11 on of monohybrid. L. Cross Mendel also proposed that in a true breeding. Tall or dwarf P vanity the allelic pair of gene FBR height are identical or homozygous, TT and TT, respectively. TT and TT are called the genotype of the PLT while the descriptive terms tan and dwarf are the phenotype. What then while DB the phenotype of opant that heterogenop TT as Mendel found the phenotype of F1 heterocy, PE to B, acui like the T parent in appearance. He proposed that in a pair of disylo 3R factors one dominates Li other as in Li F1. MCT hence is called TLLE dominant factor while TLLE Olier factor is recessive. In TLL's case TFBR tallness is Kprin or Wiltmer TTBRDW orphness. TLL is res is sive. He observed identical BH, der FBR all the other characters trait pairs that he studied. It is cornient and logical to use Li capital MDLM lowercase of an alphabetical symbol to remember TLL's concept of domeral 3 in SAMD. Re 03 SCNS, do not use for WI and D roar dwarf because, you will find it difficult to remember whether T and D are alleles of the same gene character or not. Alleles can be similar as in the case of them, Xi, PST tan TT or can be disylo 3R as in the case of the heterozygote TT. Since the TTPL3 and T is heterozygous the junior genes ron rolling 1CH Mactor hyded is a monohybrid and the cross between TI and TT is a monolybrid pro. BFRC and the observation that the recessive parental trait is E.O, pressed without any blending in Li F, generation. Lol I can infer that when Li tall died and dwarf PL3 and T produce gametes by the process of meiosis. The alleles of he parental Po, are separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of alleles is R Mictorn process MD so TLLR is a 50% 0CH3 instead of a gamete containing Giebler allele. Is H3SF. EQ been verified by the results of the crossings. In this way the garetes of the tull TIPL3 and TS hail the allele and the garetes of the dwarf TT plants have the allele T. During fertilization the TV, alleles, from one parent say through the pollen and T from the other parent. Then through the egg RWLI tend to produce zygotes that hail one TALL and one T allele. In other voles the hybrids have TT. Since TLLE's hybrids contain alleles which express contrasting TR, it's the plants are heterozygous. The product ion C garetes by the parents the Phi, drama ion 0 of the zygotes the F1 and F2 plants can be understood from a diagram collectwomet square as shown in figure 5. 4. It was developed by a British one, geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible georetes are written on two sides usually the top row and left ulvums. All possible combinations are represented. In Bo, Estelo and Li SQU math which generates a square output form the pun square shows the parental tull TTFLS 5.4 male and dwarf TT female PL3 and TS. The gametes produced by them in the F1 TT progeny, the F1 PL3 and TS of genotype TT are self-pollinated. The symbols S and D are used to denote the female, re JL TD, TT, RC8, 1 geome, low. Icanted, 1 T, TT, TT1, 1 A met square used to order stored at Tibble Moor, Hibnt cross conducted by Mendel Bet. True Tol PL, N1 SI Trube T, LMG Dwarrel Plonels eggs and male pollen of the F1 generation. Respectively, the F1 PL3 and T of the genotype TT when self-pollinated produces georetes of the genotype TANT in equal proportion. When fertilization takes place the pollen grains of genotype they'll a 50% chance to pollinate eggs of the genotype T as LLs of genotype T also pollen grow, NS of genotype T have a 50% chance of pollinating eggs of genotype T, as well as of IIEIAG genotype T as a result of wrong fertilization, the resultant zygote can be of the genotypes IT. TT or TT, from the Punnett spread it is easily seen that 141 of the rhomtome fertilizations led to TT. 1-2 led to TTMD 141 to TT, though the F1 hectaria genotype of FT but the phenotypic character seen is all. At F34T1, of Li PL3 and TS.RT all where soar of Li may re TI while all years A re TT. Lemolge fit is not possible to distinguish between the plants with the genotypes Tita and TT, hence. Within the genotypic pair Tom Lyon CH Moctor TT all is expressed, hence Li CH Rector Tor T all, is said to dominate over Li all year 3 Lel Tor DW art, CH Moctor, it is Lias due to TLL as dominance of one character over the other that all the F1 are tall though the genotype is T and in the F34 of the plants are tall though genotypically 1-2 are tandemly 1-4, are TL, this leads to a phenotypic ratio of 3-4 tall, 1-4 TT 1-2 TT and 1-4 TT, that is, a 3-1 ratio, but a genotypic ratio of 1-2-1. The 1-4, 1-2, 1-4 adiufit, TT, it is mathematically condensable to the form of the binomial X/Xi that has the gamete-sparing genes stored in equal frequency of 1. Full stop. The expression is extended as given below, 1-2T1-2T, 1-2T1-2T x 1-2T1-2T1-4TT1-4TL Mendel self-pollinated the F plants and round that dwarf F plants continued to generate dwarf plants in F8 and F4 generations. He concluded that the genotype of the dwarfs was homozygous TT1 at Dugu TLVNL, he while D-90T had. He said F, Pounododo tal, F, pant from the preceding paragraphs I was clear that though the genotypic ratios can be calculated using mathematical probability, but simply looking at the phenotype of a minute trait, it is not possible to know the genotypic position. That is, for example whether a tall plant from F1 or F2 has Tita or TT composition cannot be predicted therefore. To determine the genotype of a tall plant at F, Mendel crossed the tall plant from F2 Villiers DWR3 and T. This EC3 led a test cross. In a typical test cross an organism P plants here showing a dominant phenotype on whose genotype is to be determined is crossed. Villiers as CE parent instia D of self-crossing, the progenies of such a cross can also be analyzed to predict the genotype of the test organism. Figure 5.5 shows the results of typical test cross where violet color U where WJ is pronounced over white color flower W. U sin, IRL T square, TJ to JRV, J what the nodure of Q spear, GFO test cross, W1 latra TODJJ. U9L2 use at 929 and Upsajlves cross, 001 U9 We09 NROT Diaphonity on for O test cross. M1RE01, all F10, RAR FTCL at, MIL NMODCM and R1ACJ, Mud half of tile LLOMWT violet ball CTU, DUUR, WLL, U, Alorwamdralaladir. G, based on his observations on Rana, Abra crosses Mendel prop, said lawyer general rules to consolidate as one understanding of inheritance in Rana, Abt crosses. Today these rules are collect the principles or laws of inheritance, the first law or law of dominance and the second law or law of segregation. 5.2 Law of dominance ICH factors or control B, Y discrete WLITS collect factors 2 factors occur in pairs. 3 in a dissimilar pair of factors one member of the pair dominate the other recessive. The law of dominance is used to PL and Li res sign off only one of the paren 3.1 characters in a Ronald cross in the F1 and the pression of both in the F2. It also planes the proportion of 3, IOBL3 enacted the F2 5, 2 law of segregation. This law is based on the tact that the alleles do not show any blending. And TLL at top Li characters. Are recovered. As such in the F generation though one of these is not seen at the F1 stage, though the parents con 3 and TV Lusktuningar at F formation. The factors are 3 lels of a pair psi 8 from each other such that a G3 lel receives only one of the 2 factors. Of course a NCERT just parent produces algaretes that are similar while a heterozygous one produces two kinds of gametes each having one allele with equal proportion. P generation gametes FL generation gametes F1 generatlan phenotypic ratio genotypic ratio red 1 RR1 pink 2 RR2 RR1 white 1 biology 5, 2, 2, 1 incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower snapdragon or Anthranum sp is a good example to understand incomplete dominance. In a cross between true-breeding red-flowered RR and true-breeding white-flowered plants RR, the F1 RR was PJNK figure 5.6. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1 RR red, 2 RR pink, 1 RR white. Herethogenital parentless we are exactly as we would expect in any Mendelian monohybrid cross, but the pheno-1 ratios had changed from the 3:1 dominant, recessive ratio. What happened was that R was not completely dominant over R, and this made it possible to distinguish RR as PJNK from RR red and RR white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical, as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on. And in the next chapter, which modifies the information that particular allele contains. Figure 5.6 results of monohybrid cross in the plant snapdragon. Where any allele is incompletely dominant over the other allele. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, as is more common, that the normal allele produces a normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for production of either normal less efficient enzyme, or 11 a non-functional enzyme, or 111 no enzyme at all. In the first case, temslifidolylise equivalent to the 1-difidolylI, it will PTT, duce the sort TRNT. That is, result in the terms Ronliknov substrate S such EQIVT OLPM very CC41-41, but if the OLPLTXI uses N in function 3.1N0 or ONZRRE, the phenyl P may be attracted. The phenotype trait will only be dependent on the functioning C. The TMM, dirfeed.uli. The TMM, dirfeed functi, ning allele which represents the ONG, ni phenojope is the dominant allele, and the M, D1IED allele is narrowly reses sin 3 lel. Hence in Li.ample.data veli reses C trait is seen due to non-FWLCTI and 31E11Z allele or to cause NONZR is produced. 5, 2, 2, 2 co, dom, hank till now lolly word discussing crosses where the F1 resembled Hitlerov Li two parents culminants or was in BN incomplete minants. But in Li CS of codominance, Li F1 generation resembles both the parents. A GCX, D, ample is different types of red BLCX, D cells that determine ABO-BLCX, D grouping in human beings. ABO-BLCX, D gups are unruled by tregene I. The plasma membrane of the red BLCX, D cells have sugar dirers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has 3 alleles Z.Z, and I the alleles Z and Z. Produce a slightly different form of the sugar, while allelides S and PLTXI use any sugar. Because HUNG1S are diploid organisms, each person possesses any 2-3 gene alleles I, and D, RO, Rinklet I. In all your words, when IAMDL are present, only P expresses because LD says not pro, 2 any sum. And when P.MCT lays present, P expresses, but when Pandler present, Tojet their Thebeth express their own types of sugems. This is Tekos of Koshayarnim 3 says. Hence red blow. D cells have both A and types of sugars, since there are 3 different alleles. There are CK different combinations of LE-STLUEE3 lels are possible. A total of 6 different genotypes of the human ABO-BLCX, D-Twas table 5.2. Homonocps are possible 13 BLE 5.2, 13 BJSHOG the genetic B3 cis of blood groups. In H1, 0 TL3LL Fpushin, an S, R, LP3 cent 1 ALOB, JAIJRSLI allelom PA, rent 2 Z, Z, P, Genopayavov, S. Comma prings I, JAJA lap, pi Z, blood types of OFF, Ipxingated as AABBB0. IIEIOGVDALJ you realize that the ample of Abablo, D grouping also provides a GCX, D, ample of mu. Up alleles here LJ you can see that there are wormers MITLID, that is, TLL1EE alleles garvefling this ruhr C. Mactor, since in on Indi ua allelatolos can be present multiple alleles can be found only when population studies are made. Occasionally a single gene PLTXIX may lose more than one effect, for E.impl starch synthesis in pea seeds is controlled by one gene. It has two alleles B and B. Starch is synthesized effectively by BB heterozygotes and therefore large starch germs are PLTXI used. In contrast, BB homozygotes have lesser E1I and C in starch synthesis and produce smaller starchins. After maturation of Lee seeds, BB seeds are ROMD and the BB seeds are wrinkled heterozygotes produce round seeds. Onsob seems to be the C-dominant allele, but the starch GRMS PLTXI used are of interdiet CSA and BB seeds. So if starch GRM CSA is considered as the phenotype, then from this angle the alleles show incomplete dominance. Therefore dominance is not .m. Autonomous feature of a gene or TLLE product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product. As ITD says on the particular phenotype TLL at LOL each used to examine. In case more than one phenotype is influenced by the same gene, Mendel also worked with uncrossed pea plants that differed in TO characters. As is seen in TLLE crossed between PL3 and TTLL. It has seeds little yellow color and round shape, and one that had seeds of green color and wrinkled shape. Figure 5.7. Mendel found that the seeds resulting from the crossing of the parents had yellow-colored on round-shaped seeds. Here go EU lich of the DV, RRODers and the PWS juanine green color in Vaward for clad shape UXXS dominant. Yellow color was dominant or green in round shape dominant wrinkled. These results the are identical to TLLO's TLLD got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols YF for dominant yellow seed color. MDY for Rhesus CE green seed color. RFB all 1D shaped seeds. And RFB wrinkled seed shape. The genotype of the parents can then be written as RRYY. And RYI. The cross between TLLE2 PLNTSC and BW and 10DJ1J1 is in Figure 5, 7 showing the genotypes of the parent plants. The gametes RY and Z unite on for WIS ion to produce the F1 hybrid RRI. When Mendel self-hybridized F1 plants, he found that 3/4 of plants had yellow seeds and 1/4 had green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. Principle of Inhudeng FAHO, Uation P0, 11 Herziar 17 F8 FTS. Semicolon EUAD, AU, NLMD, WIM1, W is little Fiat 11 a 1 Figo 5.7 of the Hibn class T, LHO2 FEY, Stillery, L in Parasophone U. 2, Colo.row seed shape. IIEI 5, 3L law of independent assortment in the dihybrid cross F, Gure 5, 7 the phenotypes round, yellow, wrinkled. Yellow, round green, and wrinkled, green, paired in the ratio 9:3:3:1, such. R. Ratio was observed for cellular pairs of characters TH, T. Mendel studied. The R ratio of 9:3:3:1 B rare IL. Is. A combination series of 3 yellow, 1 green, VL 3 row WLD, 1 wrinkled. This derivation can be V, Lafitten. As talos, 3 round, I wrinkled 3 yellow, I green 9 round. Yellow, 3 wrinkled, yellow, 3 round, green, I wrinkled, green. Based upon such observations on dihybrid crosses beta-ene plants Dilberton in 2 T.S. Mendel proposed, or set C generalizations that call Mendel's law of independent assortment. The law states that when 2 pairs of TROX are combined in hybrid segregation of 1 P where of characters is independent C the other pair of characters, the PWL net square can be effectively used to understand TLLE independent segregation of P2P, irs of genes dirt meiosis and the production of eggs and pollen in the F1 RRI plant. Cider the segregation of 1 P, IR of genes rand are 50% of theg, reads H, the gene R, and E the other 50% haver. Now besides Z, CHG read having Edler or R and should also have TLLE. Or Y. The important thing to remember here is that segregation of 50% R, and E 50% RIS independent from the segregation of 50% Y, and E 50% Y. Therefore 50% of the RB, RT and G gamete H, SY, and E the other 50% has Y. Similarly 50% of the R Berton G read has Y. And TLLE Outler 50% has Y. Thus TLLR. Are genotypes of G, reads F are types of pollen, and EF are types of eggs. The 4 types are RY, RYI, and EIYE, CH with frequency of 25% or phi of the total gametes produced. When you write down the 4 types of egg stud and pollen on TLLE teosides of dot or PWL net square, it is very pseudo-derived the composition of the zygotes that give rise to the F plants figure 5. 7. Although there are 16 squares, how many different pairs of genotypes and phenotypes are formed? Note them down in the format given. Can you, using the pun out square del, work out the genot, IPCR, SHIO, TDF, stage, ND, fill in the F formagilin? Is the genotypic R, SHIO, LS09, 3, 3, I. Dot, I all are expected phenotypes I. Full stop, S no, genotypes found, LNF, 5, 3, 2 cron, TOS and 1 al. Theory of inheritance. Mendel published his work on inherit MCE of characters in 1865, but for several reasons. It remained WL recognized till 1900, firstly. Principe Felti Huda, Isano VAT, Atio I Corn WLization was not easy as it is now in TLLO's days and his work could not be widely publicized. Secondly, his concept of genes occurs in Mendel's words as stable and discrete units that control TLLE expression of traits and of TLLE pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his theory. Finally, though Mendel's work suggested that factors genes be are discrete units, he could not provide any physical proof FBRTLLE existence of factors or say which they were made of. In 1903, scientists Devnes, Odrens and von Chermak independently redisco, erect Manktet, as results on TLLE inherit MCE of characters. Also by TLL's time due to advances in microscopy, TLL at LR taking place as scientists were able to carefully observe cell division. This led to the disco area of structures in TLLE nucleus. TLL had appeared to double and divide just before each cell division. These L are called chromosomes, colored bodies, as they revisualized by staining. By 1902, TLLE chromosome new variant D, 1 Ewing retiosus had been worked out. Walter Sutton and Fector Bovary noted that the behavior of chromosomes was parallel to the behavior R of genes and used Crowersor LDR in figure 5.8 to explain Mendel's laws. Table 5.3. Recall that you have studied the behavior of Crowesorus during mitosis equational division and during meiosis reduction division. The import, MT things to remember are TLL at chromosomes as well as genes occur in pairs. The 2 alleles of a gene pair are located on homologous sites on homologous chromosomes, G, Y1 TJADFL PBM UD, LL figure 5.8 meiosis. Male FTLM cell F formal 1 male milli F are chromosomes. Can you see how chromosomes segregate when RRN cells ME formed? Lab J 5.3, a confusion between the behavior of C chromosomes and genes A beaker C and PC1S ocra and PCRSL segregate that QFG meet segregs TG meet form, RTJ on ND on I, Jorm Konacht T on EM1 QFECH PCRCS tronsum to PCRS tronsum to 2. Jamie Chi meet, independent PCRS sesreg ter 1 I segregtes CNDQ, endently JCNDQ, endently QFECH other nether PCR. JCN UTET LHCCH these co LRMSLF. Or bar Q, resent the chronosome, and ELHCCHRQ, resents the gene hue DCDU dead. Dirtmaro, pH3 set of RelioCSI the D100 Crowersor P-bars con all up the metapruseplate independently of each other figure 5.9. To uncturst third TLL is compare Lee chromosores of 4 different color in the left and ReLL columns. In the left column possibility I orange and green is segregating together, but in the right hand column possibility DTLL the orange chromosome is segregating V, little. TLLE red chromosomes. Possibility I1 long or SH, LRT gene chromosome long yellow short red chromosome L the same pole LII, I possibility 2 1 long or E short red chromosome long yellow and short green chromosome L the same pole MS mile, IIJ fig, your 5.9 R dependent treatment of chromosomes. Sultan and Bow, and argue that the pairing and separation of a pair of chromosomes would 103D to the seg. Egadion of a pair of factors the, CNET as Upland unit technold, Jeff Kronlosamaj segregation with Mendelian principles and called it the chromosomal theory of inheritance. A B figure 5.10 Drosk, HDMN stir BJM3 Lee BRML, Lee follow WIing this synthesis of ideas. Experiment 11 even Theory of the chromosomal theory of inheritance by Thomas H. UNL Morgan and his colleagues led to discovering the basis for the variation 3 sexual reproduction produced. Morgan worked WTTH the tiny fruit files. Drosophila melanogester figure 5, 10 which he found very suitable for such studies. They could be grown on simple synthetic medium in the 13 laboratory. They complete their life cycle in about 2 weeks, and a single moting could produce a large number of progeny rows. Also there was a clay, R.D. Laranuantan of E. the male and female flies. Easolidus was able. Also it has many types of hereditary variations that can be seen with low-power microscopes. 5.3. 3 Lin-Leod recombination Phi, Q and C write out several dihybrid crosses in Dr0, J and I4 to study genes that were UNL. The crosses were similar to the disjunct crosses carry out B.J. Mendel in these, for example Phi, Q and H Batiste yellow-bodied. Wu, E.D. females to brown ba, Leah, L. red E-jed males and Dinu crossed their F progeny. He observed that the twogenous did not segregate independently of each other, and the F ratio do, Tate, L. very S, Niflacanra from the 9, 3, 3, 1 raw O expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosome section 5.4 and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome LHE proper U on off-prenal gene combinations were much weaker than the non-P, renal type. Morgan attributed this due to the physical association or linkage of the two genes, and coined the term linkage to describe this physical association of genes on a do, and also mean the term reambly on to describe the genera DL and non-parental phgenet, Umbenatmukens figure 5.11. Mikrgan and his group also Hatepunchnes were grouped on the same Chia, Isu genes wherever is it Linl, Ed showed Wari low recombination figure 5.11. Cross while others were lually linked showed higher recombination figure 5, 11, Crow, 8, Furry, Ample. He found that the genes white and yellow were Ryazu I1INL, Ed and showed only 1.3% recombina U while Wood and Integer Wing SHCI 37. 2% recombination U. Lee's student 1 Latran, Elsterdavant used the frequency of recombination between gene pairs on the Sara chromosome as a measure of the distance between genes and mapped their poston on the chromosome. One-day genetic maps extensively. Parental F1 generation biology used set as a starting point in the sequencing of whole genomes, as was done in the case of the Human Genome Sequencing Project, described later. Cross A Wfit MD4 KBH, F, ID or yellow Wtety C, M, FT, IHL KCM, WCDIW wild type parental type 98. 700, W, W, M yellow white Y, W, WCBEW, W, OA wild type WKIM, L2SCIM, WMIFP Widlow, Wiltety Y, W. CC wild type DWH, IWIL, CLM yellow, White gametes recombinant types 1.3 YW. Sec Y, W, WHMIIFIH, TL Croy White Widlaw cross BUJWFJ. WC, LJ, IHWM White miniature W, 2 Ilfit IEWF, BWM, wild type parental type 62.8 W, M, T, P wild type WM Sim LDI White. Miniature W, M, PG MIEI, I. JWM wild type, EIA, DE, O.LIFII, FFID 10. Miniature DW, M, WWO wild type DLF, W, M white, Meitner recombinant types 37.2, I, MMJB. JFLLWM, CFUM, CDYIW, MWM, JFLLU, 21 MWM layer WM. Comma with colon 3 HHIIGHI, MWMLL byte figure 5.11 linkage, results of 2 dihybrid crosses conducted by Morgan. Cross A shows crossing between gene Y and W. Cross BSH shows crossing between genes W and M. Here dominant wild type alleles are represented with sign and superscript. Note, the strength of linkage between Y and W is higher than W and M. The mechanism of sex deed, or menation has ALWI's been a puzzle before TLLE geneticists. The entity al clue about the genetic chromosome al mechanism of sex determination can be traced back to source of TLLE experiments carried out in insects. Intact the cytological observations modern and number of insects led to the development of the concept of genetic chromosomal basis of sex determination. Henking 1891 couldn't said a specific nuclear structure al TLL roughspe, RM added genesis in a few insects. And it was also said by him that 50% of the sperm received this structure after spermatogenesis. Where is TLLE outlier 50% sperm did not receive it. Henking a name to TLL is true CTU re is TLLE X body but he could not, plain its C81 ificans. Further investigations by others in streets led to the conclusion that the X bod JF of honking was in T or orasme, and that is why it was given TLLE near X chromosome. It W is also absent that in a large number of insects TLLE mechanism C1 said, deteneralishile. is a TLLEXO type, that is, all XBR and .X additional ZZ, X chromosome besides the other chromosomes autosomes. On the L, your H and some of the sperms B are the BCXXXXZWX chromosome, whereas some do not. Fis, your 5.12X fertilized by sperm H having an X chromosome become females. Embryos fertilized by sperms TLL, that do not have an X chromosome become Wes. Do go Thi and K, the number of chromosomes and the mole and femole are equal, due to the DN of 9I, X by chromosome. Rudolf Menos, be both LNHILN Frost, L, low. The MNJ has a POJRL X chromosomes I the mole X, L hedrog, metacornpus 111 on, CL and Moni BTTDS. RM ter has a POJRL Dlsarn, R chromosomes ZWIM, Leonville variant of TLLE X chromosome and TLLE determination of sex. It was designated to 2Z, Z chromosomes T TLLE sex chromosome and TLLE rest of TLLE chromosomes L air red as autosomes. R a shopper is an example of XOTP of sex determination in which TLLE molecular on lion X chromosome tesides TLLE. Autosomes. Whereas females have a pair of X chromosomes, these observations led to the investigation of the number of species to others to ND. TLLE are a mechanism of sex determinal lion, in nurture of Butler insects and RN animals including man. XY type of sex determination is seen where Butler and Fae, RTL3 lihale SR number of chromosomes. IEI of MJ and G males ANX chromosome is present but its counterpart is distinctly smaller on called the chromosome. Le males however have a PROFX, chromosomes, if males and females bears are a number of auditosaurs. Hence Leos have auditosaurs plus XY while female have auditosaurs plus XX, in human beings AND. Intrasoplidal Leos have one X and one Y chromosome whereas S Fae, RTL3 lehane pair of X chromosomes besides autosaurs figure 5.12a. B. In the above description you studied about two types of sex determining mechanisms. That is, XO-type A and XY type, but in both cases M3 produce two different types of GA meets A either Vili or Wilia at X chromosome or B or GA reads Vili X chromosome. N. Some Vili Y cluomosome, such types of sex determination mechanism is designated to be the example of male heterogamity. In other organisms for example, birds a different mechanism of cell, deet, Romero 3 Cheyenne is observed figure 5. 12c. In this case the total number of chromosomes is say RRE in both males and females, but two different types of G3 reads in terms of the sex chromosomes. Are produced by Fem3 that is, female heterogamity, in order to have a distinction via mechanism of sex determination in desert bed earlier. Lead to different sex chromosomes of a female bird has been designated to be the Z and W chromosomes. In these organisms the females have one Z and one W chromosome whereas S have a pair of chromosomes besides the autosomes. 5.4L said, determination in humans. It has already been mentioned that the sex determining mechanism in cause of Hung 1S is XY type. Out of 23 pairs of chromosomes present 22 are exactly say RRE and tot laws in one Fae, NWS, a really audosknes. Apo, R of X chromosomes are present in the female whereas the presence of an X and Y chromosome are determinant of the male characteristic. Dung spermaloelenesis 1 males 2 passive gametes are PTT, deuced 50% of the total sperm PLTXI used carry the X chromosome and the rest 50% has Y chromosome besides the auditosaurs. Fenwes however produce a lion type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with a sperm coiting either X or Y chromosome. HL cos LE ovum fertilizes with a sperm cani, X chromosome the zygote develops into a female pock and the fertilization of ovum via which tomsor car-tying sperm results into a male offspring. Thus it is evident that it is the genetic makeup of the sperm TL13T determines the sex of the child. It is ALSO a variant TL13T in AOCH pregnancy. There is always 50% probability of either a male or a female child. It is on 1BRTWL3 TL13T in our society. The RNA BL3 med for pursuing female children on high been ostracized on ill treated because use of TLL is TL's notion. How is sex determinal 3 Cheyenne mechanism different in birds? Is the sperm or the E9.9 responsible for the sex of the C and E, S? Pre, I sipi of LTI Huda, I say, I ovar, Atio I 5.5 mud. Tion mutation is a phenomenon which results in alteration of DNA sequences on dune sequenuit results in CH, J's in the genotype on the phenotype of .m organism in .didion to recombination mutation is another phenomenon that leads to venation in DNA as William in CH3 6. One DNA helix runs continuously from one end to the other in each chromatid in a highly superkyled form. One error or loss, deletions or gain insertion duplication of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on chromosome.es, alteration in chromosome.es results in abnormalities or aberrations. Chromosomal alterations are frequently observed in cancer cells, in addition to the above. Mutation all sorts due to CH and GE in a single base pair of DNA. This is known as P-ointment mutation. A classical example of such a mutation is sickle cell anemia. Deletions and insertions of base pairs of DNA causes fluorescent mutations. See Chapter 6. The Rechinismoth mutation is beyond Lee scope 3 of TLL is discussing on at this level, HDA. There are many chemical and physical factors that induce mutations. These are referred to as mutagens. LN radiation CM cause mutations in organisms. It is a mutagen 5,6-GFNBTIC RDNs 5.6L Pettiger. E-analysis. The Irritrat disorders are inherited has been prevalent in Li Hongwan society since long. This was based on TLL heritability of certain characteristic features in families. After Li rediscovery of Menret, SV, ORC the practice of analyzing inheritance pattern of traits in M beings tag M. Since it is evident trait control crosses TLL, at CMB per 100 in PPLMT or SOR other organisms are not possible in case of unwell being. Study of the family history about inherit set of a particular trait provides an alternative. Such an .M3 lysis of traits in a set, array of generations of a family is called the pedigree anal, BSLS. In the pedigree analysis, Li inherit MCE of a particular trait is represented in TLLE family tree over generations. In M genetics pedigree study provides a strong tool which is utilized to trace Li inheritance of a specific trait. Abnormality or disease, SOR are the important standard symbols used in the pedigree analysis. Have been shown in figure 5.13. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other XNA located on the DNA present in Li chromosome. DNA is Li Kamarov genetic in one formation. It is hence transmitted from one generation to the other without any crew and GE or 1, D0, IIEIOG, Valiiodid alteration. However, CH3 and J or alteration do take place occasionally. Such an alter, Cheyenne or CH, and GE in the genetic matter Y is referred to S mutation. A number of disorders in hum beings have been found to be associated with trait inheritance of CD or altered genes or chromosomes. 5.2 Mendelian disorder, SAEIOMIUI, if W, C SAPU, if DLLL4 AVIUTA WFTB 411 Whitfis, Euro 5.13 symbols used TN. Ilia Huma, and Pivot TOYSL Spradley. Genetic disorders may be grouped into TOC, Tegons-Mendelian disorders, NCT chromosomal disorders. Mendelian disorders are M3 only determined by alter, TN or mutation in the single gene. These disorders are transmitted to the offspring on Li CRRE lines. As Lal Ihan studied. In Li principle of inherit MCE, the pattern of inherit MCE of such Mendelian disorders can be TR, set in, formed by the pedigree analysis. Most common and PREV3 Lentmanctuli 3N disorders are hydrophiliae cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, T1 leucemia etc. It is import, MT to Renchen here had such Mendelian disorders may be due, mingant or recessive. By pedigree 3 and 3IJ1 cis1 C3JLE assolely understand weller TLLE trait in question is dormant or recessive. Simil 3 RTYTLLET it may also be linked to the sex chromosome. As in CS of handphilia. It is evident that TLLISX link recessive trait shows transmission from carrier female to male progeny. Its percentative of XII is shown in figure 5.14 Bordeaux, Min 3 and T and recessive TR. It's discussed Wilheljuk teacher and resign XII skr CH Ders Linl, etibotl. Autosaurs. In sex chromosome, BFIS, UX 5.14 representative but teists of AJ autosomal dole. In 3NT trait F for example, myotonic dystrophy B autosorna L recess, LF, it ROX, Replan, S1 called SLO Ernie. Pre, ICBIT LT, HTTA, ISANO, AT, Ashioahimafwa, this sex-linked recessive disease. Which shows its transmission from a protected carter disorder Lea progeny juice been widely studied. In this discussion E protein clot is PM of the cascade of proteins involved in the clotting of blood, D is affected due to this. In unaffected in DU3.1 a simple cut will result in non-stop bleeding. The heterozygote is female comer FBR heme, philia may transmit the disease to SUS. The P. Sibility of a female bioorning a herific is extremely rare because either of such a female has to be at least earner MD. Lee father should be Hemthecumbrielli in the later stage of HFE. The 3 Renili pedigree of Queen Victoria shows and Ribera of hemophilic descendants as SHW as daughter earner of the disease. S1C, class Elanimwana. This is an autosort-link recessile genit that can be transmitted from parents to the offspring when both lemners are carrier FBR the gene or heterozygous. The disease is control B, Y a single pair of allele, H beyond HB8. Out of the 3 possible genotypes only homozygous individuals FBR-HB, HBS show the diseased phenotype. Heterozygous HB-HBS individuals appear apparently no-affected but they are camerately diseased as clear as 50 page 3% probability C1. Terms reniscion of the mutant gene to the progeny thus. High binding sickle cell LRoid figure 5.15. The defect is caused by the substitution of glutamic acid by NCERT1 RLL high, W, CIC, CIC 1 2 3 4 6 E7 HBLPSTLD, FIS. Your 5.15 Crographilia red blood cells. Mel LAC1 decompose, I1 of their portion of PCH, M of homoglobin N, of flamma normal individual, B from an individual with sealed ol, M sem1a. Silog, V, LLN, vol, T the S1XTH pos1 D1 on of the beta globulin chum of the hemoglobin molecule. The substitution of mino, CLDLN the globulin proton results due to the S-GLB, Se substitution, T the SL, CTH codon of the beta-globulin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under 2 oxygen tension causing the change LN the shape of the RBC from Blaconc, V, disc to Elung, Tetzlakulka structure Flagier 5.15. Phenylketonuria, THLS Limborn error of MET, Bral SMLS also hinted, S the autosomal recessive DRI. The affected 1NCL1 vidal acts. An enzyme that converts the mono, CLD phenetine nonteteroslinet. S, result of THLS phenyl. 10mls, cumit TED and converted into phenylpyruvic, CLD. And other denvats. Accumulation of these 1NBRN results 1N mental retardation. These are also excreted through 1N because of 1TS per, sorplin by Tudney. 5, 6, 3 chromosomal disorders. The chromosomal 31CB disorders on the other H3 are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Follower of segregation of chromatids Dunzel D1 is less than 1 son cyclarisalt LN thega 1N or loss of chromosome S. Called aneuploidy, for ELC, PLEC, Down syndrome results from the G, M of Ixter, copy of chromosome 21. Similarly, Turner syndrome results due to loss of N's chromosome in human females, follower of Siddle-Unslzoid or telophase stage of cell die. Slon results in M1NCNS1N. Of E, whole set of chromosomes 1NM organism. MD, flat book offid, pro, defiant F, Sanjay, loops on, fingertips, P-crease. Semicolon, 0A, Jian Klai Tong, C, genital heart disease F1A E5.16A representative. Figure shown, MTN inflicted WTTL Down syndrome 01 DTLE correspondence chromosomes of ETN DF201. Pre, ICBIF LTIETTA, ISANOVAT, ACIO this phenomenon is known, as prolic, LDY this condition is often seen in PLMTSLH, TALNL 1111H, ROF THRMLL. Alioff, I3 JH being is 4623P, IRS. Out of these 22 pairs are autosomes and 1 pair of chromosomes are sex chromosome. Sometimes though rarely either an additional copy of chromosome may be INCL at an M individual or an individual may first lack one of any 1 pair of chromosomes. These situations are no 1 NTL as trisomy or monosomy of a chromosome respectively. Such a situation leads to very serious consequences in the individual. Down, S syndrome, Turner syndrome, Klinefelter syndrome are common examples of chromosomal disorders. Down's syndrome, the cause of TLL is genetic disorder is the presence of an actid 0 and 3.1 copy of the chromosome number 21 TRT SON with 21. This disorder was first described by Langdon Down 1866. The affected individual is short-statured. Really small round head furrowed tongue, NCT partially open mouth figure 5.16. P3LM is broad vela characteristic palm crease. Physical psychomotor and rental development is retarded. Klinefelter syndrome. This genetic disorder is a 1 so Cossack due to the presence of an additional copy of X chromosome resulting into karyotype of 47. XII, such, an industrial individual has all masculine development, however the feminine development of Bray, Street. That is, gynecomastia is also expressed. Figure 5.17 such individuals are sterile. Turner, S syndrome. Such a disorder is caused due to the absence of one of the X chromosomes. that is, 45 with XO. Such females are stereoneas of MEAS are ruderentaribiceres. Other features including lack of other secondary sexu, L-core, Ters figure 5.17b0. Li, AIR, II, YILIB short SL, D and underdead octeloped felinine CH mactrophy of 5.17d tire upper, NT, Dion of Jet, Na11C disorders due to sex chromosome compos 111 on all. B. On MLS, Klinefelter syndrome, B1 on Ayres syndrome. Sumiirigmuosls abrorachif B1ology WH, Odeojage with PRW equals of LL, Hontensitis praeocias. Pioni. Blingapo, Endless LN morphob califistolog C, Elphiopus has alteract ed the autonomian of many bilogicals. Mendel was Ilya BRST to study UU's Fen, LME, non-systema Euclid, while studying Ilya pattern of in, Rit, 3 set in PPL3 and TS of contrasting CH mact, RS. Mendel proposed Uapril sipples of INL, Eric, Nsel, Hitchmetaderi, Fe, Red to as ME, Niels laws of Inhani. He proposed Ili 31 the F, Torres lot, An, Netos Grungvicha, Reuters, F found in pairs known as aols. He obscene, L Natalia expression of Ilya CH, Ractors LN the offspring F follow a different pattern LN different first D, neck, Rons F1. Second, IFJ so on, some CHC, Ractors me domin MLTOE, I others, T1 E doyon, 3 NT coact, RS me expressed when factors me in heterozy condition law of da join, Mo. In Ria is 1 ECH3 Ractors me only expressed in H, Omazi 9 1 conditions, T1 E coact. RS net doctor blend in heat, Razi 9 1 condition A Ria is 1 li CH mact, R rulet was not expressed in heterozygous conduct and may expressed egg. 3 inches when it becomes H, LMO2 F9 1 US, heme coactor segregate while forma 11 on a G its law of segrego ion, not all CH mact, RS show true do, some CH mact, RS show LN, all PK. T, sol SH, WEO dol LML, whom mil studla, D1M, in henny of 2 CHC, ract, RS togelier. It was F dulatilia factors independently assort combine in all permutations and combine 11 ons law of irredeep, Ni, NT. Assault meant, furent combinations of G, amites A, Riiorat 1 cali represented in a squamitiabu, MF form KN, LWN as PUM, et. W, Rien LEF N, LW1 monas gene on chromosomes regulating Ilya CH mact, RS me called Ilya genotype Ilya phi cyclopression of the CHR actors LS called phenolip. After Mount Ilya F, Nesme located on Ilya chromosomes a 9-1 odd correlation was drawn between Mendel's laws, segreg, a U1 and assortment of chromosomes dirt meiosis. The Mendel's laws were extended in Ilya F form of chromosome. Ru theory of Mhe, R1WL, E. Later it was F found at Mendel's law of independent assortment, NT Dejrot H, LLD true F where the gen, R.Es Iliot were located on the same chromosomes. These gen, Nes were called as one inked N.Es closely located Nes assorted Toju, at Mel DSTC, Ntwi located JMS. Due to recombina U on assorted LNDY maps therefore corresponded to an ONT of FMS on a chromosome. MCNY genes. Air linked to sexes. Russo called as sex linked Nes two sexes RM1 Efern were found to have a set of chromosomes which were common. Moilier set WCH was different, the chromosomes which were different in two sexes were named as sex chromosomes. The Rematrung set was named as autosomes. In Hton, MS a normal female has 22 PRS of autosomes. And a pair of sex CHR Roilia Sychrome 00, a male has 22 patterns of auto 5 ohms, NDP, LR of SI, XLUM. UXX, in C, HCl, 2 N SOX chromosomes 1 N Ma IEZC Mel 1 N A RZW, mutation is del O, LU, OHA and GE, TN Netlik mid turtle. A point MB Cheyenne is a chance of A Bapu and DNA Clarolanern 1AT, Kaas 81 Dilo Albu and the Gelol or Beta CH3N of hemoglobin. Inver, WIOMUUT, Ons Con B Stud 1 Ed BFG Neratringaped, Giolda Fam 11Y, S Nomurat Lor. Ian whole set of chromosomes Prolpo CLL or in a subset of etomo number aneuploidy. TTU Helpton W, Lorspa 1 DLHEMB Cheyenne Al basis of Jarrett JC Disorden, Downs SDR OIM 11. Tsami of chromosome 2 liters where their LS. E, XRA. Copy of D Yalmo, WNE, 21 and Company TLYUE total N on B, RCL Kpumoi ISIS BLR, Mel 41, in 1.LRNRS syndrome. Or X chromosome ELSM, 101 and HX chromosome TS as XO ONI 1 and Nefilter, S Certus ILA. UCX, ND1 Senile, XXX1 TTIE Consei 3511 Y Street Died by Analysis Oka, al. XERCISESI Mentlana, D of MCLN 8 Pu Plant or Oxbun.NL by Mundell 2, D Anchial Ball. NU, Lalo Domin, L1CO ond, Riotsid IB homozygous and HW, yeah, us C monohybrid Lahybrid 3, Ad PBLD org, NSM is B, Esicus lore 4 low 01H, W many types of garnas can be produod 4. Explain the law of Ornlin not 1 IIU.8 A monohybrid cross 5, Defino and D61 aleatoroa. 6 Lilna puneltua, O world cowl, U, D slugtbulin of phenotypically albues in U, lust FLH, LG norit, on after oroa between H, renazig, us liamale and IJ, us malo laura SNP buse. 1. Wabina cross in Maklobot, lol PBNTLHLO for T on BLL plant WLLHGR, NWH, L proper ruins of plurloft in the cow. BE, Pocted Lobo W bland 6 LBLDW, DN. 81010 GY8, 2 hetera Z91 us PME, NTS me crossed, Iphilia 2 Louis me link V hat would be U, E distant bu U on of Fen, Ltipi features 1 NF1G, Nara U on F or Adlibber 1 D cross 9. Pretend, fly me, and U on a contribution of TH more N1N, 10, what LS pivot T, lysis jet, street husach. W sysconda usophil 11, how on S sextile, 9 hum, M beings 12, a child H, S blood group 0. To the father H, S blood group A and mother blood group B workout Iliagen, N, types of Iliapemi, NTS the pos 1 blood T. All the other offsprings 13, E, CPLMEF follow Wu, G terms with O, pleta 0, Doral, Nobile cornthot door, Un, NCE 14. What 1S page 01 NT mutation G, 1E, MNP2, 15, who had proposed the cloamasorn, L theory of the Lanhantan, E16. Me, and you on my 2 autosomal disorders with their symptoms. Chapter 6 Molecular Basis of Inheritance 6.1 The DNA 6.21 He rich for genetic Matino, T6.3 FN Avon 6.4 Hip of Cotton. 6.5 Transcriptome 6.6 Genetic Code 6.7 Translation 6.8 Reg Genton of Geni Express Ton. 6.9 Human Gen Project 6, J0 DNA F celery in the Prevalence chapter, you have learnt the inheritance patterns and the genetic basis of such patterns. At the ire of Mendel the nature of those factors, regulating the pattern of inheritance W is not clear. Over TLLE next 100 years the nature of the putative genetic material was investigated culminating in TLLE realization that DNA T-atonucleic acid is the genetic matter E3.1, at least for the majority of organisms. In class side you have learned that nucleic acids are polymers of anucleotides. Deotonucleic acid, DNA. And ribonucleic acid, RNA are the 100 types of nucleic acids found in living systems. DNA acts as the genetic material in most of the organisms. RNA also acts as a genetic matter E31 in some R1 ruses. Endstly functions as a messenger. RNA has act addition 3L roles as V, L, it FWL xions as dad, 3pter. Structural MD and source E as a cal 3 lytic M, DEQLHL, class Xi, IOU have already learnt the structures of nucleotide S.MD. The W alleles and noil 1F willets are linked to F-form nucleic acid P. Lirers L in this chapter of our going to discuss the structure of DNA. Its replica IC and the process of roking RNA from DNA transcultiogenetic octet determines the sequences of amino acids in proteins. TLLE process of protein synthesis TR translation and eliminatory basis of their regulation. The determination. Ceology of complete nucleotide sequence of H-gen or Daniel, Street decade has set in a new era of genomics. In the last section TLLE essentials of Hern gen or say, Junk, Ingnitskensah, Juances will also be discussed. Let us beg, and our discuss by first understanding the structure of the RMST interesting molecule in the living system. That is the DNA. In subsequent sections, T Lolo 1 Alderst TLL at Y it is Li NDST. WLDMT genetic material. MD what its relationship is V, lith RNA 6.1 the DNA is, long Paul, Icher of DOJJ bonucleotides. The length of DNA is usual, irrefined, number of nucleotides or, parapnucleated refer to as base pairs present in it. This also is TLLE characteristic of M organism for X, plant, B, tenaphage known, S174 has 5,386 nucleotides. Bacteriophage 13 has 48,502 B, sepairs BP, ESDRECNA, CCI has 4.6xt08 BP, ND haploid content of HERN DNA is 3.3x10 BP. Let us discuss the structure of such long polymer 6, 1.1 structure of polynucleotide tri-U. Let us recapitulate the chemical structure of polynucleotide chain DNA or RNA. A nucleotide has TLUEE components. A nitrogenous base. A pentose sugar ribosin C, set of RNA, ND dale, J ribose FBR DNA MD, phosphate group. There, two types of nitrogenous bases adenine, ND guanine, NDP, imidine cytosine. Sil, ND-thymine cytosine is common for both DNA and RNA. ND-THJ-mine is present in DNA or Sil is present in RNA. T the PL, set of THJ-mine. A nitrogenous B set is linked to the pentose sugar through NGL, acidic linkage to form a nucleoside. Such as adenosine or CTIA, y-actenosine. Or deogeanosine, cytidine or Cl, itidine and uridine adec, ethmidine. When phosphate section cup is linked to 5O of nucleoside through phosphoester linkage, corresponding nucleotide or nucleotide depending upon the type of sugar present is FPRD. TV, nucleotides, relinked through 3S phosphodiester linkage to form dinucleotide. Mononucleotides can be joined in such manner to FBRM, polynucleotide chain. A pol, thus FPRD H, S at one end, free phosphate LL diet, Y, T. Moleculae UL of LaTriHUIAHC fest.endol NL, OS sugar which is referred to as S-endol polynucleotide chain. Similarly at the etheriodotl point where the ribose 1 is greater than to a free 3.0 group which is referred to as R.end of lipoil, IUDEC-ALDLAN. One heat backbone in a polynucleotide chain is luted QLE to sugar and phosphates. The nitrogenous base is linked to sugar-JL object from the backbone figure 6.1. In RNA rinucleotide residue has an 3D, lit NAO group present at 2 position in the ribose. Also in RNA the uracil is found at the place of LH-TMINO S methy-1 uracil another chemical name K, RLH-TMINO. DNA is an acidic substance present in nucleus was GTST longlage led by FT1 Edmshmisser in 1869. He named it S-nuclein, how, R due to technialimal, U on an esylaling such, long polymer intact. The LOC-Bu1 of structure of DNA remained 1 UCEK, R, very long period of time. It was only in 153 that James L. B. Toon and Francis Crick. Based on the X-ray de track UM.Pro, loosed by Maurice W. Ui, Inson Rulined Franklin Jlapuit a very simple but thorough double-held control of the structure OIDNA, Onial Halmeral, SDO Proposiliu. Thus pairing between the two strands of polynucleotide chains, HLHLS Proposiliu was also based on Hiobsank UOO Erwin Kwakawan with that I, replicable stranded and I Horatius between adenine and thymine and guanine and cytosine or O, stunt and equitrilson. The base P, iron A, confers a Viriaruk property to the pol, nucleotide chains, they are said to be complementary each other. Entheric, if the sequence of bases in one strand is known then the sequence in other strand can be predicted alog of each strand from adenolatocylitis apparent DNA acts as a Turk. 8K, our synthesis of a new strand the tube bubble straro, BDDNA let us call them as daughter DNA thus. Produced would Beldlantial to the pair of 431 DNA B, Aussie of THL as the genuine implications of the structure of DNA became very clear. The salient lil, terrestre double helix structure of DNA is lipose, it is made of polynucleotide joins. Where the backbone is constituted by sugar phosphate and the base is eject inside, the two chains have anti-parallel darty. It means if one C-hane has the pollen DS3 the other has 3, S2 the bases in T strands are paired through Hickel-Rickson bond H bonds F base pairs BP. Actinyl OI, RMS2HL, Ltogid bonds with thymine fran opposite strand and diltsarse, similarly. Guanine is bonded with cytosine with 3 H bonds, ASA result always appearing OCL is oppositopyrimidine. This genre, tesapro, imate the uniform distance between the T strands of the helix figure 6.2. If the T chains are coiled in a right-handed fashion, the pitch of THLL helix is 3.4 nm and nanometer is 1 billionth of a meter. That is 10 m and there, we rough 10 BP and E3CH. Ceology Fear, 6.2 double serled polynucleotide CHNISW DIPRRE TUM, O. And sequently TLLE dist MCE between data BP and data helix is APRO. Imatelli equal to 0.34 nmLJ the plane of OOE base pair slacks mer TLLE Ollier and double helix, this. In addition to H bonks confer stability of the helical structure figure 6.30, C. Compare the structure of PER, NES and FLS, 6.3 DNA double helix NSL PGNM, D. Nes. Can go found out WLVJ the D dot stands between two pot nudiot the DAINS and DNA remains street constant. The proposition of a double helix structure for DNA and its simplicity in plumping the genetic implication become revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology which states that TLLE genetic NEDRRN flows from DNA RNA protein. G. In Pra1 is less than 11, only dogma. Molecular basis of inheritance in some viruses the flow of information is in reverse direction, that is, from RNA to DNA. Can you suggest a simple name to the process 6.1.2 packaging of DNA helix taken the distance between two consecutive base pairs is 0.34 nanometer 0.34 XL 09 meters. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of BP with distance between two consecutive BP, that is, 6.6x Lu BP x 0.34 xi 09 meters BP, it comes out to be approximately 2.2 meters, a length that is far greater than the dimension of a typical nucleus, approximately LQ 6 meters. How is such a long polymer packaged in a cell if the length of E. coli DNA is 1.36 millimeters? Can you calculate the number of base pairs in E. coli and prokaryotes, such as E. coli? Though they do not have a deft nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged is held with some proteins that have positive charges in a region termed as nucleoid. The DNA in nucleoid is organized in large loops held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged, basic proteins called histones. A protein acquires charge depending upon the abundance of amino acids residues with charged side chains. Histones are rich in the basic amino acid residues lysines and figure 6.4 and nucleosome arginines. Both the amino acid residues carry figure 6.4b and picture beads on string positive charges in their side chains. Histones are organized to form a unit of 8 molecules called as histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome figure 6.4a. A typical nucleosome contains 200 BP of DNA helix. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin, thread-like stained colored bodies seen in nucleus. The nucleosomes in chromatin are seen as beads on string structure when viewed under electron microscope in figure 6.4b. Theoretically, how many such beads nucleosomes duengine are present in a mammalian so the beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as Ceology non-histone chromosomal NHC proteins. In a typical nucleus of chromatin are loosely packed and stoms light and are referred to as euchromatin. The chromatin that is raw or clumsily packed and stains DMK. Are called as heterochromatin. Euchromatin is AI to be TRM script T, LRL3 LLY active to OI-udin, whereas heterochromatin 3 tin is inactive. 6.2 th, search LOIL, 1, C. Fillet, U, T, Ellen. The discovery of nuclein by Miescher and the Prop. CION were principles of inheritance by Mendel were albums at the sort time. But that the DNA acts as a genetic material took long to be discovered and proven. By 1926 the quest to determine the mechanism for genetic inheritance had reached Liam. Decalar level pre-OS discoveries by Gregor Mendel, W. Sutton, Thomas Hunt Morgan and numerous other scientists H3D and 3 TLLE Senarch to TLLE cron source located in TLLE nucleus from cells, but the question of what molecule was actually the genetic matter E3L. H had not been. MSV, Erdthumsfondung principle in 1928, Frederick Death, in a series of experiments with Streptococcus pneumocolineae bacterium responsible for pneumonia. Witnessed a miraculous T-arms formation in the bacteria, during TLLE course of his expertron. A Leon Organism bacteria had changed in physical form when Streptococcus CCIJ, S. Nevrum GNV, Uniumcoccus bacteria. Argotan on a culture plate. Sort produced smooth shiny colonies S while others produce rough colonies R. This is because TLLESSTR and bacteria have a mucus polysaccharide while R strain does not. Mice infected with the S strain virulent die from pneumonia infection but mice infected with the R strain do not develop pneumonia. SECT, RLL object. Lomsml Lovegre 1 it was able to kill bacteria by heating them. He observed that he killed S-strain bacteria injected into mice did not kill them when he S-heat. 1 S who at 1 R kill jet local. Molecula, Sassus of inherence injected. Mix tour of heat killed, S.M. ER bacteria TLLE mice died. Moreover he removed leavings bacteria from the dead mice. He concluded that the R strain bacteria had somehow been nuanced on by the heat K, led S strain bacteria. Serwan's principle transferred from the heat-killed S strain had enabled the R strain to synthesize .sm, otl. Polysaccharide coat and before R1 rulant. This must be due to TLLET arms fur of TLLE genetic amatter dalhav, ear. The biochemical nature of genetic material W is not defined from his exp. Reruds, chemical Chanktertsa Yuan of Islands von prior to the work of Oswald Avery. Colin MacLeod and Macklin McCurdy 1933-44. The genetic material was thought to be protein. They vork to determine the biochemical natwaf transform principle in Griffith's experiment. They pump-feed biochemicals proteins, DNA, RNA etc. from the heat-killed S cells to see which ones could TRMS1 live R cells into S cells. They discovered that DNA alone from bacteria caused R bacteria to become transroared. They also discovered that protein-digesting enzymes proteases and RNA-destreating enzymes RNases did not transform ion. So the transforming substance was not a protein or RNA, DS ion with DNase did inhibit transformation. Suggesting that the DNA caused the transformation, they concluded that DNA is the hereditary material. But not all BOLOSTSL are convinced, CATCO TNNL, of MG difference between DNAs at the DNase 6.2. 1. The genetic material is DNA. The unequivocal proof that DNA is the genetic material car from the experiments of Alfred Hershey and Martha Chase 1952. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its CNJN.MD subsequently manufactures more virus particles. Hershey MD Chase Loyoth to D is Coer-Whitler at W as protein or DNA from the law uses that entered the bacteria. They grew some viruses on a red IUMTLL. It contained radioactive phosphorus and scred others and radium that combined radioactive sulfur. Viruses grown in TLLE presence of radioactive phosphorus contained radioactive DNA but not radioactive E protein because DNA contains phosphorus but protein does not. Similarly, viruses grown in radioactive E sulfur contained radioactive protein but not radioactive E DNA because DNA does not contain sulfur. Ceology R3 dialkyl EPH31S were all OD to attach to E. cart bacteria. Then, as the infection proceeded, the viral fluids were removed from the bacteria by aling them in a blender. The virus mickles were sep, L from the bacteria by spinning them in a centrifuge. Bacteria which W3S infected, V, little, viruses TLL, 3TH3DR3 diactive DNA were radioactive, indicating that DNA was the material that passed from TLLE virus to TLLE bacteria. Bactum TL13T low, air infected. V. Little uses TL13TH3CTR3 diactive proteins were not R3 diactive ETHs indicates TLL, 3T proteins did not enter the B3 Terry3 from TLLE viruses. DNA is TLL era for TLLE genetic material that is passed from virus to bacteria Phi 6.5. And Xeno RLLLDDT1 TDEC, N, B, or, it LLUDed M.UPNATA, TDW1PNCA. Exclamation point NC, 11OTCN, R, 1101, LUAACD, 1 AHO PPRKI Limited 6.2. 2 Properties of genetic matter E3 LDNA versus RNA from the ID regime discussion. It is clear that the debate beta and proteins versus DNA 3S TLL genetic matter E3.1 W3S Tmequiok 3 LLY result. From Hirsch, Y, Chase, Penmanent, it became an established fact that it is DNA TL13T3CLS3S genetic mate, Re 3.1. However it subsequently TKRRE clear TL13T. Molecula, S satisfy, Hansen sore viruses, RNA is TLL genetic mate, Re 31 tower example, tobacco mosaic uses. QB bactinopmic etc., ends to soar of the questions such as why DNA is the pro-or-lerent genetic matem. Whereas RNAP RMS dyne 3 mic functions of Resinger and adapter has to be round from the differences between chemical structures of TL1E2 nucleic acid. M. Decules, Kangurical detox, Chem, C. Ujfarin, XS.F, between DNA and RNA a molecule that can act as a genetic material must fulfill the following criteria. I. It should be able to generate its replication. I. It should chemically and structurally be stable. 3. It should produce the SUP-FBR slow changes mutation that are required for evolution. I. It should be able to express itself in the form of Mendelian characters, if one examines each requirement one by one. Because of rule of base pairing and complementarity both the nucleic acids DNA and RNA have the ability to direct their duplications. The other molecules in the leaving system such as proteins fail to fulfill the first criteria itself. The genetic material should be stable enough not to change with CL afferent street, Gs of light cycle. Or with change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself. That heat, which killed the bacteria, at least did not destroy some of the properties of genetic material. This now can easily be explained in L-GLT of the DNA that the two strands being complementary if separated by healing correlate together. When appropriate conditions are provided, further 2O group present at every nucleotide in RNA is a reactive E group and makes RNA labile and acetyldegractable. RNA is also now known to be catalytic, hence reactive E. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, along the TVO nucleic acids, the DNA is a better genetic material. In fact, the presence of TLL-amine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding of the process of repair in DNA. And you will study these processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable and rooted at a faster rate, consequently, viruses have a genome and having shorter lifespan rooted and E01E faster. RNA can directly code FBR, the synthesis of proteins, hence can easily express the characters, DNA Ho1A. Or is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The discussion indicate both RNA and DNA can function as Ceology genetic matter 3.1, but DNA being RORSU, is preferred for store genetic information. For transmission of genetic information, RNA is Bet6F, LRNA war, L, D from Tbreakring discussion. Immediate question becomes evident, which is the first genetic matter 3.1. It shall be discussed in due in the chapter on chemical evolution, but briefly. We shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest TLL at essential life processes such as metabolism, translation, splicing etc. evolved around RNA. RNA used to act as a genetic material as well as a catalyst. TLL are so important biochemical reactions in living systems that are catalyzed by RNA catalysts and not by protein enzymes. But, RNA being catalyst was reactile and hence unstable. Therefore, DNA has evolved from RNA with the chemical D6 cations TLL at MA1 set it near stable. DNA being bubble-stranded and having implorino, Y-strand further resists changes by evolving a process of repo, R. 6.4 replic LL on wall prop, Singlet bubble helical structure FBR DNA, Watson and Crick had iron redia teleproposed, scheme for replication of DNA 1B quote the origin 3 else deterrent that is expose, 1T has not escaped. Our notice TLL, at least specific pairing we hail postulated and immediately suggests, possible copying mechanism for the genetic matter E3J, Watson and Crick. 1953, the share suggested that the TO strands would separate and act as a template for LSNTLL of new, Phi. GVRESS, W:(A), TSONCRTCKM, ODA-LFORC Ampler and Tia Rice Street RMDS. After TLLEC Ampler lion of Sempcancer, a UEDNA replication each DNA molecule would have one replicant parental N. One newly synthesized STRMD, this share was termed as semiconserva UV DNA replication figure 6. 66.4.1 The experimental proof is now proven that DNA replicates sentencer with T. Ely. It was shown first in ESDRECNA cow and subsequently in higher organisms, such as plants. Molecula, S. satisfy, Hehensontum, M. cells, Matthew Messelson on from Lilin SUHL power of the following E. Pertrant in 1958, by the EQ, in a Redwink 01L1.3 Ming NH. Cl Nisthavi isotope of nitrogen is the only nitrogen source for many generations. The result was that 1N was incorporated into newly synthesized DNA as LL is clearly rho LLL Kumrung CCFLL, duals. This heavy DNA molecule will be disunused TRC and normal DNA centrifugation in a sesawink CSQ density groctant AEA seno tlat UN is not a radioactive isotope and it can be separated from 1N only based on densities 2 then they transfer the cells into a medium with normal 14NLI.CLND. Took samples ATVMIS refinitin E intervals 3s the cells multiplied, and extracted the DNA that remained as bubble STR mictotesses. The VMIS samples were separated independently on CCL gradients to measure the densities of DNA Phi 6.7. Catco recall, what centrifugal force, S at DTLVNL. WLVJ MECUL HIV BUR JMSSD ANSAT wood. Sediment jaster. The results are shown in Figure 6.7 Photo T Figure 6.7 Messelson Street 3 HLS Expenro, NT3 thus. The DNA that was extracted from TLLE culture 1 generation after the transfer from N to N ready when it is after 20 minutes, E. CoU divides in 20 minutes had a hybrid or intermediate density, DNA extracted from the culture after another generation that is Euler 40 minutes. D generation was Ceology composed of equal 3.1 or ounce of this hybrid DNA and of 1 I DNA if E cow was allowed. To 9 row for 80 newts then what walk BT me proportions of U9HT and LV breed density. ES DNA am acute. Very similar 3R experiments involving use of radioactive emitting to detect distribution of newly synthesized DNA in Lecron source was performed on Fabians by Taylor and colleagues in 1958. The experiments proved that the DNA in chlorosaurs also replicate semi-conservatively 6:4 to the machinery and the enzymes in living cells. Such as E. coli TLLE process of replication requires a set of catalyst ears. The main enzyme is referred to as DNA-dependent DNA polymerase. Since it uses DNA template to catalyze the polymension of deoxynucleotides, these enzymes are highly efficient enzymes as they have to catalyze a rension of a MGE number of nucleotide resonance. E. cow het tassenly 4.6 x 108 BP comparatively human whose diploid content is 6.6 x 109 BP completes the process of replication in 38 minutes. Tlat reans the dot of her age rate of polymerization has to the I'm telly 2000 BP per second. Not only do these raises E to be last but they also E to Kyle, is he reaction with high degree of accuracy. Enemista, E done replication VO result into mut, atom S for themo, E, energy DCLI replication is RI, pensive process. Deo, J-ribonucleoside phosphates sane dual purposes, in addition to acting as substrates they prove. De energy for polymerization reaction tetratelenophosphates in a real nucleoside phosphates are high energy phosphates. SARA as in case of ADP, in addition to DNA-dependent DNA polymerases M, my additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occur within a small opening of the DNA helix referred to as replicatlin fork. The DNA-dependent DNA polymerases catalyze polymerization only in one direction he is 5:3. This creates some additional complications at the replicating fork, ogen sequenui on one strand the template with polarity 3:5. The replication is continuous while on TLL other Lie template Vili polarity 573 it is discontinuous. The discontinuously synthesized FR grants are later joined by the NCERT DNA ligase figure 6.8. The DNA polymerases on their own cannot initiate the process of replication, also the replication does not initiate randomly at any place in DNA. There is a definite region in eCarpal DNA where replication originates, such regions are Tarek as Aralclan of replicatlin. It is molecule US, of Inhuianth because of the requirement of the rigid, of Repuca UC and that a piece of DNA if needed TDB propagated eluding retinobinate DNA proceller S. Exclamation point it's a vector, the vectors provide the ong, and all replication, further not every detrial of replication is under sticksy well. In UP3 siotes the replicate, on all DNA tal.s place at S phase all the cell cycle, the epucoano, DN on cell DC on cycle should be highly co-written 3 TED. A failure in cell DVC on after C, DNA replic, lion results into polyploidy, chronosaur, nor E. You will learn the data I'll natter off or NND the process is occurring, the Hissoit LN Hitler CL, says. 6:01 raw, MP. On the process opinth, I. Dedic information from one strand of the DNA into RNA is termed as transcripton. Here also the principle O, cu tipo. N lolwygaoms the process at drumsatplic neo, sept the adenosigned RCW on space page 3 ir with uracil instead a thite. However unlike in the processo replica U on which ucilin heteld neo and M gets duplicated. In transcript U on only a segment all DNA and only one half the strands is copied into RNA. This necessitates defining the towns that would Daryl, recode the region and the strand of DNA that would be transcribed. Why both the STR, NDS, not copied done transcription has the influencer, first if both strands, CT, leaflet. They would code for RNA RDL will different sequences remember complement, does not mean identical. Third and if they seed for proteins the sequence of amino acids in the proteins will be different. Hence one SETI after DNA would be co-forged different proteins and this WQLLDC offlooked in genetic and transformationity. Second, the two RNA molecules ILPN, X, loose simult, an ESTW, a UKTB toil JETL, a JTRA TOE other. Hence would form a double-stranded RNA. This would prevent RNA from being translated into protein and the E. We set a transcript LCN vocal B caret to ulun. 6. 5. One transcription unit. A transcription unit in DNA is defined primarily by the three regions in the DNA, IAPG or U. The structure 31 gene 3A terminator A. Ceology. There is a connection in diphylorally two strands of TLLE DNA and TLLE structural neophytrumscretion units and celli TVOSTR mixave opposite male. The DNA-dependent RNA polymer cell SO carries the polymerase T on in only one direction, that is. 5. 3. The STR, ND that has the polarity 3, CTS, S, template male is also referred to, S templates child. The other strand which has the polarity 5, 3, ND the sequence sum, sRNA except gmine, T the place of or, sil. Is displ, sec D wing TR, script ion, STR, jelly. This strand which does not road FBR anything is referred to, S codlungstsiend. All the reference P ointment while defining, transcription WL.IT is made with seeding STRMD2E, plainly P ointment. A hypothetical sequence from a TRM inscription WLIT is represented below. 3 at cat 5 template strand 5 tac 3 oxling STR, ND Congo now WLT the sequence of FNA transcribed. From OBDNA STR. W. FIS. 1. TE69 schematic structure of a TR page 11 on Willet the promoter, ND terminal or the structural gene in, transcription WL.IT. The promoter is said to be located to MDS5 and upstream of the structural net. The reference is made with respect to the palatine footing strand. It is DNA sequence that provides binding site FBR RNA polymerase and it is TLLE presence of a promoter in a transcription willet that defines the template and decoding strands. By switching its post-EICN with tinulator, fit FNE1 ion of seeding and template strands could be reversed. The terminator is located to MDS3 and downstream of the link strand and its USU. IIY defines the end of the process of transcription figure 6.9. There are additional regulatory sequences that may be present further upstream or downstream to TLLE promster. Sorry properties of the sequences shall be discussed while dealing with regudge, Shyanov gene, Procyon. 6. 5. 2 Lorenz Stipcio, NU1 did in the gen, a gene is defined, as the functional willet of inheritance. Though there is no ambiguity that the genes are LOC, TET in the DNA, it is difficult to literally molecular stasis of inheritance refine a gene in terms of DNA sequence. The DNA sequence A, Dingferternatter, RNA molecule also define a gene. Of, or bit of an Inga-Klstron 3S3 segment of DNA seeding for 3 polypeptide. The structure 3L gene and 3 transcription willet could be said as monoclstron MOSUI nuke 3 ilotes or polyclstron MSUI in bacteria or prokaryotes. In eukaryotes the NDN cistronic structural genes high interrupted seeding sequences all genes in eukaryotes are split. The seeding sequences or express sequences are defined as exons beyond RS8 to be little sequence that appear in mature or processed RNA. The E, RONs are interrupted by introns. Introns are intervernal sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the refinity of a gene in terms of a DNA segment. Inheritance CHR actor is also affected by Pi-C4 Lorito in regulatory sequences of a structural gene. Hence the regulatory sequences are loosely defined as regulatory genes even though these sequences be not seed for any RNA or protein. 6.5. 3 types of RNA. In the process of transcription in bacteria there are 3 major types of RNAs, mRNA, messenger RNA, tRNA, transfer RNA and ribosomal RNA. All 3 RNAs are never to synthesize a protein in a cell. The mRNA provides the template, tRNA brings RNA and reads the genetic seed and RNAs play structural and catalytic role during translation. There is single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription with TRIUON. It uses nucleoside triphosphates as substrate. Fig. 6.10 process of transcription TN. 8 acti, RTA. Ceology and polymerase is in a template-dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA ruralness bound to the enzyme, once the polymerase reaches the terminator region. the nascent RNF also also the RNA polymerase. This results in termination of transcription. An intriguing question is that how is TLL the RNA polymerase able to see at alice all the TLL steps, which are initiation, elongation, and termination. The RNA polymerase is only capable of catalase, and the process of elongation. It associates transiently with triuon factor RCR and termination factor P to initiate and terminate the transcription, respectively. Association with TLL's factors alter TLL specificity of TLL RNA polymerase to either initiate or terminate figure 6.10. In bacteria, since the capital R, capital N, capital A, C, B, E, S, N, O, T, R, E, Q, U, I, R, E, A, N, Y, P, R, O, C, E, S, S, I, N, active and also since TRMS eruption N. TRMSylation L3K place in Lysaracorn patron T. There is no separation of cytosol and nucleus in bacteria. Many times the translation can begin Rutger or the mRNA is fully trans-embedded consequently. The transcription and translation can be coupled in bacteria. In eukaryotes are two additional complexities. Either at least 3 RNA polymerases in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut DC of labor. The RNA polymerase I transcribes RNAs Fig. E6.11 pruas of N, inscription in Eul, myotes. Mol, call UUDIs of an HFL, NCF28S, ISS and S8SJ, whereas tile RNA polymerase is responsible for LRAN, Erlpan ctTRNA. SSRRNA MDS and RNAS homonuclear RNAIH. The RNA polymerase II transcribes precursor A-tile heterogeneous nuclear RNAH and RNA. 2. The second complexity is that the MARI transcripts con, in both the exons and the introns and are non-functional. Hence it is subjugal. L-LOA process C311 EDSPL1 CB1 Guerthine Lorenz are removed and L, ONS are joined 1 and a defined order. HNRNA undergo T, MJ acylation processing called as capping and tailing. In capping, an unusual nucleotide methylguanosine triphosphate is acyled, D-Lodi enclafn RNA and tailing. Adenylatir sitters 200-300 or adrata T3 enclafn template independent manner LD is the fully processed H and RNA. Now called mRNA that is transported out of the nucleus for translation figure 6.11. The significance of such complexities is now beginning to be unclear. XL the SPLLT gene RU, gerents represent probably an unchained feature of the duone. The presence of introns is renist and the CT antiquity and the Proteus CT splicing represents the clonerant ctRNA. Orald, in Res.NL times the undersl, dinct RN oncl RNA dependent processes in the 1 goi stembla 1 easunad, re-importance. Certain replication and transcription of nucleic acid was copied to feed JRM another nucleic acid, hence. These processes are UI to conceptualize on the BAM of complementarity. The process of translation requires transfer of genetic information from an apol, of nucleotides to a polymer of no acids. Neither does any complementarity exist BTO, and nucleotides and nosids nor coldenide drawn theoretically. There existed ample evidences though to support the notion that change 1 and nucleic acids genetic alter responsible you change at amino acids in proteins. Fossil to the proposition of a genetic code that could direct the sequence of amino acids done synthesis of proteins. If the element in the biochemical nature of genetic material and the structure ctDNA was one exciting. The XLLPO-Cillian and Cleofertin CT genuc code reads challenging in a one true sense. It record Ln 110m, rent CTS scientists from disciplines PH, science. Grgmic chemist bio, L. Emsels Ankliger to a cell was George Gam.W a PhD scientist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should constitute a combination of bossers. He suggested that in order to code you all the 20 amino acids, the code shall be made up of 3 nucleotides. THLS was a bulk 1 propulsion because a permutation combination of 4x4x4 would generate 64 codons, generating many more codons than record. Providing proof that the codon was a triplet was a more daunting task. The chemical rethidilope by HRCBBL and CL Guarana was. Ceology Instrument 3.1 and Sin ceasing RNA molecules with defined combinations of bases homopolyrers on copolyrers. Marshall-Nirenberg cell-free system or protein synthesis finally helped the XE to be deciphered. Savarilo-Cho enzyme polynucleotide phosphorylase was also helpful in trancing RNA with refined sequences in a template-independent manner enzymatic synthesis of RNA. Finally a checkerboard for netic CW3S prepared which is given in T3+6, 13B6.1, the codons. For the vaccine US amino acids, TTE, A0 I1CUL or LL at UQC low U, C. UAC, Recro 11111 VC 480 T11 M 8101, UCO 8011 WO VTQ KIIAO 8101, IIQQ TIP CLIDIAN 00J. CWL low CQU, Q10AO CCP and low IC, UA, OCUP, OCM AMOCA ACT WQ.HO, CO. Colon 1Q011, OQQ, 6QAAW DUIDJNLR MUJJ, IUACRKDCIION, CDIRD CAR Alex IIOMA1, OJ. N10T New Jersey .61, GLNLQCIIJL, Loy Fago, LBOVC 1111, GCC Alacac. OLROIIAWGC ALAQAACLHLQGOGLLR CoFlow ICQLGAOCLHLO, GIR0 The salient Faith 3 Teres of, genetic seed are 3SF follows, I the CCDCN is flit. 61 Axel, NSCDCR aminoacid, LS on 3 codons do not core for any amino acids hence they function as stop codons. 21 codon seeds FBR only one amino acid. Hence it is unambiguous and spec 111C3 ser amino acids are seeded by M, TLWL1 cogn. Hence TLLE seed is degenerate I the cogn is read in mRNA in a contiguous fashion, there are no PWLCTU actions. V the code is near tion vexel, fer, ample from bactina to human UUU would core FBR alanine alanine fact. 9. RRE, sepalients to this rule have been found in mitochondrial codons on or protozoans. VAUG is dual functions, it cores FBR methionine ret, onda. Also. Act. As in Strader Co. BN. If owing, S the sequence of nucleotides in mRNA, predict the sequence of amino acid decoded. BG, T Talks het the Checkerbu Road, AUGUUUUC. Molecular, Sassus of inherence now the opposite, FUOU89, S the sequence of amino acids coded BG mRNA predid. The sequence of nucleotide in the RNA, metrafFFFFedugauphacyandig familkin predid 9 the opposite can go. Now corrects, LLEU, NDWO properties OJ genet C code go, rulern 6, 1 mutations and genetic code the relati, and shifts between G, NES on DN or best uncl, Risto, D by rotation studies. Who have studied, Tauten Ruta ICN3JLDC Fictin Chapter 5, Effects of, MGE deletions 3JLD rearrangements in, segment of DNA or E, Site to umprahundit may result in loss or G, in of, G, Ne3JLD so, function. The effect of P ointment mutations will be explained here, a CL, sickle, plot of P, int mutation is, change of single B, separ in the G, NAFBR beta globin cham that results in the change of ornino, SID residoglutamate to valine. It results in 2 dice, sedundion called, S-cyclosalinnemia, effect of P, lint mutations tlat inserts or elites database in structural gene can be better understood by a following simple example. Consider, statement that is made up of the following words each having THM letters LLK, G, netic co, Li. Raj1 has read CAP if insert, letter bin between has, NCT read 3JLD rearrange the statums, NT. It VODD react. As follows, Raj1 has BREDCAP similarly if now insert 2 letters, T the sor playo, say BL. Now it, WOLD read.act, Raj1 has BMEDCAP now insert 3 letters together S, I big the statement would read Raj1 has BIG read CAP the sor. C's can be reap, TED by deleting the letters RE3JLDD 1 by 1 3JLD reair, NGMG this deterrent to make, triplet. Ord Raj1 has EDCAP Raj1 has DCAP Raj1 has CAP the conclusion fran TLLE ABA EE. Where C set is Eddie obvious. Insertion or deletion of 1 or 2B, says changes the reading frame from the P ointment of insertion or deletion. Insertion or deletion of UUEE or its multiple bases. Ceology insert or delete 1 or multiple codon hence 1 or multiple amino acids, ND reading from where names unaltered from the P ointment onwards. Such mutations are referred to, SLE, AMSHLFTLRU, each one CR deletion mutations. This F forms the genetic basis of proof that CC, DOM is, plit, did is read in a contiguous marmer 6, 2 tRNA 10 adapter molecule from the very beginning of the proposition of code. It W, S clam to Francis or TH, T there has to be, rechinism to read the axt and also to link it to TLLEA. Amino acids take house ed. Amino acids have no structure LSPEC3 lydies to read the code weakly. He postulated the presence of an adapter molecule that would on one hand read the code, and on other hand would bind to specific amino, acids. The tRNA then called soluble RNA YSKNCJT, TJN before Lee genetics CW is postulated, however its role LAUOS, TO.V, F:1 FIS. Your 6.12 tRNA allele adapter molecule AS and 3D8 DRM, DQLWS, SS. Net much later tRNA has an anticodon loop THS base-umblarenturido the code.mct at ALSO has an amino acid acceptor and to which it binds to amino acids. tRNAs are specific tor ARCHRNino, SID figure 6, 12, for initiationaris. Micthor specific tRNA TLAD is referred to AS in it later tRNA. There are no tRNAs that for stop codons. In figure 6, 12 the second, Y structure of tRNA has been depicted that looks LLL, E, early, F. In actual structure the tRNA is, comp, CT molecule which looks like an ERDL 6, 1FR, Ansla Tim, Ilansla urine refers to the process of irritilion of amino, EADS to form, polypeptide Phe 6. 13. The older and sequence of, mind, SIDS are redefined by Lee sequence of BOS and Lee mRNA. The amino acid S are joined by, bond which is known, S, peptide bond. Form of, peptide bond requires energy, therefore. In the first phase its alpha amino acids are aided in the presence of ADP and linked to their cognate tRNA, process commonly C, lead, S charging of tRNA or aminosyl U1 of tRNA to be Rho specific. U2 such charged tRNAs, brought close enough the formation of peptide bond between them. Molecula, S satisfy, hence MJUDB favored energetically, the presence of a catalyst would enhance the rate of peptide bond for MTIL, the cellu, M factor responsible for synthesizing proteins as ribosor. The ribosor consists of structural RNAs and about 80 different proteins HL its inactin state. It exists as TLO subunits. A large subunit and a small subunit. When TLLE small subunit encounters mRNA process of translation of the mRNA to protein begins. There are two sites in TLLE large subunit for subsequent amino acids to bind to in TLL.US. Be close enough Fig. E6.13 and U1 to each other for formation of a peptide bond. The ribosome also acts as a catalyst. 23 Ser9 bacteria is the NCERT enzyme for the formation of peptide L, and at the RMSLation LWL.IT and mRNA is Lee sequence of RNA TLL, at is oanked by the store codon AUG and the stop codon and cadesper a polypeptide. An mRNA also has SOR additional sequences O, at or not TRMSL adect and are referred as on-Shiansl, red regions UIR. The UTRs are present at both 5 and before start codon and at 3 and 3 at their stop codon. They are required for efficient TRMSLation process. For initiation, the NL, OSOR binds to the mRNA at the start codon AUG that is rec, GN, said only by the initiator tRNA. The RIL, OSOR proceeds to the elongation phase of protein synthesis. During this stage compler.es composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by Roman-implantory base pairs with the non-Shiaidin. Then Bosorovas Froenkadon to codon along the mRNA amino acids are added one by one translated into polypeptide sequences dictated BDN and represented BJ1IF, J the end of release. Acror binds to the stop codon terminating translation and releasing the complete polypeptide from the Bosor. Lion of gene pression refers to a ribroad term that may occur at famously LZA, considered that gene pression results in the formation of a polypeptide. It can be regulated at several levels. In UCA 110 test the regulation could be ordered at I transcriptional level formation of MARI transcript. U processing L relation of splicing, 3 transport of mRNA from nucleus to the cytoplasm. IJ translational level. Physiology the genes in a cell are expressed to perform a particular FWL XII or a set of functions. For example, if an enzyme called beta-galactosidase is S-jalphysese BJIE cow it is used to AIULJ7 hydrolysis of a disaccharide lactose and fructose and glucose TLLE bacteria use lean as sow. Set of energy, hence if Lee B actor to CB not have lactose to row 1D them to be utilized Phe, junior energy source. They LD no longer require the synthesis of the enzyme beta G3 lactose at 3 cent, their Phe, JRE, in simple terms. It is the metabolic Fys-Cochrane when rental conditions 3T relate the expression of genes, the development 3rd deferrin U, Cheyenne of Embage, L into 3 delta Q3 nisrans 3 re also 3 result of the CC, or denoted regulation of expression of cell, or alsets of NES. In Proc 3J10 leuntal of the R310 transcriptional in U, Cheyenne is the pressionerval site FBR control of gene, pression. In data transcript T on WLIL the 3CTI video of RNA polymerase 3T3 given promoter is in turn Reg3 TED by XII with 3 accessory proteins, which affect its 3 ability-to-recognize stored sites. 1 is irrelatory protein C3 and 3CT both positively 3 active 3 towers 3 neg3 tidally repressors. The 3 accessibility of promoter regions of PROC31J, tick DNA is in MMYC3 says Reg3 TED by the inter 3 XII of proteins with sequences termed opera towers. The operator region is adjacent to the prom.ter elements in NDSD operons and in those cases TLLE sequences of TLLE operator bind. Data repressor protein. Each operon has its specific OPR3 lower third specific repressor. For example, !Acaparotor is present only in the LAC3 NIT intero CTS specific 3LLJ with LAC repressor 6.8 L1 LTE LAC operon the elucid, Asian C1 Li LAC operon was also a result of close association bet 3 grams neticus FL3 newest 3 cob 3003 B0 chemist. J3, Zdudari TJE first toluci, low T3 Trumskmshanaloy Ergul 3 dit system in la copper and here acre forestal 3 tos. 3 polyestronic structural genes Regal 3 Tebi 3 CULIUI pi oil or integalacri as such there NT is edi call IUUI and B actor and is referred to as operon. LB name few such ampos lac operon 7 popernera othlerin ho, espronul operon etc. The lac operon consists of 1 Regal 3 tori gene the gene here the term D, S not referred to inducer. R atler it is reartfect from Li lol or DNHI but to third 3 structural gene CG thirda, the gene codes V, junior the repressor of the AC othler oil. The Z gene codes V, Trebetagmtasa 3 set 13 GoL which is primarily responsible for the HJ, irolysis of the dis3 CCH3 NDEL3 ktose into its NDFIOIE IW its. Ktose and glucose, the G gene seeds FBR pemias which incur, as is permeability of TLLE cell to page G3J, actosides. The A gene angst transacetylase, hence ali TLL regene production lac operon 3 rear acquired phi, trimetabolism of lactose. In RMST operon 3 as well, the genes present in the othler oil 3 we needed together to function in the SMNE or related metabolic page 3 THW3J figure 6. 14. Molecula, stasis of inherence TIPIL, 7 I IRC pot dota QTHC5L3D, RNA PLBO, FB. FUO, HLRT, W6 ram lactose is Li substrate tor Li enzyme beta G lactoside, asdodan. It retas S switching on off of the Kberken, hence ilus S inducer HLTLLE. Absence of preferred. Carbon sus such as glucose if lactose is provided in the galfri do not the B, tina the lactose is trans, or ted into the cells through the, XII of pair, so remember. At a low level of E, pre-cyan of AC operon has to be present in the cell. U the tiri art leftwise lactose C armor enter TLLE cells. The lactose then induces Li operon in the following manner. The repressor of the operon is incised. U, the, teruns dilute L from the I gene. The repressor protein binds to the OPER, tor egg, on of the operon, ND prevents RNA polyerror, se from transcribing the operon. In Li presence of an inducer such as lactose or 3 lol, actose Li repressor is inactivated by interaction with TLL. Li inducer, this allows RNA polyerror as access to TLL pronter and transcription proceeds figure 6.14. Essentially regulation of lac operon can also be visualized as regulation of NCERT synthesis by its SUBSTR8. Remember, glucose or galactose not ODS inducers for La, C operon, can go ding for how long the lac operon lux, uld be expressed in the presence of altos regal, XII of AC operon by repressor is referred to, S negative regulation. Lac up 3 ron is under control of positive regulation. As well but it is Li scope of discussion. At TLL is level. In Li preceding sections you have learned TLL it is Li sequence of bases in DNA that determines the genetic INF formation of a G, then organism in othler L or S. Genetic makeup of .M organism or .M individualize in TLLE DNA sequences LF jewels differ. Then their DNA sequences should also be different, at least at source places. These assumptions led to Li's quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clue any piece of DNA and ability of simple and fast techniques FBR determining DNA sequences. ARIORNBE's project of sequencing human genome was launched in the year IG-0. Human Genome Project HGFJ was collect a mega project. You can imagine the magnitude and the requirements FBR the project if we simply define the aims of the project as follows. Human genome is said to ha, approximately 3 XI0 BP. And if the cost of sequencing required is US 3 per BP, the estimated cost in the beginning, the total estimated cost of the project would be approximately $9 billion USD. Further, if the obtained sequences were to be stored and typed FBRM in books, and if each page of the book contained I000 letters and each book contained I000 pages, then 3,300 such books would be required to store the INF formation of DNA sequence from a single human cell. The enormous amount of data field to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HGPW is closely associated with the rapid development of a new area in biology called as blot format S, G, als of HGP. Sorry of the important goals of HG were as follows. I identify all the APRO immediately 20.000, 25.000 genes in human DNA. I'll determine the sequences of the 3 billion chemical base pairs that now up human DNA. I'll store this information in databases. I improve tools for data analysis, transfer related technologies to other sectors, such as industries. V address the ethical, legal and social issues LC that may answer from the project. The Human Genome Project was a 13-year project coordinated by the U.S. Department of Energy and the National Institute of Health. During the early years of the HGP, the One Elkhorn Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China and others. The project was completed in 2003. Now let's about the effects of DNA donations on Gindi, which can lead to real new ways to diagnose, treat and prevent the thousands of molecular satisfy hence disorders that affect human beings, besides providing clues to understanding human biology. Learning out non-human organisms DNA sequences led to a known 3RSL3 of their natural capabilities that can be applied to soiling challenges in health care, agriculture, energy production and true remediation. Many non-human organisms such as bacteria, Yesankin have DTSLE9 and SA3 leaving non-pathogenic nematode, Drosophila fruitoi, plants and Arab, Dopses etc. have also been sequenced. Methodologies. The Reproduction note. 2 major approaches. One approach focused on identifying all the genes that pressed as RNA referred to as XPre56 at sequence exists. The other took the blind approach of simply sequencing a whole set of genome that contained all the seeding and non-coding sequence, and later assigning different regions in Lee sequence with left XII a term referred to as sequence and not N. For sequencing the total DNA from a cell is isolated MCT converted into RMCBMFR agrants of relatively smaller Cs. As recall, DNA is a very long polymer, and there are technical limitations in sequencing very long pieces of DNA and cloned and suitable host using SPE3 lysed vectors. The cloning resulted in amplification of a CH piece of DNA FR agrants OTLLAT. It subsequently could be sequenced. Nith AOS Abacomi used hosts were bacteria.mcde street.md. The vectors were called D as Lactobacterium typhiel Crohn, SORS, and Yak-L Eastmtalicial chromosaurs. The 4 TS were sequenced using automated DNA sequencers that worked on the NCERT of a method developed by Frederick Sanger. Remember, Sanger is ALSO credited for developing method for determination of amino acid sequences in proteins. These sequences TRL and 3.1 TNG set on some overlapping regions present in TLL, and this required generation of merlapping 4 TSFBR sequencing. Alignment of these sequences was HUMA-NLY not possible. Therefore SPC3 lysed computer BOSED programs were developed. Figure 6, 15. These sequences were subsequently encoded and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of Lee 24 Huma and chromosome.es22a utosaurs.md. X.mdy to be FIS, W, E6.15a representative D1AGRA row of HUJMN, project. Ceology sequenced. Another CH3 linking T, SK was, S signing the genetic and physical maps on the genome. This W, S generated using information on polymorphism of restriction and tonicles, recombination sites, and some repetitive DNA sequences known as micros, telites. One of the plications of polymorphism in repetitive DNA sequences shall be explained in next section of DNA fingerprinting. 6. 9. 1 Salient features of human genome. Serif TLLE salient observations drawn from Hum Genome Project. Flows by the Human Genome contains 3,164.7 million nucleotide bases. ILDA, RHGME consists of 3,000 bases but sizes which Joy, with them just MN human gene bank dystrophin, T2.4 million bases. 3. The total number of lineages is estimated, T 30.000 much lower than pre. 2 estimates of 80.000 to 1.40000 lineages. Almost LL 99, 9% nucleotide B, Caesar X, Tree the Soren. U people I the functions are one known F former 50% off discover X genes. J less than 2% of the genoroids for proteins, the REP, TED sequences make up very large portion of the human genome. Repetitive E sequences. R stretches of DNA sequences to lat. R REP, TED managers sorters 100 to 1000 teras. They are thought to H, then O directed in functions but they shed light on chromosome structure and evolution. IOC per cross or I has Ross genes 2968 and the past thief street 231 I, as scientists have identified, about I. 4 million LOC, Cheyennes where SNP-based DNA differences SNP's single nucleotide proj molecules and proromicate. Data SNPs 1 occur in HUT IWLS. This in one formation promises to revolutionize the processes of finding chloroform locations for disease-associated sequences and TR single history. 6. 9. 2 applications and future challenges 10. NRRE, Rungchil L. Knowledge from the DNA sequences WUI define research through the corning decades leading to our understanding of biological systems. This enormous task will require the expertise and creativity of tens of LIOS and S of scientists from vain disciplines in both the public and PNV, test sectors worldwide. One of the grant, TestImp, CTS of HA, NEHG sequence may well be enabling, radically new, approach to BCal research. In the past researchers studied 1 or, few lineages, T, time, with whole genome sequences and new high throughput. Mole, call UUDIs of an HFL, NCF technical class, can approach questions systematically and SILA MCH.R scale. 1 bacon slew I altyl sin a genome lore 011 e altyl transglobin iular TE or organ or tura. Or how tens of cousins of gems and proteins WCRK tog Tyler and intercar are, acted networks to orchestrate the chemistry of we. 6. 10 DNAF, OOELLPLTLLMA is stated in title preceding section, 99.9% OIB, says sequence or wrong her, WL1SUE same. Assumergy human genome A, 3XJFBPH Mangba, Sokwanos would there be J Aranos it is USA differences in sequence of DNA which make every individual can UEIR phenotypic appearance. If one aims to find out genetic differences between two individuals or among individu 3 ls of a population, sequencing the DNA every tina would be a gan, pensive, task LRR, a giant trying to compare two sets of 3x10 base pairs. DNA fingerprinting is a quick way to compare DNA sequences of any two individuals DNA 6 gerpinting animus identifying diffly, renses in some specific peg, CILs and DNA sequence called as repetitive DNA because in USA sequences a small stretch of DNA is repealed maniditn. As these repetitive DNA are separated from bulk genomic DNA as deerrant peaks dirt and ginsel, Y-grodenal centrifugation. The bulk DNA for new a major peak and Tyler small peaks are referred to as satellite DNA, depending on base composition A, G. CNCB.languatsegnanella number of repu units tile satellite DNA is classified into many categories, such as macrosatellites, mini, satellites, etc. These sequences norm 3LLY do not code for any proteins, but truly form a large portion of human genome. These sequences show high degree of polymorphism and LBRM tile basis of DNA fingerprinting. Since DNA from a viral source such as blocks the hair, oblique skin, bone, saliva, permet grown individual show tile same degree of polymorphism. UE become very useful I2 and Kaiwan tool in forensic applications, further as UE polymorphisms are enhantable from parents to children. DNA effort nung is UE basis CC paternity testing in case of derputes, as polymorphism in DNA sequence is UE basis of genu CMAP, NCERT main genome sweat as CC DNA fingerpung. It is as in UE we understand what DNA polymorphism means in simple terms, morph variation at genetic level is due to mutations. Real different kind of mutations and clear effects that you have ready study, LL in chapter 5 and in UE preceding sections in this chapter. New mutations may arts in M individual either in sornox cells or in tile germ cells that generate gametes and sexui reproducing of one. Organisms, if a germ cell mutation D, S not seriously impaired individuals. Ceology ability to have offspring who can transmit the mutation it can spread to the other members of population through sexual reproduction. Allelic run recall the definition of alleles rem chapter 5 sequence 03 the nation has traditionally been described as a DNA polymorphism 2 more than one VMNT allele. Locus occurs in human population v. Little. Data frequency greater than 0. 0I in simple terms 2 unheard of the LEMUDAton is observed in data population at high frequency. It is referred to as DNA polymorphism, the probability of such venation to be obstinate in including DNA sequence old. The higher as mutations in TLLE sequences may not have any immediate impact in an industrial IVDUL's replicability. These mutations keep on accumulating generation after generation and form one of the basis of vulnerability polymorphism. There is a onity of different types of polymorphisms ranging from single nucleotide CHMGE to very large-scale CHMGEs. For evolution.md speciation such polymorphisms play a very important role and you will study these in details at higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used a satellite DNA as probe that shows very high degree of polymorphism. It was called as variable number of DMDM repeats INTR. The technique as used earlier involved southern blot hybridization using radiolabeled INTR as a probe. LT included isolation of DNA. Digestion of DNA by restriction endonucleases. 3 Separation of DNA fragments by electrophoresis. IV transferring blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. The hybridization using labeled VN1R probe and II detection OFHABLD set in fragments by autoradiography. A schematic representation of DNA fingerprinting is shown in Phi 6.16. The INT belongs to a class of satellite DNA referred to as mini-satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome. M individual. The numbers of repeats show very high degree of polymorphism as a result. TLLE CZ of the NTR varies in CZ from 0.1 to 20 kilobytes. Consequently, after hybridization with INTR probe, the autoradiogram gives many bands of differing CZs. These bands GE a characteristic pattern, or an individual DNA figure 6.16. It differs from individual to individual in a population except in the case of monozygotic identical twins. The sensitivity of TLLE technique has been increased by use of polymerase chain reaction PCR. You will study about it in MITCHI, TIIH2C, LU III 18A, 16 of I, figure 6.16 schematic reprise, NTUON of DNA FNCHET, reprinting, few representa. Chromosomes have been shown to con D1FFE, rent copy number of VN1R, for Ilias Sakavante, RSTMD1 and D1 Farrant color schemes ha. Been used to trace the ONGM of each BMD oligot, T1E2 left paternal mate, RN, 3LL of a chromosome contain D1FFE, rent copy numbers of VN1R. It is C1 amulatilia B pat, RN of DNA from chromosome, RE matches with LD1V1 dual B. Melrot with a CH3 trill. O, sequence Y, DNA from a single silicon to perform DNA fingerprinting analysis, in ACL, listen to application in forensic science. It HSM, 1CH Wichterf cation such sync teternally population act genetic diversities, currently. Many different probes are used to generate DNA fingerprints. Dialog. Summary, nucleic acids long polymers all nuclei, UDs, while DNA stores IL4 and CDI and RNA mostly helps 1 anti-archmster.mle, express 1 on of INF formation. Though 8H DNA RNA both for Kuanosmatin, but DNA being Chero 1 Calistrutor.RU more stable 1S a better na UE material. How, RNA is ilia first to UNDNA dirt.td RNA and LE howling, RK of the doublister, dead hell 1 calstruab, reoff DNA LS the hydrobonding between ilia B from opposite STRMDS. And LE rule 1S ilia hypos MTH1 hernu, E through 8H2H boards on, go, R4 now with cytosine new out Louis H bonds. This makes one strand complete, and Y2 Eli oilier and LE-DNA replicates similiancer, AU, eddy the process LSQ, IED by the corn plui, NWYH boarding. A CNET of DNA that seeds F or RNA may 1 and the sun plus up termum be rethread as netherton transcription O. One of Ilya strands of DNA acts a template to direct the synthesis of corn plementa, RNA and backtort. The TR-script mRNA 1S hum C3 and Duke GBTR8 at HL Ryotsilia go, NER1S split. NLECO, it sequences E, ZONS, me interrupted by wrong coding sequences and trans HL trans me rate, MO, D exons me joined to produce FWLC UNL RNA by splicing. The messenger RNA cont MNS the base sequences ILIT me read in a combination of ILI to RMK and flit MU code 20, DF or an M1, L, 1 is less than 1. The RUC code LS redog, 1 and on the PRV apple all corn PRmentordy by RNA THL, CISOS adapter molecule. 1 horospe, HCtRNAs L or EYMO, L3C1D, the tRNA BU DS to SPIFT Gruno AC1D, DRE, ND pairs UVA H bond Wily codes on mRNA UVA X multicodons. NLE side of TRU1 Pro 1 N Sinu, S, is 1S ribosomes V hitch bind to mRNA mal PRO VID platform F4J of MO, L.ZC1DS. Oriavillium acts as a catalyst for peptide bond formation which 1S and E, play of RNA enzyme ribosfman. Ntion 1S apruasolid.OH, LM on RNA1 dicawaning iliot BFE began MOTM, LRNAS use transcription translation RNRGD Ry pros, says. These ought to be tightly regulated. Regulation of TRCMSCRTPU on 1SLE prim step F regulation of gear expers cyan and bacteria more than 1R, ELS or on D together unregulated in UNPLS called OSOP, UNS. LCR, C on 1S the prototype upper on 1N backed, RT3 which O, dash F or NSS responsible, F or metabolism of lactose, the upper on 1S regulated by TL, E amount of lactose 1N TL, E medium V he, read the backed, R1 ME grown. Therefore, you use regulation BW, metas regal 311 on of enzyme sinu, ests by its substrate, human F, TNome project was a mega project ella.med to sequeroeoe, ribase 1N hum CM. Genome NUS project has yielded much new information, many new MES avenues have opened up as a consec 1 arrow of ULE project. DNA fulgen, RPRTN 1S a technique to BND out OMECD ions 1N first 1 bidu, 3 LS of a population at DNA 10, L. It works on the protroipolo polymorphism 1N DNA sequences. It has immense APL1 Ca11 ons 1N allele field of 4, CCNO. Ariok Biodiversity Lutinary Bilog. Exercise ES1G up U, L, Lowing is new, Ermis hoses on EN, JD, Sitl Nerwan him 1 ni, Gualasine. Ur 3C11 Edge, DUYT, 2LF a double street ARSC SEDIAN AIS 20% of SA2 NU, per 011111. Of Clarin 1 NU, DNA 3, if the, W, N, E of or Easlrani of digit TSWRTTTN if all of S. Adicat ket garo C Eric, Eslahilto down the sequenal of complamer, or ACT, 11N. A, Grekland 4, if U, SAC UE, row of the O, Dingstran, I1N A1R Anscripure on UNLTTSW11TN. L, Uaus, Adicat ket garo C Eric, ST white down the sequence of mRNA5, which P, operative on AWEX led Watson dot and Crick low HJP, I hassles, NL, conjunction Lajva mode of DHA replication X plow. E depard and UPM the DEM rat nature the learned glycoma or RTF.ANO detour of, Leocads doftlet II, NA or N, IJ1. HSL though typos of M Leocad poemerases 1, 1 bo CBD his B. Rod chose ly errant lit bet ONDNA ont POD TMTROXPOMOMTLF, OT that DNA is the RN, 18. DR, tape between U, L, LLO83, Flurpo UVA DT1A and S tele DNA BRNR11A and tRNA01 LRNPL test retar, 1 edge, deoxylstrand G, UST. 2 Watson rolls of ribosome dirt and trans BU110, in DHT midlam where ECDC was GTOWLRLG lactin Wu added VH1CH Durduk, TBLC, TC operon 11 and V high does B operon shutdown simplema.daft.TU1 of Brados 1 and the medium 11, I. Ogwana 1 and LN1 or 2 Laneju, Funkman of the L, 1 gel NATO, Piamot BLTRNA lay colon 100 NS12. 1 high as though human jerome PCOE T called a Mia P, Jockle 13, 1 highs DNA FLNGRP it's up Cotland 14. Nali Duk Bu, Folio data TRMSCRTPT John B Polymorphy SMO feed.bu on D Bio, NNX. Chapter 7 Evolution 7.1 Origin of Life 7.2 Erbt Cheyenne of Lee, Forms A1ML 7.3 Pat A Reth Evidences for EVDAT Tickn 7.4 VTS at Op var MJI Dickn 7.5 BT Oat Call Evolution. 7 FEMEC, Mismoff EVDAI.KN 7.7 Horveen.S Print Test 7.8 Abri F Cotton for the OO Cheyenne. 7.9 Ori GTNAND Evolution of LVBN Evolution Audi Biology is the study of history of life forms on animal. What exactly is evolution LB on rareston TLLEC sin orang twanatlathanakam, for millions of years on Earth? Rust have understanding of the context of life that is, Eoluian offereth of Su, RS, and Enrichtively WLI verse itself. What blows is lead longest of .dali construed .md conjectured stories, this isly story of origin of life and eoluion of life forms or big D1 or sitting planet Earth in the context of evduion see Earth and .mst the background of evoluion of universe I self. When AE looked out at stars on a clear night sky we are in W, while looking back in IR, SLR distances are RRE, short in light years. What CIJ, day is, an object whose emitted light started its journey millions of years B, CK and from millions of kilometers, W, Y chung our eyes now. However when we see objects in our immediate surroundings we see them instantly and hence in TLLE present time. There or when we see SU, RSW, the re-peeping into the P, street, the origin of life is considered a unique event in the history CW1%. The W1% is St. Rella MLY speaking. Biology in the earth itself is almost only a speck. The universe is very old, almost 20 billion years old. Huge clusters of G3.13 Xizumpres the WLIers, GQIS can't run stars and clouds of gas dust, considering the size sea livers. Earth is indeed a speck. The Big Bang Theory attempts to explain 3M Taustian of livers it books off S, killer huge explosion WLBLE in 563.1 terms. The universe EXP and hence the temperature RECBWN, H-drogenan, HEMBNEC, I sometime later. The gases und Entwilder gravitation for the G3.13 size of the present-day livers, in the solar system of the Renilquai Gaia, Y. Earth W is supposed to H18 BNE, I about 4.5 billion years back, there W is no ATM sphere on ARTY Earth. LLB Terra VAP 3 UR Retlul, Carton Dio, ID. And 3 Phil LJJ1 I rail last from 10 MSCO erect the surface. Tile LNRIs from the SWL broke up water into U, Ulgen and Xygen the lighter H escaped Oigen combined with ammonia RRE Hain to F form water. Company others, the Oscan layer was fronered, as it A, Olay the water that fell as rain to 611 all the depressions F formations. IF appeared 500 million years oiler the Fermati, LNCERT that is, Alhistar billion years back. Did life core from outer space? Our scientists believe TLL it came from outside. Early Greek thinkers thought willets of life called spores were transferred to different PL-mets including Earth. PANSP, RMIA, is still a favorite idea to some astronomers, for a long time it W is also believed that life came out of decay, and rotting matter like straw mud etc. This was the theory of spontaneous generation. Louis Pasteur by careful experimentation demonstrated that life comes only from pre-existing life. He showed, that in pre-sterile said flasks life did not come from Colette East while in all your fiascapunta air, new H-ving organisms arose from killed. Ye asked, SP111 Lmenis General and Leary W is dismissed once and FBR all have, ever TLL was did. Not answer how Lee first life FBRM Karari on Earth, Operan of Russia H3 Danioff England prop, said that the first F form of life could come from pre-E, ising non, leaving organic molecules e.g. RNA protein etc. and that F formation of life was preceded by chemic 3.1 evolution, that is, formation of diverse organic molecules from inorganic constituents. The condylions on ARTH were high-temperature volcanic storms reducing atmospheric toming CH4, NH, etc. In 1953, S.I. Miller. An Abertkin scientist created similar 3R conditions in aleatory scale figure 7. He created electric discharge in a closed flask containing CH4, H, NH8 water P at AOO, C. He observed formation of amino acids, in CERNL 3R experiments others observed formation of cigars. Nitrogen bases P, Grant and fats, analysis of redorituten 3LSO read Alex SME1R compo WLDS industrial acating TH at SME1R processes. Or occurrence elsewhere in space. With this limited evidence the first part of Lee conjectured story that is, chemical evolution W is M, re or less accepted. We have no idea about how the first self-replicating metabolic capsule of life arose, the first non-cellular F forms of life who would have originated. M. Leaklet LLLTT Orpa, KT. FIS, your 7.1 D1 Agtamaok representation of Miller's experiment 3 billion years back Halo OUD Hale bin on molecules RNA, protein. Fblisic des etc. These capsules reduce their molecules perms. The first cellular, RF form of life did not possibly originate till about 2000 million years 3.0. These air probably cine cells, all life forms were in W ateranverons, NT only, this version of A. B Genesis i.e., Lee first Vermont life arose slowly through evolutionary force from non-Lee molecules is accepted by majority. HDA once toward how the first cellular forms of life could that evolved into the complex BD, city of TC, day is the fascinating story that will be discussed below. Then a religious leader, Tor tells us, about the theory of special creation. This theory has 3 canonical icons. 1. U. Li, Eng organisms species or types TLLAT low, ECTCD, 3. Y. Were Crade Tech Day as such. 2. TLLAT Li diversity was ALWIs TLLE say RRE since Crade Tim and JILT Li say future also. 3. The Earth is about 4,000 years old. All these ideas were strongly challenged during the 19th century. Based on absurd ions made Durton, see Myagin, sail a ship called HMSB around the world. Darwin concluded at existing living forms share similarities to varying degrees not only and build themselves but, also with life forms T existed millions of years ago. Many such life forms don't more, there had been extinctions at different life forms in Li. Biology engineers go by just, new forms of life, rose, TD1 different periods of history of the Earth. There has been gradual evolution of life forms, any popul, Cheyenne has beult in WLAIon and terrace ICS. Though terrace ICS which enable sort to ser, LE better in natural conditions climate B, XT physical factors, etc. would outbreed others TLL at are less anctov, to serve in one other such natural conditions. Another word used is fitness of TLL individual or population, the sixness, according to Darwin. Refers ultimately, and only to reproductive fitness, hence TLLOs or better fit and there ain't. Leave your progeny not these TLL are for, TJIL serve in your and hence, reselected by nature. He called it natural selection, completed as a rechmism of lion. Let us so remember TLL at Alfred. W.L. is a naturalist who the Orkton Malay archipelago had also correlated to similar conclusions around. TLLE Saratiri, in Dukov set of Tiri apparently new types of organisms are recogress, all the extinct life forms share similarities and share COIIU ancestors. However TLLE's ancestors were present, different periods in the history of Earth epochs periods, and s. The geological history of the Earth closely correlates with the bioical history of Earth. According to and permissible conclusion is TLL at Earth is very old, not house, and of years, as was thought earlier but billions of years old. The evolution of life forms has indeed taken place on Earth has or from my quarters. Fossils, remained of hard parts of life FBRMS TBWLD in rocks. Rocks form sediments and a cross-section of Earth's crust indicates TLL arrangement of sediments 1 mer TLL outler dunang TLL long history of Earth. Different, rock sediments unan fossils of different life forms who lie died during the formation of the particular sediment. Some of TLLM appear similar to dem organisms figure 7, 2. They represent tinct organisms for example. Dinosaurs, a study of fossils in different sedimentary ages test the geological period in which they existed. The study shows life forms net over ire and certain life forms are restricted to certain geological ire spans. Hence new forms of life have arisen, different years in the history of the Earth. All this is called paleontological evidence. To remember the ages of the fossils are related to ued the method of Rodial, OD, IOE data 9n. The PS behind the PROBI comparative anatomy and morphology shows similarities differences among organisms of today and those that existed years ago. Such similarities can be interpreted to one Alderst, and Whitler Corner JN. McKesters v. Air shared. Or not, furry, ample whales bats, cheetah and human. ULWLL share similar to in Thep, Temeth Buzaf Brelins Re 7.3b. Though these F4 perform different functions in these animals they have similar anatomical structure. U of them have. Ceology Fig, your 7.28 moily tree of dinosaurs UEU lived modern-day Cowlta, RPML org, amsums like crocodiles Malbudsaurus. Aradius ulna carpals reticorpals and pH3 alongian lear forlums, hence in Lee's animals. OLSR structure developed along different directions due to adaptations to different needs. This is divergent evolution. And TLLE structures. Are homologous. Homology indicates common micastry 0 T her examples. Are ertibrati. Arts or brains, in plants also. The thorn and tendrils of Bauger Alpho and Kuat, to represent horology figure 7.3 homology is based on divergent evolution whereas 3J13 allogy refers to assiduity on exactly opposite. Wings of butterfly and of birds look alike, they are not anatomically similar. Biologian structures though they perform similar functions, hence ATL3 logos structures mere result of convel and Lutland different structures EOLV, and for the SOR function and hence having similarity. Other examples of N3 logi are the eye of TLL octopus MD of RN3 anal 1 als or the oipers of penguins and D. Elephants. One can say that it is the similar habitat that has resulted in selection of similar adaptive features in different groups of organisms but Taura. D. The SOR function, sweet potato root smectification and potato stem modification is another lit example FBR and 3ln. 161, in the same line of argument similarities in proteins and genes. Performing a given function. The wrong LLDers organisms give clues to corn JNMLM chemistry. These be a chemical similarities JP. Into the CRRESH3 red. Ancestry. A structural simil 3 riddies. A wrong diverse OQ, MLISRNS. Man has bred selected plants and animals FBR agriculture horticulture sport or security. Man has domesticated many low-lyld NRIWs.MCT crops. This intensive breeding program has created breeds that differ from other breeds, for example, dogs, but still are of the SOR group. It is rare that if within hundred of years man could create new breeds. Could not nature hide done this over millions of fires? 11, B. V. I. Bet. Another interesting observation supporting evolution by natural selection comes from England. In a collection of FLUG 7.3x and play of homologous organs in a PL, NTS, NDB Anring, LSNDTLLS made in 1850s. That is, before industrialization set in, it was absent that there be mere white winged myths on trees than DSRL, winged or L3 nisect NDTLL. SHCN ever in Lee collection comment out from Lee Sane area, but after industrialization, that is. In 1920 there were mere dork-winged NDTLLS in Lee Sane. Area that is, TLLE proportion W is reversed. The explanation put LBRTHFBR TLIS observation was that P. daters will spot NDTH. Against contrasting B background. During post-industrialization PERT, TLLE tree TRWLKS becare D, arc due to industrial smoke and soots. Under this condition the Win LD moth did not. 1-2 Biology. A B figure 7.4 figure showing white-winged moth and dark-winged moth Melanese on a tree trunk A in unpolluted area. B in polluted area survived due to predators. Dark-winged or Melanese moth survived. Before industrialization set in, thick growth of almost white-colored lichen covered the trees in the background. The white-winged moth survived, but the dark-colored moth were picked out by predators. Do you know that agents can be used as industrial pollution indicators? They will not grow in areas that are polluted. Hence, moths that were able to camouflage themselves, that is, hide in the background, survived. Figure 7.4. This understanding is supported by the fact that in areas where industrialization did not occur, e.g., in rural areas, the count of melanic moths was low. This showed that in a mixed population, those that can better adapt, survive and increase in population size. Remember that no variant is completely wiped out. Similarly, excess use of herbicides, pesticides, etc., has only resulted in selection of resistant varieties in a much lesser time scale. This is also true for microbes against which we employ antibiotics or drugs against eukaryotic organism cell. Hence, resistant organism cells are appearing in a time scale of months or years and not centuries. These are examples of evolution by anthropogenic action. This also tells us that evolution is not a direct process in the sense of determinism. 'Tis a stochastic process based on chance events in nature and chance mutation in the organisms. 7.4 WHLIT1 Napt v. Asian. During his journey Darwin went to Galapagos Islands. There he observed an amazing diversity of creatures. Of particular interest, small black birds later called Darwin's finches amazed him. He realized that there were many varieties of finches in the same island. All the varieties, he conjectured, evolved on the island itself, from the original seed-eating features. Many other forms with altered beaks arose, enabling them to become insectivorous. Biology Run finches FIRE 7.5. This process of evolution of different species in a geographical area is starting from a point and literally rotating to other areas of geography habitats is called Yuan. Darwin's finches represent one of the best examples of this phenomenon. Annullary. Ample as OSTR3 Li 3 N marsupials. A number C1 marsupials EH different from the other FI7, 6 evolved from an ancestral stock. But all V, little and Leoster 3 Li 3 NISL and congenant, when N, or TLL and 1. Adaptee irradiation appeared to have occurred in an isolated geographical area representing different habitats. One can call this convergent evolution. Anteater Numma V, lemur biology. Placental mammals in Australia also exhibit adaptive radiation in evolving into varieties of such placental mammals, each of which appears to be similar to a corresponding marsupial, e.g., placental wolf and Tasmanian wolf marsupial. Figure 7.7.5 Biological evolution by natural selection, in a true sense would have started when cellular forms of life with differences in metabolic capability originated on Earth. Figure 7.7 Picture showing convergent evolution of Australian marsupials and placental mammals. The essence of Darwinian theory about evolution is natural selection. The rate of appearance of new forms is linked to the life cycle or the lifespan. Microbes that divide fast have the ability to multiply and become millions of individuals within hours. A colony of Bacteriaceae growing on a given medium has built-in variation in terms of ability to utilize a feed component. A change in the medium composition would bring out only that part of the population say that can survive under the new conditions. In due course of time this variant population outgrows the others and appears as new species. This would happen within days. For the same thing to happen in a fish or fowl would take millions of years, as lifespans of these animals are in years. Here we say that fitness OFB is better than that of A under the new conditions. Nature selects for fitness. One must remember that the so-called fitness is based on characteristics which are inherited, hence, there must be a genetic basis for getting selected and to evolve. Another way of saying the same thing is that some organisms are better adapted to survive in an otherwise hostile environment. Adaptive ability is inherited, it has a genetic basis. Fitness is the end result of the ability to adapt and get selected by nature. Branching descent and natural selection are the two key concepts of Darwinian theory of evolution, figures 7.7 and 7.8. Even before Darwin, a French naturalist Lamarck had said that evolution of life forms had occurred but driven by use and disuse of organs. He gave the examples of giraffes who in an attempt to forage, Evolage 110 and leaves on tall trees had to adapt the elongation of their necks as they passed on this acquired elongated neck to succeeding generations. Leufeses lolio with HRSE to acquire long necks, nobit balilatus conjecture NEMXE. Isiolation aprosu the result a process the W, RLD we see in Emelinden, makes on the, uxis teresa emluan. While we describe low storia this D we decrypt beoiu on a prey, on the other hand when we describe the sterile on earth, we treat evolution as a consequenza a process calec natural selection, vol, are still not f clear will regard evolution in natural selection as processes associated resalto, WILTNCJWN processes. It is possible that the work of Thomas Malthus and Populaticans influenced Darwin. Natural selection is based on certain observations or tactual fogs awanillo. Natural resources are limited p. Pulatlands are stable in size except I. R seasonal fluctuation rare versat p. Pulatland vary ch eric lertsticks and tacno tvo individuals are al, even though they look softly ishally similar. Rosta variations are inhertful etc. The tact that theoretically population C8 will p. Extneral ife, Arab, die reproduced maximally this tact can be seen in data gravang baclaral p. Pulatland and thedex that pupil atlan success in real time means that there had been competition to our sources, only servibed infju at the cost of others that could not loudish. The LTY and brilliant insight at Darwin was this, he observed that W rotations which are hand table and which mal, resource utilization bet bar I, are few adapted to habitat betler will enable only those to repro, Luca and Lee, roar progeny. Hence a period time over many general survivors will leave roar progeny and there would be a change in population characteristic and hence new LBRRNS appear tones. 7. 6 MBCILANLs emojn on what is the ongunaut thistardolin and how does speciation occur even though Mendel had tool, edit inheritable actors influencing phenotype. Darwin neither, these observations were kept silence ln the worst decade 20th century H. Du Ties Bassett in his work Millet Rose brought forth the idea at mutons I urged it. Aaron's rising socle, Lenley in a p. Pulation, he believed that, us mutation which causes evolution and not the minor variations heritable that Darwin tool, idaxlet. Mutations are random and direct loss-led Darwinian variations are small and directional. Evolution br Darwin was gradual while Divnes belly easdem return caused speciation and hence called it Salatin step la jg mtlawan. Studies in population genetics later brought out same dante. Ceology 7, 7H, WB, ILL or PLLC NCLL Lumina and population 1 can find out the frequency of occurrence of, ools of, gene or, locus. This frequency is said to remain fixed on Dean remained the soar through generations, Hordy-Weinberg principle stated. It using 3 algebraic equations, this principle says that allele frequencies in population, restable and is constant from gener, Cheyenne to gener, Cheyenne. The gene pool to genes on their alleles in population remains constant, this is called gene-nuke equilibrium. Sum total of all the oolic frequencies is I individual a BC Phys. 1, TE 7.8 diamat represented the operation of natural selection on D1 FFE, rent TMTS, a stabilizing B2, T1 on all ICDL through POO. Volagin frequencies F for example Conbiru, red PQ etc., in a diploid P and Q represent the frequency of allele A and allele A. The frequency of AA individuals in a P population is seam flip, this is simply stated in our other ways. That is, the probability that an allele A with a frequency of the pure L, of the chromosomes of a diploid individual is simply the PLTXI of the probabilities. That is, P, similarly of AA is Q of A2PQ, hence P2PQQI. This is a binomial pansion of P, QJ, when frequency measured differs from E, pected V3 Lewis. Lead difference terrestum indicates the extent of the evolutionary TRAFL. Disturbance in genetic equilibrium or Hardy-Weinberg equilibrium, that is, change of frequency of alleles in a population would then be interpreted as resultant, G annihilation. Phi, E factors are known to affect Hardy-Weinberg equilibrium. These are gene migration or gene flow, genetic drift mutation, genetic recombination and natural selection, when going of a section of population to our other place and population occurs. Gene frequencies TRAFL in the original one as well as in the new population. New genes alleles are acted to the new population and these are lost from the old population. There would be a gene now too. This gene alligration happens from tipple teres. The sum change occurs by chance. It is called genetic drift. So it is the change in allele frequency is so different in the new Sornpelsi population that they become a different species. The original drifted population Tekors folders and Lee effect is called folder effect. Microbial experiments show TLL at pre-existing advantageous mutations when selected will result in observation of new phenotypes. Over few generations TLLs would result in speciation. Natural selection is a process in which heritable variations bring better survival or led to a greater number of progeny. A critical Melissus and Watezus believe that V-meation due to mutation or V-meation due to recombination keratogenesis, or due to gene flow or genetic drift results in changed frequency of genes. MD alleles in future generation. Ojupal to enhance reproductive success Natural selection makes it look like different population. Natural selection can lead to stabilization in which raw individuals acquire character value. Directional change Reindustrial individuals acquire value Otler TLL, mean character value or disruption. More individuals acquire peripheral character value at both ends of the distribution curve. Figure 7.8. 7. 8 HBRWSL.CCO1JN1 Opevix, MMN1 About 2,000 million years ago the first cellular worms appeared on Earth. The mechanism of how non-cellular aggregates of Jimt macrum, decals cadero into cells with Rembermus emelop is not KNCNJN. Some of Lee's cells had TLLE ability to release the reaction. Rhyloglocl could have been similar to Lee-light reaction in photosynthesis where water is split with the help of Sol3 energy captured on Tunnelisect by appropriate light harvesting pigments. Slowly single-celled organisms became multicellular life forms. By the time of 500 invertebrates reformed active. Jawless fish probably at around 350 CX on few PLTSE, isted probably around 320 That is our total TLL at Lee first organisms TLL at invaded land low, air plants, they air widespread on land when 3 Jlilils invaded land fish with stout and strong fins could rove in land and go back to water. This was about 350 In 1938 a fish caught in South Africa happened to be a Ogelact which was thought to be extinct. These animals called lobefins evolved into the Evolution Quaternary Tertiary Cretaceous Jurassic Extinct Permian LS Euro Carboniferous. Figure 7.10 Representative evolutionary history of vertebrates through geological period. First amphibians that lived on both land and water. There are no specimens of these left with us, however, these were ancestors of modern-day frogs and salamanders. The amphibians evolved into reptiles. They lay thick shelled eggs which do not dry up in sun unlike those of amphibians. Again we only see their modern-day descendants, the turtles, tortoises and crocodiles, in the next 200 million years or so. Reptiles of different Ceology shapes and sizes dominated on Earth. Giant ferns Turkphytes present but they all fell to form cold deposits slowly. So these LMD reptiles went back into water tovoli into fish-like reptiles probably 200 e.g. LCHTLV Josuat SJ, the land reptiles of course the dinosaurs, the biggest of them that is, Tyrannosaurus rex was about 20 feet in height and had huge fears or dagger-like teeth. Out 65 the dinosaurs suddenly disappeared from the earth, we do not know the true reason. Source climatic CHMLS killed them. Source most of them evolved into birds, the truth mainly one in between. Small-sized reptiles of it is today, the first mommy WST like shrews, their sills are small-sized. Mammals were parous and protected their unborn young inside the mother's body. Martin mammals were more intelligent in sensing and avoiding time to release. When reptiles came down Martin mammals took over this earth. There, reigned South America Martin mammals resembling horse. hippopotamus, deer, rabbit, etc., due to continental drift when south. Arerica join North Arerica these animals Aramaic, i.e. and by North Arerican fa wla. Due to the Saroon in and out drift pouched marn mouths of Australia cern wanted because of lack of coil pay on fominea their mammal. Lest forget soar mammals live wholly in water, whales, dolphins, and sea cows are some XIS. Evolution of horse, elephant, dog, etc. are special stories of evolution. You will learn about these in higher classes. Tilna's successful story is the evolution of mvili language skills and self-consciousness, a rough sketch of the evolution of life forms. Their times on Agile cow scale are indicated in figure 7.9 on 7.10L9 GGMAFF5 Oj, on op man about 15 Ria. Primates called Trichopithecus and Rom3P, Theru Esri, Ising, they were hairy and walked like gorillas and chimpanzees. Romopithecus was more man-like while Tragopithecus was more ape-like. Pupsilsof like bones Haib and Discovered in Euliopia and Tanzarwa figure 7.11. These revealed hominid features leading to the belief that about 3-4 NW, like mates walked in eastern Africa. They were probably not taller than 4 feet but walked up RTGHT, till, Maya, Australopithecus probably lived in East African grasslands. Evidence shows they hunted with stone weapons but essentially ate fruit. Some of Lee Bones' AFLDFLG Lee Bones discovered were deodorant. This creature was collect the first like being the hominid was called Horohobjls. The brain capacities re between 650-800 cc. They probably did noted read fossils to CO in Java in 1891 re-yield Lena.ct stage that is. Biology and Phys. W. E 7.11a Compensinovilia skulls of adult MIGD, RN M being baby clump, Z adult CHLRNL. The skull of baby CHLRNLLS Morlcan, Delhernan skull LH, and adult churn skull Aridus. About 1.5 Maya. Aridus had LMGE brain. Around 900 cc, Homo erectus probably 8 meet, the Neanderthal man width. Brain CZ of 1400 cc tiled in Nem East. In Central Asia between 1,000,000 40.000 years back. They used hides to protect their BCXL, on bury their dead Sages and arose in Africa. Across continents and re-eloped 3D into distinct races, during ISAG, between 75,000,000 years ago Dem Homo sages and arose. Afari historic cave. Art developed. About 18.000 years ago, agriculture come around 0.000 years back and human settlements started. The rest of what happened is part of human history of growth and decline of C. Lucilian S. F.ij. Z Theology Summary. The origin of life on Earth can be understood only against the background of work 1 of WLIRSE spec 1 ALLYML. Most scientists spell me chemical evolution, that is, formation of B1 molecules preceded U, EAPM, me of U, EBRST OLU, MF forms of life. NLE subsequent to E1TS2, had happened to U, EBRSTF form of life is a conjectured story based on D1 D3S or LLIE. Evolution by natural selection. D. Diversity of life F4S on E3LL has been over millions of S. It is gen. RLLY Belai.OEDLAT. Machines in a population result in table fitness. Oilier phenomena like habitat fragmentation, netic drift, mayako and U8LEs. Machines li CDTNG to API of news B1SL. Arrow elution. Homolog is accounted F4 by LE idea of BR Deso, NT. Study of Kumpma 11 The anatomy of fossils Kumpmau. B1OCL. Ernst provides Zool. Sever E.Elution. Among all stones of evolution of individual species, all story of evolution of modern M is most int, rising up MS to P mal LE. Evolution of human brain E. Zeph, Cises 1, explain in 11B10 ok resistance obs, ND and B3 cleg, RTA in light of D cell equinolary. 2, F1N, L out on Marus Pape, RS Popl 3R71E, no articles, my new ethos, al discoe, Retez are contrawis about E.Elution. 3. LRNPLG Ving a Clio, art definition of the term spec S4, TDY to trace all E13 retus conform 1 TS of way MNE. Ulet 1 on GMT, brain S10 on Funo Yuan skit TLSTN, OUE. D1 artery proferos etc. 5, FLRD out THR8HLN, net IPOPU 1, Z, Uckleswi, Thur, LS oilier OLM, M has self-consequencelessness. 6. List 10 modern-day S1 LLGLE1 NTE, net resources link it to a corresponding A, NC1E, NTFOS, IL. Na, RNE Boily 7, Practice DR, MGW, R10S.NRNJI plants 8, Descript 1e, XII, Plevadapu, radiation. 9 that is KLL hum CME lot ion adapti, radia 11 oral 10, Uston VC, Utus resources such as We School UBRMF or LE Internet D1 Skustans Watili or Tiche, R. Trace LE. Illusion Mesov, My One Sehors. Unit VLLL Biology and Human Welfare Chapters Human Health and Disease Chapter 9 Strategies for Enhancement and Food Product Line Chapter 10 Microbes and Human Welfare Biology is the youngest of LHE formalized disciplines of natural science. Progress in physics and chemistry proceeded much faster than in biology. Applic alliance of physics and chemistry in our daily life have a higher visibility than those of biology. However, 20th century and certainly the 21st century has demonstrated the utility of biological knowledge in furthering human welfare, be it in health sector or agriculture. The discovery of antibiotics and synthetic plant-derived drugs, anesthetics have changed medical practice on one hand and human health on the other hand. Life expectancy of human beings have dramatically changed over LHE years. Agricultural practices, food processing and diagnostics have IOC ultra-I changes in human amunilis. These are briefly described in the following 3 chapters of his Unal. M.S. Swaminathan 1925 born in August 1925 in Kumbakonam, Tamil Nadu, Munkambu Sambasavan Swaminathan did his graduation and post-graduation in botany from Madras University. He worked in different capacities in large number of institutions in India and abroad and developed his expertise in genetics and plant breeding. The School of Cytogenetics and Radiation Research established at the Indian Agricultural Research Institute IRI enabled Swaminathan and his team to develop short-duration high-yielding varieties of rice including scented basmati. He is also known for the development of the concept of crop cafeteria, crop scheduling and gene ticketing improving the yield in Quailili. Swaminathan initiated collaboration with Norman Borlaug, which culminated in the Green Revolution through introduction of Mexican varieties of wheat in India. This was highly recognized and appreciated. He is also the initiator of Lab Lowland, food security and several other environmental programs. He has been honored with Padma Bhushan and several other prestigious awards, medals and fellowships by institutions of excellence. Chapter 8 Human Health and Disease 8, Common Diseases TN, H 8.2 liters I, J 8.3 AIDS 8.4 Cancer 8.5 D9 S on AICDD. Abuse health F for a long theory was considered as a state of XII.MD mind where Lear W is about 3 NCERT NHUFDFS. This is what early Greeks like Hib, U, R8 is low, L is land in Ayurveda system of medicine asserted. It was thought that persons with plague bile belonged to hot person 3HLF. And would have fevers. This idea was arrived at by pure reflective thought. The desanery of blocks of circulation by Imlion honey using experimental rests long the demonstration of normal XII temperature in persons with plague bile using thermometer disproved the good horror hypothesis of health in later years. Biology stated that mind influences through neural system and endocrine system or immune WLE system.n. Tlat our immune system noon lanes our health, hence mind. And mental state can affect our health. Of course health was affected by IgNEDC disorders deficiencies v. LTLL which a child is torn on defeines defects which the child inherits from parents from birth, IL infections on 3 lifestyle including food and water we. Rest and D. Rare is we guile to erdodes H3 pitsulit v. E have or lack etc. 8KIIOGY The term health is very frequently used by everybody. How do we define? T health does not simply mean sense of to see our physical fitness. It could be defined as a state of complete physical, mental and social well-being. When people are healthy, they re more efficient, work. This anchor, says Brexlogty, and brings economic prosperity. Health. Also increases longevity of people. And reduces infant and maternal mortality. Balanced diet personal HJL and Andrea Braces are very important to int and good health. Yoga has been practiced since Laramorial to achieve physical. And mental health. Awareness. About diseases and TLL effect on different FWL Xions vaccination immunization. Against infectious disease, as is proper disposal of wastes control of vectors. And maintenance of hygienic FBCD. And water resources. Are necessary FBR, feeding good health. When the functioning of one or more organs or systems of the XII is adversely affected char, 10 said by V new signs on symptoms. Say TLL, at V, E are not healthy that is, low, E have a disease. Diseases can be broadly grouped into infectless. And non-infectless. Diseases which are easily transmitted from one person to another. Are called infectless diseases. Infectious diseases. Are very idle. And every one of us suffers from TLLs at sorta or other. Serif TLL infectious diseases like AIDS. Or FIDL, non-infectious diseases cancerous TLL major cause of CTE at drug and alcohol, abuse also, fleet our health, D1 Ersly 8.1 Co1 TT1 TT on LLSSL, SK, SLN HY, SAD, wide range of organisms belonging to bacteria. Viruses FWLG protozoans Helmini and TLLS etc. could cause diseases in all. Such disease causing organisms are called pathogens. All parasites are TLL P. Pathogens, STC, use harm to the host by leaving in or on them the P. Pathogens can enter orgslebi nutrients. Multiply and interfere with normal TAL, CTI villies resulting in morphological and functional damage. Pathogens H1E2, D, PT to life within the environment of the host, for example the P. Phagns that enter the gut rust no way of surviving in the stomach, T low pH and resisting the venous digestive enzymes. A few representative members from different groups of pathogenic organisms are discussed here. Along with TLLE disease is caused by the Prevent1E and UN1 raw reagers, against thesides, says in general, also briefly described. Salam, NEUO, TJPU is, P, pathogenic bacterium which C, uses typhoid Phe1 or in human beings. These pathogens generally enter the small intestine through food and water contaminated with them, NCT migrate to other organs through blood. Suswind high fever 39, to 4C weakness stomach pain unstip, cheyenne headache and loss of appetite. Some of the common symptoms of this disease intestinal perfor, cheyenne, 10, th may occur in rare cases. Typhoid fever could be confirmed by will now test a classic case in Weediine that of M3II long Nickner TGJ, IO, D. Marg is worth mentioning here. She was a cook by profession. L was a typhoid CM1er who continued to spread typhoid roar several years through the FCX, D. She prepared bacteria like S. Tazakikru S. Pneumorvae and Liaum, PLVBTS and C are responsible for Odysseus and Unanwana in Hunwals which infects the alveoli air-filled sacs of the LWLGS. As a result of the infecti on the alveoli GT61 led with fluid leading to severe problems in respiration. The symptoms of Nunditia insular Phe, or chills cough NCT headache. In severe cases the lips and fingernails may turn greater bluish insular. A healthy person exuires the infection by inhaling the droplets aerosols released by M infected person or even by SH mingluses. And utensils whittle. An infected person, dysentery plague diphtheria, etc. are sore of the other bacteria. 31 diseases in NWL, M3 viruses also cause diseases in huttile beings. Rhinoviruses represent one such group of viruses which cause one of TL1E and DST infectious it's the common cold. They infect the nose and respiratory passage but not the lungs. The Umaran cold is characterized by nasolaryngeal and discharge. Sore throat, hoarseness, cough, headache, tiredness, etc., which usually last for 3-7 days. Droplets resulting from cough or sneezes of an infected person or either INH3 led directly or transmitted TLU roof contaminated objects such as pens, books, cups, portknobs, computer, keto, RD or news, etc. and cause infection in a healthy person. Sore of the human diseases are caused by protozoans too. You might have heard tout malaria. A disease has been fighting since MMY years. PKTSM, D. Amatonipactazone is responsible for this disease. Different species of FTOSM, DMPOXP malum and P. falciparum are responsible for differential pasophenol. Of these Mo-G8 land RNA1 are caused by FTOSM, Diamphocyporum is RMST serious one and can even be fatal. Let us a glance at the life cycle of PASM. DM figure 8.1. FTOsmodium enters the human Xlia sporozoites infectious form TLU roof the bite of infected female anaphil. ESM, squiddo. The parasites initially relatively within the Lear cells and then attack the red BLCX, D cells RBCs resulting in their rupture. The rupture of RBCs is associated with real as of data to substhrene sehal which is responsible TBR chill and high fever recurring ERE3 to FR days. When a female allergy ESN squiddo bites an infected person TLLE's parasites enter the mosquit cell, and undergo further development. The parasites relatively within them to form sporozoites that are stored in their salivary glands. When Lees mosquits bite HUFWL, Lee rhozoites are intro, lucet into his herbxley. Thereby ion erasing to note TLL email re3.1 parasite requires 2 hosts human. N. From squios to complete its life cycle figure 8.1, the female anoples from squitois the vector transmitting agent 2. Full stop. F is, UE 8.1 in U, E life cycle of CR smodclementomojxtltist, K, is a protozoan parasite in Lee JMGE intestine herm which causes moblastols DJ celtici. Symptoms of TLL is dysphagia, insular constipation, abdominal pain, and cramped stools with E. Sesmucuescentplo, D. Clots, houseflies act. As rich 3 Nicalcine and serve to transmit parasite from feces to infected person to odd. Human health and disease and food products, thereby contaminating them. Drinking water and food contaminated by the fecal matter are the main source of infection. Ascaris, the common roundworm and Wuchereria, the filarial worm, are some of the helminths which are known to be pathogenic to man. Ascaris, an intestinal parasite, causes ascariasis. Symptoms of these disease include internal bleeding, muscular pain, fever, anemia and blockage of the intestinal passage. The eggs of the parasite are excreted along with the feces of infected persons which contaminate soil, water, plants, etc. A healthy person acquires this infection through contaminated water, vegetables, fruits, etc. Wuchereria W. bancroftshi and W. malayi. The filarial worms cause a slowly developing chronic inflammation of the organs in which they live for many years. Usually the lymphatic vessels of the lower limbs, and the disease is called elephantiasis or filariasis. Figure 8.2. The genital organs are also often affected, resulting in gross deformities. The pathogens are transmitted to a healthy person through the bite by the female mosquito vectors. Many fungi belonging to the genera Microsporium, Trichophyton and Epidermophyton are. Figure 8.2 diagram showing inflammation in one of the lower limbs due to elephantiasis, responsible for ringworms, which is one of the most common infectious diseases in man. Appearance of dry, scaly lesions on various parts of the body such as skin, nails and scalp. Figure 8.3 are the main symptoms of the disease. These lesions are accompanied by intense itching. Heat and moisture help these fungi to grow, which makes them thrive in skin folds such as those in the groin or between the toes. Ringworms are generally acquired from soil or by using towels, clothes or even the comb of infected individuals. Figure 8.3 diagram showing ringworm affected area of the skin. Maintenance of personal and public hygiene is very important for prevention and control of many infectious diseases. Measures for personal hygiene include keeping the body clean, consumption of clean drinking water, food, vegetables, fruits, etc. Public hygiene includes proper disposal of waste and excreta, periodic cleaning and disinfection of water reservoirs, pools, cesspools and tanks, and observing standard practices of hygiene in public catering. These measures are particularly essential where the infectious agents are transmitted through food and water, such as typhoid, amoebiasis and ascariasis. In cases of airborne diseases such as pneumonia and common cold, in addition to the above measures. Close. 8KIIOGY contact VATLLE infected persons or TLL or telungings should be aware for diseases such as malaria and filariasis LLI arms mitted TLL rough insect vectors. The most important measure is to control or eliminate the vectors on their breeding places. This can be achieved by avoiding stagnation of water in and around residential areas. RLR cleaning of household coolers. UC1 mosquito nets introducing fishes like OMFX.SA. In ponds that feed on Roscudo 13N, OE, spraying of insecticides in ditches, drainage areas and SWOMPs, etc. In addition, doors and wink should be protected with wire mesh to prevent the entry of mosquitoes. Such precautions be Rome are important, especially in the light of recent low-lying spread incidences of Leicester-borne Aedes nusquitzes diseases like dengue and chikungunya in many ports of India. The active microbes made in BLOCL science HE.armed us to detect RRW1Y infectious diseases. The use of vaccines and iron mineralization programs have enabled us to completely eradicate a deadly disease like smallpox. A large number of other infectious diseases like polio, diphtheria, pneumonia and tetanus have been controlled to a large extent by the use of vaccines. Biotechnology, about which you will read more in Chapter 12, is at the verge of making available on safer vaccines. Discovery of antibiotics MD drugs has also enabled us to affect the illiterate infectious diseases. Bujni, why every day are exposed to a larger number of infectious agents, however only a few of TLL's exposures result in disease. Why this is due to the fact that the body is able to defend itself from LDS of these UEI agents. This overall ability of the host to fight the disease using organisms conferred by the immune system is called immunity. Immunity is of two types, innate immunity and acquired immunity. 2. 1. Innate immunity. Innate immunity is non-specific type of defense. TLL, is present at the time of birth. This is accomplished by putting different types of barriers to the entry of the fibrin GN agents into our body. Innate immunity consists of FR types of barriers. These are physical barriers. Skin on our body is the membrane which prevents entry of microorganisms. Mucus coating of the epithelium lining the respiratory, gastrointestinal and urogenital tracts also help in trapping microbes entering our body. Euphysiological barriers, acid in the stomach, saliva in the mouth, tears, foamies all prevent microbial growth. 3. Cellular barrier S. Certain types of leukocytes WBC of our body like polymorphonuclear leukocytes PMNL, neutrophils and human health and disease monocytes and natural killer type of lymphocytes in the blood as well as macrophages in tissues can phagocytose and destroy microbes. 5. Cytokine barriers. Virus-infected cells secrete proteins called interferons which protect non-infected cells from further viral infection. 8. 2. Acquired immunity. Acquired immunity, on the other hand, is pathogen-specific. It is characterized by memory. This means that our body when it encounters a pathogen for the first time produces a response called PRIL, 8-lie response which is of low intensity. Subsequent encounter with the same pathogen elicits a highly intensified secondary or anamnestic response. This is ascribed to the fact that our body appears to have memory of the first encounter. The primary and secondary immune responses are carried out with the help of two special types of lymphocytes present in our blood. That is, B lymphocytes and T lymphocytes. The B lymphocytes produce an army of proteins in response to pathogens into our blood to fight with them. These proteins are called antibodies. The T cells themselves do not secrete antibodies but help B cells produce them. Each antibody molecule has 4 peptide chains, 2 small called light chains and 2 longer called heavy chains. Hence, an antibody is represented as H2L2. Different types of antibodies are figure 8.4 structure of an antibody molecule produced in our body. LGA, IgM, LGE, IgG are some of them. A cartoon of an antibody is given in figure 8.4. Because these antibodies are found in the blood, the response is also called as humoral immune response. This is one of the 2 types of our acquired immune response, antibody-mediated. The second type is called cell-mediated immune response or cell-mediated immunity CMI. The T lymphocytes mediate CML very often, when some human organs like heart, eye, liver, kidney fail to function satisfactorily. Transplantation is the only remedy to enable the patient to live a normal life. Then a search begins to find a suitable donor. Why is it that the organs can't be taken from just anybody? What is it that 8 KIIOGY the daughters check or 3 FTS from just any source. M.animal. Mother primate or any beings cannot be mode since the graft cell D be rejected sooner or later. Tissue matching gluco, L group matching, re-assemble before undertaking any graft transplant and after this the patient has to, EMA presence. U his or life. The XII is able to differentiate self.md non-self. MCT Lee cell radiated immune resp 01 says resp, NCERT F with graft rejection. 8. 2. 3. Active and passive immunity when a host is exposed to antigens which may be in the F form of living or dead microbes or all proteins. Antibodies are PM, TUCD and Lee host Bhakti. This type of immunity is called ACT-UVLM will T. ACT tolerability is slow. N. T3 has time to give its full effect ERSP 01 cent. Injecting Lee microbes reliter 8 leak tunning immulize a cheyenne or infectious organism gaining. Access into body during an natural infection induced. Active immunity. When reacting mocked antibodies are directly G, then to protect the body against foreign agents it is called passive immunity. To go no co WHGM. Thursmac S. Constv, L. Regessent, U for the newborn in feign the jello wish fluid colostrum secreted B, Y rather during the initial days of lactation has abundant antibodies I get to protect the infant. The fetus. Also reset ES sort. Tib, dies from TLL or Mottler through the placenta during 11G pregnancy. These are sorry amples of passive immunity 8, 2, 4 vaccination and immunization. The principle of immunity honor of vaccination is based on TLL property of marriage of the immune system. In vaccination a preparation of antigenic proteins of pathogen or inactivated fucking pathogen vaccine or introduced into the body. The antibodies produced in the XII against these antigens would neutralize TLL pathogenic agents actual infection. Tile vaccines also generate IUFBMD T cells that recognize the pathogen quickly on subsequent Posure.md. Overwhelmingly in V actors via MSC. Reproduction of antibodies, a person is infected. The source CTEADL microbes to which quick immune response is required. As in tetanus we need to directly inject TLL pre-1 med. Antibodies are antito in a preparation unlaning antibodies to the twin, Ellen in cases of snocobites. The injection which is gene to the patients contain per-honored. Antibodies. Agonist TTLLESMKNM. This type of immunization is called passive immunization. Recombinant DNA technology has allele, D the production of antigenic pol, if applies of pathogen in bacteria or yeast. Vaccines produced using this approach allow 13 RGE scale production and hence Jeter availability for immunization. For example hepatitis B vaccine produced from yeast. S2, 5 AURGs did this Rupeng 2, UN, U113 go to a new place 3 JLD Sokl, Lenley, you started sneezing wheezing for no explained reason. 3JLD when you can't your symptoms disappeared as of us are sensitive to some particles in the environment. The above mentioned reaction could take because of allergy to pollen, mites, etc., which are different in different places. The exaggerated responsible immune system to certain antigens present in the environment is called energy. The substances to which such iron-1E responses PM, TUC are called allerg, MS, tile antibodies produced to these are of I type. Ojran examples of allergens are mites and dust, P, lens, animal dander, etc. Symptoms of allergic reactions: inhaler, sneezing, watery ES, running nose and difficulty in breathing. Allergy is due to the release of chemicals like histamine and serotonin from the mast cells. For determining the cause of allergy, the patient is exposed to or injected with LARISFLL3LL-P, S-cyble allergens. MD reaction studied, the use of drugs like anti-histamine, adrenaline and steroids quickly reduce the symptoms of allergy. How, Dem, day lifestyle rules resulted in lowering of ILLD and more sensitivity to 3 allergens N. Children in retro cities of India suffer from NERG, S and asthma due to city tight environment. This could be because of the protective environment provided early in life. 6 autoimmunity memory, Basect acquired ILLD evolved in higher vertebrates based on the ability to differentiate to organisms, for example. Palo, ins from self cells. While we still do not understand the basis of this, Coral 3 rise of this ability to be understood. 1. Hypnovertebrates can distinguish ROR, GN molecules as well as ROR, GN organisms. Most of the parental amyloid deals with this aspect. Sornal due to genetic and other W1KMWN reactions, the body attacks self cells. This results in damage to Lb, and is called autoimmune rheumatoid arthritis, which affects MMY people in our society as to immune disease. 7 immune cyst, in the body. The human immune system consists of LMPH tissues. Cells 3JLD soluble molecules like antibodies, as HO113E read, iron GNE system is unique in Lee sense TLL, and it recognizes foreign antigens. Response to these 3JLD remembers them. The immune system also plays an important role in allergic reactions. Autoimmune diseases. And organ transplantation, lymphoid organs. These are the organs where under maturation 3JLD proliferation of lymphocytes occur. The primary lymphoid organs are bonomato 3JLD thymus, where immature lymphocytes differentiate. Figure 8.51 Thymus diagrammatic representation of lymph nodes biology into antigen-sensitive lymphocytes. After maturation, the lymphocytes migrate to secondary lymphoid organs like spleen, lymph nodes, tonsils, Peyer's patches of small intestine and appendix. The secondary lymphoid organs provide the sites for interaction of lymphocytes with the antigen, which then proliferate to become effector cells. The location of various lymphoid organs in the human body is shown in Figure 8.5. The bone marrow is the main lymphoid organ where all blood cells including lymphocytes are produced. The thymus is a lobed organ located near the heart and beneath the breastbone. The thymus is quite large at the time of birth but keeps reducing in size with age, and by the time puberty is attained it reduces to a very small size. Both bone marrow and thymus provide microenvironments for the development and maturation of lymphocytes. The spleen is a large bean-shaped organ. It mainly contains lymphocytes and phagocytes. It acts as a filter of the blood by trapping blood-borne microorganisms. Spleen also has a large reservoir of erythrocytes. The lymph nodes are small solid structures located at different points along the lymphatic system. Lymph nodes serve to trap the microorganisms or other antigens, which happen to get into the lymph and tissue fluid. Antigens trapped in the lymph nodes are responsible for the activation of lymphocytes present there and cause the immune response. There is lymphoid tissue also located within the lining of the major tracts: respiratory, digestive and urogenital tracts, called mucosal-associated lymphoid tissue MALT. It constitutes about 50% of the lymphoid tissue in human body. 8.38 The word AIDS stands for acquired immunodeficiency syndrome. This means deficiency of immune system, acquired during the lifetime of an individual, indicating that it is not a congenital disease. Syndrome means a group of symptoms. AIDS was first reported in 1981, and in the last 25 years or so, it has spread all over the world, killing more than 25 million persons. AIDS is caused by the human immunodeficiency virus HIV, a member of a group of viruses called retrovirus, which have an envelope enclosing the RNA genome. Figure 8.6. Transmission of HN infection generally occurs by a sexual contact with infected person, B by transfusion of contaminated blood and blood products, C by sharing infected needles, as in the case of intravenous drug abusers, and D from infected mother to her child through placenta. So, people who are at high risk of getting this infection includes individuals who have multiple LLLM, Health A, OOMFLL, all, a CDLLLT, WRNAMP:01BJT, HLM, 6DCD null, Li4-ocbidol. Willy Bolif FT1JTCD Fe1-EC, FIS, Ur 8.6 replication of Retric MRU, SXW, L partners. Drug octics who took drugs intravenously in D.modules who require repeated BLCX, D transfusions on children born to an HIV-infected her. Do go, now and do PXE need repeated blood, to Rufus on FTND out at DM, all, EAI street of such conjugations. It is important to note that HIV-1 AIDS is not spread by rear touch or physical contact, it spreads only through XII fluids. It is hence imperative for the physical and psychological well-being that the HIV AIDS-infected persons are not isolated from family and society. There is ALWI's a time bed and the infection and appearance of AIDS symptoms. This period may vary from a few months to many years, usually 5-10 years. 8KIIOGY after Jitingantalb die after Perskin the was enters into myas where RN agenor of Tre virus replicates to F-worm WLDNA with the help of the ENSG, Ironerstrom scriptase this WOLDNA. Ilzino, UP, latidentalized cells DNA and directs the infected cells to produce virus particles figure 8.6. The Microbes untenutipro, loose was and in this way acts like, H, factory, simultaneously, hive enters into helper TIJM phocytes T. J replicates and loose progeny uses, the progeny is released in the blow, D addict the helper IJM phocytes. This is repeated leading to a progressive decrease in the number of helper lymphocytes in the XII of the infected person. During this period the person suffers from bouts of feller diartho and GHT loss, due to decrease in the number of helper lymphocytes. Tre person SWTS suffering from infections that would have been otherwise 011 errors such as those due to Bacteries PEUM cogs or denim. Viruses, fungi and even parasites like toxoplasm, the patient BRSOMRTNO deficient she is unable to protect himself/herself against these infections. A widely used E-stick test FBR AIDS is enzyme-limited non-IDL060I beta-species Treatment of AIDS with antiretroviral drugs is only partially effective. They can only prolong the life of the patient but C3JLLLD. Pre-entry, which is inevitable, prevent, N of AIDS, as AIDS has no cure. Prevention is the best option. Murmur, HIV infection M, often spreads due to conscious THVR patterns and is zooronal the happens inadvertently like pneumonia or typhoid of course. Infection in blow, detransfusion patients newborns from etc., may take place due to poor monitoring. The only excuse may be ignorance and it has been NGHUI so, CTCB and die of ignorance. In our country the National AIDS Control Organization NACO and other non-governmental organization NGOs are being to educate people about AIDS. WHO has started number of programs to prevent the spreading of HIV infection, making BLCX, D from BLCX, D banks safe from HIV, ensuring the use of disposable needles and sarin. Is in public and private hospitals and clinics free distribution of condoms, controlling drug abuse. Educating safe set, and promoting regular checkups. The junior hive in susceptible populations are some such steps free can up. Infection with HIV or having AIDS a oral head should not be hidden since then the infection may spread to many rower people. HIV/AIDS infected people need help and sympathy instead of being shunned by society, unless society uses it as problem to be dealt with. In a collective NWLLR the chances of ICID spread of GLLE disease increase manifold. It is, malady health can only be tackled by the society and medical fraternity while acting together to prevent the spread of the disease. Cancer is one of the most dreaded diseases of human beings and is a major cause of death all over the globe. More than 1 million Indians suffer from cancer and a large number of them die from it annually. The Organisms TLL adjuncture development of cancer or oncogenic transformation of cells. Its treatment and control have seen some of the NDST intense areas of research in biology. MCTM, design. In arbxley cell growth and differentiation is highly controlled and regulated in cancer cells. TLL is breakdown of Lee's regulatory mechanisms. Normal cells show a property called contact inhibition by virtue of which contact with all your cells inhibits Lee or WL control growth. Cancer cells appears to have lost this property. As a result this cancerous cells just continue to divide giving rise to masses of cells called tumors. One tumors are of TO12, benign and malignant. Benign tumors only remain confined to their origin 3 location and do not spread to other parts of the body and cause little DE. The malignant tumors on the other hand are a mass of proliferating cells called neoplastic or tumor cells. These cells very rapidly invading and eating the surrounding normal tissues, as these cells actively divide and they also starve the normal cells by impelling through vital nutrients. Cells sloughed from such tumors reach distant sites through BLC, XXL on where they get lodged in the body. They start a new tumor there. This property called metastasis is the most feared property of malignant tumors. Causes of cancer: T-arms formation of normal cells into cancerous neoplastic cells may be induced by physical, chemical or biological agents. These agents are carcinogens, long-lasting radiations like X-rays and gamma rays on non-ionizing radiations like UV cause DNA DE leading to neoplastic transformation. The chemical carcinogens present in tobacco SNDK have been identified as a major cause of cancer. C3 NCERT using whose is called oncogenic for uses genes called V1 RAL oncogenes. Furthermore, several genes called cellular oncogenes C1 or proto-oncogenes have been identified in normal cells which when activated under certain conditions could lead to oncogenic transformation of the cells. Cancer detection and diagnosis. ERDIC detection of cancers is essential as it reloads lead disease treated successfully in NWLF cases. Cancer detection is based on biopsy and histopathological studies of the tissue. In blood, DN. Bone marrow tests FBR increase cell count TS in the case of leukemias. In biopsy, a piece of the suspected tissue cut into thin sections is stained and R9 under microscope. Histopathocal studies by a pathologist. Techniques like radiography, use of X-rays, CT computed tomography and MRI magnetic resonance imaging are very useful to detect cancers of the internal organs. A. Computed tomography uses X-rays to generate a three-dimensional iron eye of the internals of an object. MRI uses strong Fro3L81 fields and idealizing radiations to elucidate L1CL3 tech pathological and physiological changes in the LLW, G tissue. Antibodies against cancer-specific antigens are also used for detection of certain cancers. Equal of molecular biology can be biology applied to detect genes in individuals with inherited susceptibility to certain cancers. Identification of such genes, which predispose an individual to certain cancers, may be very helpful in prevention of cancers. Such individuals may be advised to avoid exposure to particular carcinogens to which they are susceptible, e.g., tobacco smoke in case of lung cancer. Treatment of cancer. The common approaches for treatment of cancer are surgery, radiation therapy and immunotherapy. In radiotherapy, tumor cells are irradiated lethally, taking proper care of the normal tissue surrounding the tumor mass. Several chemotherapeutic drugs are used to kill cancerous cells. Some of these are specific for particular tumors. Majority of drugs have side effects like hair loss, anemia, etc. Most cancers are treated by combination of surgery, radiotherapy and chemotherapy. Tumor cells have been shown to avoid detection and destruction by immune system. Therefore, the patients are given substances called biological response modifiers such as interferon which activate their immune system and help in destroying the tumor. 8.5 Drugs and alcohol abuse surveys and statistics show that use of drugs and alcohol has been on the rise, especially among the youth. This is really a cause of concern as it could result in many harmful effects. Proper education and guidance would enable youth to safeguard themselves against these dangerous behavior patterns and follow healthy lifestyles. The drugs, which are commonly abused, are opioids, cannabinoids and coca alkaloids. Majority of these are obtained from flowering plants. Some are obtained from fungi. Opioids are the drugs, which bind to specific opioid receptors present in our central nervous system and gastrointestinal tract. Heroin figure 8.7, commonly called smack, is chemically diacetylmorphine, which is a white, odorless, bitter crystalline compound. This is obtained by acetylation of morphine figure 8.7, which is extracted from the latex of 0 figure 8.7 chemical structure of morphine figure 8.8 opium poppy. Human health and disease poppy plant Papaver somniferum figure 8.8, generally taken by snorting and injection. Heroin is a depressant and slows down body functions. Cannabinoids are a group of chemicals figure 8.9, which interact with cannabinoid receptors present principally in the brain. Natural cannabinoids are obtained from the inflorescences of the plant Cannabis sativa figure 8.10. The flower tops, leaves and the resin of cannabis plant are used in various combinations to produce marijuana, hashish, charas and ganja. Generally taken by inhalation and oral ingestion, these are known for their effects on cardiovascular system of the body. Figure 8.9 skeletal structure of cannabinoid molecule coca alkaloid or cocaine is obtained from coca plant Erythroxylum coca, native to South America. It interferes with the transport of the neurotransmitter dopamine. Cocaine, commonly called coke or crack, is usually snorted. It has a potent stimulating action on central nervous system, producing a sense of euphoria and increased energy. Excessive dosage of cocaine causes hallucinations. Other well-known plants with hallucinogenic properties are Atropa belladonna and Datura figure 8.11. These days cannabinoids are also being abused by some sportspersons. Drugs like barbiturates, amphetamines, benzodiazepines, lysergic acid diethylarnides LSD, figure 8.10 leaves of cannabis same and other similar drugs that are normally used as figure 8.11 flowering branch of Datura medicines to help patients cope with mental illnesses like depression and insomnia, are often abused. Morphine is a very effective sedative and painkiller, and is very useful in patients who have undergone surgery. Several plants, fruits and seeds having hallucinogenic properties have been used for hundreds of years in folk medicine, religious ceremonies and rituals all over the globe. When these are taken for a purpose other than medicinal use or in amounts frequency that impairs one's physiological or psychological functions, it constitutes drug abuse. 8KIIOGY smoking also paves the way to HMD drugs. Tobacco has been used by human beings for more than 400 years. Lettuce smoked. Chia, Dora used as a snuff. Tobacco contains a IMGER number of chemical substances including nicotine, an alkaloid. Nicotine stimulates adrenal gland to release adrenaline and noradrenaline into blood, circulation. Bottle of which rise blood, pressure. And increase EMT RA10, SM is associated really increase incidence of cancers of lung. Uniriblet, IRANDTLUOOT bronchitis emphysema coronary heart disease gastric ulcer etc. Tobacco chewing is head, really increase risk of cancer of TLL or alcar1 type. Smoking increases carbon monoxide content in blood and reduces Lee concentration of hemalda, IgN. This causes IgN deficiency in the body. When one buys packets of cigarettes 1C3.1 a lot miss TLL street a tutorial warning that is present on the packing which warns against smoking and says how it is injurious to health. Yet smoking is very prevalent in society both young and old knowing the dangers of smoking and chewing tobacco. And its addictive nature the youth and old need to avoid these habits. Any addict requires counseling and medical help to get off the habit. 8.5L Adolescence and drug alcohol abuse. Adolescence bottle, a process, during which a child becomes mature in terms of his her activities and behaves for effective participation in society. The period between 12-18 years of age and 3 YBTLL out of adolescence period, in other words. Adolescence is a bridge linking childhood and adulthood. Adolescence is accompanied by several biological and B3B0 or ALCH3 changes. Adolescence is a very vulnerable phase of mental and psychological development of an individual. Curiosity need FBR adventure and excitement and experimentation constitute and causes which motivate youngsters towards drug and alcohol use. A child's natural curiosity motivates him her to efferent. This is complicated further by effects that might be personally led as benefits of 3 alcohol or drug use. Thus TLL first use of drugs or alcohol and 3 YB out of curiosity or experiment alien but later TLL child. Street ARTS using these to escape ing problems of late stress from pressures to excel in academics or exam ENTIONs. Has played a significant role in persuading the youngsters to try alcohol and drugs. The perception among youth that it is cool or progressive to smoke, use drugs or alcohol is also in a way a new use for yoli to street art these H3 bits, television movies newspapers. Internet also help to promote this perception. Other actors that have been seen to be associated with drug and alcohol abuse ARRDNGA plus NCERT slabla or unsupported family structures and peer pressure. 8.5. To addiction and dependence. Because of the perceived benefits drugs are frequently used repeatedly. The important thing which one has to realize is the inherent addictive nature of alcohol and drugs. Addiction is a psychological addict to certain effects such as euphoria and a temporary feeling of well-being associated with drugs and alcohol. These DTLE people to L3L, learn and when the are not nettered, L, are used tech or self-destructive. With repeated use of drugs the tolerance level of the receptors present in our top T increases. Q. Consequently the receptors respond only to higher doses of drugs or alcohol leading to greater and addiction. However it should be clearly borne in mind that use of these drugs even once can be a forerunner to addiction. Thus the addictive potential of drugs and alcohol pull the user into a vicious circle leading to their regular abuse from which she may not be able to get out in the absence of any guidance or counseling. The person GTS3 ticked and because dependent on their use. Dependence is the tendency of the body to manifest a characteristic and unpleasant will syndrome to regular dose of drugs when alcohol is abruptly discontinued. This is characterized by anxiety, shockiness, nausea, MD, sweating, which may be relieved when uses Resurrect XII. In sore cases, withdrawal symptoms can be severe and even life-threatening, and the person may need medical supervision. Dependence leads the patient to ignore social norms in order to get sufficient funds to satiate her needs. These result in many social adjustment problems. 8.5 Effect of drug alcohol abuse. The immediate adverse effects of drugs and alcohol abuse are manifested in TLL form of reckless behavior. V3JLD, 3-lism. In violent excessive doses of drugs may lead to coma and death due to respiratory failure, heart failure, or cerebral hemorrhage. A combination of drugs or their intake, along with alcohol, generally results in overdosing and even deaths. The most common warning GNS of drug and alcohol abuse among youth include drop in academic performance, unexplained absence from school, college, lack of interest in personal hygiene, withdrawal, isolation, depression, fatigue. Aggressive. In rebellious behavior. Deteriorating relationships with family and feelings, loss of interest in hobbies. CH3 and GE in sleeping. And eating habits, fluctuations in GHT, appetite, etc. There may even be some reaching implications of drug alcohol abuse. If the abuser is unable to get money to buy drugs alcohol, she may turn to stealing. The adverse effects are just not restricted to the son who is using drugs or alcohol. J.I. Irons. A drug alcohol addict because the cause of rental and financial distress to his current family and friends. 8KIOGY. Those who L3K drugs intravenously direct injection into leaving using a needle on syringe are much more likely to acquire serious infections like AIDS and hepatitis B, the viruses which are responsible for these diseases, or transferred from one person to another. Be shoring of infected needles and syringes. Both AIDS and hepatitis B infections are chronic infections and ultimately fatal. AIDS can be transmitted to one's life partner through GHC, UAL intact, while hepatitis B is transmitted through infected BLCX-D. The use of alcohol during adolescence may also have long-term effects. It could lead to overdrinking in adulthood. The chronic use of drugs and alcohol damages nervous system and liver cirrhosis. The use of drugs and alcohol during pregnancy is also known to adversely affect the fetus. Another misuse of drugs is what certain sports persons do to enhance TLL or per one Burnwell said. They misuse narcotic L3-LGE6. Matic steroids, diuretics, and certain hormones in sports to increase muscle strength, MD bulk, and to promote DTE aggressiveness, and as a result increase athletic performance. The side effects of the use of metabolic steroids in females include masculinization features like NWS, increased aggressiveness, NDCDS, means repression, abnormal menstrual cycles, XII, hair growth on the face and body, enlargement of clitoris, re-edging of voice. In males it includes acne, increased aggressiveness, autism, depression, reduction of size of the testicles, decreased sperm production, potential for kidney and liver dysfunction, breast enlargement, premature baldness, enlargement of the prostate gland. These effects may be permanent, really prolonged. Use in TLL adolescent male or louisa or facial on body acne and premature closure of the growth centers of the long bones may result in stunted growth. 8.5 For prevention and control. The old adage of prevention is better than cure holds true here also. It is also true that habits such as smoking, taking drug or alcohol are more likely to be taught and up at a young age, more during adolescence. Hence it is test to identify these situations that may push adolescent to MDS use of drugs or alcohol, and to tolls rare leisures TL in time HL. This regard, TLL parents and teachers have a special responsibility. Parenting that combines with high levels of nurturance and consistent discipline has been associated really lowered NSK of substance, alcohol, drugs, tobacco abuse. Some of the measures mentioned here would be particularly useful for prevention on control of alcohol and drugs abuse among adolescents. I avoid undue peer pressure. Every child has his her own choice and personality, which should be respected and nurtured. A child should not be pushed unduly to perform beyond his her threshold limits, be it studies, SP, RDS, or other lilies. Education and Counseling 719 Educating on CO of selling him her to face problems and stresses. And to accept disappointments and mules as a PMT of life. It IOUID also be worthwhile to canalize the child's energy into healthy pursuits like sports, reading, RRU, sick yoga, ANCT, artillery.ct, rockhomeku1m. Activities 3 Source and 9 Help from parents and peers. Help from parents and peers should be sought immediately so that they can use appropriately. Help may even be sought from close and trusted friends, besides getting proper advice to sort out their problems. This LD help, long to vent their feelings of anxiety on GULT IJ LULD 719 for DA 719 are signs alert parents and teachers need to look to and identify the dangerous GNS discussed Ellen Nens 2 They find Sorian using drugs or alcohol should not hesitate to bring TLLs to TLLE notice of parents or teacher in the best interests of the person concerned appropriate measures. LD then be record to diagnose the malady and the underlying causes. This old help in initiating proper remedial steps or treatment. JCK9 National and medical help. A lot of help is available in the form of highly qualified psychologists, psychiatrists, anti-addiction and rehabilitation programmers to help individuals who have et fortune intelligent in their of drug alcohol abuse. With such help the affected individual with sufficient efforts will PD can rid of the problem completely and lead a perfectly normal unfulfilled. Sumiiri Haru else not just the absen, of Dice said. It one as a state of com page 1 8 ph monosoc, a on psychological well-being, DLCOs is like typhoid. Color pneumonia FLML affections of skin M3L and MMY oiliers me a major cause of distress to human. Banks, Victor Bori dies like IMLMTAS colon 1 aliore caused by SMOD CP rum 1F not treated. May pro fatal, is 1 des personal cleanliness mel hygiene public RUL measures like proper disposal of W3 sta, deco N of DN and K1 and water. Control of RS like mosquitoes use alien me I helpful in pre-owned MGULES diseases. Our immune system plays a major role 1 and pre-anti-ing ila east 3 says when we me exposed to tsquas, ing NTS. The humid deferos of our body like SK1 and mucus me, RNB arpmas. Turn 1 crob 1 al sub as present in our TEMs. Saliva on the pH, cyoxy, else help to block the entry al pathoms into our body, if the patho ms su, C, E, LLNG, 1 and 1 ang on 1 right to our body. Spec, NCONL the body's humoral immune response C, elzo mediated UMW, E response M to kill ila east patho else. In E system E, urinary, on subsequent exposure to same paleage, and IMT response is rapid more intense. Nusephoros the Bas 1S of protection 163. IIG ordered by N and TMMUI ISA Yuan, among other dice AIDS, E, R kill a 1M number of MDHWs worldwide. AIDS caused, T, by the HUMCM immunodeficiency WS HIV1S fatal bocom B, pre. OE, TED1 FOE, RTA1 NPR, COT1 ONSMEO 4 CAN-M, 3 NAI, RSA, recur WLE1F DEDIO, RLYI, PropNet Throput 1 Semioswas, token. Of late, drug and alcohol abuse among Oulia adolescents, NTS1S became mother cause of course, RN. Because of the addictive nature of alcohol, drugs and their EIE, LB, nefts like relief from stress. A person may try isolifao of pressure, examina ret dead, comp, it 1 orlet, L stresses. In doing so she may get addicted, to educat 1 on without their harmful effects. Cotton selling seeking, in 1/8 professional medical help would totally relt Elia, DYUA1 from these evils. FXFRC, is FS1, what me the imtus public L, milli measures which ho would SU, F, street os soligo, Zioto NSL and Fiawas 2. In which way has the study of biology helped us to control infectious diseases 3, how does Ilya TRN of each of Ilya following says tar 1, E place Arnold 1, SSBMT Nasiasco, R, SLSDP Nia 4. What me 3 Suviol you take to pre.E, ND whale, R born D1 Se 3 says 5, discuss WTH0 U teacher what does a sub L3 BLETME means. Olilia coned, XT of DNAV ACM 6, known the PRTRN my I second, Rylernfo 1 DE, G, R, S7, the following me some well-known abbreviations. VHI been used all, ULAS chapter, XECCH1 to its fun form, AMWAL BCMIC AIDS D NACO HIV 8. DLF Arenu Tevi following examples all EA, AJLNN, D. Melikwood Ulimmunity BKTIE. Malpassi EGMMWIID 9. Draw a VL label D1 AGRMN of M. T body molecule 10, what me the VMS roots by which LR, LNSIIIs, strong of Hern, MULMO, Delic, Onceilris voice PL, set 11. What LS the metrojournalism by which the AIDS illris is Delic, Once of immune system of Ilya 1 and nicted person 12. How on S arouse old E1 FFE, rent from a normal ol 13, exp 31 and what 1 S me and T by M14, us the H, Renful FX, S caused by a Bahul DRVG abuse. 15. Do you THL and Kavif nons convergluence 1 to toka Bahul DRVGS if yes H, LW may reprotect learn self he, self from SLCH. Empton flu, row 16, Y1S E. A person stalls taking alka, LL or drugs at 1SD1 called to farid of use habit discuss it WL. We teacher 17, in review what motivates OS to T, 3K to alcohol or drugs. Melhoamulis be a .01 dead. Unit VLLL Biology and Human Welfare Chapters Human Health and Disease Chapter 9 Strategies for Enhancement and Food Product Line Chapter 10 Microbes and Human Welfare Biology is the youngest of LHE formalized disciplines of natural science. Progress in physics and chemistry preceded much failure than in biology. Applicable lines of physics and chemistry in our daily life have a higher visibility than those of biology. However, 20th century and certainly the 21st century has demonstrated the utility of biological knowledge in furthering human welfare, be it in health sector or agriculture. The discovery of antibiotics, and synthetic plant-derived drugs, anesthetics have changed medical practice on one hand and human health on the other hand. Life expectancy of human beings have dramatically changed over LHE years. Agricultural practices, food processing and diagnostics have IOC ultra-I changes in human communities. These are briefly described in the following 3 chapters of his Unal. M.S. Swaminathan 1925 born in August 1925 in Kumbakonam in Tamil Nadu, Munkambu Sambasavan Swaminathan did his graduation and post-graduation in botany from Madras University. He worked in different capacities in large number of institutions in India and abroad and developed his expertise in genetics and plant breeding. The School of Cytogenetics and Radiation Research established at the Indian Agricultural Research Institute IRI enabled Swaminathan and his team to develop short-duration high-yielding varieties of rice including scented basmati. He is also known for the development of the concept of crop cafeteria, crop scheduling and gene ticketing improving the yield in Quailili. Swaminathan initiated collaboration with Norman Borlaug, which culminated in the Green Revolution through introduction of Mexican varieties of wheat in India. This was highly recognized and appreciated. He is also the initiator of Lab Lowland, food security and several other environmental programs. He has been honored with Padma Bhushan and several other prestigious awards, medals and fellowships by institutions of excellence. Chapter 8 Human Health and Disease 8, Common Diseases TN, H 8.2 liters I, J 8.3 AIDS 8.4 Cancer 8.5 D9 S on AICDD. Abuse health F for a long theory was considered as a state of XII.MD mind where Lear W is about 3 NCERT NHUFDFS. This is what early Greeks like Hib, U, R8 is low, L is land and Ayurveda system of medicine asserted. It was thought that persons with plague bile belonged to hot person 3HLF. And would have fevers. This idea was arrived at by pure reflective thought. The desanery of blocks of circulation by Imliam honey using experimental rests along the demonstration of normal XII temperature in persons with plague bile using thermometer disproved the good horror hypothesis of health in later years. Biology stated that mind influences through neural system and endocrine system or immune WLE system.n. Flat our immune system new and lanes our health, hence mind. And mental state can affect our health. Of course health was affected by IGNEDC disorders deficiencies v. LTLL which a child is torn on defeinius defects which the child inherits from parents from birth, IL infections on 3 lifestyle including food and water we. Rest and E. Rare ease we guile to erdodes H3 pitsulit v. E have or lack etc. 8KIIOGY The term health is very frequently used by everybody. How do we define? T health does not simply mean sense of to see our physical fitness. It could be defined as a state of complete physical, mental and social well-being. When people are healthy, they we more efficient, work. This anchor, says T and brings economic prosperity. Health. Also increases longevity of people. And reduces infant and maternal mortality. Balanced diet personal HJL and Andrea Braces are very important to int and good health. Yoga has been practiced since Royal to achieve physical. And mental health. Awareness. About diseases and TLL effect on different FWL Xions vaccination immunization. Against infectious disease, as is proper disposal of wastes control of vectors. And maintenance of hygienic FBCD. And water resources. Are necessary FBR, thieving good health. When the functioning of one or more organs or systems of the XII is adversely affected 10 said by V new signs on symptoms. Say TLL, at V, E are not healthy that is, low, E have a disease. Diseases can be broadly grouped into infectless. And non-infectless. Diseases which are easily transmitted from one person to another. Are called infectless diseases. Infectious diseases. Are very only idle. And every one of us suffers from TLLs at sort or other. Serif TLL infectious diseases like AIDS. Or FDL, non-infectious diseases cancerous TLL major cause of CTE at drug and alcohol, abuse also, fleet our health, D1 Ersly 8.1 Co1 TT1 TT on LLSSL, SK, SLNY, SAD, wide range of organisms belonging to bacteria. Viruses FWLG protozoans Helmini and TLLS etc. could cause diseases in all. Such disease causing organisms are called pathogens. All parasites are TLL P. Pathogens, STC, use harm to the host by leaving in or on them the P. Pathogens can enter or be nutrients. Multiply and interfere with normal DAL, CTI villies resulting in morphological and functional damage. Pathogens H1E2, D, PT to life within the environment of the host, for example the P. Phagns that enter the gut rust no way of surviving in the stomach, T low pH and resisting the venous digestive enzymes. A few representative members from different groups of pathogenic organisms are discussed here. Along with TLLE disease is caused by the Prevent1E and UN1 raw reagers, against disease, says in general, also briefly described. Salam, NEUO, TJPU is, P, pathogenic bacterium which C, uses typhoid Phe1 or in human beings. These pathogens generally enter the small intestine through food and water contaminated with them, NCD migrate to other organs through blood. Suswind high fever 39, to 4C weakness stomach pain stip, cheyenne headache and loss of appetite. Some of the common symptoms of this disease intestinal perfor, cheyenne, 10, th may occur in severe cases. Typhoid fever could be confirmed by will now test a class sick case in Weediine that of M3II long Nickner TGJ, IO, DMARG is worth mentioning here. She was a cook by profession. L was a typhoid CM1er who continued to spread typhoid for several years through the FCX. D she prepared bacteria like S. Tasakicru S. pneumorvae and Liaum, PLVBTS and Z are responsible for Odysseus and Unanwana in Hunwals which infects the alveoli air-filled sacs of the LWLGS. As a result of the infecti on the alveoli GT61 led with fluid leading to severe problems in respiration. The symptoms of Nunditia insular Phe, or chills cough NCT headache. In severe cases the lips and fingernails may turn greater bluish insular. A healthy person exuires the infection by inhaling the droplets aerosols released by M infected person or even by SH mingluses. And utensils with an infected person, dysentery plague diphtheria, etc. are sore of the other bacteria. 31 diseases in NWL, M3 viruses also cause diseases in human beings. Rhinoviruses represent one such group of viruses which cause one of TL1E and DST infectious and the common cold. They infect the nose and respiratory passage but not the lungs. The Umaran cold is characterized by nasopharyngitis and discharge. Sore throat, hoarseness, cough, headache, tiredness, etc., which usually last for 3-7 days. Droplets resulting from cough or sneezes of an infected person or either INH3 led directly or transmitted TLU roof contaminated objects such as pens, books, cups, port knobs, computer, keto, RD or news, etc. and cause infection in a healthy person. Sore of the human diseases are caused by protozoans too. You might have heard tout malaria. Disease has been fighting since MMY years. PKTSM, D, Amatonipactazone is responsible for this disease. Different species of FTOSM, DMPOXP malum and P. parum are responsible for differential pisophenol. Of these Mo-G8 land RNA1 are caused by FTOSM, Diamphocyporum is RMST serious one and can even be fatal. Let us a glance at the life cycle of PASM. Dium figure 8.1. Ftosmodium enters the human Xlia sporozoites infectious form TLU roof the bite of infected female anaphil. ESM, squito. The parasites initially relatively within the Lear cells and then attack the red BLCX, D cells RBCs resulting in their rupture. Therapy of RBX is associated with real as of to Zik substhrine sehal which is responsible TBR chill and high fever recurring ERE 3 to FR days. When a female energy ESN squiddo bites an infected person TLLE's parasites enter the mosquit cell, body and undergo further development. The parasites relatively within them to form sporozoites that are stored in their salivary glands. When Lees mosquits bite HUFWL, Lee SP, rhozoites are intro, lucet into his herbxley. Thereby iaing the unsem ion octavo and erasing to note TLL email re 3.1 parasite requires two hosts human.n. From squios to complete its life cycle figure 8.1, the female anoples from squitois the vector transmitting agent 2. Full stop, F is, UE 8.1 in U, E life cycle of CR smod Clementomojxtlatist, K, is a protozoan parasite in Lee JMGE intestinate herm which causes Moblasils Mobic DJ celtici. Symptoms of TLL is dysphagia, insular constipation, abdominal pain, and cramped stools with E. Sesmakuesenplo, D. Clots, houseflies act. As rich 3 Nicalcine and serve to T-arm-Smithley parasite from feces at infected person to fee, odd. Human health and disease and food products, thereby contaminating them. Drinking water and food contaminated by the fecal matter are the main source of infection. Ascaris, the common roundworm and Wuchereria, the filarial worm, are some of the helminths which are known to be pathogenic to man. Ascaris, an intestinal parasite, causes ascariasis. Symptoms of this disease include internal bleeding, muscular pain, fever, anemia and blockage of the intestinal passage. The eggs of the parasite are excreted along with the feces of infected persons which contaminate soil, water, plants, etc. A healthy person acquires this infection through contaminated water, vegetables, fruits, etc. Wuchereria W. Bancroftshi and W. Malayi. The filarial worms cause a slowly developing chronic inflammation of the organs in which they live for many years. Usually the lymphatic vessels of the lower limbs, and the disease is called elephantiasis or filariasis. Figure 8.2. The genital organs are also often affected, resulting in gross deformities. The pathogens are transmitted to a healthy person through the bite by the female mosquito vectors. Many fungi belonging to the genera Microsporum, Trichophyton and Epidermophyton are. Figure 8.2 diagram showing inflammation in one of the lower limbs due to elephantiasis, responsible for ringworms, which is one of the most common infectious diseases in man. Appearance of dry, scaly lesions on various parts of the body such as skin, nails and scalp. Figure 8.3 are the main symptoms of the disease. These lesions are accompanied by intense itching. Heat and moisture help these fungi to grow, which makes them thrive in skin folds such as those in the groin or between the toes. Ringworms are generally acquired from soil or by using towels, clothes or even the comb of infected individuals. Figure 8.3 diagram showing ringworm-affected area of the skin. Maintenance of personal and public hygiene is very important for prevention and control of many infectious diseases. Measures for personal hygiene include keeping the body clean, consumption of clean drinking water, food, vegetables, fruits, etc. Public hygiene includes proper disposal of waste and excreta, periodic cleaning and disinfection of water reservoirs, pools, cesspools and tanks, and observing standard practices of hygiene in public catering. These measures are particularly essential where the infectious agents are transmitted through food and water such as typhoid, amoebiasis and ascariasis. In cases of airborne diseases such as pneumonia and common cold, in addition to the above measures. Close. 8KIIOGY contact VATLL infected persons or TLL or telengings should be aware for diseases such as malaria and filariasis LLI RT arms mid TLL rough insect vectors. The most important measure is to control or eliminate the vectors on their breeding places. This can be achieved by avoiding stagnation of water in and around residential areas. RLR cleaning of household coolers. UC1 mosquito nets introducing fishes like OMFX.SA. In ponds that feed on Roscudo 13N, OE, spraying of insecticides in ditches, drainage areas and SWOMPs, etc. In addition, doors and winkposts should be protected with wire mesh to prevent the entry of mosquitoes. Such precautions be Rome are important, especially in the light of recent low-lying incidences of Leicester-borne Aedes nusquitzes diseases like dengue and chikungunya in many ports of India. The active microbes made in BLOCL science armed us to detect RRW1Y infectious diseases. The use of vaccines and iron mineralization programs have enabled us to completely eradicate a deadly disease like smallpox. A large number of other infectious diseases like polio, diphtheria, pneumonia and tetanus have been controlled to a large extent by the use of vaccines. Biotechnology, about which you will read more in Chapter 12, is at the verge of making available on safer vaccines. Discovery of antibiotics drugs has also enabled us to affect the illiterate infectious diseases. Why every day are exposed to a large number of infectious agents, however only a few of TLL's exposures result in disease. Why this is due to the fact that the body is able to defend itself from LDS of these UEI agents. This overall ability of the host to fight the disease using organisms conferred by the immune system is called immunity. Immunity is of two types, innate immunity and acquired immunity. 2. 1. Innate immunity. Innate immunity is non-specific type of defense. TLL, is present at the time of birth. This is accomplished by putting different types of barriers to the entry of the fibrin, GN agents into our body. Innate immunity consists of FR types of barriers. These are physical barriers. Skin on our body is the barrier which prevents entry of microorganisms. Mucus coating of the epithelium lining the respiratory, gastrointestinal and urogenital tracts also help in trapping microbes entering our body. Physiological barriers, acid in the stomach, saliva in the mouth, tears, foamies all prevent microbial growth. 3. Cellular barrier S. Certain types of leukocytes WBC of our body like polymorphonuclear leukocytes PMNL, neutrophils and human health and disease monocytes and natural killer type of lymphocytes in the blood as well as macrophages in tissues can phagocytose and destroy microbes. 5. Cytokine barriers. Virus-infected cells secrete proteins called interferons which protect non-infected cells from further viral infection. 8. 2. Acquired immunity. Acquired immunity, on the other hand, is pathogen-specific. It is characterized by memory. This means that our body when it encounters a pathogen for the first time produces a response called PRIL, 8-Li response which is of low intensity. Subsequent encounter with the same pathogen elicits a highly intensified secondary or anamnestic response. This is ascribed to the fact that our body appears to have memory of the first encounter. The primary and secondary immune responses are carried out with the help of two special types of lymphocytes present in our blood. That is, B lymphocytes and T lymphocytes. The B lymphocytes produce an army of proteins in response to pathogens into our blood to fight with them. These proteins are called antibodies. The T cells themselves do not secrete antibodies but help B cells produce them. Each antibody molecule has 4 peptide chains, 2 small called light chains and 2 longer called heavy chains. Hence, an antibody is represented as H2L2. Different types of antibodies are figure 8.4 structure of an antibody molecule produced in our body. LGA, IgM, LGE, IgG are some of them. A cartoon of an antibody is given in figure 8.4. Because these antibodies are found in the blood, the response is also called as humoral immune response. This is one of the 2 types of our acquired immune response, antibody-mediated. The second type is called cell-mediated immune response or cell-mediated immunity CMI. The T lymphocytes mediate CML very often, when some human organs like heart, eye, liver, kidney fail to function satisfactorily. Transplantation is the only remedy to enable the patient to live a normal life. Then a search begins to find a suitable donor. Why is it that the organs can't be taken from just anybody? What is it that 8 KIIOGY the daughters check or 3 FTS from just any source. M.animal. Mother primate or any beings cannot be mode since the graft cell D be rejected sooner or later. Tissue matching gluco, L group matching, re-assemble before undertaking any graft transplant and after this the patient has to, EMA presence. U his or life. The XII is able to differentiate self.md non-self.mct Lee cell radiated immune resp 01 says resp, NCERT F with graft rejection. 8. 2. 3. Active and passive immunity when a host is exposed to antigens which may be in the F form of living or dead microbes or higher proteins. Antibodies are PM, TU-seed and Lee host bacti. This type of immunity is called ACUVLM will T. Actal immunity is slow. N. T3 has time to give its full effect ERSP01 cent. Injecting Lee microbes well either 8 leak tunning immulize cheyenne or infectious organism SGAning. Access into body during an natural infection induced. Active immunity. When reacting mocked antibodies are directly G, then to protect the body against foreign agents it is called passive immunity. To go no co WHGM, Thursmac S cons, L erdoregescent, U for the newborn in feign the jello wish fluid colostrum secreted B, Y rather during the initial days of lactation has abundant antibodies I get to protect the infant. The fetus. Also reset ES sort. Antib, dies from TLL or Mottler through the placenta during 11G pregnancy. These are sorry amples of passive immunity. 8. 2. For vaccination and immunization the principle of immunolicity honor of vaccination is based on TLL property of marriage of the immune system. In vaccination a preparation of antigenic proteins of pathogen or inactivated fucking pathogen vaccine or introduced into the body. The antibodies produced in the XII against these antigens would neutralize TLL pathogenic agents actual infection. TIL vaccines also generate IUFBMDT cells that recognize the pathogen quickly on subsequent Posure.md. Overwhelmingly in V actors via MSC. Reproduction of .mtibds, a person is infected. The source CTEADL microbes to which quick immune responses required. As intent must we need to directly inject TLL pre-1I, med. Antibodies are antido in a preparation unlaning antibodies to the 2N, Ln in cases of Snocobites. The injection which is gene to the patients contain for honored. Antibodies. Agonist TTLLESMKNM. This type of immunization is called passive immunization. Recombinant DNA technology has allele, D the production of antigenic pol, if applies of pathogen in bacteria or yeast. Vaccines produced using this approach allow 13 RGE scale production and hence greater availability for individualization. For example hepatitis B vaccine produced from yeast. S2, 5 AURGs did this Rupen 2, UN, U113 go to a new place 3 JLD Sockle, Lenley, you started sneezing wheezing for no explained reason. 3JLD when you can't your symptoms disappeared as of us are sensitive to some particles in the environment. The above mentioned reaction could take because of allergy to pollen mites etc. which are different in different places. The exaggerated responsible immune system to certain antigens present in the environment is called energy. The substances to which such iron responses PM, TUC are called allerg, MS, tile antibodies produced to these are of I type. Ojran examples of allergens are mites and dust, P, lens, animal dander, etc. Symptoms of allergic reactions inhaler sneezing, watery ES, running nose and difficulty in breathing. Allergy is due to the release of chemicals like histamine and serotonin from the mast cells. For determining the cause of allergy, the patient is exposed to or injected with LARISFLL3LL-P, S-soluble allergens. MD reaction studied, the use of drugs like anti-histamine, adrenaline and steroids quickly reduce the symptoms of allergy. How, day lifestyle rules resulted in lowering of ILLD immunity and more sensitivity to 3 allergens N. Children in retro cities of India suffer from NERG, S and asthma due to city environment. This could be because of the protected environment provided early in life. 6 autoimmunity memory. Basect acquired ILLD immunity evolved in higher vertebrates based on the ability to differentiate to ignore organisms for example. Palo, ins from self cells. While we still do not understand the basis of this, Coral 3 rise of this ability to be understood. 1. Hypnovertebrates can distinguish ROR, GN molecules as well as ROR, GN organisms. Most of the parental immunology deals with this aspect. Sornal due to genetic and other W1KMWN reactions, the body attacks self cells. This results in damage to Lb, and is called autoimmune rheumatoid arthritis which affects MMY people in our society as MAU2 immune disease. 7 immune cyst. In the body the human immune system consists of LMPH tissues. Cells 3JLD soluble molecules like antibodies, as Ho113E read, iron TNE system is unique in sense TLL, and it recognizes foreign antigens. Response to these 3JLD remembers them. The immune system also plays an important role in allergic reactions. Autoimmune diseases. And organ transplantation, lymphoid organs. These are the organs where under maturation 3JLD proliferation of lymphocytes occur. The primary lymphoid organs are bonomato 3JLD thymus where immature lymphocytes differentiate. Figure 8.51 Thymus diagrammatic representation of lymph nodes biology into antigen-sensitive lymphocytes. After maturation the lymphocytes migrate to secondary lymphoid organs like spleen, lymph nodes, tonsils, Peyer's patches of small intestine and appendix. The secondary lymphoid organs provide the sites for interaction of lymphocytes with the antigen, which then proliferate to become effector cells. The location of various lymphoid organs in the human body is shown in Figure 8.5. The bone marrow is the main lymphoid organ where all blood cells including lymphocytes are produced. The thymus is a lobed organ located near the heart and beneath the breastbone. The thymus is quite large at the time of birth but keeps reducing in size with age and by the time puberty is attained it reduces to a very small size. Both bone marrow and thymus provide microenvironments for the development and maturation of lymphocytes. The spleen is a large bean-shaped organ. It mainly contains lymphocytes and phagocytes. It acts as a filter of the blood by trapping blood-borne microorganisms. Spleen also has a large reservoir of erythrocytes. The lymph nodes are small solid structures located at different points along the lymphatic system. Lymph nodes serve to trap the microorganisms or other antigens, which happen to get into the lymph and tissue fluid. Antigens trapped in the lymph nodes are responsible for the activation of lymphocytes present there and cause the immune response. There is lymphoid tissue also located within the lining of the major tracts respiratory, digestive and urogenital tracts called mucosal-associated lymphoid tissue MALT. It constitutes about 50% of the lymphoid tissue in human body. 8.38 The word AIDS stands for acquired immunodeficiency syndrome. This means deficiency of immune system, acquired during the lifetime of an individual indicating that it is not a congenital disease. Syndrome means a group of symptoms. AIDS was first reported in 1981 and in the last 25 years or so, it has spread all over the world killing more than 25 million persons. AIDS is caused by the human immunodeficiency virus HIV, a member of a group of viruses called retrovirus, which have an envelope enclosing the RNA genome. Figure 8.6. Transmission of HN infection generally occurs by a sexual contact with infected person, B by transfusion of contaminated blood and blood products, C by sharing infected needles as in the case of intravenous drug abusers, and D from infected mother to her child through placenta. So, people who are at high risk of getting this infection includes individuals who have multiple LLLM, Health A, OOMFLL, all, a CDLLLT, WRNAMP:01BJT, HLM, 6DCD null, Li4 ocbidol. Willy Barloff FT1JTCD Fe1 eq, FIS, Ur 8.6 replication of Retric MRU, SXW, L partners. Drug octets who took drugs intravenously in D.modules who require repeated BLCX, D transfusions on children born to an HIV-infected her. Do go, now and do PXE need repeated blood, to Rufus on FGND out at DM, all, EAI straight of such conjugations. It is important to note that HIV-1 AIDS is not spread by rear touch or physical contact, it spreads only through XII fluids. It is hence imperative for the physical and psychological well-being that the HIV AIDS-infected persons are not isolated from family and society. There is ALWI's a time bed and the infection and appearance of AIDS symptoms. This period may vary from a few months to many years, usually 5-10 years. 8KIIOGY after Jitingantalb die after Perskin the was enters into myas where RNA of Tre virus replicates to F-worm WLDNA with the help of the ENSCH, Iron scriptase this WOLDNA. Ilzino, UP, latidentalized cells DNA and directs the infected cells to produce virus particles figure 8.6. The Microbes to lose was and in this way acts like H factory, simultaneously, hive enters into helper TIJM phocytes T. J replicates and lose progeny uses, the progeny is released in the blow, D-addict helper IJM phocytes. This is repeated leading to a progressive decrease in the number of helper lymphocytes in the XII of the infected person. During this period the person suffers from bouts of Feller diartho and GHT loss, due to decrease in the number of helper lymphocytes. Tre person SWTS suffering from infections that would have been otherwise 011 errors such as those due to Bacteries PUIM cogs or denim. Viruses, fungi and even parasites like toxoplasm, the patient BRSOMRTNO deficient she is unable to protect herself, against these infections. A widely used E-stick test FBR AIDS is enzyme limited non-IDL060I beta-species Treatment of AIDS with antiretroviral drugs is only partially effective. They can only prolong the life of the patient but C3JLLLD. Pre-entry, which is inevitable, prevent, N of AIDS, as AIDS has no cure. Prevention is the best option. Murmur, HIV infection M, often spreads due to conscious THR patterns and is hing the happens inadvertently like pneumonia or typhoid of course. Infection in blow, detransfusion patients newborns from third etc., may take place due to poor monitoring. The only excuse may be ignorance and it has been NGHUI so, CTCB and die of ignorance. In our country the National AIDS Control Organization NACO and other non-governmental organization NGOs are being to educate people about AIDS. WHO has started number of programs to prevent the spreading of HIV infection, making BLCX, D from BLCX, D banks safe from HIV ensuring the use of a disposable needless and sarin. Is in public and private hospitals and clinics free distribution of condoms controlling drug abuse. Educating safe set, and promoting regular checkups. The junior hive and susceptible populations are some such steps free can up. Infection with HIV or having AIDS is oral head should not be hidden since then the infection may spread to many rower people. HIV AIDS infected people need help and sympathy instead of being shunned by society, unless society uses it as problem to be dealt with. In a collective NWLLR the chances of ICID spread of GLLE disease increase manifold. It is, malady that can only be tackled by the society and medical fraternity while acting together to prevent the spread of the disease. Cancer is one of the most dreaded diseases of human beings and is a major cause of death all over the globe. More than 1 million Indians suffer from cancer and a large number of them die from it annually. The Organisms TLL adjunctive development of cancer or oncogenic transform alien of cells. Its treatment and control have seen some of the NDST intense areas of research in biology.mctm, design. In ARBXLE cell growth and differentiation is highly controlled and regulated in cancer cells. TLLR is breakdown of Lee's regulatory mechanisms. Normal cells show a property called contact inhibition by virtue of which contact with all your cells inhibits Leer WL controlled growth. Cancer cells appears to have lost this property. As a result this cancerous cells just continue to divide giving rise to masses of cells called tumors. One tumors are of TO12, benign and malignant. Benign tumors normally remain confined to their origin 3 location and do not spread to other parts of the XII and cause little DE. The malignant tumors on the other hand are a mass of proliferating cells called neoplastic or tumor cells. These cells are very rapidly invading and eating the surrounding normal tissues. As these cells actively divide, and they also starve the normal cells by impelling through vital nutrients. Cells sloughed from such tumors reach distant sites through BLC, XXL on where they get lodged in the body. They start a new tumor there. This property called metastasis is the most feared property of malignant tumors. Causes of cancer. T-arms formation of normal cells into cancerous neoplastic cells may be induced by physical, chemical or biological agents. These agents are carcinogens, long-lasting radiations like X-rays and gamma rays on non-ionizing radiations like UV cause DNA DE leading to neoplastic transformation. The chemical carcinogens present in tobacco SNDK have been identified as a major cause of cancer. C3 NCERT using whose is called oncogenic uses high genes called V1 RAL oncogenes. Furthermore, several genes called cellular oncogenes C1 or proto-oncogenes have been identified in normal cells which when activated under certain conditions could lead to oncogenic transformation of the cells. Cancer detection and diagnosis. Aortic detection of cancers is essential as XII lead disease treated successfully in NWLF cases. Cancer detection is based on biopsy and histopathological studies of the tissue. In blood, DN. Bone marrow tests FBR increased cell count TS in case of leukemias. In biopsy, a piece of the suspected tissue cut into thin sections is stained and R9 under microscope. Histopatho studies by a pathologist. Techniques like radiography, use of X-rays, CT computed tomography and MRI magnetic resonance imaging are very useful to detect cancers of the internal organs. Computed tomography uses X-rays to generate a three-dimensional iron eye of the internals of an object. MRI uses strong Fro3L81 Ig fields and idealizing radiations to evaluate L1CL3 tech pathological and physiological changes in the LLW-G tissue. Antibodies. Against cancer-specific antigens are also used for detection of certain cancers. Likes of molecular biology can be biology applied to detect genes in individuals with inherited susceptibility to certain cancers. Identification of such genes, which predispose an individual to certain cancers, may be very helpful in prevention of cancers. Such individuals may be advised to avoid exposure to particular carcinogens to which they are susceptible, e.g., tobacco smoke in case of lung cancer. Treatment of cancer. The common approaches for treatment of cancer are surgery, radiation therapy and immunotherapy. In radiotherapy, tumor cells are irradiated lethally, taking proper care of the normal tissue surrounding the tumor mass. Several chemotherapeutic drugs are used to kill cancerous cells. Some of these are specific for particular tumors. Majority of drugs have side effects like hair loss, anemia, etc. Most cancers are treated by combination of surgery, radiotherapy and chemotherapy. Tumor cells have been shown to avoid detection and destruction by immune system. Therefore, the patients are given substances called biological response modifiers such as interferon which activate their immune system and help in destroying the tumor. 8.5 Drugs and alcohol abuse surveys and statistics show that use of drugs and alcohol has been on the rise, especially among the youth. This is really a cause of concern as it could result in many harmful effects. Proper education and guidance would enable youth to safeguard themselves against these dangerous behavior patterns and follow healthy lifestyles. The drugs, which are commonly abused, are opioids, cannabinoids and coca alkaloids. Majority of these are obtained from flowering plants. Some are obtained from fungi. Opioids are the drugs, which bind to specific opioid receptors present in our central nervous system and gastrointestinal tract. Heroin figure 8.7, commonly called smack, is chemically diacetylmorphine, which is a white, odorless, bitter crystalline compound. This is obtained by acetylation of morphine figure 8.7, which is extracted from the latex of 0 figure 8.7 chemical structure of morphine figure 8.8 opium poppy. Human health and disease poppy plant Papaver somniferum figure 8.8, generally taken by snorting and injection. Heroin is a depressant and slows down body functions. Cannabinoids are a group of chemicals figure 8.9, which interact with cannabinoid receptors present principally in the brain. Natural cannabinoids are obtained from the inflorescences of the plant Cannabis sativa figure 8.10. The flower tops, leaves and the resin of cannabis plant are used in various combinations to produce marijuana, hashish, chars and ganja. Generally taken by inhalation and oral ingestion, these are known for their effects on cardiovascular system of the body. Figure 8.9 skeletal structure of cannabinoid molecule coca alkaloid or cocaine is obtained from coca plant Erythroxylum coca, native to South America. It interferes with the transport of the neurotransmitter dopamine. Cocaine, commonly called coke or crack, is usually snorted. It has a potent stimulating action on central nervous system, producing a sense of euphoria and increased energy. Excessive dosage of cocaine causes hallucinations. Other well-known plants with hallucinogenic properties are Atropa belladonna and Datura figure 8.11. These days cannabinoids are also being abused by some sportspersons. Drugs like barbiturates, amphetamines, benzodiazepines, lysergic acid diethylaronides LSD, figure 8.10 leaves of cannabis same and other similar drugs that are normally used as figure 8.11 flowering branch of Datura medicines to help patients cope with mental illnesses like depression and insomnia, are often abused. Morphine is a very effective sedative and painkiller, and is very useful in patients who have undergone surgery. Several plants, fruits and seeds having hallucinogenic properties have been used for hundreds of years in folk medicine, religious ceremonies and rituals all over the globe. When these are taken for a purpose other than medicinal use or in amounts frequency that impairs one's physiological or psychological functions, it constitutes drug abuse. 8KIIOGY smoking also paves the way to HMD drugs. Tobacco has been used by human beings for more than 400 years. Lettuce smoked. Chia, Dora used as a snuff. Tobacco contains a number of chemical substances including nicotine, an alkaloid. Nicotine stimulates adrenal gland to release adrenaline into blow, d, circulation. Bottle of which rise blow, d pressure. And increase EMT raw 10, SMG is associated really increase at incidence of cancers of lung. Uniriblet, IRANDDLUOOD bronchitis emphysema coronary heart disease gastric ulcer etc. Tobacco chewing is associated, really increase at risk of cancer of TLL or alcar1 type. Smoking increases carbon monoxide CO content in blood and reduces Lee concentration of hemalda, IgN. This causes IgN deficiency in the body. When one buys packets of cigarettes 1C3.1 a lot miss GLLE Street a tutorial warning that is present on the packing which warns against smoking and says how it is injurious to health. Yet smoking is very prevalent in society both arm young and old knowing the dangers of smoking and chewing tobacco. And its addictile nature the youth and old need to avoid these habits. Any addict requires counseling and medical help to get off the habit. 8.5L Adolescence and drug alcohol abuse. Adolescence bottle, a process, during which a child becomes mature in terms of his her activities and behaves for effective participation in society. The period between 12-18 years of age and 3 YBTLL out of AS adolescence period, in other words. Adolescence is a bridge linking childhood and adulthood. Adolescence is accompanied by several biological and B3B0 or LCH3 changes. Adolescence is a very vulnerable phase of mental and psychological development of an individual. Curiosity need FBR adventure and excitement and experimentation constitute and causes which motivate youngsters towards drug and alcohol use. A child's natura el curiosity motivates him her to efferent. This is complicated further by effects that might be personally led as benefits of 3 alcohol or drug use. Thus TLLE first use of drugs or alcohol and 3 YB out of curiosity or experiment alien but later TLLE child. Street ARTS using these to escape ing problems of late stress from pressures to excel in academics or exams. Has played a significant role in persuading the youngsters to try alcohol and drugs. The perception arm youth that it is cool or progressive to smoke, use drugs or alcohol. Is also in a way a new use for yoli to street art these H3 bits, television movies newspapers. Internet also help to promote this perception. Other actors that have been seen to be associated with drug and alcohol abuse ARRDNG8 plus NCERT slabla or unsupportive family structures and peer pressure. 8.5. To addiction and dependence because of the perceived benefits drugs are frequently used repeatedly. The RMSD important thing which one taught us to realize is the inherent addictive nature of alcohol and drugs. Addiction is a psychological attachment to certain effects such as euphoria and a temporary feeling of well-being associated with drugs and alcohol. These DTLE people to L3L, learn and when the are not nettered, L, are used tech or self-destructive. With repeated use of drugs the tolerance level of the receptors present in our top T increases. Q. Consequently the receptors respond only to higher doses of drugs or alcohol leading to greater and addiction. However it should be clearly borne in mind that use of these drugs even once can be a forerunner to addiction. Thus the addictive potential of drugs and alcohol pull the user into a vicious circle leading to their regular use abuse from which she may not be able to get out in the absence of any guidance or counseling. The person GTS3 ticked and because dependent on lear use. Dependence is the tendency of the body to manifest a characteristic and unpleasant will syndrome. 2 regular dose of drugs 1 alcohol is abruptly discontinued. This is characterized by anxiety, shockiness, nausea, MD, sweating, which may be relieved when used as Resurrect XII. In sore cases, withdrawal symptoms can be severe and even life-threatening, and the person may need medical supervision. Dependence leads the patient to neurosocial norms in order to get sufficient funds to satiate her needs. These result in many social adjustment problems. 8.5. Effect of drug alcohol abuse. The immediate adverse effects of drugs and alcohol abuse are manifested in TLLE form of reckless behavior. V3JLD, 3-lism. In while excessive doses of drugs may lead to coma and death due to respiratory failure, heart failure, or cerebral hemorrhage, a combination of drugs or their intake, along with alcohol, generally results in overdosing and even deaths. The most common warning SGNS of drug and alcohol abuse among youth include drop in academic performance, unexplained absence from school, college, lack of interest in personal hygiene, withdrawal, isolation, depression, fatigue. Aggressive. In rebellious behavior. Deteriorating relationships with family and FLE's loss of interest in hobbies. CH3NGE in sleeping. And eating habits fluctuations in GHT, appetite, etc. There may even be some far-reaching implications of drug alcohol abuse. If the abuser is unable to get money to buy drugs alcohol, she may turn to stealing. The adverse effects are just not restricted to the FT son who is using drugs or alcohol. FJI irons. A drug alcohol addict becomes the cause of rental and financial distress to his or family and friends. 8KIIOGY those who L3K drugs intravenously direct injection into leaving using a needle on syringe are much more likely to acquire serious infections like AIDS and hepatitis B, the viruses which are responsible for these diseases, or transferred from one person to another. Be shoring of infected needles and syringes. Both AIDS and hepatitis B infections are chronic infections and ultimately fatal. AIDS can be transmitted to one's life partner through GHC, UAL intact, while hepatitis B is transmitted through infected BLCX. D. The use of alcohol during adolescence may also have long-term effects. It could lead to heavy drinking in adulthood. The chronic use of drugs and delusional damage nervous system and liver cirrhosis. The use of drugs on delusional during pregnancy is also known to adversely affect the fetus. Another misuse of drugs is what certain sportspersons do to enhance TLL. Per one Burnwell said, they misuse narcotic L3LGE6. Matic steroids, diuretics, and certain hormones in sports to increase muscle strength, MD bulk, and to promote DTE aggressiveness, and as a result increase athletic performance. The side effects of the use of metabolic steroids in females include masculinization features like NWS, increased aggressiveness, NDCDS, means repression, abnormal menstrual cycles, XII, hair growth on the face and body, enlargement of clitoris, re-edging of voice. In males it includes acne, increased aggressiveness, autism, depression, reduction of size of the testicles, decreased sperm production, potential for kidney and liver dysfunction, breast enlargement, premature baldness, enlargement of the prostate gland. These effects may be permanent, really prolonged. Use in TLLE adolescent male or female or facial on body acne and premature closure of the growth centers of the long bones may result in stunted growth. 8.5. For prevention and control. The age-old adage of prevention is better than cure holds true here also. It is also true that habits such as smoking, taking drug or alcohol are more likely to be taught and up at a young age, more during adolescence. Hence it is test to identify these situations that may push adolescent to MDS use of drugs or alcohol, and to tolerate rare deal leisures TL in time. HL this regard, TLLE parents and teachers have a special responsibility. Parenting that combines with high levels of nurturance and consistent discipline has been associated Vili Laverde SK of substance, alcohol, drugs, tobacco abuse. Sarah, the measures mentioned here would be particularly useful for prevention on control of alcohol on drugs abuse arming adolescents. I avoid undue peer press. Every child has his her own choice and personality, which should be respected and nurtured. A child should not be pushed unduly to perform beyond his her threshold limits, be it studies, SP, RDS or Atlaracti lilies. Education and Counseling 719 educating on excelling him her to face problems and stresses. And to accept disappointments and mules as a PMT of life. It IOUID also be worthwhile to canalize the child's energy into healthy pursuits like sports, reading, RRU, sick yoga, ANCT, artillery.ct, rockhomeku1m. Activities 3 Source and 9 Help from parents and peers. Help from parents and peers should be sought immediately so that they can use appropriately. Help may even be sought from close and trusted friends, besides getting proper advice to sort out their problems. This LD help, long to vent their feelings of anxiety on GULT IJ LULD 719 for 719 are signs alert parents and teachers need to look to and identify the dangerous GNS discussed And thence too they find Sorian using drugs or alcohol should not hesitate to bring TLLs to TLLE notice not parents or teacher in the best interests of the person concerned appropriate measures. LD then be record to diagnose the malady and the underlying causes. This old help in initiating proper remedial steps or treatment. JCK national and medical help. A lot of help is available in the form of highly qualified psychologists, psychiatrists, anti-addiction and rehabilitation programmers to help individuals who have et fortune intelligent integrar, off drug alcohol abuse. With such help the affected individual with sufficient effort will PD can rid of the problem completely and lead a perfectly normal and fulfilled. Sumiiri haru else not just the absen, of dice said. It one as a state of calm page 1 8 ph monosoc, on psychological well-being, DLCOs is like typhoid. Color pneumonia FLML affections of skin M3 LMA and MMY oiliers me a major cause of distress to human. Banks, Victor Bori dies like IMLMTAS colon 1 aliori caused by SMOD umthig CP rum 1F not treated. May pro fatal, is 1 does personal cleanliness mel hygiene public RL measures like proper disposal of W, 3 sta, deco N of DN and K1 and water. Control of RS like mosquitoes use alien me I helpful in pre-owned MGU lease diseases. Our immune system plays a major role 1 and pre-anti-ing ila east 3 says when we me exposed to tsquas, ing NTS. The humid deferos of our body like SK1 and mucus me, RNB arcmas. Turn 1 crab 1 al sub as present in our TEMs. Saliva on the pH, cyoxy, Ls help to block the entry al pathogens into our body, if the patho ms su, C, LLNG, 1 and 1 ang on 1 right to our body. Spec, NCON alibodies I humoral immune response C, Lzo mediated UMW, response M to kill ila E's patho Ls. In system urinary, on subsequent exposure to same paleage, and response is rapid more intense. Nusephoros the Bas 1S of protection 163. IIG ordered by N and TMMUI ISA UON, among other dice AIDS, are kill a 1M number of MDHWs worldwide. AIDS caused, by the HUMCM immunodeficiency WS HIV1S fatal bocom B, pre. OE, TED1FOE, RTA1NPR, COT1 ONSMEL4 CAN-M, 3 NAI, RSA, recur WLE1F DEDIO, RLYI, PropNet Throput 1 Semioswas, token. Of late, drug and alcohol abuse among Oulia Dolaso, NTS1S became. Mother cause of course, RN. Because of the addictive nature of alcohol, drugs and their LB, nefts like relief from stress. A person may try ileazoliliafao of pressure, examinoans ret dead, comp, it one oralit, L stresses. In doing so she may get addicted, to educat one on without their harmful effects. Cotton selling seeking, in 1-8 professional medical help would totally relt Elia, to YUA1 from these evils. FXFRC, is FS1, what me the imtus public L, milli measures which would SU, F, street os soligo, zioto NSL and fiawistlaciosis 2. In which way has the study of biology helped us to control infectious diseases 3, how does Ilya TRN of each of Ilya following says TA 1, place Arnold 1, SSBMT Nasiasco, R, SLSDP Nia 4. What me 3 suviol you take to pre.E, ND whale, R born D1 sa 3 says 5, discuss WTH0 U teacher what does a sub L3 blatme means. Olilia coned, XT of DNA VACM 6, known the PRTRN my I second, Rylernfo 1 DE, G, R, S7, the following me some well-known abbreviations. VHI been used all, ULAS chapter, XECCH1 to its fun form, Amwal BCMIC AIDS D NACO HIV 8. DLF Irenu Teddy following ioxamples all EA, AJLNN, D. Meliquid Ulimmunity BKTIE. Malpassi EGMMWIID 9. Draw a VL label D1 AGRMN of M. T body molecule 10, what me the VMBus roots by which LR, LNSIIIs, Strong of Hern, MULMO, Delic, Onceilris voice PL, set 11. What LS the metrojournalism by which the AIDS illris is Delic, Once of immune system of Ilya 1 and nicted person 12. How on S arouse old E1 FFE, rent from a normal old 13, Exp 31 and what 1 S me and T by M14, us the H, Renful FX, S caused by a Bahul DRVG abuse. 15. Do you THL and Kavif nons conwardluence 1 to toka Bahul DRVGS if yes H, LW may reprotect learn self he, self from SLCH. Empton flu. Row 16. Y1S E. A person stalls taking alka, LL or drugs at 1SD1 called to farid of use habit discuss it WL. We teacher 17. In review what motivates OS to T. 3K to alcohol or drugs. Melhoamulas be a .01 dead. Chapter 9 Strategies for Enhancement in Food Production 9.1 Anima, H. Espand, Y 9.2 Ron, Breed 9 JS, N. Glasil Rhodius 9. 4 TSSVE Culture with ever-increasing population of the world, enhancement of FBCD production is a major necessity. BJO Jog, Cal principles as applied to Merle 3L husbandry and plant breeding Honem 3 jaw role inner efforts to increase food production. Savero new techniques like MH10 transfer technology on tissue culture techniquis orego, and to play a pividine further enhancing food production. ELANI, HT is Sanuju animal husband JFIsh the cultural practice of breeding. Met RAI sing livestock. As such it is .oil. A skill for fatmers. MD is as much science. As it is art, anernal husbandry reals with the care on breeding of LLS stock like buffaloes. Rose pigs hoxes cattle sheep corrals goats etc. that are useful to humans. Protected includes P. Ultra-iming on D fisheries. FL Sheens Inchler reorgan catching selling etc. of fish raw lists shellfish and crustaceans prawns. Crabs etc. Since Tyrial rendertal. N and W's like T's, silk, T, Iofi prawn crabs. Fishes birds pigs cattle sheep on carols have been used by HL3 JLS rower produce CTS like milk. Eggs VO, L, silk honey etc. It is estimated that more than 70% of the world livestock population is in India. N. China, however it is 81,010 GV. Surprising to note that the contribution to the world form produces only 25%. That is, the productivity per WL.IT is very low, hence in addition to all practices at normal bleeding male T, i.e. Nevel 3R technologies also have to be applied to achieve improvement in quality and productivity. TTY 9, 1 management of farms and farm animals. A professional approach to what have been traditional practices of farm man, Gmet gives then ruch netted boost to our food produce CIOO. Let us discuss the Emmet procedures MPI and now well form systems 9. JJJDY Fatmo Najimar NT Dairying is the managerant of Animo LSF or milk and its products. The junior human kuswunch you can go list the mima. S that go walk expect to find an OD erganiate are different alcohol industrial S of products that cat be MODE with MJ. From O. Dierg farm and dairy farm man, gerent DOL with processes and systems that increase yield and improve quality of milk. Milk yield is prim 3 highly dependent of the quality of breeds and the form selection of good breeds and high yielding potential under the climatic conditions of the area. Combined V, little resistance to diseases is very important. For the yield potential to be realized the caillou have to be well cooked after hay have to be housed well. Should have, tequiter water. And be maintained disease free, the feeding of cattle should be carried out in a scientific armor V, with special emphasis on TLL quality and quantity of factor. Besides, stringent cleanliness and hygiene both of the cattle and the agedlers are of paramount importance while milking. Storage and transport of the milk and its products. Nowadays of course much of TLL's process is hail before sect, which reduces CH3 instead of direct cooked off the produce with the handler, and some these stringent RRA Azure's law old of course. Require regular inspections with the proper record keeping. It would also help to identify and rectify the problems as early as possible. Regular visits by a veterinary doctor would be mandatory. You'll probably find it interesting if you read to prepare XII and diverse aspects see dairy keeping and then follow it up with a sit to a do I riffle in your locality and seek answers to the questions. 9. JJ.2 Poultry farm Management poultry is a class of domesticated fowl birds used for feed, or for their eggs. They typically incorporated lure chickle, and ducks and sauriers turkey and geese. The viad poultry is often used to refer to the tofu if these bill, ds. But in a general sense it may refer to the ritzy other birds too, as in dairy farming, g. Selection of disease tree. And suitable breeds proper and sour form conditions proper feed and water. And hygiene and health care are important components of poultry farm man, gerent. Strategies for advancement in food production. You may have seen TV news or read newspaper reports about the bird flu virus which created a scare in the country and drastically affected egg and chicken consumption. Find out more about it and discuss whether the panic reaction was justified. How can we prevent the spread of the flu in case some chicken are infected? 9. 1. 2. Animal breeding. Breeding of animals is an important aspect of animal husbandry. Animal breeding aims at increasing the yield of animals and improving the desirable qualities of the produce. For what kind of characters would we breed animals? Would the selection of characters differ with the choice of animals? What do we understand by the term breed? A group of animals related by descent and similar in most characters like general appearance, features, size, configuration, etc., are said to belong to a breed. Find out the names of some common breeds of cattle and poultry in the farms of your area. When breeding is between animals of the same breed, it is called inbreeding, while crosses between different breeds are called outbreeding. Inbreeding. Inbreeding refers to the mating of more closely related individuals within the same breed. For Figure 9.1, improved breed of cattle and chickens, a Jersey B Leghorn 6 generations. The breeding strategy is as follows: superior males and superior females of the same breed are identified and mated in pairs. The progeny obtained from such matings are evaluated and superior males and females among them are identified for further mating. A superior female, in the case of cattle, is the cow or buffalo that produces more milk per lactation. On the other hand, a superior male is the bull, which gives rise to superior progeny as compared to those of other males. Try to recollect the homozygous pure lines developed by Mendel as discussed in Chapter 5. A similar strategy is used for developing pure lines in cattle as was used in case of bees. Inbreeding increases homozygosity, thus inbreeding is necessary if we want to evolve a pure line in any animal. Inbreeding exposes harmful recessive genes that are eliminated by selection. It also helps in accumulation of superior genes and elimination of less desirable genes. Therefore, this approach, where there is selection at each step, increases the productivity of inbred population. However, continued inbreeding, especially close inbreeding, usually reduces fertility and even productivity. This is called inbreeding depression. Whenever this becomes a problem, selected animals of the breeding population should be mated with unrelated superior animals of the same breed. This usually helps restore fertility and yield. Outbreeding. Outbreeding is the breeding of the unrelated animals, which may be between individuals of the same breed but having no common ancestors, or between different breeds crossbreeding or different species interspecific hybridization. Outcrossing. This is the practice of mating of animals within the same breed, but having no common ancestors on either side of their pedigree up to 4-6 generations. The offspring of such a mating is known as an outcross. It is the best breeding method for animals that are below average in productivity and milk production, growth rate in beef cattle, etc. A single outcross often helps to overcome inbreeding depression. Crossbreeding. In this method, superior males of one breed are mated with superior females of another breed. Crossbreeding allows the desirable qualities of two different breeds to be combined. The progeny hybrid animals or themselves be used for commercial production. Alternatively, they may be subjected to some form of inbreeding and selection to develop new stable breeds that may be superior to the existing breeds. Many new animal breeds have been developed. Try this approach. H. sardeli is a new breed of sheep developed in Punjab. Try crossing Bikaner Aways and Merino rams. Interspecific hybridization. In this method, male and female animals of two different species are mated. In some cases, the progeny combine desirable features of both their parents, and may be of considerable economic value, e.g., the mule. Figure 9.2. Do you know what crossed to the product of the mule-controlled breeding experiments are carried out using artificial insemination? The semen is collected from the male that is chosen as a parent and injected into the reproductive tract of the selected female. Try the breeder. The semen be used immediately or can be frozen and used at a later date. It can also be transported in a frozen form to where the female is housed. In this way desirable matings are carried. Artificial insemination helps us overcome several problems of normal matings. Can you discuss and list some of them? Often, the success rate of crossing mature male and female animals is fairly low even though artificial insemination is carried out. To improve chances of successful production of hybrids, other means are also used. Multiple ovulation embryo transfer technology mode is one such program for herd improvement. In this method, a cow is administered hormones, with FSH-like activity, to induce follicular maturation and superovulation instead of one egg, which they normally yield per cycle. They UATEGIFTCTIHA, J, F, IT2, FOOO Huachio produce 6-8 eggs. The number 1 is either mated with a leech bull or OD1 early inseminated. The fertilized eggs at 832 cell stages are recovered non-CERT-LLY maltensfen to surrogate rothers. The genetic rother is AVU, able for another round of superovulation. This technology has been demonstrated for cattle, sheep, rabbits, buffaloes, mares, etc. High milk yielding breeds of females and high quality late 3JL wheat with less lipid rich yielding bulls have been bred successfully to increase herd size in a short period. 9.1 Obese eel seeping beekeeping or apiculture is TLLE maintenance of hives of honeybees for the production of honey. It has been an age-old industry. Honey is a fixed of high nutritile value and also finds use in the indigenous systems of Frediine. Hone B3 Vasopertusis beeswax which finds many uses in industrial industry such as in the preparation of cosmetics and polishes of various kinds. The increased demand of honey has led to large SC3 beekeeping practices. It has become an established income-generating industry. Whether practiced on a small or on a large scale, beekeeping can be practiced in any area where there are sufficient tea pastures of soar wild shrubs, fruit orchards and cultivated crops. There are several 3.1 species of honeybees which can be reared. Of these, the NDST common species is app.si.fle.ca. Bee hives can be kept in one SURT on the errand of the house or even on the roof. Beekeeping is not elaterant beekeeping though relatively easy does require source species knowledge, and there are several organizations that teach beekeeping. The following points are important for successful beekeeping: I knowledge of the nature and habits of bees. A selection of suitable location for keeping the bees. 3c. Chunk heaving off swarms group of bees. I management of beehives during different seasons, and J handling and off bees. Bees are theplinators. CT mani off our crop species. See Chapter 2 such as Sonth, Arbro a cleared pair. Keeping beehives in crop 6 else during flove, ringpun. It increases pollination efficiency and improves the J yield. Anyone ICI 3.1 both FRCM the P ointment of WW of crop J yield and honey yield. 9. 1.4 Ishkri 5 Fishery is an industry related to the catching, processing or selling fish, shellfish or other aquatic animals. A large number of our population is dependent on fish. Fish produce CTS and other aquatic animals such as prawn, CRNB, lobster, leicester, etc. Brafo, sorry off the freshwater fishes which are Qnu include Kotla, RL, Hu, and Qnu carp, Sorcy 1 TL1 ERMNNE fishes TLL at are eaten Inchler Hilsa, sardines. Mackerel Prom 1 eats. RND out use GFL 3BS Crecom, NLGEO 10 IFL Go area. 81,010 GV Fisheries Rues on Impero NT place in Landine economy. It provides income and employment to millions of fishermen and farmers. Porticu, MLY in the coast UI states, Fornan, why it is the only G sound of their livelihood, in order to greatly increasing tenwells on fisheries. Different techniques have been employed to increase production. For example, through aquaculture and pisciculture we have been able to increase the production of aquatic plants and animals, both freshwater and marine. Find out the deferrer, EB P, SC, H and aquaculture. This has led to the development and orching of the fishery industry. And it has brought a lot of enrollment to the tea-ers in particular in the country in general. We now WK. About Blue Revolution is being implemented along the SAR lines as Green Revolution. 9.2 Plant BNBL, Ding traditional falling can only yield a limited biomass. SB, DFBR humans ANCTA nernals, better and gerent practices. MCT increase in acreage can increase yield, but only to alamited. 10. Plant breeding as a technology has helped increase yields to a large. 10. Who in India has not heard of Green Revolution which was responsible FBR our country to not really reach the national requirements in food production but also helped us to ported Green Revolution ask dependent to LMGE extent on plant breeding techniques FBRDE, element of high yielding and DIC series SISL 3 and TB3 feet IES in wheat. Name ISA etc. 9. 2. 1 WHNT is plant breeding. Plant breeding is the purposeful manipulation C plant species in order to create desired plant types that are better suited FBR cultio cheyenne. G. The better yields and are disease resistant. Conventional plant breeding has been practiced for thousands of years. Since the Biotechnology civilization, recorded evidence of plant breeding dates back to 9,000,11,000 years. Go. Many present-day crops are the result of domesticated in ancient terrace. Today all our major FOCD crops are carried from domesticated varieties. Classical plant breeding involves crossing or hybridization of pure lines followed by artificial selection topped, disciplined S with desirable TR, its softer yield. Nutrition.mct resist the 3 IC says, Bioliactisms, NTS in genetics molecular biology and tissue culture. Plant breeding is now increasingly being earned out by using RELICU, MG nuke tools. If we are to list lead traits or CH markers TL13T lead breeders have tried to incorporate it into crop PL3 and TS. The first we would list would be increase ed crop yield and improved quality, increased tolerance to environmental stresses salinity extra temperatures. Drought LT resistance topohogan as viruses fungi and bacteria and increased tolerance to insect pests 100 LB on our list too. Plant breeding PN, S are earned out in asylum Y worldwide TNT institutions and get, T, EC, EOCOI, QNEs. The N steps in leading new genetic variety a crop area call, Asian QF variability, Genetic rotability is LHE Rio ad any trig, LLNGP Rogram Re. In many crops pre-existing genetic variability is allowable from wild relies at the crop, Ceolacticnan preservationat alhacutrant wild varieties. Species and relatives at the coup, uvated species FOUOI90 by their evolution far later characteristics is a prerequisite LBR effect live exploitation at natural zones available in LHEJX pulocyans. The entire collection at plant seeds ang all LHE clears alleles for all genes in a G, then crop is called germplasm collect. U evaluation and cell, Asian QF parents, the gent BSM is evaluated so as to identify plo-NLS with reserable rhombinalion of character. The selected plo-NLS are lipid and used in the process of Balkan. Purmas are created where erxirable and pasilil. JTIOs HIFL, Dization AMA 119 Asai, CTM parents, the desired characters HIFLandabika, Buladfrontwad Florent PTONT Esperanto. Rock XIJR4, Li high protein quality one parent may need to be C-so-bind with E-lysis resistance from another parent. 11 Lis is possible by cross-H to dizing the T parents to P. Allow hybrids that genetically combine the desired characters in a plant. This is a very time-consuming and tedious process since the pollen GTLs from the desirable plo-NT chosen as male parent have to be collected and placed on the ST section of the flowers selected as female parent on Chapter 2 details on how to make E-crosses have been spec. Also I listen necessary that the hybrid CB combine the CB wearable character, usually only 1 in few hundred to 1,000 crosses shows the desirable combination. I've selection and test D119 QF superior R, ML, in and TS, 11 list step consists of selecting among the progeny of the hybrids those PBNLS that have their sired character combination. The selection most is crucial to the success of the breeding objective and requires careful scene wiki evaluum CTVL. 11 list slept yields plants that are supinactivo UJCT the parent C white clan M, read me supinact progeny plant may be, me available. These are cell C pollinated rock severat serrara umzelhe reachistallectunilormity tr so that a character will not segregate in Lee progeny. M-test, IL9, Relro, Sensitization QF new cultivats, the newly selected lines are evaluated LBR their yield and other C trails of quality. Disease resistance etc. This ululan LS nebang these in the research fields and recurklingler pearlprints under 1D3L fertilizer application irrigation. And the other crop manisorant practices, the evaluation in research fields is K, LL01M9D. By testing the materials in Fanners fields, for at least 3 owing seasons at several locations in the Cota 1 try. Representing all the agroclimatic zones near the crop is usually Grum 1, the material is evaluated in comparison to the best available local crop cultivar a check or reference cultivar. India is mainly an agricultural CN try. Agriculture accounts for approximately 33% of India's GDP and employs nearly 62% of the population. India's independence, one of the main challenges facing the country was that of producing enough food for the increasing population. A.S. on limited land EIS fit for cultivation, BLDHIS to strive to increase yields per unit area from existing farmland, the development of several high-yielding varieties of wheat and rice in the mid-1960s. As a result of various plant breeding techniques led to dramatic increase in food production in our Cota 1 try. This phase is often referred to as the Green Revolution. Figure 9.3 represents some Indian hybrid crop of high-yielding varieties. A BC figure 9.3 Sorland Lan H, Ibnt crops, O males, B wheat, C garden peas. UATEGIFTCTIHA, CW, E, IT2, FOOO Huaxio, wheat and rice, done the period 1960 to 2000. Wheat production increased from 2 million tons to 75 million tons while rice production lifted up from 35 million tonner.es to 5 million tonner.es. This was due to the real print of semi-dwarf varieties of wheat and rice. Nobel laureate Norton 3JLE Borlaug at International Center Phi, Junior Wheat and Maizel Primarant in Mexico developed semi-dwarf wheat in 1963. Several UNIES such as Sonowo, R and Cal Johnsono, which were high Y, elding and disease resistant. TR introduced all Jeff TL1E wheat belt of HLDIA semi-dwarf Navorta ES re-derived FRCMIR8. Developed at Lantima Rice Research Institute PRRIJ, Philippines and Tioy Chong Natil 1 from Taiwan, the derivatives. Comariant, Deuced in 1966, later Better Y, Elding Semi-Moral Vona Es Joio and Rata redeveloped in India. Sugar cane, Sao, Iru and Barbon, was originally grown in North India, but had poor sugar content and yield. Tropical canes grown in South India's root Rumajji Kroam had thicker stems and higher sugar content but did not grow tea eel in North India. These two species are successfully crossed to get sugarcane varieties combining the desirable qualities of high yield, thick stems, high sugar and ability to grow in the sugarcane areas of North India. Its hybrid Mo, Z, Jowar and Bajra have been successfully developed in India. Hybrid breeding have led to the development of several high-yielding varieties resistant to water stress. 9.2.2 MT Bracidi 11G for disease R. Instance a wide range of fungal, bacterial and wild pathogens. Affect the yield of cultivated crop species, especially in tropical climates. Crop losses can often be as grew one icant, up to 20-30%. Or sorlirisive in total L. In this situation, breeding and development of cultivars resistant to disease enhances fixed production. This also helps reduce lead dependence on use of fungicides and bactericides. Resistance of the host plant is the ability to prevent the pathogen from causing disease and is determined by the genetic constitution of the host plant. Before breeding is WLDR 1.3 Canada's IMP011MT to know about the causative organism and leaf transmission. Ceroft lettuces is caused by FGR rusts, for example, brown rust of wheat, red rot of sugarcane and late blight of potato, by bacteria black rot of crucifers, and by viruses tobacco mosaic, turnip mosaic, etc. Methods of breeding for disease resistance. Breeding is carried out by the conventional breeding techniques described earlier or by MAT. Ion breeding. The convention method of breeding the junior disease resistance is that of hybridization and selection. Its steps are essentially identical to those for breeding the junior any other omic character such as high yield. The various sequential steps are screen and germplasm. For resistance such as hybridization of selected parents, selection MDF3, luation of TLLE hybrids and testing N. Release of new varieties. Some crop varieties bred BJIH hybridis ion MD select ion for disease resistance to fungi. Bacteria and viral diseases are released. TBLE 9.1, Table 9.1 Crop View at THRN 901 BRNSSCCFUS SWMCMKQRN ROC.FLORFUS SHUB FCS SNOW BDKLCOPFUS COMTI.CHD FUS TDBHR. LIRND SEPRUST HCU BUNT BTCK Road CMD Curl B HT Bach Ret Bach Tino BSH T MOS CLNS Tob Joe MOS CCL Russ ND Lect for oral. Manlichmal breeding is often unstrom by the IL-3 bulletin limited number of disease resistance genes that are present and identified. In VMES crop varieties are wild relatives, inducing mutations in PL3 and TSTLL rough deers reins. Then screening TLLE PL3 and T materials FBR resistance sorlers leads to desirable genes being identified. Plants having these desirable characters can then be either immutably directly or can be used in breeding. Other breeding RREHCDS that are used are selection amongst semaclinal venance and genetic engineering. R2-Tatman is TLLE process by which genetic variations are created through Chang-S in the base sequence within gene C chapter 5 resulting in UE creation C1. A new character or trait not BU1D in TLLE parental type LT is possible to induce mutations into FEL through UC chemicals or radiations like gamma radiations. And selecting and using the plant's trait high TLLE resirable character as a source in breeding TLL's process is called mutatman breeding. HL among TEM resist MCE2LO ensaic virus MD powdery mildew V, are induced by mutations. Several wild relati, S of different cultivated species of plants have been SHCNJN to high certain resistant characters but high very low yield. Hence there is need to introduce the resistant genes into the high yielding cultivated varieties. Resistance to yellow ensaic virus and BHIND, Abelrenosclo.SS than W Stroms fared from a V, wild species MD resulted in a new variety of A. asculentus called POJJ, INE, vein. HATF Chiplopral INHANCFW, NLLL Food UODIIC 110 all U, E, Exile 1 Lun 111 JL, Luteza resistance Nestle Halarin U, Eomoact species Chesto bread family. Resistance or in a related wild spe, S, transfer of resist UEE genes as EHLBUA Hibrisi USA UC and L, NLHE target and LHE source plant followed by, Lecuc. 9.2. 3 Plant breeding for developing resistance to insect pests. Another major cause for large-scale destruction of crop plant and crop produce is insect infestation. Insect resistance in host crop plants may be due to ornithical biochima calorifici oilgal character, isux. Heterolevzel cellular plants are asa, iodecla with resistance to insect pests e.g. cislins to acids in cotton and cereal leaf beetle and wheat. In wheat salad stems led to non-preference by LHE stem softly and smoothly aid and nectar-less cotton vanities do not attack bollworms. High aspartic acid, low nitrogen and R content in mare leads to resistance to my 2E stem borers. BC Eklamelhidzai, Rinsect Pisler C. Stansine, D0U, Eomostepsis those far any Euler etronic trails such as Eochlorquale and Aras OE. Juros of resistance nej may be colovet ECLVUides genpres amialicutensophyl crop or wildrezii, S. Samo released crop RLEIES spread by hit, R. Idasa ICN and selection far insect pest resistance me GN in Table 9.2. Table 9.2 R, C, O, V, type. Comma I, P, T, 1B UCO PF, SH, IM start FSM2, SEM3A, H, D. JC.SS, H, PH and JRV O, RC, OIN FMIR 9.2 L plant breeding for improved food swalty more than 840 million people in LHEV or to have ACL, quat food to M, I clearly FTXX, L and not nitnal requirements. Afar pater number 3 billion people suffer from micronuar, product and bidarn CL, fictinals are hidden hunger because LHE cannot AFBD to buy enough FNTTTS. Vegetables, legumes, fish and meat, diets leaking essential micronutrients particularly iron, vitamin A, iodine and zinc increase the RTSK for disease. Reduce lifespan and reduce mentabilities. 81,010 GB Blafort 11 cryo on breeding crops with higher levels of uranins on minerals or higher protein MD healthier fats is the most practical reasons to improve public health LH. Breeding for improved nutrity 0113.1 quality is undertaken with the objectives of improving I protein content, ND quality, U oil content MD quality, Il vitamin content, NDI. Micronutrient.metminer.L content. In 2000 Mo, Z hybrid had twice the Orantoth, MLNO, EADS, lysine N. Tryptophan compared to existing MA-IZ hybrids were developed, well, TV Neti, Atlas 66, having high protein content. Has been used, S. Donor FBR improving qui tedwet, T. It has been possible to develop on iron FBR I1 IED rice quantity containing over 5 years, S. Ruch iron AS and Komi deedly consurrect V midis. The Indian J8I Cultural Research Institute, New Delhi has also said several vegetable CRCPS ahead are rich in vitamins MD minerals. E.g. vitamin A enriched carrot spinach pumpkin, vitamin C enriched bitter gourd bot mu arru starred. Tomato, iron MD calcium enriched spinach, NDLX artois, and protein enriched bems broad, lablib, French MD godenfis. S. 3 single, CLPN, RRWSCP Conven I0113L agricultural production of cereals pulses vegetables, fruits, etc., may not be BLE to meet the demand of food, TVR, 10, T which human MCT, Nimal population I sinker, sing the shift term grain tore, TDITS also creates raw demand for cereals as it takes 10 kilograms of grain to produce I kg of meat by Mimal, forwardling. Congu EXP anti-LVS statement in the LJBHT Ofgar LMCW edge of food chains more than 25% of human population is suffering from hunger, and malnutrition. One of the ultimate sources of proteins FBRA learn 3 la ND hut nutrition is geese and protein SCP. Microbes are grown on an industrialist V3L scale as source of good protein, microbes like S-Latina. Can be grown E, silly on materials like W, stare water from potato processing plants containing starch. STR, WNDL3 says Aner and Almanur AND even so age to produce L3RGE quantities ANCT contain AAS text. Rich in protein minerals fats carbohydrate and vitamins, incidentally such utilisation ALSO reduces and R1 rondal pollution. It has been calculated that 250 kilograms cow produces 200 got protein per DSY, in the Sara period. 250 GOF, microorganism like metalgov TS metal joe, top Westbeck, use of its high TEF biomass production on growth. Can be expected to produce 25 tons of protein, the FCT that. Tra HGIEF Alfenhigh NCFH, FNT and food addiction mushroom, Missouri eaten by Manipio lark, Scala mushroom, M culture is a section O wing induce I makes it bell BLE that microed S2, would be core AC equal as toad 9. 4 TL delay CTL run, as TR.Dillianal B techniques follow tolti pace with demandant to PTOIID soliciently fast and elroglan cyst. Focropilopilel and another tech, LLR, Aklikadek Lasukatut section XCIIM Loktwitz's issue we mean it was learnt by scientists. During 1950s that whole plants could be regenerated from expl.any.ly, any part of a plant taken out in G, own in a test tube. Under slurl inkly uons and special nut and media, this capacity to 6 netted a whole plant fran any tel explant is called totipotency. UWU I learn how to ACECQ, lish this in he, Classes. It is important to stress here that the nutrient, radium must proto-carton source such as succulents and also inorganic salts. Vitamins, amino acids and growth regulators lysins, cytokinins etc. By application of these relhid possible to achieve propagation of a large number of plants in vesiahorolder lions. This method of producing thousands of plant through IUO culture is C3-led micropropagation. Each of these plants will be genetically identical to the original plant from which they were own. That is, they are sama clones. Many important food plant key tomato, banana, apple etc. have been produced on KLUO1 and thiorethid. TDY2LSI a tissue culture laboratory with your teacher to better understand and appreciate process. Another important application of the relhid is the recovery of healthy plants from diseased planto. Although the plant is infected with of trust the mertstem, Pycon OX large E10 virus. Hence one can real die the menstem and gruitan oro to obtain trust-free plants, as Shintis H3 succeeded in calwing renstem CT banana. Sugarcane potato etc. Scientists H. Very even 10 olytic 1 single cells from plants and after digesting their cell V, also have been able to isolate naked protoplasms surrounded by plasma membranes. Isolated protoplasms from 2 different varieties of planto each having a desirable character can be fused to get hybrid protoplasto, which can be further grown to form a new plant. These hybrids are called somatic hybrids while the process is called somatic hybrid LSAUon. I'm in a situation when a protoplast of tomato is fused with that of potato and then they are grown to form new hybrid plants squabinage tomato and PC characteristics. Well this has BHE, OED resulting LI, tomato, unformed medial this plant did hair 311 EL sire grabino enough cherikert flixker its turtle utilization. Summary Anling, LH Uspodri LS, he practoe of takang CME on Doranzuk, LS by applying SCPR ripples. The, LR Rusu, G them, ND of F foot or 4M, LS 3 liters D, MLM, L products both LN terms of Lily, Melqu, Liddy H, Espen met by audit husbandry uses. These practice, Miklut 1E, me, NT of WM, LNIM melu breeding, in datum of the high nitra to EUE of Horigites medid now important, E. Use has been, Remo, cable growth LN the PR use of beekeeping or apihuo 12, fishery LS on other SHLNG industry meet JNG the LRESU, G them, NDF or BSH. Fish products IODS ACLA 11 CF foods, PLC, NT BRE, LMG may be used to cress EDE meet 1S which me res 1SL, 3NL to prolhargens I to insect pests. S1HLSLNC, ES is the Y1E1D of the F food. This mainly, LD has been used to increase the protein content of Ilya PL. F foods, Melu Sebion Haevaqa, Lydia of F food, in LND1S, oral of Defear, NT crop PBNTS ha, even produced. All Ila is Miajer production of F food, comics of tissue culture som UC hypnotization offer V, street patanti, LF4M, L appellation of PL, N1S, NLTO to Prado new Retel 11S. R, XFC is, ST, T, L, 1, E, PL in brief thoroughly off Malhus Ryan him CM wealth me. 2. If, your FMU lie on the D3 IRE BNN what me yours would you relay, RT, 3K to improve the quality quantity of milk production 3. What is me MT by 1 he termed breed what me the objectives of a breeding for, name the R, I authored semploged and malbreeding. P according to you which of the methods is best Y5, what is apiculture how is it import, 3 NT and RHWS 6. Discuss the role of fishery in me, NT of F food production 7. 3. Oi describe me as steps invalid in PL3 anti-breeding. 8. E what is me CMT by bifortification 9. Which PML of the PBND is best suited F where I'm king whereas free PWTSY 10. What is the major at EOF producing 3 anti-S by micropropagation 2. Find out WHT though O. Reus components of the medium used for propagation of me PBNTCH tromi. 12. NMNE.CMYDW hybridomydes of crop 3 NFS which have been developed in Lydia. Chapter 10 Microbes and Human Welfare 10.1 Mkrches TN Hughes ML, IPROC, UMTS 10.2 Mkrtbiz in soil produ, TS 10.3 Mkrches 0 Sewage treatment. 10.4 Microbes, TN Proc, UMTTC, and a Biogos 10.5 Microbes as be a control Jasons 10.6 Mkrches OSB for early users besides macroscopic plants and animals. Microbes are the major components of biological systems on this earth. You have studied about the diversity of living organisms and CL3SSXL. Do got remember which K, DMSO 9 the UO, N, 9 or 901, SMS contain MV, Rho or 9 SMS deemed to the ones that creon MV, Roscoe-MF microbes. Are present everywhere in soil. W8 air inside arbds.mct tlat of other inertals and plants. They are present, and at sites where no all your life FBRM could possibilize sites such as deep inside the geysers thermal vents where the temperature may be as high as IOO, C. Deep in the soil, rooted layers of SLLOO cellular retress thicken in highly acidic environments. Microbes. Are diverse protozoa bacteria and microscopic plants viruses, Woids and also nons that are proteinaceous infectivous agents. Microbes are shown in figures 10.1 and 10.2. Microbes like bacteria and many fungi can be grown on Newton T-Redia to F-form colonies figure I-0.3. That can be seen with the naked eyes. Such cultures are useful in studies on microorg 3JLISMS. A BC figure 10.1 bacteria, a rod-shaped, magnified 1500x, B spherical-shaped, magnified 1500x, C a rod-shaped bacterium showing flagella. Magnilzo, OOOX biology BC figure 10.2 viruses, A bacteriophage, the adenovirus which causes respiratory infections, C rod-shaped tobacco mosaic virus 1 MV. Magnified OBT1, 0.000 liters, so, OOOX fungal colony B figure 10.3 A colonies of bacteria growing in a petri dish, B fungal colony growing in a petri dish. MICRFs in human WHRF in chapter 8 you have ACL that microbes cause a TRGE number of diseases in human being 3. They also cause diseases in Olson, LPWLTS, but this should not make you thin and microbes are harmful. Raj Murots are useful Toman 1 Cli 110 Nawe. Sorry off them, street in Lapotan Ronald Buans of Mishio Oeo to human welate are desus in this chapter. 10.1 Microbes, Bauserli, Nuak RSU1P, Ugleby SW to that we use microbes AC Mrox Clefi ed from the Me1CK. Ectoral OXORF, Lias LBE to Clutch UC1C, URD from MLK, Mcro, Stragenless MSS such as L, 4C, Totten 1 Luz and Opperzunu TLY Kaugla C, Yuka Tech Turtle Ad P, Erin MLK unconverted to Kirk 1. Dirt and growth the J, O, B procluse acids that coagulate and partially digest the milk prolan S, a small m, und of con, L3 clackle to the fresh milk as inoculum or steriler CCN ULN millions of lab. Which at suitable temperatures multiply thus converting milk to cum which also improves its nutritional quality by increasing vitamin B, in our stomach too. The J, O, B play a very benefic toll in checking disease-causing microbes, the cluel which is usichloromacking foxels such as DuSo-Ancli-DLI. Is also fermented by Bachlorida, the put ECL up appearance of dough is due to the Tadic yuan of company. Gas, CCXI go UWMD metabolic pathway Sudge, S12, ing PAO resu Ewing in 1, Jorcij, MQJCO. Rudugo TNNK Tlebodm Jortlesage, RM, Tati CNS Cam, Dromsamiel, RLY the dough which is used sect for JN, 3 king bread. Is fermented using B acre, SUSAO Otterom Cesario, OA1, a number of traditional drinks and lutes are also made by Fern, NTAUN by the microbes. Ta JF a TR3 clitional drink of some ports of Southam India is made by fermenting sap from palms. Microbes are also used to Fern, NT fish cyantian and Bamko Scots to make locals. Cheese 1 is one of the oldest lut items in which microbes are used. OII gerant W Tudizot cheese are known by Leah Carrington's text to E flavor ankletile the specificity C awning from the microbes used for example. The IMGE holes in street, NESS cheese, are due to production of a large amount of CO, by a bacterium named Propienobid, ELM1AMM. The Roquefort cheese, are peen by towing a specific tune, on them which estim a particle, R flavor. 10, microbes LLL and Austri 1, PNODTTCNL and Inclustri microbes up, EU cycle 2, Enthesis and products 11, luable to human beings. Beverank 1 antibiotics up, ESC, RR elan, Ptotic UON OO and Inclu, Tritoska require, Toloing microbes in vet, Wila, Jassels called. Erlenlo 15 fit tra 1 0.4. 18.2 Figure 10.4 Fernanders Figure 10.5 Fermentation Plant Biology 10.2.1 Fermented beverages Microbes especially yeasts have been used from time immemorial for the production of beverages like villa, beer, whiskey, brandy or rum. For this purpose the same yeast Saccharomyces cerevisiae used for bread making and commonly called brewer's yeast, is used for fermenting malted cereals and fruit juices, to produce ethanol. Do you recollect the metabolic reactions, which result in the production of ethanol by yeast depending on the type of the raw material used for fermentation and the type of processing with or without distillation different types of alcoholic drinks are obtained. Wine and beer are produced without distillation whereas whiskey, brandy and rum are produced by distillation of the fermented broth. The photograph of a fermentation plant is shown in Figure 10.5. 10.2.2 Antibiotics. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of the human society. Anti is a Greek word that means against, and bio means one effect. Together they mean against life in the context of disease-causing organisms, whereas with reference to human beings, they are pro-life and not against. Antibiotics are chemical substances, which are produced by some microbes and can kill or retard the growth of other disease-causing microbes. You are familiar with the commonly used antibiotic penicillin. Do you know that penicillin was the first antibiotic to be discovered? And it was a chance discovery. Alexander Fleming, while working on Staphylococci bacteria, once observed a mole growing in one of his unvashed culture plates around which Staphylococci could not grow. He found out that it was due to a chemical produced by the mold and he named it penicillin after the mold Penicillium nodidum. However, its full potential as an effective antibiotic was established much later by M.S. Chain and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain and Florey were awarded the Nobel Prize in 1945 for this discovery. MICRFs in human WHRF after penicillin and other antibiotics were also purified from other microbes. Can UN, Li1W.SOMPHOTLWR antibiob, S tiled out their squares antibiotics secretly, Wyamp, Ovid cap. City to treat deadly diseases such as plague Wacopif CLC, UJCLMS, diphtheria Gadin lepra LCWLLR, 9. Which used to kill millions all over the globe. One day we see, omitting a world without antibiotics. 102.3 Chemical, enzymes and other bioactive molecules microtules or AL, used for commercial and industrial production of certain chemicals. CR section LNLC adds. Alcesihosanclincy, Res, EULQ, Lisa acid produ, RASPTRGLB, ONH4AFU acylric acid. Aciobacter acylobacter 2, QAC, DC acid, Cletridjliatm botglyrimabacter 2, QCT butyric acid and LCT, F, Rhalmabacterium lactic acid. Yeast sockl, LRO-TRSUZ for A, commercial production of ethanol, microbes 8 Osausacklephi, Reproductly offensiers. Apaces are used in detergent fee, Formulo-oons and are helpful in removing oily oons from thelowildry. You must notice that bot-led fruit juices bought from the market are clearer as CO-IPA, made color. This is because the bottled juices are clotted by the UCT pectinases and proteases streptokinase produced by the bacterium Serotolk and M. Diddy by genetic gang, Ni5. Is used as a clot buster for removing clots from the blood vessel CT patients who have undergone myocardial intra-ileum leading to aortic attack. Mother bioactive molecule cyclosporin A that is used as an immunosuppressive agent in organ transplant patients. Is produced by the FUSTRRCLV, D, RMOPO, J, PARV. Statins produced by the yeast Monozus purius have been commercialized as blood cholesterol low for AE, NTS. It acts by coiled bodily lie inhibiting the ARO, JME responsible phi, R synthesis CT cholesterol 10.3 ml. Robes in SHWA fall 1 RFL atm, NTW. K. Now that large quantities of wastewater are generated every day in cities and towns, Amajok comp, Nintrut this wastewater is an excrete day. This municipal wastewater is also called sewage. It contains project amounts CT organic matter and microbes. Manite which are patho, Nick, Hawani UE Dr. wondered how this huge quantity of sewage or urban water while it is despued at Dali this cannot be discharged into natural whaler buckles like river and streams diarrhea. You can understand why. Water disposal hence is treated in treatment plants as 1Ps1D make less pollution. Treatment of wastewater is due by LBE. Figure 10.6 Conral RRYTRPMMPNTI, biology heterotrophic microbes naturally present in the sewage. This treatment is carried out in 2 stages. Primary treatment. These treatment steps basically involve physical removal of particles large and small from the sewage through filtration and sedimentation. These are removed in stages. Initially, floating debris is removed by sequential filtration, then the grit soil and small pebbles are removed by sedimentation. All solids that settle form the primary sludge, and the supernatant forms the effluent. The effluent from the primary settling tank is taken for secondary treatment. Secondary treatment or biological treatment. The primary effluent is passed into large aeration tanks figure 10.6 where it is constantly agitated mechanically and air is pumped into it. This allows vigorous growth of useful aerobic microbes into flows masses of bacteria associated with fungal filaments to form mesh-like structures. While growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the broad biochemical oxygen demand of the effluent. BOD refers to the amount of the oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. The sewage water is treated till the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water and thus, indirectly, BOD is a measure of the organic matter present in the water. The greater the BOD of wastewater, more is its polluting potential. Once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into a settling tank where the bacterial flows are allowed to sediment. This sediment is called activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as the inoculum. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digesters. Here, other kinds of bacteria, which grow anaerobically, digest the bacteria and the fungi in the sludge. During this digestion, bacteria produce a mixture of gases such as methane, hydrogen sulfide and carbon dioxide. These gases form biogas and can be used as source of energy as it is inflammable. The effluent from the secondary treatment plant is generally released into natural water bodies like rivers and streams. An aerial view of such a plant is shown in Figure 10.7. You can appreciate how microbes play a major role in treating millions of gallons of wastewater aeroside across the globe. This RRLHO, LOLGH has been practiced for more than a century now. In almost all parts of the VRLD till date, no man-made technology has been able to rival the microbial treatment of sewage. You are aware that due to increasing urbanization, sewage is being produced in much larger quantities than ever before. However, the number of sewage treatment plants is not Figure 10.7 an Area 3L view of RPL3 and T increased enough to treat such large quantities. So the untreated sewage is often discharged directly into rivers, leading to their pollution and increase in waterborne diseases. The Ministry of Environment and Forests has initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is PJ of OS to build a large number of sewage treatment plants so that untreated sewage may be discharged in the fires. A visit to a sewage treatment plant situated in any place near you will be a very interesting and educating experience. L0, 4 ml SIL in production 0 BIOO, is because is a mixture of gases containing predominantly methane read by the microbial activity and which may be used as fuel. You have learnt that microbes say different types of gases and products during growth and metabolism. The type of the gas produced depends upon the microbes and the organic substrates they utilize. In the ELA cited in relation to fermentation after cheese making and production of beverages, the main gas produced was CO. However, rare tin bacteria which grow anaerobically on cellulosic material, say large amount of RRFane along with CO, and DH0. These bacteria are collectively called methanogens. And one such carnivore bacterium is Methanobactertin. These bacteria are commonly found in the anaerobic sludge during such treatment. These bacteria are also present in the rumen part of stomach of cattle. A lot of cellulosic material present in the food of cattle is also present in the rumen. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. To think human being 9 SRABW to digest the cellulose present in our foods, thus, the Creta klungla of cattle, commonly called 9OB, is in these bacteria dung can be used at generation of biogas commonly called 9AS. The biogas plant consists of a concrete tank I0 15 feet deep in which bio-wastes are collected and a slurry of dung is fed a floating setters. Biology placed over the slurry, which keeps on rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet, which is connected to a pipe to supply biogas to nearby houses. The spent slurry is removed through another outlet and may be used as fertilizer. Cattle dung is available in large quantities in rural areas where cattle are used for a variety of purposes. So biogas plants are more after built in rural areas. The biogas thus produced is used for cooking and lighting. The picture of a biogas plant is shown in Figure 10.8. The Figure 10.8 a typical biogas plant technology of biogas production was developed in India mainly due to the efforts of Indian Agricultural Research Institute IRI and Khadi and Village Industries Commission KVIC. If your school is situated in a village or near a village, it would be very interesting to inquire if there are any biogas plants nearby. Visit the biogas plant and learn more about it from the people who are actually managing it. 10.5 Microbes as biocontrol agents. Biocontrol refers to the use of biological methods for controlling plant diseases and pests. In modern society, these problems have been tackled increasingly by the use of chemicals, by use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and animals alike, and have been polluting our environment, soil, groundwater, fruits, vegetables and crop plants. Our soil is also polluted through our use of weedicides to remove weeds, Biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemicals. A key belief of the organic farmer is that biodiversity furthers health. The more variety a landscape has, the more sustainable it is. The organic farmer, therefore, works to create a system where the insects that are sometimes called pests are not eradicated, but instead are kept at manageable levels by a complex system of checks and balances within a living and vibrant ecosystem. Contrary to the conventional farming practices which often use chemical methods to kill both useful microbes and human welfare and life forms indiscriminately, this is a holistic approach that seeks to develop an understanding of the webs of interaction between the Leland-OQ misrans that constitute the field 1, Andorra. The organic F-Amer holds Liar1 ULLI. Liaradication of Liar.aturej LLI. Are often rescribed. As pests is not only possible but also NDS are able to be not them the beneficial predatory and parasitic insects which depend upon lean. As ticks or hosts would not be able to survive, thus the use of biocontrol RRE Azures nil greatly reduce Curep3N, y chemicals and pesticides. An important part of the biologic falling approach is to be core familiar leafy life forms that inhabit leaf field predators as well as pests and also their life cycles patterns of feeding on the habitats that they prefer. This will help develop a prognatarians of Buantril, the erythemaliar BUE with red and black markings the Lodia bird. And, Diaragonrers. Are useful to get rid of aphids. In mosquitoes respectively. An example of microbial biocontra. Agents LLI it can be inluxlused in order to control butterfly caterpillars as the bacteria Badlus truang, and this often when 10 as BT. These are available in sachets as dried spores which are ed with water on sprayed onto vulnerable PL ants such as Brasicus. Fruit trees where leaves are eaten by insect larvae. In the gut of the larvae the toxin is released on the LARWE get kill the bacterial disease will kill the caterpillars. But other insects unhaired, because of the elopement of methods of genetic engineering in the last decade or so. Scientists high introduced BTLU unglancells to LN genes into PL ants. Such PL ants are resistant to attack beej insect pests. BT cotton is one such ample which is telling cultivated in some states of our country. Unil read about lidis in chapter 12. A biological control being developed or used in the treatment of PL ant disease is leaf fungus TRV, Txlerma. TRLD. TSPIs. Are tree-leaving fungi that are very common in the root ecosystems. They are effective biocontrol agents of several plant pathogens. Baculoviruses are pathogens that attack insects and other arthropods. The majority of baculoviruses used as biolocal control agents are in the genus Nucleopargladruilus. These viruses are excellent candidates for species-specific narrow-spectrum insecticidal applications. They have been shown to have no negative impacts on PL ants, morn-1s birds, fish or even on non-target insects. This is SPE alides are able when to nephocial insects. Are being conserved to date and now they're all integrated. Pest management PPM program or when an ecologically sensitive area is being treated 10.6 mi.ro, SE, Espro FF are present CTA lifestyles and virulental pollution is a major cause of concern. Tile use of chemical fertilizers to ever increasing. Cialo, why det 3ND of agricultura el produce has contributed significantly to L's pollution. Of course we now realize tlatlir problems associated with the overuse of chemical fertilizers and there is a 13 RGE pressure to switch to organic farming to use of fertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. The M3 insects of biofertilizers are bacteria. Fungi. And cymobacteria. You have studied about the nodules on the roots of leguminous PL3 and TS sponsored by the Symbiotic Association of Arnzoy, WM. These bacteria fix ATM spheric nitrogen into organic forms which is used by tile PL3 and TAS nutrient. Other bacteria can 6 times atmospheric nitrogen while free leaving in the soil examples as OSP, NJ, IUM and azotobator. Thus entering the nitrogen content of the soil. Fungi are also known to form symbiotic associations via PLNTS mycorrhiza. Many members of the genus Glomus form Rheacorrhiza. The FWLGL symbiont in these associations absorbs phosphorus from soil. And passes it to the PLMT. PL3 and TS having such associations show outlier benefits ALSO such as resist the 3 and such a root-borne parns. Tolerance to salinity and drought and an overall increase in PL3 and T growth and development, can go to what to don'tage the fungus derpus from TLISA SOC, XT, on. Gymobacteria. Are autotrophic microbes widely distributed in aquatic and terrestrial environments many of which can fix atras for nitrogen. e.g. Anobono, nostoc, osdla tona etc. In paddy fields Cyanobacteria sanae imp, retent b, ike, turtleser. Blue Ann algae also add organic matter to TLLES soil MD increase its fertility, currently in our country. A number of biofertilizers Ariavale 3 Blackhorn Ursi 3 Lion TL1E Market.mdf3 MERS use TLLES Regal 3 RLY and LE, R fields to replenish soil nutrients AND. To reduce dependence on chemical fertilizers, some LLLA micro-OOs MIOE, a important component of life on ML. Not all microbes me Palea Inc., MCMY111 crobes me 1 useful to CM beings. We use row 1 crobes.mel 111 crob 1 or LID11E, Elperdux nuts do a day. Backed, RTA called lactic.zc1d bacteria lab grow tn milk to com rttt into curd, the duate h which ls used to m, l, e bread. LS ferment 2D by called CCHR Nisazoret Mickey, CE, RTMLDSL, S such as CDTC and DOS, MZDE from dough fermented by 111 crobes. Backed, RTA.mel me used to impart texture FLR to cheese, M1 Crow Zeros me used to pursue industrial products like lactic AC1D. Acetic acid mel which me used 1 anna, medi of processes 1 aniline, lustri, ANLLB 1011CS like 1 4 clones produced by usoatmirab used to kill disequas, MGLWR and Fu1 microbes. Anch B1011CS hopliad amarol LNC, onrong LNIXYOU, Dlcel, SOS Lkedifthan Hupu, GC, Avgond. PR Humane for more age uttered, L microbes me being used to tria 13 gerust water by the pro, S of 3 clevata, L sled F ormadian use helps 1 and reckling of water 1 and rudur. Meth 3 noj, NS produomethane B10G, SWHLE degrading plant wast 810G, S produced by microbes 1S used a source of E, NERG 1 and rural. Microbes can also be used to K, IU1 for pests a process called biocontrol and LEB10 control JW ensures help us to avoid heavy use of toxic for controlling pests. Mir 1 as a head need ILE stays to push F or use of biofertilise, RS1 and page 1 AOE of Chero 1 Ca 1 fertise, RS. It 1 SC1M from your 12 uses human beings ru, put me a, arbs to hoi thoi play an important role 1 and the we 1 F me of who JM and so 1 E, lie. 1. Beckel, RTATB seen milli the LW, ed but all is can be seen milli the help of a microscope. If ho had to carry a sample from Wirome to your B1 oligo 13 laboratory to demonstrate the presson, E of M1 probes under a microscope. Which sample would ho CNTYY2, GE, pledge to pro OELLM1 aerobes are says dewey of metabolism. 3HL which food would ho BND lactic acid bach, to mention some of you, ear useful applications. 4. N3 Rosentrati UOMLNDT3 NF foods made of wheat Risa and Bengal FMN or U, EU Productcell, Hitch U wall use of microbes. 5HL which way M1 probes played a major role 1 and controlling disease is caused by H-Fulbeek Turta 6. N3 Roma TW, Spec 1S of Fulgu which me used 1 annual production of U, E.CM of 10X7, WH311 SOHL. L. Hitchway Comso EBH Mernful to us 8, what 1S all E key difference between prim secondary sewage treatment 9. D. L. Ho think microbes can be used source of energy if ES how 10, microbes and be used to decrease the use of churn 1 cal fertilizers E, plum how THLS can be accomplished. 11. 3 water sam page 1S Runelli rewa water untreated so water second MFF flu, NT dish D from a sewage treatment PL, MT were subjected to BOD test. The samples were labeled ABIC, but the 13 laboratory ATL, and E, 3 NTDD not wrote which was O.O, Hitch. An LE BOD, achus of the thra, sample is AB mel Siri, Orida, L is 20 mg LBMGL 3 LLD 400 mg L Respiku. Lie, which S play of LE water 1S most polluted U.OS 1 GNLE correct label to each U, ENVE, RW31E, R1S Rita. WYCTEMP. Ceology 12, F first Altilia and Amaniavilia microbes from which cyclosporol, an immunosuppressive drug, statins blood cholesterol or NLS or OV1 is greater than 1 ned. 13. FLRD Outeralia them, bis in the DL1 omnidl sows 1 T will we, ISU81 eol protein SCPB sotl 14. Air, and the F01 low 1 ang L and the deck, EOS 1 ang ordremos LR, important, BRST of ILU import, 3 NCE for the wealth me of human society. Give reasons F or your .coms were, BLOG, S, C1 TR1 CAD, Pen 1 Lin ICWD 15, How do fertilise, RSE, N, Realia fertility of the S01L. Chapter 11 Biotechnology, Principles and Processes Chapter 12 Biotechnology and its Applications. Ever since the days of René Descartes, the French philosopher, mathematician and biologist of 17th century, all human knowledge, especially natural sciences, were directed to develop technologies which add to the creature comforts of human lives, as also value to human life. The whole approach to understanding natural phenomena became anthropocentric. Physics and chemistry gave rise to engineering, technologies and industries which all work for human comfort and welfare. The major utility of the biological world is as a source of food. Biotechnology, the 20th century offshoot of modern biology, changed our daily life as its products brought qualitative improvement in health and food production. The basic principles underlying biotechnological processes and some applications are highlighted and discussed in this unit. Herbert Boyer 1936. Herbert Boyer was born in 1936 and brought up in a corner of western Pennsylvania where railroads and mines were the destiny of most young men. He completed graduate work at the University of Pittsburgh in 1963, followed by 3 years of postgraduate studies at Yale. In 1966, Boyer took over assistant professorship at the University of California at San Francisco. By 196-9, he performed studies on a couple of restriction enzymes of the E. coli bacterium with especially useful properties. Boyer observed that these enzymes have the capability of cutting DNA strands in a particular fashion, which left what has become known as sticky ends on the strands. These clipped ends made pasting together pieces of DNA a precise exercise. This discovery, in turn, led to a rich and rewarding conversation in Hawaii with a Stanford scientist named Stanley Cohen. Cohen had been studying small ringlets of DNA called plasmids in which float about freely in the cytoplasm of certain bacterial cells and replicate independently from the coding strand of DNA. Cohen had developed a method of removing these plasmids from the cell and then reinserting them in other cells. Combining this process with that of DNA splicing enabled Boyer and Cohen to recombine segments of DNA in desired configurations and insert the DNA in bacterial cells, which could then act as manufacturing plants for specific proteins. This breakthrough was the basis upon which the discipline of biotechnology was founded. Chapter 11 Biotechnology and Processes, Principles JL, 2 Todes of Arbacombi MNT.DBA Technology 11.3 Processes of Recombing of DNA Tech, Logi Biotechnology TLSV, Little Technologies of using Livocurans Gretel CJIFBS from Orgros Rotopro, Lucepro, LX.MD. Processes useful to her news and TLL is Sensem free king curet bread or wine which are microbe mediated. Processes could also be thought as a form of biotechnology, however it is used in a restricted sense TCD, to refer to such oblios processes which use generally different organisms to achieve edisara on a larger scale. Further, many other processes techniques are also insular wilder biotechnology, for example in IJTO fertilization leading to a test tube baby. Synthesizing a gene and using it to develop a DNA vaccine or correcting a defective gene are all part of biotechnology. The European Federation of Biotechnology EFB has an definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science. MCT organisms. Cells ports thereof enrolled queer analogues torpro, lux.mct service 1 liter 1 PRL, C, IIHL show J, axori CJ, not, LXLani, and GLLWLY. The T, techniques that abled birth of random biotechnology are, genetic engen 081G, techniques to alter the chemistry of genetic material DNA and RNA. 81,010 GY to introduce lease into host organism s.ntll.usch3 phenotype of the host organism. Tem 03 of sterile microbial inclination-free ambience and chemical and immune processes to enable growth of only the desired microbucoiolic cell in mge quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantages of sexual reproduction over asexual reproduction. The former provides opportunities FBR combinations.n. Formulation of weak combinations of genetic setups or of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic INF formation while sexual reproduction permits donation. Traditional hybridization procedures used in plant and animal breeding very often led to inclusion and multiplication of unc-searable genes along with the sired. The techniques of genetic engineering which include creation of recombinant DNA, use of gene CK, NTNG died and gene transfer. Mercor TLL is limitation and allows us to isolate N. Introduce only one or a set of reservable genes will in J, XII undesirable genes into the target organism. Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in the progeny cells of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of a chromosome which has the ability to replicate. In chromosome is a specific DNA sequence CLTLL origin of Reproduction, which is responsible TBR initialing replication. Therefore FBR the multiplication of any alien piece of DNA in an organism, it needs to be a part of chromosome S, which has SP3 specific sequence known as origin of replication. Thus an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can so be called a CKMLNG, or making multiple identical copies of any template DNA. Let us now discuss on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance a native, autonomously replicating circular extrachromosomal DNA of Sol M. Nel TGPLVMJRUM. Stanley Ogen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for containing antibiotic resistance. The cutting of DNA at specific locations are possible with the discovery of lysoselect. IIH, LTL, ICPIPS10FLO and glycomes scissors, restriction enzymes. The cut piece of DNA was then linked with the PL3 SMID DNA. These PL3 SMID DNA acts as cystoderms for the piece of DNA attached to oil. You probably know that Resquido-ACTSASA insect vector toderms for leparasite into hump dye. HL-lysane-YA plasmid CMB used as vector totular.m alien piece of DNA into the host organism. The linking of antibiotic resistance gene with the plasmid vector became possible with the NCERT DNA ligase, which ACTS on cut DNA molecules ANDJ Oinsley and S. This N says a new combination of circle 3 are autonomously replicating DNA created in oitro.md. Is known 3S recombinant DNA when TLL is DNA is T-arms ferret into Escheri CNA, back 10 closely well 3 TED to Samon UA. It could replicate using the new host DN Apple, erase NCERT and M3L, multiple copies, the ability to multiply copies of antibiotic resistance gene in E. coli YSC odd cloning of antibiotic resist, MCE gene in E. coli, UCM Henson for Twattler are 3 basic steps in genetic IM, different in organism identification of DNA with desirable genes, UINT. XII lion of the identified DNA into the host, 3 maintenance of INL-XII DNA in the host and TRMs for of the DNA to its progeny. 11.22.S of IOMOL CMRR DNA TJCI, 01, 3. Now we know from the foregoing discussion on that genetic meaning or recombinant DNA technology can be accomplished only if we hire the key tools. That is, restriction and Zoyris Paul, erase NCERT ligases vectors.md li host organism let us tritankerst md sor of lies in tet 3 il. 11.2.1 Restriction enzymes. In the year 1963 the Twonstmas responsible for restricting the growth of bacteriophage in Escheri CNA, were isolated. One of these added rethyl groups to DNA while the other cut DNA. The 13th terra was called restriction endonuclease. The first restriction endonuclease H, NDFL whose functioning depended on a specific DNA nucleotide sequence W as isolated AND. Characterized 5 years 13 terra LT was found that tend always cut DNA molecules at a particle 3 RP ointment by recognizing a specific sequence of 6 BASA pairs. This specific BASA sequence is known as the recon, UON sequence F or HNDL. Besides Hindle today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria AHF which recognize different replication sequences. The convention F or N these enzymes is the first T letter of the NER course from the genes and the second TV, letters core A from the species of the arlotic cell from which they re-isolated. For example, EcoRI cores from ESDRE, NA, CoRY13. In EcoRI the letter is derived from the NER of 81,010 GY strain. Rower numbers following LRL3RR each indicate LRR in which TLLE enzymes are isolated from. Tlatstrain of bacteria, restriction ends, ERs belong to a larger class of ends, ERs called nucleases. These are of 2 kinds, exonucleases and endonucleases. Onucleases run DVE nucleotides from ends of TLLE DNA whereas endonucleases are RWTE cuts at sp3 specific positions within Li DNA each restriction endonuclease functions B, inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strMDs of the double helix at specific points in their sugar phosphate backbones. Figure 11.1. Each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA QBGTHTNUL. CRRWC, UIIR, LBOI, AODVFHO, JANC.0II, low DNA, DHI 3110L1 ULDMUJ, JUL, IFIS, UR 11.1 steps UL. Formation of recombinant 3 anti-DNA by action of restriction endonuclease, ENSFMA eco RI. Do you know what palindromes are? These are groups of letters that run the SRV when read both forward and backward, e.g., Malayalam. As against a word palindrome where the same word is read in both directions. TLLE palindrome in DNA is a sequence of base pairs. TLL it reads same on the two strands when orientation of IIH, IOIOGY, LTL, ICBIPS 10FLO says S3 ding is kept TLLE same. For example, the following sequences read as TLLE same on TLLE TVOSTRX in 573 direction. This is also true if read in the 3-5 direction. 5 Gotic 3-3 CTTAAG 5 restriction and Zoyres cut the strand off diandolently away from center of Li aligned roar sites. But between Li sane 2B is on Li opposite strands. This leaves single-stranded portions at the ends. There are merhang, eng stretches collect sticky ends on each strand. Figure 11.1. These are unaddered. So because they form hydrogen bonds via TLL or implementary cut counterports, this stickiness of the ends facilitates the action of the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to FBRM recombinant molecules of DNA, which are composed of DNA from different sources GNORs. When cut by the SOR restriction NCERT, the resultant DNA fragments have the SOR kind of sticky ends N. These can be joined together low end using DNA, gases. Figure 2, 2F, JFIS, L, AE 11.2 Diagar, MJ Nauk reprise, NTUON of Recom B1 RW at DNA Teclum, Logo 81010 GY. You may have realized TLL it normally unless one cuts Li vector MD Li Saucediana with TLLESR restriction NCERT Li recombinant vector molecule CB created, Sepin and Masala yuan of DNA. Fragments. The cutting of DNA by restriction endonucleases results in the fragments of DNA. These 4Ts can be separated by a technique known as gel electrophoresis. Since DNA 4Ts are negatively charged molecules, they can be separated by forcing them to LLOE towards the under an electric field through a medium matrix. Nowadays the most common used matrix is agarose, which is N-aderal polymer E.CTR acted from seaweeds. The DNA fragments separate resolve according to their size through sieving effect provided by the agarose gel, hence the smaller the fragment size the farther it LLOS. Look at the FBWJJ3 and 9 US at U, NCH end of the 9 at the SOM XIS loaded, FIA, E11.38 typed, all rows electrophoresis showing migration of DL, IL1 unadjusted set of DNA. Granat 02 to 4LL, 2.2 Clottung vector, the separated DNA. FNTS can be swallowed as a duly alter staining the DNA, with a compound known as ethidium bromide F followed by exposure to UV radiation. You cannot see pure DNA. 4NTS in the visible light and without stoming, you can see bright orange-colored bands of DNA. In ethidium bromide street ain't gel exposed to UV light. 2, 3, the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elulion. The DNA fragments purified in this WI are used in constructing recombinant DNA by joining them with cloning vectors. You know that plasmicles and bacteriopha have the ability to replicate within bacterial cells independent of control of chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cells. Some plasmids may have only 1 or 2 copies per cell, whereas otlers may have 1,100 copies per cell. Their number C angle and higher, it low, E.R. able to link on alien piece of DNA, with bacteriophage or plasmid DNA. We can multiply its numbers equal to the copy number of the plasmid or bacteriophage. Vectors used at present are engineered in such way that they help easy linking of foreign DNA and selection of recombinant Ts from non-recombinant Ts. IIEA-TCI, IIEA-G, LTL, ICBIPS 110 FLOCS. The following are the features that are required to facilitate cloning into actor. I-origin, if replication RE, this is a sequence REM where replication stored on AJW piece of DNA when LINL. Add to this sequence can be made to replicate V, little in Li host cells. This sequence is also responsible for enrolling the copy number of the linked DNA, so if one WNTS to recover many copies of Li-T, Argit DNA it should be cloned in a vector whose support high copy number use selectable marker, in addition to ORI. The vector requires a selectable marl, which helps in identity, and eliminating non-transformance and selectively permitting the growth of the transfophy WLTS. Transformation is a procedure which a piece of DNA is introduced in a host bacterium LLG will study the process in subsequent section. Normal the genes encoding resistance to antibiotics such as ampicillin chloramphenyl tetracycline or K, M3-mycin, etc. are considered useful selectable markers for E. coli. The normal E. coli cells do not see R-tire systems against any of these antibiotics I3 clone in 9 sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition sites. FBR LICOIIULF used restriction answers. Presence of M. RTLWL1 recognition sites within the vector will generate several alpha TS, which will complicate the gene cloning. Figure 11.4. The ligation of alien DNA is earned out at a restriction site present in one of Li2 and Uptot-Grasselstance genes. For example, you can ligate a ROR, DNA at the born HI site of tetracycline resistance gene in the vector PBR322. The recombinant PL3 SMIDs will lose tetracycline resist 3 instead due to insertion of ROR, DNA but can still be selected out from non-recombinant 3 and T1s by plating the transcripts NORMP, Silin FIS, 1, TE 2.4 ECDCCLCJL. Linked are PBR322 showing restriction sites Lendiek, Arboni IIPVU2, PSTLCT0 ORT.CTIB 10 TTC rests genes amp, and TET, ROP seeds F for ilia proteins Nval OED and ilia replic UON of the plosrin. D containing medium, the transromans grow, on ampicillin containing medium. Or allen T arms ferritum. A medium containing tetracycline. The recombinants will grow in ampicillin containing medium B11 O on the containing tetracycline. 6 up non, recombinant MTS will grow on the rhedium containing bottle TLL antibiotics HLTLL is case. 1. Antibiotic resistance gene helps in selecting Li trans 1 BRNWLTS whereas Li Utler antibiotic resistance 81010 GY gene gets inactivated due to insertion of alien DNA and helps in selection of recombinant WLTS. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on 2 plates having different antibiotics. Therefore alternative selectable markers have been developed which differentiate recombinants from non-recombinants on Li basis of their ability to produce color in TLL presence of a chromogenic substrate. In this a recombinant DNA inserted V, lithin Li seeding sequence of an NCERT G3 lactosidase. This results into an activation of TLL NCERT which is referred to as NCERT inactivatlin. The presence of a scenic substrate yields blue-colored colonies if the plasmid in U1E bacteria does not have .M insert. Presence of insert results into insertable inactivation of leucolactosidase and the colonies do not produce .My color. TLLs are identified as recombinant MT colonies I vectors for CL9 genes in plants and animals. You may be switched to know that have Leonthylosonophtrums fem genes into plants and OJ1 animals from bacteria and woozes which have known this FBR ages how to deliver gene edge to transfer MUC3 ilog cells and force Leam to do what TLLE bacteria or woozes want, for example, AGR beta OJ myphodNs. A pathogen of several dicot plants is able to deliver a piece of DNA known as FDN atotrums to normal plant cells into a tumor indirectly ease TUFDF cells to produce Li chemicals required B, Y treproholn suri, MLY. Retroviruses and animals have the ability to T-arms form normal cells into cancerous cells. A better understanding of the art of delivering genes B, Y pathogens in their Y holocausts has generated knowledge to transform these tools of pathogens into useful one actors for one genes of interest to humans. The tumor-inducing T plasmid of Agribacterium T and has now been defigured into a cloning lector which is no mere pathogenic to the plants but is still able to use the organisms to deliver genes of our interest into a vanity of plants. Similarly retro uses hail also teen disarm, D and are now used to deliver desirable genes into an INLA1 cells. So once a gene or a DNA fragment has been ligated into a suitable lector it is transferred into a bacterial plant or mammal host where it trolls. I1.2.3 compared MT host for transformation with recombinant DNA since DNA is a hydrophilic molecule. It cannot pass through cell membranes, WL1FJ in order to force bacteria to take up the plasmid. The bacterial cells must first be made compage 310 to L3K up DNA. This is done by treating Leam V with a specific concentration of a divalent cation, such as calcium which increases Li efficiency V, lit which DNA enters. OHCHNology, principles and processes The bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock, and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cells, in a method known as microinjection. Recombinant DNA is directly injected into the nucleus of an animal cell. In another method, suitable for plants, cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA in a method known as biolistics or gene gun. And the last method uses disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA into the host. Now that we have learned about the tools for constructing recombinant DNA, let us discuss the processes facilitating recombinant DNA technology. 11.3 Rose SSESOF recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of a desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale and extraction of the desired product. Let us examine each of these steps in some details. 1.1.3. 1. Isolation of the genetic material DNA. Recall that nucleic acid is the genetic material of all organisms without exception. In majority of organisms this is deoxyribonucleic acid or DNA. In order to cut the DNA with restriction enzymes, it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, proteins, polysaccharides and also lipids. This can be achieved by treating the bacterial cells, plant or animal tissue with enzymes such as lysozyme bacteria, cellulase plant cells, chitinase fungus. Figure 11.5. DNA that separates out can be removed by spooling. You know that genes are located on long molecules of DNA intertwined with proteins such as histones. The RNA can be removed by treatment with ribonuclease, whereas proteins can be removed by treatment with protease. Other molecules can be removed by appropriate treatments, and purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen as collection of fine threads in the suspension. Figure 11.5. 81,010 GYLLA2 cutting of DNA at specific locations. Restriction NCERT digestions or PCR by incubating Pun6 DNA molecules with a restriction NCERT. At the optimum 3L conditions FBR, that specific NCERT, agarose gel electrophoresis is employed to check the progression of the restriction or cligostion. DNA is a negatively charged molecule, hence it moves tau RDS the positive electrode A not E. Figure 11.3. The process is repeated with the vector DNA also, the joining of DNA in 01S processes. After having cut the source DNA as well as T vector DNA via a specific restriction NCERT. TLLE cutout, gene of interest, from TLLE source DNA ANCT TLLE cut vector via spa or mixed.n. Ligase is extract. This results in the preparation of recombinant DNA LLA3 amplification of C. Negative arrest using PCR. PCR stands for polymerase chain react, N. In this reaction, RMDI copies of TLLE gene or DNA of interest is synthesized and oil row using TVORLI SIA TD. CMIS, E2, RR3.I, 1I1 FISW, E11.6 polymer ECHML. Reaction PCRL, each cycle UV steps, I denaturation, uniner, net IU1E, tons 10 N of pruners. IIODH, IOIOGY, LTL, ICPIFS.A, IOPIASes S sets of PL near small chemically synthesized oligonucleotides are implementary to the regions of DNA on the NCERT DNA polymer cell. The NCERT extends the pruners using the nucleotides provided in the reaction on the genomic DNA S template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to iron 8 billion years, that is, I billion copies are made. Such amplification is achieved by the use of thermostable DNA P, Lyrer, Seisyl, TED from B, tenum. Thermosacclity cus, which remain tied during the high temperature induced denaturation of bubble-stranded DNA. The amplified 42 desired can now be used to ligate with vector FBR. Further Klum. Figure 2. 611, 3.4 Insertion of recombinant DNA into the host cell. Org3 there are several RREHCDs of introducing the ligated DNA into recipient cells. Recipient cells. After M3 King Lim, competent to receive T3K up DNA present in its surrounding So too, recombinant DNA bear gene FBR resistance to .M antibiotic, for example. Ampicillin is transferred into ECDI cells, the host cells, before T-arms formed into ampicillin resist MT cells. If spread, the transferred cells OOAGARPL, test containing ampicillin. TRMS1 BRMMTS will grow. WLTRMS1 burned recipient cells slowly will die. Since due to ampicillin resistance gene, one is able to select a T-arms FUD11 al cell and lepresin EOFMPLN. 1 reacillin assistance egenine to this case is called a selectable marker. 1.1.3. 5. Obtaining the foreign gene product when you insert a piece of 3 lean DNA into a CLCI-ling actor.md. T-arms for it into B. Bacterial, plant or animal cell. The alien DNA gets multiplied in almost all recombinant technologies. The ultimate O.M. is to produce reservable protein, hence there is a need for recombinant DNA to be expressed. The F foreign gene gets expressed under appropriate ions. The E. Pre-cio foreign is in host cells in Ryan understanding many technical details. After H, they include the gene of interest, and E haloing optimize the ions to induce the expression of the target protein. 1H, S to unsire or producing at OO, large scale, con G, UTNNL, of ang reason U, LVJ there SO need jor R9 ESO, RLE production at any proteinina, ding gene is E, pressed. In heterologous host to see all recombinant protein, the cells harboring clone lineages of interest may be grown OO, small scale in the laboratory. The cultures may be used for tracking the desired protein and then purifying it by using different separation techniques. The cells can also be one pleaked in a culture system wherein the use medium is drained out from one side while fresh red iron is added from the other to mom and the cells in their physiologically most add base morph control biology active log exponential phase. This type of culturing method produces a larger biomass leading to higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors, where large volumes 1,000 liters of culture can be processed, was required. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc., using microbial plant, animal or human cells. A bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions, temperature, pH, substrate, salts, vitamins, oxygen. The most commonly used bioreactors are of stirring type, which are shown in figure 11.7, JOM. R. Foam increase surface area for oxygen transfer Bragger 2.2. D1 flat-plated impeller, H culture broth. The Ka bubbles dramatically increase the oxygen transfer area. B figure 11.7 a simple stirred tank bioreactor. B sparge stirred tank bioreactor through which sterile air bubbles are sparged. A stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system, an oxygen delivery system and a foam control system, a temperature control system, pH control system and sampling ports so that small volumes of the culture can be withdrawn periodically. 11.36 Downstream processing. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished. BT01 ECN, ology, IRT, CPLFS.UNLL Pro Sapro, locked. The processes include separation and purification, which are relatively referred to as downstream processing. The PRCDTCT has to be formulated with suitable Presina tires. Such formulation has to undergo thorough clinical trials as in case of drugs. Strict quality control testing for each PRCDTCT is also required. The downstream processing and quality control testing vary from PRCDTCT to Pro, locked. Summary. Biotechnology, Gilles WL1M Skull Production N, ML, editing of products and processes using Organisms. Cellular enzymes, modern biotechnologies, and Genuk 31 Li modified organisms was made possible only WTNM Liam to alter. He cherishes one rye of DNA and construct recombinant DNA. This key process is called recombinant 3 anti-DNA tech, malo or ing. 1b, S process involve 9s the use of restricted endonucleases, DNA lig 3 appropriate plasmid or viral vectors to isoy ALMD ferry U1EI, U, in DNA into hast or 3 SMS. Pression of U1E foreign gene purification of EE, neprodual that is, U1E functional protein MD fin 31 Li making a suitable GNULAT IO04, ml, editing. Lore, scale Prodwatman involves use of PRES1 St10 recombinant proteins which are used in medical practice. Find out where U1E are used as therapeutics usulintimate. 2. Make a CH3RT vol diagrammatic represented in SH doing a restriction at del, ZFL lead the substrate DNA on which it A, TS. Treside at which it cuts DNA RT proto it produces 3. From what you have Leon can you tell ready, enzymes are bigger or DNA is bigger in molecular SLZ1, H, LWDUKMVL4. What would be U1 emoconcentration of human DNA in a human cell? Consult your teacher. S. D. Eukaryote says how, E. Restriction endorin, D. says. Just a D. Your answer 6. Besides better rare one ion and moon properties V. Bat all her advantages do stirred tank biory, Lars Hal, E. Overspell, E. Flask 7. O. Gile, I send Pesoft 31 I drum, Adna Bayan Solngire Te share, better try to cray 1 year palindromic sequence bite blowing pa, Arus. 8. Can you rec 311 meiosis and in, LC31E at water recombinant DNA is M3, DE9. Can you U1 Inc. LNFL answer how a reporter enzyme can be used to monitor transgranulin of H, street status bite bread, GNDNA in a didion to a select ABLA F, F, I. 10. 10. Desk BR1 EFI the F01108, Ors of replication BB1 reactor C downster MN Pruis 1 Ang 11. E. CPLM Nefly PCRB Restant 11 on 2 JR, iOS IDNAC Chil, SE 81010 GY12. DSCS with your testure.mel FTN, L out. H, LW to distinguish between a PL, SRND DNA I chloronosornal DNA BRNA on DNA C exonic Mazzini armed. Sen. Chapter 12 Biotechnology and its applications 121 Technologiolog gel applications TN, JL Shri CV JBT 122 Technologiolog gel applicaton STN. Medi-CTNE 123 TRC, Nisjan TCNM, DS 124 at LSSV.ES Biotechnology, S, UDI, MT from the previous chapter. ISLT 31IG deals with the just MLS reproduction of biopharmaceuticals on biologicals using genetically mictified microbes. Fungi plants MD, Nimals, the, Plic, science of biotechnology include therapeutics D3, Gnostics. Genetically M, diffy crops 4, section I culture processed B, D bioremediation waste treatment, ND energy production. 3 Critica L Resi RCH. Area S of biotechnology, iding the best C, Tau list in the F form of improved org 3 LLism. Usmli data microterpure and zerile Craig, Ting optimal conditions through Kuang, Ninifber, C, T Alice to court. ND3 downstream processing technologies to Puri, Jit Proteinok, Nick compound, let us now learn how human beings have used biotechnology to improve the quality of human life especially in the field of food production, and health. 1. 1 blow TPCHN old TRHR, AJ, S, I, A11, L45 TLLAGR1C, T, R, 1 Ru 3 let us take, look, T the 3 options Pet Condi thought FBR Anchor, SMGB, D production I, grow chemical B, said, section I culture. Iolog Vilo Q3 Nick 3 Greaseculture, third 3 Genetically Nirek Cropbo said 3 Agriculture, the Green Revolution succeeded in tripling the food supply but G8L at W, S not enough to feed the growing H, Populo. Lion, instead JL Zami page 3 Tlibine due to the use of improved crop varieties but mainly due to GLLE use of better management practices. N. Use of agrochemical as fertilizers third bestiires, HD, very FBR, MERS in the developing world. 3F. Chemicals. Are often too expensive. And further increases in yield net. Existing varieties are not possible using conventional breeding. Is TLL my alternative page third 3TR understanding of generally CSM show so that 4ers RMY atom maximum yield from their fields. Is there 3 to minimize TLL use of fertilizers. And chemicals sought let TLL harmful ethics on TLL and vir or urine. Are reduced use of genetically diffy crops is a possible solution. PLMTS bulk taro, fungi 3 mernals whose genes H3 vebinalterad B manipulation 3 rec 3 lead gene eucal Z model 11 at organisms G10. GM plants hydine useful in many ways. Netic M, defecation has, IFFI 3D crops more tolerant to 3B0 lick stresses cold. Drought salt T3TU reduced reli 3 cell on chemical pesticides pestwe sisla 3 anti crops 3 health D to reduce post H3 revest losses. IJ Anchor 3 said efficiency of bind IS 3 Jebitlo NTS this pre, and Samajex Hoaslian of fertility of S oil. BENH3 NCERT Nutritione 3L Volume of food, for example, VIL 3 minutes A enriched rice, in addition to TLLE's uses. GM has been used to create tailor-made clones to supply alternative sources to industries in the form of S, mix. Fuels MD Pharmaceuticals, SOR of the 3 science of biotechnology and 3 agriculture that you will study in DEL 3 ILME the production of pest resistant 3 and TPLMTS, which could decrease TLLE and DUNT of pesticide used. BT Tizin is produced by a B3 bacterium called Ladlues laryngoensis BTFBR short. BT2 and gene H3 has been cloned from the B3 chlor 3/3 been expressed in PLMTS to provide resistance to insects with low TLLE need FBR insecticides in effect created. A bit pesticide. B3 plasma BT cotton, BT com rice term to Pro 3 to MD soy abanet, BT cotton, SOR SLR3 ins of Bacillus strvnngins produce proteins 3T kill certain insects such 3S lepid, NS tabo CCO budworm. Coleopters beetles 3 dipterms mosquitoes. Beetle ringoens, S forms protein crystal 3LS during 3p molecular pH 3 of their growth. These crystals conlo in 3-2, iconsecoclidal protein, white bezis 2, inoculate bacillus. Actually the BT lolene protein is 3S and 3 type prot, LLMs but once M insect ingest the inactive tizin. It is converted into and active at form of tizin due to the 3 alkaline pH of the gut which solubilize the crystal 3LS. The 3-3 TED2 binds to the CERN set of midgut epithelial cells MD. Biotechnology and its education, S create pores that cause cell swelling and lysis and eventually cause death of the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton figure 12.1. The choice of genes depends upon the crop and the targeted pest, as most Bt toxins are insect group specific. The toxin is coded by a gene named CRY. There are a number of them. For example, the proteins encoded by the genes CRY-IOB and CRY-IOB control the cotton bollworms, that of CRY-IOB controls corn borer. Figure 12.1 Cotton boll, A destroyed by bollworms, B a fully mature cotton boll pest-resistant plants. Several nematodes parasitize a wide variety of plants and animals including human beings. A nematode Noida incognitia infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation which was based on the process of RNA interference RNA. RNA takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to a complementary dsRNA molecule that binds to and prevents translation of the mRNA silencing. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements transposons that replicate via an RNA intermediate. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant figure 12.2. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs being complementary to each other formed a double-stranded dsRNA that initiated RNA and thus silenced the specific mRNA. Biology of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite figure 12.2. Figure 12.2 Host plant generated dsRNA triggers protection against nematode infestation, a roots of a typical control plants. B transgenic plant roots 5 days after deliberate infection of nematode but protected through novel mechanism. 12.2 Technological.pr.ppll Cations M1LLM CLAN are the recombinant DNA technological processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological responses as is common in case of similar products isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. 1 genetically engineered insulin management of adult-onset diabetes is possible by taking insulin at regular time intervals. What would a diabetic patient do if enough Hunan insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from other animals be just as effective as that secreted by the Hunan body itself, and would it not elicit an immune response in the Hunan body now? Imagine if bacterium were available that could make human insulin. Suddenly the whole process becomes so simple. You can easily grow a large quantity of the bacteria and make as much insulin as you need. Think about whether insulin can be orally administered to diabetic people or not. Why? 6 IOIH, IIOGFALNR peer, AIIOI insulin used for I3 bet was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from M-mernal source though caused sort patients to develop .NRG or other chips of reactions to the F4 protein. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by Eli sulfate bridges, figure 12.3. In MLS including Hums, insulin is synlysis. Is .pro, horone Nike .pro and zerli pro horone. Also. Exclamation point. CL needs to be processed B1 braic because a fully mature functional IUE which contains an E.CTR is stretch fig 1U, called the C peptide. This C peptide is not present in mature insulin. And is remoed matudation into insulin. The mom challenge. Pro, Luction of insulin 12.3 mature UON of PRLs Ulin MTO Ulsulin removal of CPAP to be Sanpl1 EDLU Sing. DNA Techniques was getting insulin assembled into a mature F-form in 1983. LL Lion American Company prepared TV, DNA sequences corresponding to A and B chains of human insulin and introduced them in PL Smith S of E. coli to produce insulin CA chains, CA chains A. And below, air produced separately extracted and combined by creating disulfide L, ons to FBRM hot-diol insulin. 2C, now 1 therapy if a person is born, ili herited, airy disease can corrective TLL or APBL3 can FBR such DIs as gene therapy is M. Attempt to do TLL is. Gene therapy is a collection of relics allows correction of a gene defect that has been diagnosed in a child embryology. Here genes are inserted into a person, S cells in tissues to try DIs as, Q, action of genetic defect invos area of normal gene into the industrial livid UL or embryo to TLs over TLLEFWL xion of and compensate roar the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl, with adenosine deaminase 88 deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to the release of gene for adenosine CTEMS. In sore children ADA deficiency can be cured by bone marrow transplantation, in others it can be treated by NCERT therapy, in which functional ADA is given to the patient by injection, but the problem with both of these approaches is that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the BXLE. A functional ADA adeno using a retroviral actor is then introduced into these lymphocytes which are subsequently returned to the patient. H011 ever as these cells are not ILL the patient requires spirit DC infusion of such genetically engineered lymphocytes. Or 2 lead gene isolate from MRO cells using ADA is intro, loosed into cells at the early embryonic stages. It could be a PEM-blend cure. Iolog vel 2, 3 moleculage, diagnosis. You know that for effective treatment of a disease irlidiosis and understanding its pathophysiology is irrelevant, LRLMT. Using conventional REFT, DS of cis-serum and urine analysis etc. early detection is not possible. Recombinant DNA technology, polymerase CH, and reaction PCR on enzyme link runosorbent assay use or SOR of the techniques that set, RVE the purpose of irlidiosis. Presence of a pathogen bacteria, rhesus etc. is normally suspected only when the pathogen has produced a disease symptom. By this time the concentration of pathogen is already GH in the body. However, very low concentration of a bacteria or RUS at a time when the symptoms of the disease are not JLV-sible can be detected by amplification of nucleic acid by PCR. Congo explain how PCR001D at Overglau Emeroons if DNA PCR is now routinely used to detect HIV in suspected AIDS patients. It is being used to detect mutations in lineage in suspected cancer patients too. It is a PDA, referral technique to identify many other genetic disorders. A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridise to its complementary DNA in a clone of cells FBLUV by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. USA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins etc. or by redeclaring the antibodies synthesized against the pathogen 12,3-FN, NSGENC.RLT. Mouse animals that had their DNA manipulated to possess and express on extra foreign gene are known as UAN and animals. Transgenic rats, rabbits, GS, sheep, cows and fish have been produced alpha over 95 page 3%. All existing TR enzymic.mejnals. Are mice. TNHGR these LIM, RLSL, iron produced, how can MML, Nate from Sudmod JFO Kaj, Onslatus Tri 3 JLD. E.O, Plurisor of Lee Karun JN Reactions, I normal P, Ig Theology and Development, Transgenic aromals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of a body and its re-output. For example, studying complex factors involved in growth such as insulin-like health factor. By introducing genes from other species that alter the formation of this factor and studying the biologic 11F tech that result. Information is obtained about the biological role of a factor in the body. IL-2, FUQFD SUS, Transgenic kernels are reassigned to increase RSMDing of TO. LNish contribute to the development of DC. These are specially made to say as NXX, IOSBR human Eli, Sofidin Tiank Nutritmonutrically, SU made possible. Day transgen, C. Migdalzeris DT Manaham 21L diseases NCH.Cancer, cystic fibrosis rheumatoid arthritis and A1's HRFS. W. Biological products, medicines required to treat retain human DC.S can contain B0 Logic 31 Pro, LUX. But such PO, LUX are often expensive to make. Transgenic.ONIJN, ALs that produce useful B0106C31 Pro, LU EDS can be created by the INTM, LUCON of the portion of DNA or genes which axles RORA Portula Pro, LUC such as Hun protein. Exclamation point antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenyl, L-Generta PKU and cystic fibrosis. In 1997 the first transgenic CCM, Rosie produced human protein-enriched milk 2.4 grams per liter. The milk contained the human 3-lacth-31-bumin and was nourished 3-LLYM, rebalanced POUET for human babies in natter, L-cow milk. LJ Vokes and Escfidi, trans-CMCE are being developed to use in testing the S31 ETCT vaccines before they are used on humans. Transgenic Mikir being said to test the Sayalic Thecliovacine, if successful and fun to be reliable. They could replose the UCTRMNKEYS to test the Cephatiop Bachisophtavac, MLMLSCFD test in 9, this is KNCMNSID, Citisophetidasling. The procedure is the SARAs that used or testing Corsatiop drugs. Transgenic animals are made that carry genes which MA1, LO-thermer or sensitive to crox-subs1 and cysthanin transgenic anim, LS. They are then exposed to that crox substances and the Edreitz-Doodle D2 testing in such animals will 31 loas 2 boin result in less teary. 12. 4 Li, NTJ.111, ISTFI, S1 humanipulon of living organisms by the human race cannot go on any further. Without regulon, Sun, ethic 31 standards, required to evaluate the morality of all human activities that might help CR harm leaving agan SMS. Loing beyond the more 3 litif such issues the blolog, el sigil, I can CT such things as 31 so important lonetic different UANA organisms can ha 10 unpredicatally results men such organum. Lantro, loosed into the ecosystem there but the Indian Gulemarat has set up again Sayun such as GEAC genetic engineering a Prod 31 corn T. Which will make decisions regarding the V31 identity of GM research and the safety of intro, loosing GM agonisms for public services. Silicon, usage of leaving agorisms or public services as FCO, L&MD, CINI sources, ROR example has 31 so created problems with patents granted are the same. Iolog there is growing public anger that Cherdo companies are being granted patents the junior Reproduction technologies that make use of the genetic matter is. Plants MD other biologic resources that have long been identified developed and used by farmers and indigenous people of a SF civic region country. Rice is an import tea food grain the presence of which goes back thousands of years in Asia's cultural history. There are an estimated 200,000 varieties of rice in India. Alone, the diversity of rice in India is one of TLLE richest in the world. Basmati rice is distinct for its WL-aroma. And FR. In 27 counted varieties of Basmati. GRCRWN in India. There is reference to Basmati in ancient Te.O, TS folklore MD Sutri. As it has been grown FBR centuries. In 1997 Amabert Company got patent TS on Basmati rice through the U.S. Patent and Trademark Office. This allowed the company to sell a new variety of Basmati in the U.S. and abroad. This Ruvineti CT Basmati had actually been derived from HTMFRNI SV Midis, Indi 3 NB Basmati W is crossed with Lasemi DWRV Midis. NCLR. As. In Engineer and Melty. The patent runs to FW, CTI 0113.1 equivalents implying that other people selling Basmati rice could be restricted by the patent. Several attempts also been made to patent uses products and processes based on Indian TR additional herbal medicines. For example, Turk name if we died are not vigilant and we do not immediately owe to these patent applications. Other countries individuals may encash on our rich legacy and may not be able to see anything about it. Biotree is TLLE term used to refer to the use of bioresources by multinational companies. And all your organizations without proper authorization from Ultrates MD people concerned without compensatory project. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity MDTR additional knowledge related to resources. Strategy 0113.1 Knowledge related to resources can be exploited to develop STEM applications and can also be used to save TNE. Effort.nd. Expenditure during their commercialization, there has been growing realization of the injustice. Inadequate compensation and benefit sharing and WFD and developing countries, their fee, JRE. Some nations are developing laws to prevent such unauthorized exploitation of TLL or bioresources MDTR additional knowledge. The Indian Parliament has recently cleared the C and R entrant of the LD3 and patents bill. TLL takes such issues into consideration including patent terms emergency provisions. In Research MD Development initiative. Biotechnology IDGYI to useful products B, using FTRobes. Plant and Metab, Lickmac Benefi, Recombin, MTFDN Electrology has possible to near FTRodes, PLTS3LD anim, LS such that they have novel capabilities. Genetically, Mode I1 IED organisms have been created by using methods other 1H3N and 3 Chural methods to TR30 or one or more from our organ, SM to C another. Narrowly using techniques such Siri, Ombineo DNA to Crollo, LLIGM plants have been useful in crop J, yields. Redo, EP, Street IH, nest losses 3rd M3LRE crops more toler 3LF of stresses 1 are several GN crop plants with Imprimagel Wishiol 90 Lu F Fox 3rd Ridurui Trarelli, S on C, Chemical FLSI sides J, S, Resi STT crops IPA, Ambiant DNA technology Calpru S have M3D immense, IMPA, T in the of Heal 1 Care B enabling mass production of Sol, 3rd more effective TL, Therapeutics. Since Trari, Ombinant therapeutics Jamie identical to human proteins they do not induce unwanted immunoresp, OSAS 3LD are free from risk of infections was I observe in case of simil 3 are products isolated from raw M sources. Human insulin is M3D in bacteria yet its structure is absolutely identical low that of Tranotter 31 mol, U. I, R, N3NS Ni animals Me3LSO used to understand how genes contribute to the development of a D, C spy serve, enzymic, dels for ham DCS. Such as cystic fibrosis rheumatoid R1 ritus J.UVLL, UGDLI, gene therapy is insertion of INLO in individual cells. Tissues to treat dysresp, C3LLY hereditary diseases. It does, so by replicating a directive mutant allele, first of on, SHIOL1 or 1. Geartor which involves Z, amplification viruses. At ACL, Trier hosts .00 introdu, E-Triri. Redic material into TRAR1 NST cells EF part of their replicable, LN are used as soders to transfer healthy, Geracermari, NUIP, Rashians F genes. Exclamation point 1 current interest in tramanable Cheyenne of microtes PLTS 3rd, one has raised serious ethical questions. Arzerk is L, S1. Street LS of BT to Zin perdurate by some bacteria do not kill the bacteria TB. Selves because the bacteria are resistant to the lozin B to Zin is immature, CJ to Zin is inigai, D bacteria ear places lozin in a special sac. 2. What are TR bacterial lustrale using any one example 3. Per 3rd contest the odd 3rd disadvantages of production of L-modified crops. 2 T5. 1, 4, 5, 6, 7, 8, 9, 10, 810 L0 GYWHC1 microprotuls and 3 Rolamorg, Amzamulet produced TT. Human exploit add this protein to his benefit what? S gene herapja ULS RIEL, ULG he example of adenosine deoroin, CAD8 off J. DTGR, emmatically represent the experimental steps 1 and cloning and expert eng on hernanar, ECEFR growth hormone into a biocternum like Eutkin Hosui. F. SL Emiliad Teremo oil QL Drosim Bonl from seeds based on OTNG of Rona Technology. Melcherist rye of oil F1N, L out on 1 Turnitwit 1S golden recent. Does our blood ha proteases and reduces consult internet FND out how to make Ali active protein PLWR and solical. FAT1SU, E major problem to be E, ROALLE, red. See Hapliker I30RGANISMS and Popul Alliance Chapter 14 Ecosystem Chapter, S Biodiversity and Conservation Chapter 16 and Environmental Issues. Diversity is not only a Cherich Laris look of living organisms bolo of Conlinland biology lexl books. Biology is present deal her as botany, zoology and microbiology are as classical and modern. The latter is a euphemism for molecular aspects of biology. Luckily we have many threads which weave LHE different areas of biological and form align in a unifying principle. Ecology is one such thread which gives us a holistic perspective of biology. The essence of biological understanding is know-how organisms. While remaining an individual, in Morocco will all her organisms and physical habits as a group and hence behave like organized wholes, that is, population, communally, ecosystem or even as LHEW whole biosphere. Ecology explains Loas Alice a particular aspect of his as LHE study of anthropogenic environmental degradation and LHE socio-political issues IL has raised. This unit describes as WL as Lake's critical view of LHE above aspects, capital I. IRAMD Fo Misra 1908, 1998 Ramdeo Misra is revered as the father of ecology in India, born on August 26, 1908. Ramdeo Misra obtained Ph.D. in ecology 1937 under Professor W. H. Pearsall, W. JFRS, from Leeds University in UK. He established teaching and research in ecology at the Department of Botany of the Banaras Hindu University, Varanasi. His research laid the foundations for understanding of tropical communities and their succession, environmental responses of plant populations and productivity and nutrient cycling in tropical forest and grassland ecosystems. Misra formulated the first postgraduate course in ecology in India. Over 50 scholars obtained PhD degree under his supervision and moved on to other universities and research institutes to initiate ecology teaching and research across the country. He was honored with the fellowships of the Indian National Science Academy and World Academy of Arts and Science, and the prestigious Sanjay Gandhi Award in Environment and Ecology. Due to his efforts, the Government of India established the National Committee for Environmental Planning and Coordination 1972 which, in later years, paved the way for the establishment of the Ministry of Environment and Forests 1984. Chapter 13 Organisms and Populations J3, J or 9 NSM und SRTNM and 13.2 Population SRL, Eng 100 RLD is fascinatingly diverse and organismally complex. We can try to understand its complex by investigating processes at various levels of biological organization macromolecules, cells, tissues, organs, individual organisms, population, communities and ecosystems and biomes. My level of biologic 3L organization we can ask types of questions. For example, when we hear the bulbul singing earlum, running in the garden, may ask how does the bird sing or why does the bird sing. The how type questions seek the meton SM behind the process while the why type questions seek the significance of the process. For the first question in example the answer might be in terms of the operation of the iOS on the vibrating bone in the bird. Whereas for the second question the dot's low, mahian bird, need to communicate with its mate during breeding season. When you observe nature around you little a scientific FRR of mind you will certainly roar up with many interesting questions of both types are nigdel, human. Jowers General 19U, and how does the BL, now wick jower 1 is nectar WI does catu s hayasum and G thorns how does the DLICK recon. Sef larocon. M, third dot and so on. Full stop. 110 II1 Joule Biology. You have already learnt in previous classes that ecology is a subject which studies the interactions among organisms and between the organism and its physical abiotic environment. Ecology is basically concerned with 4 levels of biological organization: organisms, populations, communities and biomes. In this chapter we explore ecology at organismic and population levels. Ecology at the organismic level is essentially physiological ecology which tries to understand how different organisms are adapted to their environments in terms of not only survival but also reproduction. You may have learnt in earlier classes how the rotation of our planet around the sun and the tilt of its axis cause annual variations in the intensity and duration of temperature, resulting in distinct seasons. These variations together with annual variation in precipitation, remember precipitation includes both rain and snow, account for the formation of major biomes such as desert, rainforest and tundra. Figure 13.1. 32.520 Tropical 15 10 506 10 15 50 100 150 200 250 300 350 400 450 Mean annual precipitation. Figure 13.1 Biome distribution with respect to annual temperature and precipitation. Regional and local variations within each biome led to the formation of a wide variety of habitats. Major biomes of India are shown in Figure 13.2. On planet Earth, life exists not just in a few favorable habitats but even in extreme and harsh habitats: scorching Rajasthan desert, perpetually rain-soaked Meghalaya forests, deep ocean trenches, torrential streams, permafrost polar regions, high mountain tops, boiling thermal springs, and stinking compost pits, to name a few. Even our intestine is a unique habitat for hundreds of specks of microbes. Organisms and populations of BCD Figure 13.2 Major biomes of India, a tropical rainforest, B deciduous forest, C desert, D sea coast. What are the key elements that led to so much variation in the physical and chemical conditions of different habitats? The most important ones are temperature, water, light and soil. We must remember that the physico-chemical abiotic components alone do not characterize the habitat of an organism completely. The habitat includes biotic components, also pathogens, parasites, predators and competitors of the organism with which they interact constantly. We assume that over a period of time, the organism had, through natural selection, evolved adaptations to optimize its survival and reproduction in its habitat. 1 major abiotic factor is temperature. Temperature is the most ecologically relevant environmental factor. You are aware that the average temperature on land varies seasonally, decreases progressively from the equator towards the poles and from plains to the mountain tops. It ranges from sub-zero levels in polar areas and high altitudes to 50 degrees Celsius in tropical deserts in summer. There are, however, unique habitats such as thermal springs and deep-sea hydrothermal vents where average temperatures exceed 100 degrees Celsius. It is general knowledge that mango trees do not and cannot grow in temperate countries like Canada and Germany. Snow leopards are not found in Kerala forests and tuna fish are rarely caught beyond tropical 81,010 GY latitudes in the ocean. You readily appreciate the chickens of temperature to organisms when you realize that directs the kinetics of NCERT the row at the basal metabolism. Actal, LTY3JLD. Other physiological functions of the organism. A few organisms can tolerate and thrive in a wide range of temperatures. They are called euthermid. But a vast majority of them are resistant to a narrow range of temperature. Such organisms are called stemmerm. The levels of thermal tolerance of different species determine to a large extent their geographical distribution. Can go TLVNL, of thermal and stenothermal animates and plants. In recent years there has been a grow and concern about the gradually increasing average global temperatures. Chapter 16. If TNS trend cont, NUES, wout go exp the DSCTLF, UTL on it row and check UJ some sped.es to be affected. Water, next to temperature. Water is the RMST iron potent factor in 11 unting TLLE life of organisms. HLF3CT light on earth all igen 3 TED. In W3 terra and is unsustainable without W8er, it's .F3 ilability is so limited in deserts TLL it only special adaptations make it possible to live there. The productivity and distribution of plants is also heavily dependent on water. You might TLL link TLL, at organisms LR1 anginosans, 13 Kes.N. Rivers should not face any water-related problems, but is not true. For aquatic organisms the quality chemical composition. pH of water BR is important. The salt concentration measured as salinity in pmls per thousand is less than 5 page 3% in INL and waters 30-35% Lyseodidin, 100% in sour hypersaline 13 goons. Some organisms are tolerant of .IR range of salinities urohaline, but others are restricted to a narrow range stenohelm. Many freshwater animals cannot live for long in seawater. N. Vice versa because of LEOS and Duke problems. TLLEIOL phase, LLGLLT, since plants produce B, D through photosynthesis, a process which is only possible when sunlight is available. Is a source of energy. We can quickly understand the importance of light for living organisms, particularly autotrophs. Many species of small plants, 3JLD shrubs growing in forests, are adapted to photosynthesize optimally under very low light conditions because they are instantly overshadowed by tall canopy trees. Many plants are also dependent on sunlight to reach their photoperiodic requirement. For flowering, for many animals too less important in that they use the diurnal and seasonal variations in light intensity and duration photoperiod as cues for timing their sprawling. Reproductive and migratory activities. The availability of light on land is closely linked via TLL of temperature, since TLLESWL is Lee source FBR both, but deep 500 m in the oceans the environment is perpetually D. Arc. And its IRL bit 3 and TS. Are not. Are awfully existence of a celestial source of energy called sun. TFM Nan, SD Mir Salsaviner 9G. The spectral quality of solar radiation is also important for life. The W component of the spectrum is harmful to MMY organisms, while not all the color components of the visible spectrum are available for marine plants living at different depths of the ocean. And the red-green and algae that in OTTMC. UID, SW, KG to before J in the deepest waters. Why solely, the nature and properties of S-oil in different places vary. It is dependent on the climate. Levy, Ealing process wetler S-oil is rotator sitter and torianto S-oil developer in occurred. B. New CHR characteristics of TLLES oil such as S oil composition GRN size and awegation determine the percolation and water holding capacity of the S oils. These CH3 characteristics along V, little parameters such as pH mineral composition and topography determine to LR. Exclamation point E. 10th the education in any OA this is interdicted as the type of animals that can be supported simul 3 RTY. In the aquatic environment less adherent CH macteris often mean the type of benthic animals that can lull either. L3L, 2 responses to abiotic factors and realize that the abiotic conditions of many habitats may vary drastically in theory. Lol E now ask W do the or, gan, SMSUO, N, 9, NCERT LLO bidnits koper monaju at stressifu.com dt once but before attempting tones are this question. We should perhaps ask first why a highly runnable external environment should bother organisms at all. One would, that during the course of millions of years see their existence many species would have evolved a relatively constant internal within the body and, had permits all biochemical reactions and physiological functions to, proceed with maximal efficiency and thus enhance the overall fitness of the species. This constancy, for example, could be in terms of optimal temperature mictos and concentration of F body fluids. Ideally then the organism L should try to maintain the constancy of its item 3L environment, a process called 1 homeostasis, S despite varying external environmental conditions TLL it tend to upset its homeostasis. Let us L3K. An analogy to see with this important concept. Suppose a person is able to perform her best when TLLE temperature is 25 degrees Celsius and wishes to maintain it so. Even when it is scorchingly hot or freezingly cold outside. Fis. Euro 13.3D1 agrarum at 1C representation of org3L.les, M1C response it could be achieved at Hoer. In the car while traveling and at Oak Place by using air conditioner and server and heater in winter. Then her performance while be always maximal regardless of the weather ld hiln her. The person's me is uses as not TLL rough physiologic albit. Artificial me ns. How do other Leo LR9 or 9 SMS Cope W, TH the SIG JATV, let us look at the me as possibilities figure 13.3. 81,010 GYIR LIE, SOR organisms or 3 blatomom tom homeostasis by physiolog, Kodso, Metters Beth 3V0 Euro 3 LSO Rions which ensures constant bow, like Telcler 3 Ur. Cons MT Oswert Tick constant 13 ion etc. All birds. In mammals and very few lower vertebrate and invertebrate species. Are indeed capable of such regulation. There are low and third as more regulo lion. Ealuli 0113 IY biologists believe the success of our MLSs, due to their 3 ability tom 3 consto antibody temper 3 Turon thrive whether they live in Antarctica or in Lee SR or resort. The Richoni SMS used by RMSTLW, OS Torregulo tether body length F.Atcher.R similar to Lee or STLATHUFWLSUs. AENUMF, and 3 Amston body temperature of 37 C in summer when outside temperature is rumor 3 N Arbo, like temper 3 Twerwe. 3 profusely, the resilient jet 3 pour 3 Lee cooling semi 1 M to WH3TH3 pens with 3 desert A, Oler in Oper 31 ion. Bring CBW and the body temper 3 tour, in winter when Lee temperature is much lower TLWL 370 C, Tay start to shear. A kind of Seas which produces here. And RAs is TLL bow, tie temperature, PL ants on the other HN. Do not have such mechanisms tolerant and into maltayarachers ILCO, I form, an overwhelming or jaunty 99% of 3 nimals on nearly 311 PL ants. 3.1 ULM main and a constant intimal environment. Thirbo, little creature CH3 and jet LTH the ambient left for geature and aquatic animals TLL the osmotic concentration of lead difluid CH vili trat of lead ambient W8 OSFI tick concentration. These 3 nimals third pools or simply conroarers, considering the benefits of a constant intern. A environment to TLL the organism. They must ask why these unirers H3D note 01 ed to be Rome regulators, recall the Hernan Ono IOG we used 3 above, much 3 as they like. How many people can really afford on 3 iron or M3 ni simply sweat 3T it out. And resign themselves to suboptimal performance in hot summer and bliss. Hermoregulation is energetic ally, pensive FBR monoorganisms, this is Pimtaku, MLY true for SRMLL on 3LS like shrews. N. Hummingbirds, he dotted loss or he ATGN is a function of surface. RA since small animals HAve a TRG surface. R relative to their valor. They tend to lose body heat 3 LREF third when nearly asuled outside, then they H3 vet 2, Pend much energy to gener 3 body he at GLU of metabolism. This is the main reason why or small 3 nimals 3 we are pre rely round and po, mreg, ons. During the course of evolution the cost and benefits of maintaining a constant intimal ENW current RT, 3L, and into can see, ieration ser species HEROT. The 3 ability tower 3 tabit only mer 3 limited R0 JLA del of environmental conditions beyond which they simply conform if the stressful extra malundilians or la calcita remom only for a short duration the organism. H is twulier. Alternatives. LWAI grot, the organism can away temporarily friend the slarat, sval lubidictor or hospital 3 blary andrew WII when susful perb, L is 011 or. In human anuji this STNT edgius lilta per men, eng fran deli to shimla for the dur UCN asernmar. Many animals particle 311 ibirds do, in winter undertake ALNG clistance retrations to roar hospitable areas. Every winter the F31LL0 Uskialaton 800031 Park BH3 Rupurin R3 Jaslahan host thousands of aleatory birds oro, eng from Siberia and other extremely cold northern reg, ons. LJSU spend, in Bactinaton and lower plant LS kinds of thick-walled spores are formed which help them to discern unfavorable conditions the, egernetin of the oilability, ultible environment. In higher plant seeds and some other vegetative reproductive structures the enias means taught Eckerpank CLST respastled. S helping in dispersal they germinate to form new plants under favorable moisture and temperature condition. Tyly do so by reducing their metabolic activity anga, eng into a date occupancy, in animals the organism. I funabled omligrid niglta 110 I'd the stress by Eska and Thierry 1 Bitam, Arcasa bears goins into enbornadon during winter as an example at Escape and lime, Sarah snails and fish GP into East Duty on 2A, oid summer related problems heat and desiccation. Under horrible conditions many, see a plankton species in Lacas and partles are known to enter Kuopo Street. A strive suspended D110 Lapo, and LL adaptations while considering the Vanus alternati Sava, bled to organisms for coping with the trends in their environment. We have seen that some are able to respond through certain pH physiological adjustments while others CB so behave, tenurally retreating temporally to a less stressful habitat. These responses are also actually their adaptations, so we can say that Akpajon is any atl one butte of the organ, SMM, pharological. Pedagogical behavior, alpha that enables the SM to serve EIO and reproduce in its habitat. Manak taptons have E101 bred Mr. LCMG Eoluiri white time and Arnalikani fee, ed in the ints of an external source of water. He kangarks, rad in north. Atral 1 kankterert, is capable of meeting all its water requirements LH1 if its internal fat OO, ida UCN in which water is a bank slug. L also has the illegal to uns and rate 1s urine so that mouth kluma water is used to redder 01 excretory ink slugs. Many desert plants have a thick cuticle on their layout faces and have their stomata arranged in deep pits to mini OOIOE warlo around transpiration. They so have a special photosynthetic pathway CIM hat enables their stomata to remain ing daytime. Some earth plants like Opawil have no leaves they are reduced to spines and the phyllostotic function is taken 0110R by the flattened stems. 81,010 GYMWM3LS from culture climates generalia, short terra und limbs to minimus ahiat loss. This is collectivo answer, in the P. Larcius, qualigram noas like seals hail a thick layer awful blood below their skin X.m insulator. And reduces loss of At. Some organisms possess adaptations that are of J's, 009 cal, which allow them to respond quickly to a stressful situation. If you had ever been to any high-altitude place, 3.500 m Tang Pass near Mannerley and NCERT. In China occupied Tibet you must have page 3 next what is called OJJI Jude SLCKNS. Its symptoms include nausea, fatigue. MD heart palpitations. This is because in TLLE low. ATM spheric pressure of high altitudes. The body does not get enough oxygen, but gradually you get acclimatized on stop experiencing altitude sickness. Howdy de Gurbod 9 Sal TNS Proner the body compensates low oxygen availability by increasing red blood cell production. Decreasing the binding capacity of hemoglobin and by increasing breathing rate. Meng tribes li oe and tlen altitude of Himalayas. FTR1D out leg normal 19 ruo 9 mer red blood su count or total hema 9 and then PRPT. F and TRPONs and NDST animals TLLE metabolic reactions. And hence. All the physiological functions pursued optimal in a neuro temperature range in humans. It is 37°C, but there are microbes archaebacteria that flourish in hot springs and deep sea hydrothermal vents where temperatures FME, exceed 100 degrees Celsius. How is TLL is possible? My fish live in Antarctic waters where TLLE temperature is ALWIs below zero. How Diothegmage to Eptliobod 9 Lu DS drum freezing Svahorhaven etiophemerine invertebrates and fish live at griot depths in the ocean where the pressure could be 00 times than Grima 1 atmospheric pressure Tratwi penance. How Diotheglioi under Sudsea and Slater 9 pressures N. Diothegruang spedalensms organisms living in such extra environments showed a fascinating array of biochemical adaptations. Organisms show better 10 Ural resp, and says to cop 3 with VRDations in their environment. Certainly ds lack the physiological ability that mammals have to deal with the high temperatures of their habitat, but manage to keep their body temperature fairly constant by behaviorally as they bask in TLLE sun and absorb heat when their body temperature drops below the conv T zone, but move into shade when TLLE ambient temperature starts increasing. Sarah species are capable of burrowing into the soil to hide and escape from the above ground heat. 1A2.1 Population attributes in nature were defined isolated, single individuals of any species, marginal, Y of them lie in groups in a well-defined geographical area. Share or compete or similar resources potentially interbreed. In TLL.US constitute a organisms and populations population. Although the term one interbreed one implies sexual reproduction, a group of one individuals resulting from even asexual reproduction is also generally considered a population for the purpose of ecological studies. All the cormorants one and a wetland, rats one and an abandoned well one ang, teakwood trees one and a forest tract, bacteria one and a culture plate and lotus plants one and a pond, are some examples of a population. In earlier chapters you have learnt that although an individual organism is the one that has to cope with a changed environment, it is at the population level that natural selection operates to evolve the desired traits. Population ecology is, therefore, an important area of ecology because it 11 NKS ecology to population genetics and evolution. A population has certain attributes that an one individual organism does not, and one individual may have births and deaths, but a population has birth rates and death rates. In a population these rates refer to per capita births and deaths, respectively. The rates, hence, are expressed as change one and numbers one increase or decrease with respect to members of the population. Here is an example. If one and upon there are 20 lotus plants last year and through reproduction 8 new plants are added. Tak one and the current population to 28, we calculate the birth rate as 820.4 offspring per lotus per year. If for one individuals one and a laboratory population of 40 frutiles died during a specified time one interval, say a week, the death rate one and the population during that period is 440 all individuals per fruit fly per week. Another attribute characteristic of a population is sex ratio. An individual is either a male or a female, but a population has a sex ratio, e.g., 60% of the population are females and 40% males. A population at any given time is composed of one individuals of different ages. If the age distribution percent individuals of a given age or age group is plotted for the population, the resulting structure is called an age pyramid, figure 13.4. For human population, the age pyramids generally show age distribution of males and females in a cone 1-ned diagram. The shape of the pyramids reflects the growth status of the population and whether it is growing, B stable or C decla 1 and 1 ang. Post-reproductive R and reproductive T, R, pre-reproductive ID 1I, L, U, I, SF, A2, II, S, rule expanding E. Stable decline 1 ang figure 13.4 representation of age pyramids for human population. The size of the population tells us a lot about its status in the habitat. Whatever ecological processes we wish to investigate in a population, be it the outcome of competition with another species, the impact of a 81,010 GY predator or the effect of a pesticide application, we always evaluate them in terms of .NECH engine Liu Lian CZ. The size in nature could be as low as I0 Siberan CR, Mesad Birat per V, Chlans in NEG or go into millions CH under mono S in a pond. Population CZ, more technically called popula U on density designated as N, need not necessarily be measured in numbers only. A little total number is generally the most appropriate measure of population density. It is in sourcey as is Etler RRE meaningless or difficult to determine. In N.R.A if there are 200 part man, plants but only a small huge banyan tree with a MGE Kankby, stating that the population density of banyan is low relative to that of part man, amounts to underestimating the enormous role of lead mayan and trat canronblity. In such C as is Li percent CO or B, Lernasis.m, RRE meaningful RRE azure of lead population size. Total number is again not an easily adoptable measure to the population is huge and counting is impossible or very teary concern. Exclamation point FGO TV, We a dense obor at GAD12 XJ de re and OPNDC what, s the best meoslanet repti extensive 9 sometimes. FBR over to ecological investigations, there is no need to know lead. Absolute population densities, relative densities serve the purpose equally LL. For instance, the number of fish caught per trap is good NO, measure of its total population density in the LAL, E. We are mostly obliged to estimate population C says indirectly, without actually counting them or seeing them. The tiger census in our national parks and tiger reserves is often based on pugmeral.s and fecal pellets. 1.3.2 population grow. 1. H the CSAN of a population FBR and SPE is not a static parameter, it keeps changing in time, depending on news factors including 2. D availability. Pred. Alien pressure. And reduced DAE other HL factor is ECH junction population density that has some idea of what is happening to the population, whether it is flourishing or declining, whatever might be the ultimate reasons. The density of a population in egg, then habitat ton. A given period, fluctuates due to CH junction Verbiasic processes. 2 of which natality and immigration contribute an increase in population density and T, morugity and immigration to a decrease. A natality refers to the number of births during egg, then period in the population that are added to the initial density. Mortality is the number of deaths in the population during a GN period. 3 at Nugroshyan is the number of industrial individuals of SR species that have come into the habitat from elsewhere during the time period Juncture consideration. Emigration is the number of individuals of the population who left the habitat and gone elsewhere during the time period under consider or alien. Pit pil 11 OADIN, I, so if N is the population density at Tiri T, then its density at Tiri T, I is in, N. B, NDE you can see from TLLE. Above equation TLL a population density will increase if Li number of births plus Li number of IML1 IGRMTSBI is more than the number of deaths plus the number of emigrants DE. Otherwise it will recurrence. Under normal 1 conditions births and CTE at less are the RMST important factors in one leaning population density. The other T100 factors assuming importance only under SF style conditions, for instance, MCEF. A new H habitat is just being colonisect. Immigration contribute more significantly to population growth than birth rates. Growth models. Does the growth of a population with Tiri show any species 1 icon predictable pattern? W have been concerned about unbridled human population growth and problems created by it in our country. MD. It is Lirat Baran Altaras to be curious at different animal populations in nature tenaili SR way or show sore restraints on growth. Perhaps lol ECM learned a lesson or two from nature on how to control population growth. I exponential grow L, resource rock, D and space AO availability is obviously essential F for the unimpeded growth of a population. Attorney when resources in Li habitat are unlimited each species has the ability to realize fully its innate potential to grow in number. As Darwin observed while developing his theory of natural selection, then the population grows in exponentially, L or geometric fashion. If in a population of size then the birth rate's not total number but 81,010 GY per cap, to births are represented as band death rates, in, per cap, to wreath rates as D. Then increase ER decreasing Tunga unit Tiri page 3 Rio, LTDNDT will be DNDT BDXN let BDR. Then DNDTR and the R in TLL's equation is called Li, intrinsic rate OFN Adderall increased N. Is a tie important P rarator chosen tower. S as sing impacts of any biotic or abiotic factor on population growth. To give you sore idea about the Tudo FR volus FBR the Norway rat the R is 0.015 and FBR the flower beetle it is 0.12. In 1981 the Arlu FBR human population in India was 0.0205, FTR1D out U, I at the CJ Rentar UB JES Fro, UQDIT, N, 9, T. Go need to know the B, RTH rotes and AOJH rotes, the above equation decelbes the exponential or GCRLC growth pattern of a population re 13.5 and results in a Shafod curve when plot N in relation to Tiri. If you are 3 reeling with basic calculus you can do LVE the integral FBRM of the Ponenchild-Goth equation as Fis, W, E 13.5 population growth curve when response is me not limiting the growth. Plot is experiment, 1b when response is limiting the growth PBT is low 8 SI 10 KTS Canatan Kappa and you know where N. Lapula ion density after I ran 0 population density at Tiri 0 R intrinsic rate of natural increase E the base off natural log, RTTHMS2. 71,828 any species grow wing exponenti 3 Lyonder Willemided, resource conditions reach animation ren cities and a short Tiri. DN Win showed how even, slow section O wing on M3 Laka elephant could reach generous numbers in E. Absence of checks. The following is an anecdote popularly narrated to demonstrate the arithmetical how fast a huge population could build up when growing Penenti 3 LLY. 1 mlo, Engart the Meerster Sulforo, Cheskomi 1 ml, Ing, Confo Dientof winning the 9 omi. Wo s read 9 to accept o bet proposed B9 the Meerster mum side lo if he wanted. On G some we at 9 raw, NS, TL equant, TGOFWD, S to J, EO added by DN9 on the chessboard. 19 row in square J, then wo in square 2, Sfmjo UJ in square 3, art, GR, T in square 4 on so on, 100 O, me the Priya is squanted 9 of U, L eat on the next square Ertlige. Art 64S, charges were filed, the L, I know, O Ted Platessim Sol JB Artstmik the 9 Omi Rotan 1 Uklo War 1 VM. The JMU 3 or 1, the L, in Ogel Thatfi DJ Bert 9. Orga, MA, O Popuatio I the MN Dotsters Bet UXXSO Esk, he started TTXTHSLE 9 GN the dot first square MD proceeded to foil, the other squares fo UOW, and Minsters suggest D procedure, but BGTL1J 12 he court Hoij TL1J chessboard. The low, R9 Re said to HSD, smag.aj, T the wheat proj J said FL highest entire KLN9 don pool to 9 ether would stat BLNO, dequade to Kuro, U the 64 squares. Now UDNK of a Tiang pora mequum ang with just 1, DOI dual, Ontlavau B, Nordfo.m, Drubling, and numbers E ergdag. At D, Mag, Nevu I at M, ND boggling pop daddy MC set at Woutreach, N64 do JS Puidi at food at D space Rima, and Ali M Tej UL, 9 I stick grow L. No population of any species in nature has its disposal limited resources to permit exponential growth. This leads to competition batlo, in individuals FBR limited Rezo says, Elentu, LLY the one it est individual will cerny 3 JLD reproduce. The governments of MMY countries have also realized this fact and introduced MES restraints with a view to limit population growth. In nature and habitat has enough resources to support FFI3 Zimmern possible number B in which no further growth is possible. Let us call this limit as nature's Kerg, Ankapru, TGK for that species in that habitat. A population growing in a habitat with limited resources show initially one phase. Slow by phases of acceleration and deceleration and finally an asymptote when the population density reaches a Cartan capacity. A plot of N in relation to T results in a sig-elded Qn. This type of population growth is called vanished per log stick growth figure 13s and is described by the following equation. DNDTRNKN where N population density at Timitar intrinsic rate of N Adderall increase KC3 Rhine capacity since resources tor growth tor RMST animal populations are finite 3 JLDB core limiting sooner or later. The logistic growth LDL is considered realistic 1. Gali from government census data the pop diet on figures for India for last years, Piat Thamanst, UELD Graux pattern SEOID end. 3 life history variation populations Evo Tomazamis TLL Reproductive fitness. Also called 03 Irwinian fitness higher value. In the habitat in which they live, under a particular set of selection pressures organisms EVDE to apps TLLENDST. 81,010 GY efficient reproductive strategy, organisms breed only once in TLL lifetime Pacific salmon fish. Balmux, while others breed many tiers during their lifetime most birds male are ROJLNUS, pro use a large number of small sea-set offspring oysters. Pale sea fishes while others produce a small number of large sea-set offspring birds marginals, which is desirable for maximizing fitness. Ecolo STS suggest that life history traits of organisms have evolved. In relation to TLLE constraints imposed by the abiotic malbiotic components of the habitat in which they LLE. Evolution of life history traits in different species is currently an important area of research being conducted by ecologists. L2, 4, population L can you think of any natural habitat on Earth that is inhabited just be, why a single species there is no such habitat. In such a situation is inconceivable. For any species TLLE minimal requirement is one more species on which it feed, in a plant species. Which N were says it's 01 NTL fixed, C armed slough R1 V3 lone, it needs soil microbes to CTANTLL organic matter in soil and return the inorganic nutrients for absorption. And then how will the plant age pollination without an animal agent? It is obvious TLL in nature animals. PL3 and TS. In microbes do not in C armit live in isolation but interact in various ways to form a biological corm WLID. Even in minimal corm WLIDs MMY interactive link, ages exist although all RNA not be readily apparent. Interspecific interactions arise from TLLE interaction of populations of two different species. They could be beneficial, retroental, or neutral, neither harm nor benefit to one of the species or both. Assign FBR beneficial interaction, C for detrimental, and 0 FBR neutral interaction. Let us look at all TLLE possible outcomes of interspecific interactions. Table 13.1. Table 13.1, Population Intention Species B00 Name of int, R, Action Muta T, SM Scene Peb, B, on Pred T Con P SM.con TCSM Omens TCSM Bot. TLLE SP3 size Terafit individually SM and Bethlus Pedidion in their interactions with each other. In both parasitism and predation, I1 species benefits parasite and predator respectively, ELJL, and the interaction is detrimental to the earlier species host and prey respectively, the interaction where one species is benefited and the other is neither benefited nor RUU, net is cruel commensalism, in immensalism on the other HN. One species is harmed whereas TLL the other is one all tect. Predation parasitism and commensalism's SHR. Corn JN crew rectenstic the interacting species LLE closely together. I predation, what while D happen to A, the ENER 9G FO ZBG autotrophic or 9 and SMS if the TG has no anima L. S to eat the plants. You can think of predation as nature's way of transferring to higher trophic levels the energy fixed by plants. When we think of predator and prey RMST, probably it is the tiger and the deer that readily come to our mind. But SPROE eating any seed is no less a predator, although animals eating plants are categorized separately as herbivores. They are in a broad ecological context not very different from predators. EZ.ES. Acting as conduits, energy TRNs for across trophic levels. Predators play other important roles. They keep prey populations under control, but FBR predators. Prey species could achieve very high population on densities and see ecosystem instability when certain neurotic species are introduced into a geographical area. They be core in VCE and start spreading fast because the environment tosnet high its natural predators. The prickly pear cactus intro, Lusect into Australia in TLLE early T92 S caused havoc by spreading rapidly into millions of hectares of our Angel 3ND. Finally the invasive cactus W was brought WLDR control only after a cactus, feeding predator LDTH from its natural habitat was introduced into the country. S0, 9 Kyle, Con, tetrapods adopted in agricultural pest control are based on the ability of the predator to regulate prey population. Predators also help in maintaining species diversity and corn WLED by reducing TLLE intensity of COLLFII on our link prey species. In the rocky intertidal communities of TLLE or Urkin Pacific 03 as TLLE street starfish Pisaster is an imp. Predator. In field expectifront when all TLLE starfish da area remect friend in enclosed int, relidal area. AATLWL10 species of invertebrates became extinct within YER because of inter-SF, civic competition. A predator is too efficient and overexploits its prey, then the prey might be core extinct and following it. The predator will also be core extinct FBR lack of fixed. This is TLLE reason why predators in nature are prudent. Prey species HA vero viardus retensis to lessen the impact of predation. Sarah species of insects and trogs are critically jerdum, glitch being detected easily by the predator. Sarah P. Isonus and therefore avoided by the predators. The monarch butterfly is shylitus testful to its predator. 81,010 GY bird because of a special chemical present in its body. Interestingly, the butterfly acquires this chemical during its caterpillar street by feeding on a poisonous weed. For plants, herbivores are the predators. Nearly 25% of all insects are known to be top WBS feeding on plant sap and other parts of plants. The problem is particularly sad, Airphi, Junior PLMTS because like animals cat all run away from. TLL are predators, plants their fee, JRE hail evote an astonishing V-neti of morphological and chemical defenses against herbivores. Thorns cacas are the RMST common morphological reins of defense. Many plants produce and store chemicals that eat the herbivore sick when they are eaten. Inhibit feeding or digestion, disrupt its reproduction or even kill it. You must have seen TLLE weed callow, tops towing in abandoned fields. The plant produces highly poisonous cardiac glycosides and that is why you never see any cattle or goats browsing on this plant. A wide V-neti of chemical substances tladlol-E.O, tracked from plants on a corner, commercial scale nicotine. Coivine, quinine, suicnine, opium etc. are produced by LEAM actually as references against GRNAR's MD browsers. Competition, when Darwin spoke of the struggle Junior, is tense and survival of the fittest in nature. He was convinced TLL at interspecific competition is a potent force in organic evolution. It is generally that competition occurs when closely related species compete to FBR same resources TLL are limiting. But TLL is not entirely true. Firstly, totally unrelated species could also compete for TLL same resource. For instance, in shallow South Oregon lakes sighting flamingos and resident fishes compete their corn food. The zooplankton in the lake. Secondly, resources need not be uniting for competition to occur, in interference Italian. The feeding at XII of one species MGHT be reduced due to the interfering and inhibitory presence of the other species. EI jet live resources D and space are of WLDAAT, therefore. Competition is defined as a process in which TLL fitness of one species measured in terms of its TLL intrinsic rate of increase is significantly larger in presence of milder species. It is relatively easy to demonstrate in laboratory experiments as Gosendal experimental ecologists did. When resources are limited, the competitively sub-3 rare species will eventually emanate the earlier species. But evidence for such competitive exclusion occurring was not always conclusive. Strong and persuasive circumstantial evidence exists however in some cases. The Abingdon tortoise in Galapagos Islands became extinct within a decade. 3 turgoats were introduced on Iceland. Apparently due to the greater browsing F16 of the goats. Another evidence the occurrence of competition in nature course from what is called competitive release at a species whose distribution is restricted to a small geographical area because of the presence of a competitively superior species. Is found to expand its distributional range dramatically when the competing species is effortmentally window. Nell's elegant field appearance showed that on the rocky sea coasts of Suland, the larger and imperatively superior barnacle Beloneus dominates interdid 3L. area, and excludes smaller barnacle Chathamalis in that zone. In general, herbivores and plants appear to be more adversely affected by competition than carnivores. Goss's comp. Itwelptis on Prime J.I. states that D100 closely related species competing for LISARRE resources C3 Ju10T coexist indefinitely and the competitively inferior one will be eliminated to ally. This may be true if resources are limiting but not otherwise. More recent studies do not support such gross generalizations about competition. While they do not rule out the occurrence of interspecific competition in nature, they point out that species-focusing competition might EMLE mechanisms that promote coexistence for earlier TLWL exclusion. One such mechanism is resource partition yield. If two species compete for LISARRE resource, they could avoid competition by choosing FBR instance different tiers for feeding or different FBR patterns. MacArthur Schall, D. that 5 closely related species of warblers Lil, LNG on the SARRE tree were able to avoid competition and is due to behavioral differences in their foraging activities F. tacitism, Q. NCERT parasitic mix of life ensures free lodging and reals. It is not certain that parasitism has evolved in so many taxonomic groups from plants to higher vertebrates. Many parasites evolved to be host-specific. They can parasitize only a single species of host in such a way that off-host MD. parasite tend to co-evolve. That is if host arrow species 31 Reg 3 nism ta rejecting or resisting parasite. Parasite is to arrow Reg 3 RLism to cowl terra CTMD neuteralis in order to be successful vili LISARRE host species. In accord, 3 severely linear lifestyles parasites evolved special.aptatim such as loss C1. Necessary sensorum's presence of adsive organs or suckers to cling on to the host loss of digestive system and high reproductive capacity. The life cycles of parasites are C10 compolinvolving 1 CC2 intermediate hosts or vectors to facilitate parasitization of its prill host. The human or nuka trematode parasite depends on 2 intermediate hosts, a snail and a fish, to complete its life cycle. The malarial parasite needs a ectoderm squido to spread to other hosts. Majority of the parasites, 81,010 GY, harm the host. They may reduce the survival, growth and reproduction of LE host and reduce its population density. They might tender LE host RR, reluller able to predation by NW, get physically okay, do go belly EOE trot on idea. Parasites will be able to bear with V and the host without more miflo W than X1 IJDD and T9, Wa, said on led to the EOO adon of subtod algomias parasites PRSite sulet feed on Leextem alsurf.ace of Lee Hostorg 3JLism or collect data parasites. The most familiar examples of this group are Lee lice on Hung1 SMD ticks on togs, MMY man nefish. Are infested with ectoparasilic copepts. Coscudo, a parasitic plant that is usually found growing on hedge, Plants has lost its chlorophyll. In leaves in TLLE course of evolution. It rents its nut nuon from the host PLNT which it PRC uses. The Phe, RN3 mosquito is not Kanzai red. A parasite. Leo it needs blow. D for reproduction, can go explain WLVJ in contrast. Endogress. Are those who live inside TLLE host body at different sites, liver, kidney, lungs, red blood cells, etc. The life cycles of ectoparasites. Rm, recomplex because of TLL or extra specialization. Their morphological and anatomical features are grow simply while emphasizing their reproductive potentially 3. Bruparacete smin birds is. Epicinatinge example of parasitism in which PR, acidic bird, lays its eggs in TLLE nest of its host MD lechly host incubate them, during TLLE course of evolution. TLLE eggs of the parasitic bird have arrow to resemble the host's egg in size and color to reduce the chances of the host bird detecting the foreign eggs and ejecting them from the nest. Try to follow the marents of the cuckoo coil and the crow in your net, boarhawk Paul, done the breeding season spring to summer and watched breed. Parasitism in action, W. Canon NCERT. This is the interaction in which one species benefits and the other is neither harmed nor benefited. An orchid wing is an app, PLVJTE on a mango branch and barnacles growing on the back of WHL benefit while neitherly mango tree nor WHL there less any apparent benefit. The cattle egret and grazing cattle in close association a site you are RMST likely to catch if you will in FARD. FULROS is a classic ample of commensalism the egrets. Always for 3L, equals to where TLLEC addle. GR3 because the cattle, as they move, stir up and flush out from the vegetation insects that otherwise might be difficult to the egrets to find and catch. Analyr. Amply of commensalismusly interaction beta insea. Anon DN tlad H is stinging tent, echoes MD Lee CLONTL. Fish TH, at Organisms and Evolution of E. Figure 13.6 Mutual relationship between fig tree and wasp. A fig flower is pollinated by wasp. Bee wasp laying eggs and a fig fruit lives among them. The fish gets protection from predators which stay away from the stinging tentacles. The anemone does not appear to derive any benefit by hosting the clownfish. V. Mutualism. This interaction confers benefits on both the interacting species. Lichens represent an intimate mutualistic relationship between a fungus and photosynthesizing algae or cyanobacteria. Similarly, the mycoteethesi are associations between fungi and the roots of higher plants. The fungi help the plant in the absorption of essential nutrients from the soil while the plant in turn provides the fungi with energy-yielding carbohydrates. The most spectacular and evolutionarily fascinating examples of mutualism are found in plant-animal relationships. Plants need the help of animals for pollinating their flowers and dispersing their seeds. Animals obviously have to be paid fees for the services that plants expect from them. Plants offer rewards or fees in the form of pollen and nectar for pollinators and juicy and nutritious fruits for seed dispersers. But the mutually beneficial system should also be safeguarded against cheaters, for example, animals that try to steal nectar without aiding in pollination. Now you can see why plant-animal interactions often involve coevolution of the mutualists, that is, the evolutions of the flower and its pollinator species are tightly linked with one another. In many species of fig trees, there is a tight one-to-one relationship with the pollinator species of wasp. Figure 13.6. It means that a given fig species can be pollinated only by its partner wasp species and no other species. The female wasp uses the fruit not only as an oviposition egg-laying site but uses the developing seeds within the fruit for nourishing its larvae. The wasp pollinates the fig inflorescence while searching. Figure 13.7 shown be a pollinator on orchid flower Iology for suitable egg-laying sites. In return for the favor of pollination the fig offers the wasp some of its developing seeds, as food for the developing wasp larvae. Orchids show a bewildering diversity of floral patterns many of which have evolved to attract the right pollinator insect bees and bumblebees and ensure guaranteed pollination by it. Figure 13.7. Not all orchids offer rewards. One Mediterranean orchid offers employs sexual deceit to get pollination done by a species of bee. One petal of its flower bears an uncanny resemblance to the female of the bee in size, color and markings. One male bee is attracted to what it perceives as a female, pseudocopulates with the flower, and during that process is dusted with pollen from the flower. When this same bee pseudocopulates with another flower, it transfers pollen to it and thus, pollinates the flower. Here you can see how coevolution operates. If the female bee's color patterns change even slightly for any reason during evolution, pollination success will be reduced unless the orchid flower co-evolves to maintain the resemblance of its petal to the female bee. Summary. As a branch of biology, ecology is the study of the relationships of living organisms with the abiotic physico-chemical factors and biotic components other species of their environment. It is conceived with 4 levels of biological organization: organisms, populations, communities and biomes. Temperature, light, Water and soil are the most important physical factors of the environment to which the organisms are adapted in various ways. Maintenance of a constant internal environment homeostasis by the organisms contributes to optimal performance. But only some organisms' regulators are capable of homeostasis in the face of changing external environment. Others either partially regulate their internal environment or simply conform. A few other species have evolved adaptations to avoid unfavorable conditions in space migration or in time estivation, hibernation, and diapause. Evolutionary changes through natural selection take place at the population level, and hence, population ecology is an important area of ecology. A population is a group of individuals of a given species sharing or competing for similar resources in a defined geographical area. Populations have attributes that individual organisms do not: birth rates and death rates, sex ratio and age. Distribution NLE proportion of different G, ROBs of MS FOM, GES LN at populi 11 on LS often tessin to grapple 4 CUIS, pair or D, its shape decales a population 1S street Asian my. GDOling or declinling, ecological F-tex of my factors on a population regen, ner lyri, flected in isles S1 z populi 11 on dens, iti. Which may be expressed in different ways: numbers B1 or MSS per R1 TC, OV, R etc., depending on Ilias spec 1S. Populations grow true 08 H baths on LMM, gration on decl new deaths.md aragra 11 on, WLML. Resources me unlimited. The growth is usually exponential, but when resources become progtas 1, lilaroiding. Growth pattern turns logosok, in EIU, case growth LS alternately united by the rim capacity of the environment NLE1 NLL1 NS1C rate of natter 1 and CRE 3 SER 1 SME 3 sure of the inherent potential of a population to grow. In nature, populations of different spe: 1s in it do not 11 inches sol 11 on but interact in MMY ways. Depending on E outcome ILE is INLL, reactions bet VE, N2 species me disfleti as competition both SF, S suffer, Preda 11 on ontpo, Rostalism 1 benefits I the oilier suffers. Commensalism 1 benefits ilia other is unaffected, M.D, Salism 1 1s1 medot erected mutualism boal. Spe 1s bereft, predation 1s array 1 port, 3 NL process UA which theropic E, nergitra, NS, for 1s FCC1 litated.md some predators telp 1 and contra 111 and theta prey populations. PLMLS Hyavadi diverse morphological and chemical defenses Ag 3 inst He, RBI, REHL compu U on TLTS presumed dulet U, E soup, Retor competitor L1 runets U, E inferior 1 DO, BDU exclusion prink 1 plec. But MMY closely related species have E.all.E, L, Mus S which fac 1 litit ili 1 are coexists team. Some of the most fascinating cases of mutualism in nature me seen in PL map polynol are interactions. 1. FR, S, VV, 1, how is diapause diff, re, NT from hibernation 2. If a M6SH is placed in a freshwater aquarium, will the fish be able to survive or why not 3? 9. Phenotypic adaptation, G1 page 1E4, most living organisms can survive at temperature above 4 soc, how many some microbes able to live in habitats with temperatures E, relating IOOC. 5. List the attributes that populations but not individuals possess. 6. If a population growing E, pong, initially double in size in 3 ms. What is the intrinsic rate of increase R of the population? 7. May import, 3 NT defense mechanisms in PLCM1S against He, Rabiri. 81,010 GY8. An orchid PL3 NT is growing on the BRCMCH of O3. How do, so describe this inter-action between the orchid the entry 9. WH, TSMology E.L premip H, here M bilegal, J, all RMTLD of M with pest insects 10. Distinguish bet, NTMF following, AJ hibernation P, estivation BE autothorm S, NDE, malthorm S 11. Write a short note on AJP adaptations of desert PBNTS ALS BP adaptations of PL3 NTS to water scarcity C behavioral adaptations in ALS D importance of light to PL, NTSE effect of 10, RNP, RHR or water scarcity the adaptalians of S. 12. List of EMIAS abiotic E, environment Intel factors 13, GELH Vimple 4, anondomormic or imel B. An ectothermic alc. An orgasm with benthic zone 14. Net population. And community 15. Net following terms bear 1 E, Mple for each, AJ Comrani, Salism BPN City SMC Con Alpha, Jetty Mutualism E interspecific corn petition 16. With the help of suitable diagram describe the logistic population growth curve 17. Select the statement which is best pensatism. AJ Oni Arges MISP, Nefi Ted be both the armisms me benefited C Ono Elism is Benny, Ted. Other is rejected D1 or lism is Benny, TED other is .3 affected 18, list.my 3 imb, RT, MTCH mact, ristics of a population E, PLML. Chapter 14 Ecosystem 14.1 Ecosystem TITS Meont PT, OL XII 14.3 Decustion 14.4 Energy Tau J4. 7 Nitrogen Sig, 14.8 Ecosystem Serodos an ecosystem can be visualized as functi and 3.1 WL.IT of NatWe. Where LLing org, ni SMS interact and also with the surrounding physical and environment. Ecosystem varies greatly in size from a small pond to a MGE breast or a sea. Many ecologists regard the entire biosphere as a global ecosystem. As a composite of all local ecosystems on E3RTH, since TLL's system is too much big. MCT cornpole, to be studied. It is immediate to divide it into two basic categories, namely TLL TLT1L and the aquatic. Forest section is London desert or sorry, amples of terrestrial ecosystems, pond lake V, ETL3 NCT. Enter. And S23 tie. Or sorry. Apples at aquatic ecosystems, crop fields and an aquatic one may also be considered as man-made ecosystems. We will first look at the structure of the ecosystem in order to appreciate the input productivity. Transfer of energy food chain A, B nutrient cycling on the output CE section adds ion on energy loss. We will also look at the relationship cycles CH3 and V, EBS TLL, at are created. As a result of T heat energy US Willian Lee system. MCT Lear interrelationship. 810 L0GY14, 1 FCOS YS1, MSRRRT, TIJRET, F. Junction in Chapter 13 UI looked at the various components of the environment abiotic and biotic. You studied how the individual biotic and abiotic factors affect each other and their surround and let us look at Lee's Corp. 111 and AAE integrated in Wooler. And see how the flow of energy L3 has place within Lee's components of the ecosystem. Interaction of biotic and abiotic components result in a physical structure that is characteristic for each type of ecosystem. Lithification. Generation of PL, and MIMAL species of M ecosystem gives its species composition. Vertical distribution of different species occupying different levels is called Chayaduka Yuan. 4. Ample trees occupy top vertical strata or layer O. TAF forest shrubs the second and herbs and grasses occupy the bottom layers. The COMP 111 ends of TLLE ecosystem. Are seen tough WL XII as willet when you consider the following aspects: I productivity, U composition, 3 energy now, and IOG nutrient cycling, to W Elderst and Lee Elias of an aquatic ecosystem. Let us L3K. A small pond is an implec. This is fairly a self-sustainable lid and rather simple example that explain even the complex interactions that exist in an aquatic ecosystem. A pond is a shallow water in which all the above mentioned basic components of M ecosystem are well divided. The abiotic COMP 111 and is water with all TLL dissolved in organic MD organic substances and the rich soil deposit at the bottom of the pond. The solar input, a cycle of temperature, day length and other climatic conditions regulate the rate of function of the entire pond. The eutrophic components include the phytoplankton, some algae and the floating submerged and marginal plants round at the edges. The customers are represented by the plank to the free-swimming and bottom-dwelling forms. The decomposers are the fungi. Bactin and flagellates especially abundant in the bottom of the pond. This system parents all the functions of any ecosystem. N of TLLE biosphere is a whole that is. Conversion of inorganic into organic material with the help of the radiant energy of the sun by the autotrophs. Consumption of the autotrophs by holotrophs. Decomposition and mineralization of the dead matter to release them back for reuse by autotrophs. TLLE's event. Are repeated over and over again. There is unidirectional movement of energy towards the higher trophic levels. And its dissipation. N loss. Is he. To Lee and their onland 14.2. Productor 1. Instant input of solar energy is the basic requirement for any system to function and system. IP. Autocton is defined as the amount of ECO-BTEM biomass or organic matter produced per unit. Over a period by plants during photosynthesis. It is expressed in terms of weight Rg or energy LTCM. The rate of biomass production is CD productivity. It is expressed in terms of G, your or cal m, your, to compare the productivity of different ecosystems. It can be divided into gross and Y product GPP on net and Y productivity NFFL. Gross massy productivity of an ecosystem is the rate of producing ion of organic matter during photosynthesis. A considerable amount of Ga is utilized by plants in respiration. Gross and Y productivity minus respiration losses are is the net massy productivity Na, GPPR NPP net and Y productivity is the availability, biomass FBR the consumption to heterotrophs herbivores and decomposers. Secondo Y productivity is defined as the rate of formation of new organic matter by consumers. Friendly productivity depends on the pion species inhabiting a particular area. It also depends on a netty of environmental factors. Availability of nutrients and photosynthetic capacity of plants, theraphoid and different types of ecosystems. The 3.1 LUL net PLI1 nary product of the whole biosphere is apro, iron 170 billion tons country weight of organic matter. Of this, despite occupying about 70% of the surface, the productivity of the oceans are only 55 billion tons. Rest of course is on LN, DLSQSS the Ma, and reason for the to produ D Witgavosi and UFKWR. 14. 3DERWCSN1 on, you may have heard of the earth farm being referred to as the farmer's friend. One says so because they help in the breakdown of complex organic matter. As low, L as in loosening of Lee soil. Similarly, decomposers break CBL nil compt, org, aeric matter into inorganic substmix like CR undioide. W8 nutrients. Lee process is called decomposition, dead plant RNA and such. As leaves. BARC, ores and dead remains of animals including fecal matter constitute detritus, which is raw material FBR decomposition. The important steps in TLLE process of decomposition are fermentation, leaching, catabolism, humidification and mineralization. Deader, UVORs for example, HWCR embryoc, and I11 us into cernol, particles. This process is called fragmentation. By the process of leaching water soluble in rugnutnants got one into the soil horuan and get precipitated as unavoidably, salts. Bacterial and fung, enzymes degrade J11 us into simpler and oigenic substances. This process is called as catabolism. It is important to note all the abm steps in decomposition operate simultaneously on the J11 us figure 14.1. Humidification and mineralization occur during decomposition in the soil, on leads. Leaves partially consumed by decomposers such as fungi, some are eaten by insect and other animals. Nutrients and CNC, food web, a green leaf falls to the ground. Biology and bacteria. They begin to lose form and become litty, Some nutrients leach into SU by chemical action. Figure 14.1 diagrammatic representation of decomposition cycle in a terrestrial ecosystem to accumulation of a dark-colored amorphous substance called humus that is highly resistant to microbial action and undergoes decomposition at an extremely slow rate. Being colloidal in nature, it serves as a reservoir of nutrients. The humus is first degraded by some microbes and release of inorganic nutrients occur by the process known as mineralization. Decomposition is largely an oxygen-requiring process. The rate of decomposition is controlled by chemical composition of detritus and climatic factors. In a particular climatic condition, decomposition rate is slower if detritus is rich in lignin and chitin, and quicker if detritus is rich in nitrogen and water-soluble substances like sugars. Temperature and soil moisture are the most important climatic factors that regulate decomposition through their effects on the activities of soil microbes. Warm and moist environment favor decomposition, whereas low temperature and anaerobiosis inhibit decomposition, resulting in buildup of organic materials. EcoVTEM 14.4, LMRGYLLOV except for the deep CHJ, LRO thermalia a system STM is the only source of energy FBR all ecosystems on Earth. Of the incident solar radiation, less than 50% of it is photos and nucleoactive radiation PAR. We know that plants and photosynthetic and chemosynthetic bacteria autotroph 6 times sun's radiant energy tomok D from simple inorganic materials. Plants capture only 10% of the PAR, and this small amount of energy systems finliral, also. Is important to know how the solar energy captured by plants 11 outs through different organisms of a system. All organisms are dependent for their food on producers. Ether directly indirectly, so you lind will it directly 0113.1 with energy from TLLESWL to producers.n. All to consumers, is TLISNL, KEPIF, you have the forced law of therm, DGNOM, CS further ecosystems A not D, BM'd from TLLE second. Law of therm, ZLIN, RNX, they need a constant supply of energy to synthesize the molecules they require. To counteract the universal tendency toward increasing dicertin SS, the green plant in the EA system terminology are called producers. HLA Terra star I3.1 ecosystem Nuger producers are herbaceous. And Wo, di plants, Euquise Priffy white producers in aquilic ecosystem. RV Meus species like phytoplankton algae and higher plants, you have react about the B, DCHS and EBS that EO in nature. Starting from the plants or producers B, D chains or other BS are formed such that an animal feeds on a plant or on another animal and in time is fixed. FBR. The CH3 or web is formed Tika use of this interterpanctancy. No energy TL13D is tropect into an org3JLism remains in it for Eller. The energy trop head by Lepro, TUCR hence is Elir posset on to a consurer Li Orgarus dies. DTLL of organisms Liebigierling off that Elredusn C web. Animals depend on plants directly or indirectly FBR there be, D needs. They are Hunskul consumees and also heterotrophs. If the feed in the producers the plants they are called Mycomswinners. And 2 the mammals eat other mammals which in time eat the plants or their produce Li called second, 3 Consumers. Eukarys, well could H3 Veterati consumers 2, obviously Li Prior 11 Ari consumers will take herbivores. Sercarun JN herbivores Ari insects birds ANCTM3 NLLL 13 LS in terrestri 3.1 ecosystem.n. Gluskin. Aquilic ecosystem the conswinners TL13T feed on leaves herbivores or carnivores. Or Reoractly JY carnivores though secondary consumers, those animals that depend on the primary carnivores for B, D are labeled secondary carnivores. A simple GROsing B, D CHO and GFC is depicted below, GRA SS, CBAT, Mayan producer primary consurge secondary conswinner. 810L0GY the detritus root chain DFC beg, NS with dead or gruk matter, ills made up of decomposes which, reheterotrophic organisms. Mainly Fungi bacteria, they lead theorem JIRD hull 1F at re, JRFLHTS by degrading dead organic matter or detritus. These are also KNONTL saprotroph so pro, to decompose. Decomposers secrete digestive enzymes that break wounded waste materials into simple inorganic materials which are subsequently sorbed by them in aquatic ecosystem. GF major conduit TBR energy flow. Gattensthin, terrestrial system, much larger fraction of energy flows through the detritus FCX, D-chom than through the GFC. Detritus FCX, D-chain may be connected with the grosm B, D-chom, T-sorlelels, scra of the organisms of DFC are prey to the GFC animals. RDN, natural ecosystems or animals like cockroaches, crows etc. are omnivores. These natural interconnection of O, D. CA chains make it a food web, how while D go nos if human beings, organisms occupy a place in Li and natural ROW eldings or in cornrunity, according to their feed dingrel, shyanship with other organisms. Set on Li source of their nutrition CC fixed organisms occupy a specific place in the FCX, D trump feed is known, as their trophic level. Wu users belong to the first trophic level herbivores primacy insurge to the SIA and MD carnivore SIA ondity consumer to the third figure 14. 2 TTTZ, JMTMR1 HB11 SAW, Fig WE, 14.2 Tagern Ma 11C representation of trophic TEDS1 anim ecosystem. EcoVTEM the IMB, written P ointment to note as TLL at TLLEMD unit of energy decreases at successe trophic liels. When any organism dies it is converted to CL311 to sorted L, by Armstead Senesis, Menergy source the CDFLL, XJ source. Organisms at each trophic level depend on those at the LOL, our trophic level J are their energy demands. Each trophic level has a certain mass of LL Vingranate and L adiparticle, MTRI called as the standing crop. The street, DIN crop is measured as the mass of living organisms biomass or the number in a WL.IT area. The biomass of a species is expressed in terms of fresh or Y, 1/8. Measurement of biomass in terms of TR weight is more accurate. TNHG. The number of trophic levels in the GRAS, NB, XL chain is restricted as the transfer of energy follows I0% law. I0% of the energy is transferred to each trophic level from the law, R trophic level. In nature it is possible to have some ILLs pro, TUCR herbivore pre-1 I weak carnivore secondary camera in the GRAS, Toxel chain figure 14.3. To go TNNL, MRE, Sang Sud limited on an adentus food and you must be familiar with the shape of a pyramid. The base of a pyramid is broad and it narrows DCNJN at TLLE apex. 1 TS a similar shape Wetler Ho expressed TLLE FO, DR energy relationship at 1 in organisms 247. 810 L0GY. At different trophically, LDAS relationship is expressed in terms of number biomass or energy. The base of each pyramid represents TLLE pro, Lucers are the first trophic level while the apex represents Ursha, JI or Topli L Comsrer. The 3 eological PJ Rornids that are USU3LLY studied are a pyramid of number, B pyramid of biomass and C pyramid of energy. For retail CFIRI 14.4a, B, C and D, is CLRWLWEFT 57WLEPCM, RJL, S3.54, 000, 70, 11, 000, 6, 34, 1, 000 Fig. W. E14.4 appear NID of numbers 1 Anagrast MD ecosystem. Only UUE top canwers me supported 1 Anam ecosystem ba, say, L on production of NAML 6 mil 11 ons PL3 and TSN, IJCPC pages 1.11 1137 feet. 14.4b Pirm, all B1 Ornis shows a short deco os LNB1 Ornis at HJ8 her trophic tails PC, IR, 1, UFP. Full stop, I4 Lydia Ur 14.4 CLML, Pirm, all B1 Ornis from Wall Street MDLNGE, op of FitOpl, ton supports ARB. Stored Ni, op all Zupakton. LJ 100:1 1000:110, 000:1 Fig, Ur 14.4 D1, NI Peer MODAV, NERG, OBS, RE that ma, Reproductors com, RT only 1 of Ilya energy 1 and your last 1 light available to you, M into NPP any calculations of energy content. Biomasaur numbers has 2 insular all organisms at that trophic level, no generalizations make will be true if take only a few individuals at my trophic level into account. Also a given organism may occupy or THM1 trophic level simultaneously, sly, 1 RRU street remember that the trophic level represents a funcional level. Not a species as such, a given species may occupy or THM1 trophic level in the soar ecosystem at the soar IR, for example. A sparrow is a primary consumer when it eats seeds fruits peas and a secondary consumer when it eats insects and diorphus can go work out momong. Topic ELS1WM, RN beings junction at NA Fudian and RMSTEA systems all the pyramids of number of energy and biomass are upright that is, producers are M, we inert terra and biomass TLWLTLLE herbivores. And herbivores are M, we inert terra and biomass TLWL the cambiores. Also energy at a lower trophic level is always M, re THM at a higher level. There are exceptions to TLL is generalization, it ULR to quality number of insects feeding on a big tree. What kind of pyramid would you GT? Now at an estimate of the number of small birds depending on the insects is also number of larger birds eating smaller. Draw shape you would get, the pyramid of biomass in C is also generally inerted because biomass of fish is F3RE, seeds that of PHJ top plankton. Isn't that a paradox how I'll go explain TLVS pyramid of energy is always UPRTGHT canellerte inverted. Techos when energy flows from a particular trophically 1L to the next trophic level, some energy is always lost as heat at each step. Each bar in the energy pyramid indicates the amount of energy present at each trophically 1L in a given terra or annually per unit area. 24. 810 L0GYHCN ever TLLR. Are certain limitations of ecological PI. Amid such as it S not taken to account TLLESR species belonging to 2 or more trophic levels. It, sirs, simple foe, RL cho in sorrel hingdat, Rost never ists in nature it xej not com, D, 8 foe, D. Web, moreover S, profites or not G, then any place in ecologue, cal pyramids even though they play, W role in the ecosystem UH, Velernt and CH, Chapter 13. The core characteristics of population and community MD. Also their response to environment and how such RESP 011 says V3F from an individual RESP, LLL said. Let use examine a clear aspect of corn which responds to an ornament over time, an iron important CHR, characteristic of all communities is TLL, at composition N. Structure constantly change in response to TLLE changing environmental conditions. This CHFL is octary and sequential. Parallel with the changes in the physical environment, these changes led finally to community TLL is in near equilibrium with TLLE and R1 Ronratan. T is called, e-community, the GRAC2, L and fairly predictable change in TLLE species composition of a given area is called ecological succession, DU for success science or species colonized in area in TLL or populations T or Roenurus. Whereas Pula ions of other SJE sized to C19 and L and D is a PPE are the entire sequence of cornwalities that successively change in G, then or called CRS. The indole transitional Lumruni IES or tered Seral stages or Seral cornwalities HL. TLLE successile Seral stages TLLR is a channel Eiler city of species of organisms increase in the Nernter C1. Species and organisms as well. As 311 increase in TLLE 1 bionas, the present take cornwalities in TLLE world. Have come to because of succession TLL it has occurred over millions of years since life started. On Earth, actually succession and evolution would have been parallel processes. At TLL at Tiri. Succession is hence a process TLL. It starts where no living organism s. RTLL or TLLs could be areas where no living organisms ever existed. S. Y. Rock, or in or is that somehow lost. U. The living organisms that existed there. The former is called primary succession while TLLE latter is termed secondary succession. V. Amples of areas where IV succession occurs. Are newly cooled. L. Have a bare rock newly created pond or reservoir. The establishment of a new biotic community is generally slow. Before a biotic community of diverse organisms can or established there must be soil, depending on in the climate. It W.'s natural process is several hundred to several thousand years to lose fertile soil on rock. ECOVTEM second, 3 Rise succession begins in is where an natural biotic communities have been destroyed. Such is in abandoned farmlands burned or cut forests LMDs that have been floored, since soil or sediment is present. Succession is a faster TLWL Britain Y succession. Description of ecological succession usually focuses on junction vegetarian. However these agility 0113.1 changes in turn affect food, NCT shelter for various types of animals. Thus succession precedes the nurtures. In types of animals. In decomposers. Also change. At any time dirt pl ironary or sequential succession natural or who will induce disturbances fire. Redress. On etc. See and convert a p article r sur al street age of succession to an her stage. Also such disturbances create new conditions that encourage or species. And disco. Age or eliminate or species. 14.6 L succession of plants set on nature of habitat wetler it is water or very v. It is or it is on eric tree. Succession of plants is called HDR acres or arch respectively. Hydro succession L has place in V. Eder. R. is in the succession 3 L senas progress from HDRTC Tarther secundillions. S. Goms TLL is zero succession L has place in dry. R. Is in research progress from xeric to resic conditions. Hence both Hector arch. And zerach successions to medium water conditions were tesic neither to dry xeric nor to wet HDRTC. The species V3D. A bear. R. See all pioneer species HL primary succession on rocks east. Are usualigens which are able to secrete SIDS to dissolve rock. Helping in we, 13 and soil formation, these later their way to soar various SM3LL plants like bryophytes, which are BLE to take hold in the SM3LL, or month of soil, they, with layer succeeded by bigger plants. NCT oil or several stages. Ultimately, stable Clim3XF forest community is roared the climax community Remom Street, Blair, S long, the environment remains unchanged with layer the Xerophile C habit, T gets submerged into, mesophilic 1. In primary succession in water TLLE pioneers. Are the SM all phytoplanktons they, replaced with layer by free-floating angiosperms. Then by rooted hectrophyte cyclages GR, says and finally the trees, the climax. Again would be a forest. With Cherry Lee W8 or T is converted into land figure 14.5, in secondary succession TLLE species and V3D depend on the condition of soil availability of waterly environment as. Also the seeds or other propagules present. Since soil is already there the rate of succession is much faster. And hence climax is. Also react. Quickly, WH it is important to understand as TLL, at succession P articularly primary succession. Is. A very slow process making my TLL and S of 2-1. 212 phytoplankton submerged plant stage submerged free-floating plant stage forest reed swamp stage marsh meadow stage scrub stage figure 14.5 diagrammatic representation of primary succession biology years for the climax to be reached. Another important fact is to understand that all succession whether taking place in water or on land proceeds to a similar climax community the mesic. You have studied in class that organisms need a constant supply of nutrients to grow, reproduce and regulate various body functions. The amount of nutrients, such as carbon, nitrogen, phosphorus, calcium, etc., present in the soil at any given time, is referred to as the standing state. It varies in different kinds of ecosystems and also on a seasonal basis. What is important is to appreciate that nutrients which are never lost from the ecosystems, they are recycled time and again indefinitely. The movement of nutrient elements through the various components of an ecosystem is called nutrient cycling. Another name of nutrient cycling is biogeochemical cycles. Bio, living organism, geo, rocks, air, water. Nutrient cycles are of two types, A gaseous and B sedimentary, the photosynthesis terrestrial food CH1NS photosynthesis B. Aquatic food CB, AIN C021NT, water, burning of forest fuel wood and organic debris. XII, LT, combustion of fossil fuels for vehicles, electricity and heat. Coal. Figure 14.6 simplified model of carbon cycle in the biosphere. Reservoir for gaseous type of nutrient cycle, e.g. nitrogen. Carbon cycle exists in the atmosphere, and for the sedimentary cycle, e.g. sulfur and phosphorus cycle, the reservoir is located in Earth's crust. Environmental factors, e.g. soil, moisture, pH, temperature, etc., regulate the rate of release of nutrients into the atmosphere. The function of the reservoir is 810 L0GY2M, at Whitney deficit, which occurs due to Toynbee 3 and suddenly rate C1 influx. Tank to mux. You have one IWLE do ed study of Ardoj, Iticle Iltlas I. Here we discuss carbon on phosphorus cycles. L4.7.L, Kuistum Cal, Bonsiakov. When you study the sillion of living organisms, carbon constitutes 49% of dry weight of organisms and is next only to water. If look at the total quantity of global carbon, find that 71% carbon is foul dissolved in oceans. TH's oceanic reservoir relates Li 3 nt of carbon dioxide in the atmosphere. Figure 14. 6. Dugomo that the SBLORONL9 contains AFX, % QF total Goxr carbon fossil fuel. Also represent a reservoir of carbon. Carbon cycling occurs through atmosphere, ocean on through living on dead organisms. According to one estimate, 84x1013 kGof is FIX in the biosphere through photosynthesis nearly. A considerable amount of carbon returns to the atmosphere as CO, through respiratory activities of the producers and consumers. Decomp.Source. Also contribute substantia light to CO, pool by their processing of waste materials on dead organic matter of land or oceans. SOR, E3 of the FIX CRUN is lost to sediments on the road from circulation fuming of VO, D, F forest fire on Jumbuslian of orgs regelic matter. Fossil fuel volcanic. Activity.R, additional sources FBR releasing C02 in the atmosphere. Hern Activilisha 1ES, Gin, Chikmui influenced the carbon cycle. Rapid deforestation en masse C1E burning of fossil fuel F4 energy on TRMSP, RTH, AEC section Lificantly incurusedly R8 of relative carbon dioxide and totally atmosphere. See greenhouse effect in chapter 16. 14.2e, a system phosphorus SID. Phosphorus is a major constituent of biological membranes. Nucleic acids and cellular energy transfer systems. Many NMOLs also need large quantities of TLL as inherent to NWTE shells. Bones and teeth. The Adderall reservoir of phosphorus is rock which contains phosphorus in the F form of phosphates. When rocks are low, around subly E phosphates dissolve in soil solution and are absorbed by the roots of the plants. Figure 14. 7j. Herbivores and all your animals are TLL is eliminated from plants. The waste products MD. The dead organisms are decomposed by phosphate solubilizing bacteria releasing phosphorus. Unlike carbon cycle, there is no respiratory release of phosphorus into atmosphere, can go differentiate between the carbon and the plus the all 1.0 meters ajor. And import, MT differences bet1 in carbon and phosphorus cycle are firstly. Atmospheric inputs of phosphorus through our main toll. Are much smaller TLL and CRUN inputs. And secondly gaseous. FCOTFMI, IT, I, AG, D3, R3 up. Kamil, Figlu 14.7 ASTMCL MOCL, TLLPH, SPH, Rus 1 and arrestal ecosystem exchanges of phosphorus between organism and environment are negli BLE. 14.8 ICOSERS, U.MSM, VLCT1S healthy ecosystems either BSEF or a wide range of economic environment. 31N, aesthetic goods and services. The products of ecosystem processes. RD is ecosystem services, for example He31I forest ecosystems purify oil and water. RNI yuga droughts and phytoids, cycle nutrients generate for your soils provide wildlife habitat maintain biodiversity. Pollinate crops provide storage site for carbon and 3 liters, provical, stayup culture 31 and spiritual WLUEs. The V31UE of such services of big diversity is difficult to determine. It seems reasonable to think that biodiversity should carry a hefty price tag. Robert Constanza and his colleagues have very recently tried to put price tags on nature's life support services. Researchers put an over 3 J price tag at USS 33 trillion a year on these fundamental ecosystem services, which are largely taken for granted because they are free. This is near twice the V31UE of the global gross national product GNP, which is USS 18 trillion. Out of the total cost of various ecosystem services, the soil ilmidium accounts ILR about 60 per ENL and billions RT for services like Recre UA1 and nutrient cycling. are less than I0% each. The cost of climate regulalia and habitat and wildlife are 31.6% each. Summary. An ecosystem 1 an al unit of nature. Mal complex as tick. Mal biotic components. Abiotic components meet Norg 3 lick matter 3 ls. Mr. Water. Mal S01 LWL. E3SB 1011 C compone. NTS me perturbers consumers decomposers. Each ecosystem has CLW. Eteri suck phase 1 cow structure resulting from interaction MNO street abiotic biotic components. Species composition and stratification. Mealia. 2 main structural features of an ecosystem set on source of nitrogen E.Terry org 3 Lism. Occupies a pleo in ecosystem. PRCD. Activity decomposition energy OVL nitrogen cycling. Mealia FR1 port. 3 NL components of an ecosystem. Pi 1 LMLY productive diazilia RCDE of capture of solm energy or BRM's production of the prod CERS. It 1s divided into TVO types, gross PRTMI productivity GPP net prayer MJ productivity TLPP. Rate of capture of Sol 3R energy or Ta true prod. C110N OI or 3 Nick matter 1SS GPP, MPP 1SU, ERB or Sorelia energy left, ITO utilization of producers. Second my prod. CTIT1S Iliarda of S1 mylation of FOCD energy Biolia consumers, into compass, ITI, LN. Complex or LLIE compounds of detritus mecone, reted to seam bond eoside water. Mel 1N1 Regan 1C nut, NTS Biolia decomposers. Decomposition immortalizatory processes namely fragmentation of detritus leaching catabolism. Energy flow 1s unidirectional, FOSPL mats capture sow energy and island food is TRAONs fell, read from the perturbers to decomposers. Or RSMS of different trophically. Ols in nature mesiolu, tied to each other L or F wood or RMHA rela launch the formulate F wood CH in the slore 36L, Mauane, NT. Of nut, NT elements Iliraul, Q, it is compone, NTS of Ilia ecosystem 1s called nutrit, NTC cling, nutrients me are tedly use Iliraul use press S. Nubtant cycling 1s of 2 types gaseous sedime, NY, atmosphere or H drosplum 1SU, E resin however U, E gaseous type of cycle seam bond, Harry C. Millis crust 1s the res, I ho for say, Ltmet, NT my type phosp, Rus. PRCD.CTS of ecosta, RN processes me as ecosystem services for example, Pertka 110N of Mr. Water by forests. The biotic CXM, mu, UDLS dine, IIEI undergoes MTH the passage of time. These me seek, NTILI ordered and constitute a colo.co Sion Su, says, Sion begins watilla UW Aston of a beamy lifeless me by Toriers which lail, R pateway F or sucosers. Mel ultimately a stable clear MX community 1s formed. NLECLGLMX community re, MNS stable as tongue as the E, Retatron, NT-REM MNS, FXFRC, ISFS. 1. FLLN the BLMLCSAJPL, MTS me called as because you, E-fuxine bond E1 oxide B in an ecosystem dominated by trees. The Pierre Arnold of Mont, RSLS type C in aquatic ecosystems Ilia learned factor F or Ilia productivity is. EcoVTEMD common detritu, Orzalar ecosystem E the major reservoir of seam bond on E3LTHTS, 2. Which one of the fall of N8H, S the lore, street popple, lon LNF woodchaw, RLAJ producers B pi 1 MMFC alls. On C second MF consumers D decomposers 3. The second drophically OELLAL3 KTS, Fitopl, Tong B Zupl, Tong C Benilius D Flushes 4. Sikoni 3 Jipro, IUOE, RS me AJE, RB1.ORS B producer CCMNI.ORS D none of you, E.WO5. WH, LLS the Parontojafatus in 11 Caliao Tirati, Lion Par LNU, ELC1 Dent Sol 3R radiation. A 100B50C1D2106, D, STJNTLSH beta GR, Zing Fwood chain identitas Fwood CH, 1 and B production, Meldec, Orn position CUPR8HT and U worded PIRM, IID food CH3, 1 N. Mel food. Ebiotter, Meldetritis 0 PRTMM. Mel second Empro, I.CT islet 7, Descriptilia components of an ecosystem. 8. D. BN ecological pure MODS. Mel describe tilly E, X plus pure nids of number. Mel biomas 9. WH, LLS per reproductivity G Bertoft SA, option of factors that fool reproductivity. 10. DFRE decomposition describe Ilia processes products of decomposition 11. GMZC, cow LL of energy flow UL. Ecosystem 12, right to port C, NT features of a sediment, Rice Eclairlum Ecosystem 13, Oline Soj Tent. Features of Corbon Cycling TN, system. Chapter 15 Biodiversity and Conservation 15.1 Biodiversity 15.2 Biodiversity Conservation. If an alien from a distant galaxy were to visit our planet E3RTH, the first thing that would and baffle him would probably be the enormous diversity of life that he called NCERT. LNFCRHLLWLS, the NCH vein 1, of living organisms with which they share this planet Neller C says to astonish and fascinate us. The CORDNRLWL would find it hardy to believe that there are 20,000 species of ants. 3,000 SFC soft beetles 28,000 species of fishes and nearly 20,000 SF size of orchids. Ecolo-STS and Eoluionary biologists have been trying to understand the sinuan icons of such diversity by asking important questions. Are there some spec, SDD sud 9 react diorsic explavaudrets historical d this diors, Joe Kodge, uncome AFX, how on d. WHGL's TLIS dig, TG important T to the JSOS XR would LT junk, Ang Farent G if the DO, Recitquo as much less how DO mu man spenny fought from the DO Recitga VFA 15.1 Biod 1Y1.R. SILS, in our biosphere immense diversity or heterogeneity exists not only at the species level but at all levels of biological organisms ranging from macromolecules within cells to buyers. Biodiversity is the term popularized by the sociobiologist Edward Wilson to describe Leotiv UILI.N Deconukta LLNO, Vnecldy, Siljet 311 Lili LSATL, IL, LQ Itaotalamswan. Thermstyle F4L, Exontathammer, Genotic DW, T. A single species might show big D resilge at the genetic level its distributional range. The genetic V3 fiation shown by the reticent 3LPLMT raw wolf OA umanum wing and C, IJ6 errant Himalayan ranges might be in terms of the potency and concentration or the actile chemic 3L reserpine at the PLMT pro, 2cis. India has more than 50.000 Genuk 3 little for instance or rice, and I,000 varieties or mango, U species DO, LT city, the diversity at the species level. From the Western Ghats hail a greater amphibian species diversity than the Eastern Ghats. Elecal 09 ELDU, LT city, at the ecosystem Lul, India instance, WLTH its NCERTs are, in forests. Mangroves coral reeds trans estuaries and 3 alpine meadows has a grant, tech ecosystem dealer city tha and a Scandinavian roundtree like Norway, it has tolerant millions at as of E1 Tulian. 1 OEM Allet is rich diversity in nature but we OOUJD lose 311 that altham less than 2 cent wise at the present rates or species losses untnu. Big DR, ILJ and its conserva yuan are now vital and vironor, until the use of 1 timation alkmsamas roar melmore people jlau. LD the WCRLD begin to real LHE critical INQ, Orden cellar beetle or citifer RSU1 Viv 3L and well, being on this planet. 151. 1 How many species are there on Earth and how many in India since there are published records or 311 the species discovered and named? Know how many species in all have 10 records so FM but it is not easy to answer the question or how many species there are on Earth, according to UCN 2004. The TD, 3L number or planted animal species deals so far as SL, true more than 1.5 million but we hello no clear, D lower how many species are yet to be classed oil or described. Estimates, ori widely and many of them are only attic, T at G tesses, J, L, Armanita, non-with groups. Species inventories, Winks Resi, LN11 eat in tetraf, mate than in tropical countries, consif, len that and relmany.y targe. Comma on at the species idlen to be dice, M, I are in the tropics BLCL, FSLS renalua statistical or in parts and at the La 14. Al edel cleo species retinessarani, hostilialis to klage reference xan, trappa, this ratio to other groups or 3. Limels and PLMTS2, or not with a gross estimate or the toe number or species on Earth S or te extreme estimates range around 20 or 50 million. But Aurora Conserva 1 and 10 IFAC 3 LLY Sound Estern, Temode by Robert May places the S about species clear city at about million. 81,010 GY let us locked out at so interesting aspects at earth, S big diversity based on Lee currently available species inventories. More TLWL 70% of the species record, RRE lirals while PBNTS including GFTMG bryophytes. GJMN osperms NDN, osperms untens and no more MN 22% of the TOD, L, M, LNG animals insects daughterly NDST species rich T-lomic group. M3 king up more hone 70% of the O31, that runs, out of every I0, NIM3LS on this planet. 7 are insects. 1 Go and how CB we explain this enormous diversification of insects. The number of fungi species in Lee world is M, Re 3 and Lee combined. Total of Lee species of fishes, amphibians, reptiles, NDLWLM3LSLN Re 15.1 Biodiversity is depicted SHCJB, Ling species num terror of major L3XA. I, 1 IFIS, Euro 15.1 representing global bio, IT, RS, ITI, proportionate number of species of major Y of PL3 and TS. Tebrets.mel, Tebrets it should be noted TH, these estimates do not give only figures for Proc 3 loads. Biologists, we're not sure about how many prokaryotic species there might be. The problemi SMT unvention 3L3 xenomic refts are not suable Lee JR identif. Y. Microbe species MMY species, RRE simply not culture able WL raral, laboratory conditions. If we accept biochemical or molecular cardinalbrictline, ting species Lee JR Fiskruppen and their diversity alone might run into millions. 81,001 VU80A, OC, Uvatio, although India rues only 2.4% of the world cell third area it's shortly about species diversity as a min per CE8. L%, that is what rogues are called try one of the 12 regadiversity countries of the world. Nearly 45,000 species of PL3 and TS. And twice as NW1 Yof Me 1 Nals High been record from India, how my living species are actually there woe, ting to be discovered and named we accept Mage global estimates. Only 22% of the total species RUV have been relooked so far. Applying this proportion to India's diversity figures estimate that there are probably more than 1,000,000 plant species and more than 3,000,000 anima 1 species yet to be discovered and described. Would we or be able to complete treatment the biological vithof Kirko will treat or the imrans R3 Eindman our taxonomus in the time required to complete the job. The situation appears hopeless when lolly realis TLL, at LRGFR action of these species faces Lee TLL read of Bikofl. In tinktivan B1 Bray lolly disco or Lee managers biologic caliber areas burning even before lolly Kylo the titles of all the books stocked there. 1.5.1.2 Patterns of Biod, Corsidi latitudinal gradients, the dear city of plants and animals is not WL brim throughout TLLE world. But shows WL even distribution. From any group of animals or plants there are interest, and patterns and diversity. The RMSD well, known B, and the L3 tidid algae are adient and diversity. In general species diversity techer as we move. I from TLLE equator to, Ardsley P, with very few exceptions tropics latitudinal range of 23.5 degrees N to 23. S warmer species than temperature polar areas. Lombie allocated near the equator US near Y 1.400 species of birds while New York at 41 degrees N has 105 species. N. Greenland at 71 degrees N only 56 species. Industrial IA with much of its L and R in Lee tropical latitudes. RUS more than 1.200 species of birds. A forest in a tropical region like Ecuador has up to I0 years as many species of vascular plants as a forest of equal area in a temperate region like the mid-street of the USA. The largely tropical M32 Onion Rain forest in South America H is the greatest diversity on Earth. It is hard to re than 40.000 species of plants. 3.000 of fishes, 1.300 of birds, 427 of mammals, 427 of amphibians, 378 of reptiles, and of more than 1.25,000 invertebrates. As scientists estimate at in Lee's RN forests Lear might be at least 2 million insect species aiding to be discovered. And an arid what s so sped all about, top, CS that MGHT account for their 9 reator L009. Cat Deoresid ecologists and evolutionary biologists Ruve Prop 1 Sect 1 Litris HJP, Theses, RRE ILP, tones speciation is generally a function of theory. Unlike temperate ons subjected to frequent glaciations in the past tropical latitudes 113 Evern 3 and relatively undisturbed FBR millions of years and thus H3D along 81,010 GYE Dushio WY Thierry Rohr Spe East, Ursificolik, B tropical environments unlike temp H3 R81s. Are less seasonal relatively more constant and predictable, as such constant in Lero LRTB and TS promote Rouge specialization and led to a greater species diversity and there is more solar energy. Available in Lee tropics, which contributes to higher PLTXIUCI tie. This in turn might contribute indirectly to gender diversity. All species area relationships, during his pioneering and extensive explorations in the wilderness of South American jungles. The great German naturalist and geographer Alexander von Humboldt observed TLL at Whittle. A region sp3 size less increased. Whittle increasing, plot area but only up to a limit, in Fig. 1u, E15.2 showing species me relationship. No dulet on Togesilia relationship becomes linum tactli relation to in species clueless MD. Area for a wide variety of angiosperm plants, birds, bats, freshwater fishes turns out to be a rectangular hyperbola figure 15.2. On log art Looney C scale. TLLE relationship is a straight line described by the equation logs logo where 8 sp size richness A area Z slope of the line regression comb sheen TCY intercept. Ecologists have discovered that the OLOFZ lies in the range of 0.1 to 0.2. Regus of the Telamic group are whether it is the plants in Britain, birds in Galeromia or mollusks in New York State. The slopes of the regression line are amazingly similar, but if you analyze the species, or relationships on very large areas like the entire continents, you will find that the slope of the line much steeper Z values in the range of 0.6 to I2, furry. Ample for frugivorous fruit-eating birds and RNA MEWs in Lee Tropica 1 forests of different continents. Lee Slop 3 ISBMD to T 1.15. What do steeper slopes mean in TLVS context 15.1? 3. The importance of species diva, city to th. Ecosystem. Does the number of species in a why really matter to the functioning of the ecosystem? This is a question for which ecologists have not been able to give a definitive answer. For Midicax ecologists believe TLL, at Cornwallides with M, species generally tend to more stable than those with less species. What exactly is stability for a biological? 81001 VU A, OC, Uvatio comwility. A stable comwility should not show too much WN, Asian in Pro, TUC D1. Tie from year to year, it must be equal resist MT resilient to a CAS, onrudistertlum 03S natural or RNM renacla 3. MD. It must also be resist MT to NCERT by alien species. We don't know how these attributes are linked to species richness in corn WLED, but David Tillman, S long-term ecosystem Penman S using outdoor plots provide SOAR 10.E. Slow, Ers. Tillman found TLL, at plots with LEM, species SHCN ed less ER to ER variation in total biomas. He also showed TLL, at in his Palerents increased diversity contributed to higher productivity. Although may not understand completely how SF size richness contributes totally to an ecosystem. They know enough to realize rich biodiversity is not only essential for area system health but imperative for the very sum of the hum race on this planet. At a theory when we are losing sp3 size in 3 liter at world pace one might ask does it really matter to us if a few species tech or extinct would? Western Ghats ecosystems be less FWL XII if one of its tree frog sp3 size is lost fee, trailer how is our quality of life affected too? So instead of 20.000 have only 15.000 SFC soft on Earth there are no direct MWers to such IE questions but we see develop a proper F. RSF tied through an analogy the rivet popper HJ pohesis one used by Stanford ecologist Paul Ehrlich. In an airplane ecosystem all parts are joined together using thousands of rivets SF size. ULRE passenger traveling in it starts popping a rivet to take or causing a species to be our X-line. It may not affect flight safety per functioning of TLLE ecosystem initially but as M, re.N. M, re-rivets are rare no ed, TLLE plane becomes dangerously weak over a period of time. Furthermore, which rivet is rare no ed may also be critical. Loss of rivets on TLLE wings key sp3 size TLL, that drive major ecosystem functions is obviously a M, serious threat to flight safety TLL, and loss of a few rivets on TLLE seats or ank was inside TLLE plane. 15L, for loss of biodiversity, city. While it is doubtful if any new species are being added through SFCICN into early Earth's treasury of SF size, there is no doubt I'd cut their continuing losses. The biological health of our planet has been declining rapidly and the accusing finger is clearly pointing to human acti. 1 IES. The colonicity on of tropical Pacific islands by humans is said to have led to the extinction of more than 2.000 species of native birds. The IUCN Redus 2004 documents the extinction of 784 species including 338 laribrats, 359 invertebrates and 87 plants in the last 500 years. Rare examples of recent extinctions include the dodo Mauritius quagga Africa thylus in Australia, stellar sea cow Russia and 3 subspecies Bali, Jayan, Caspian of T. Jair. The last 20 years alone hail witnessed the disappearance of 27 species. Careful analysis of rewards. 81,010 GY shows flat extinctions across T3 X are not random. Some groups like amphibians appear to be more vulnerable to extinction. Pound symbol linked to the GNM scene of extinctions is the fact that more than 15.500 species. JD are facing the threat of extinction. Presently 12% of all BD species, 23% of all mammal species, 32% of all amphibian species on 31% of all gymnosperm species in the world face extinction. From a study of the history of life on Earth through fossil records learned that large-scale loss of species like GLLE1 we are currently witnessing have also happened earlier. Ellen Terrorhams appeared on the scene, during the long period, 3 billion years since the non-diversification of life on Earth are 5 epochs of mass extinction of species. How is the SIXTH extinction presently in progress different from the previous epochs? That the differences in rates, the current species extinction rates are estimated to be 100 to 1,000 tiers faster TLWL and pre-human tiers and R.ACTR1 ties are responsible for faster rates. Ecologists warn that if the present trends continue, nearly half of all the species on Earth might be wiped out within the next 100 years. In general, loss of biodiversity in a region may lead to a decline in plant production, beloved resistance to environmental perturbations such as drought and sea increased vulnerability in certain ecosystem processes such as plant productivity. Water use and pest and disease cycles, causes of biodiversity losses, the accelerated rates of species extinctions TLLEV, world is facing now are largely due to human activities. Their are four major causes are the evil quartet, is least used to describe the habitat loss OND.FR intento This is the most important cause driving animals and plants to extinction. The most dramatic examples of habitat loss core from tropical rainforests, once cmeffing we 3 and 14% of TLLE Earth's land. Surface these rainforests now cover no TLWL 6 page 3%, they are being destroyed fast. By the time you finish reading this chapter, 000 meters, hectares of rainforest would have been lost. The Amazon rainforest it is so huge that it is called the 1/1 of the planet. One probably millions of species is being cut on cleared FBR cultivating so go beyond FBR conversion togger as the third S for raising beef cattle. Besides total loss, the degradation of many habitats by pollution also threatens the survival of many species. When large habitats are broken up into small environments due to Vienna's human lives more animals on BDS requiring large territories on certain animals with migratory habits are badly affected, leading to population declines. Lowry, Q. Lloyd O'Shien, humans have really stepped into nature for food. MD shelter but when, neat, turns to, Greek that leads to 81,001 VUADA, OC, U ratio over E.O, PLoid addion of natural resources, many species extinctions in the last 500 years stellar 0. Passenger pylon due to overexploitation by herons, presently marine fish populations around the world are harvested, endangering the continued E.O, instance of sourcio ILL really IRNPOITNT species 3 alien species Iwasians, when alien species A reintroduced. Will intend in 3 Lyrdel Baratella for whatever purpose some of them come in VCE, and use decline or extinction of new species. The Nile perch introduced into local Latona in East Africa Lidelentualidoda, tinction of an ecologically weak assemblage of we 3 and 200 species of cichlid fish in the Lalt. You must be familiar with the environmental damage caused and threat posed to our native species by INVSCETED species like Caragra SS Porthonaire MJ. Lantano and water hyacinthi, Cediana, the recent illegal introduction of the African catfish CREOS 9 REF US for aquaculture purposes is posing a threat to the indigenous catfishes in our rivers. Co-extinctions, when a species becomes extinct TLLEPL3 and TAND. Animal species associated with it in obligatory WAY also tech or extinct, when a host fish species becomes extinct its unique assemblage of parasites also meets the sore fate. Another example is TLLE cause of evolved PLMT prognator mutualism where extinction of one invulnerable leads to the extinction of the other. 15.2 BTO Laverson, Go NSM via Noro, 152, 1 Why should we consent biodiversity to lear our NW1 sons? Some obvious and others not so obvious but all equally important. They can be grouped into 3 categories, narrowly utilit nien, broad YUDILLT nien, and EDHE Kaid and narrowly utlitarlan AR rents FBR conserving biodiversity are obvious. Hun 1s rent accomplished direct economy C benefits from nature T, odd cereals pulses fruits 5F. OCD, fiber construction MA Terry 3.1 industrial products tannins, lubricants dyes resins perfumes and products of medicinal importance. More than 25% of the drugs currently STD in the market oral via RCL, related from plants, and 25,000 species of PL3 and TS contribute to TLLE traditional medicines used by native peoples around the world. Nobody knows how many more medicinally useful plants there are in tropical rainforests waiting to be explored. With increasing resources put into bioprospecting, exploring molecular genetic and species level diversity have reproductive economic importance. Nations endowed with NCERT Biodiversity can expect to reap enormous benefits. The broadly utilitarian argument says that biodiversity plays a major role in MI ecosystem services TL13T nature provides. The fast 81,000 wide dwindling Amazon forest is estimated to produce TLL route photosynthesis 20% of the TOJ in the EOR1HS atmosphere. We put an economic low on this as by nature you can get SOR ICL by finding out how much your neighbor hospital spends on a cylinder of ICN. Pollination, without which plants cannot give us fruits or seeds, is mother SEMS ecosystems provide TLL rough pollinators layer bees, bumblebees, BDS and bats. WMTU, JTB. The costs of accomplishment 9 partner without help from nature OT pollinators. There are other intangible benefits that CL derive from nature. The aesthetic pleasures of walking throughout thick DS, watching spring OORS in full bloom, or waking up to a bulbul song in the morning. Can put a PRL set tag on such things. The ethical argument FBR conserving biodiversity relates to what ECN to millions of PLMT. Animal and microbe species whom ESH are this planet, philosophically or spiritually. We need to realize that every species has an intrinsic value, even if it may not be of current or any economic value to us. We have a moral duty to see our for TLL or VL being and pass on our biological legacy and dare to future generations. 1.5.2. How do we consent biodiversity when we consent and protect TLL whole ecosystem? Its big diversity at all else is protected. ETLL entire breast to save TLL. This approach is called NSU on-site conservation. HCN when TLL are situations where animal or PL3 and T is endangered or TLL attend. And needs urgent measures to save it from extinction. XII off-site conservation is TLL-ECL, cerebral approach. In situ conservation phase nith TLL economic between revet and conservati, LNMMY nail s find. It realistic and canonical not feasible to serve all their biological wealth invariably. The number of species whaling to be saved from extinction FME.O. CDS TLL conservation resources of oily, bled. On a global basis this problem has been addressed by eminent conservationists. They identified for maximum protection certain biodiversity hotspots, regions with very high levels of species richness and high degree of endemism, that is, species zone find to that region and not round anywhere else. Initially 25 biodiversity hotspots were identified, but subsequently 9 more have been added to TLLE list, bringing the total number of biodiversity hotspots in the world to 34. These hotspots are also regions of accelerated habitat loss. 3 of these hotspots, Western Ghats in Sri Lanka, Indo-Burma and Himalaya, cover our country's exceptionally high biodiversity regions 1 through 0. All the biodiversity HOPOS page 11 together, or 11.2% of the Earth's land area. The number of species they collectively harbor is arbitrarily high, and strict protection of these hotspots will reduce the ongoing mass extinctions by almost 30%. Leod of Fuvankan F. Elevation in India. Ecological ironic and Biodiversity NCH Reg. ONS are legally protected as biosphere reserves. National parks and sanctuaries. India now has 14 biosphere reserves, 110 non-peril and 448 wildlife sanctuaries. India has also a history reg, a stud and cultural traditions that has East Proak, TLC and nature. In many cultures LRA, UC6 dressed, we set aside and all the trees and cliff within were located in lion. Such sacred exhibits are found in KG and Jia Hills in Meghalaya Arava, UHWC Rajasthan, Mustamghat regions Karnataka and Maharashtra and Hisar Gaya. Sibianda and Bastura C. Madhya ISH. In Meghalaya those sacred are the IJ are a large number of rare and threatened plants. Situ conservation. In this approach threatened animals and plants are taken out from their natural habitat and taste in speedy setting where they can be protected and given special care. Zyogical Prk SBJ Oracle Groans and Lyle Ward Prk S. Sane Ulispuk, Os. There are many animals that have become extinct in the wild but continue to be moined in Tog, Cal parks. In recent years, XII conservation has advanced beyond keeping threatened species in enclosures. NJWG01 Reds at threatened species can be preserved in viable and low condition for long proto, ls using CT opnorva UCN techniques. Yeast can be fertilized if foitro and plants can be propagated us, Angus susi ultra refo, ls, s, edsit c farent genetic strains of CL11 RCU important plants can tai, eptel or jung pino, ts in seed banks, Biodiversity k nows no poiucal boundaries and its conservation is therefore eclectal, l responsibility at all nations. The Hisland Convention on Biological Diversity RNEERth Summit 1 held in Rio, Janeiro in 1992. Called upon all nations to tackle appropriate measures for conservation not biodiversity and sustainable utilization at its benefits. In a follow-up the World Summit on Sustainable Development held in 2002 in Johannesburg, South Africa. 100 countries pledged their commitment to achieve by 2010 reduction in the current rate at biodiversity while at global, regional and local levels. F summary SN, ELFOD and Earth RLY 3.8 liters, Alan Yixis, go there had been enormous DXOATs and of wit forms on Earth. Biodiversity refers to the sum total of div, adihati exists at all levels of physiological or n, so n. Of partlekub, LM, istedl, at chinux bagozani ecosystem loot 1s.con nr4u1ets.r1.prodec, T1 and deers 1 tied off T low than 1.5 million horico dead 1 and the old. But their MFLT still be nearly 6 mil barn spa, on Earth waiting to be at on the ananakl, of hanameklixt D, 10% or at mac of OICH 10 per oral. R, though up FUQPB, Mare. 81,010 GYOL-D, Retebert Sp, Tescumbered. WL about 45.000 Sp, Tes of PL3N1S.mel TVIE is MMY Sp, Tes of 1S1 of the 12 mega G1, S, ID count for Tes of Ilya world. Species D, S, Idion M1S Rotella Fornly D1S GL1 Buted but SH, RWS Lentor Estlin patterns, IDLS firm.2IHJ8 Hestel N Lrop 10 SID, EOSIS to MDS Ilya poles. Import, 3 NTX 3 Noluans F4U, E species richness of U, E tropices me, no piece had more evolution myrni, I provide a relati alicons C, NTE, Environ me, NTN. U, E reclt2.e more sawi, NERG which contributes to greater productivity, species comis 1s FW Xian of the me of RN, Ilias Beck 1s me rel 11 on she page 1s cheer, IYRHR0011C function at 1s BLIOED Ilio Corn W. 2 ties Tilly high CB, was, iditech, ND to be less I'm table, more productive more as 1 SBNT to be 1 all 1 NV as 1 ons. EMTHSFOS, I'll history V1 and C1D, no of Massic still 11 ons 1 Anlia past but U, E present are ales of E, on 1 on. James Jitty attributed to him, MACL1 VTT1SA, Re 100 to 1000 times liar, Ne3, 1L 700 species have become extinct all. Rees, NT times and more 3N 15.500 sp, Tes of which, 65, me from Hilltalk currently follow Iliowood of extinction. The causes of high, still Q1 rates at present include habitat PA, Rachika MLY forests loss CMD fragmentation. Or exploitant tilog investance coexunctions, AA, realus NCHB1 occult, US1 Taiwan Lytle for Ilioi servio of IMLLK1N, L. The reasons F were concert WLG biop, was, idi mean tala ulic, RT3 and broadly ilitort, and non-ethical. BC, Lebedria at Bur FLTS, WAD BTE Fire Eva PH Maceuticals etc. Ilirmiam, my indirect benefts we resei.elhre, Ausius, MSEDESA Jospol 1 Nation. Exclamation point control comate moderalia nukcon 1 roll R, E also ha, E immoral response 1B1 liddy to take 9 CDCME of Earth's B1 odd 1. Tercity on all. That order to RRX gene, IAU on, B10D1 vet, Versity cons, Alien maybe NSK2 well as XII. HLNSK2 conser, AU on Ilia Reds B1 SME protected in ILU natural habitat so Ilia the entire ecosystem is protected. Recently 34 B1 odd 1 vet, Versity hotspots all Ilia world hot AB, and proposa, L for INLL, NS10E consonation efforts. Of these, UV Western Ghats Sri Lanka, Hernalaya.mel HDL Burmal Kawa HDS Re CHB1 odd 1 was, IDRNS. Our country's NSK2 cons, Asian efforts me reflected all its 14 TUSPL, Air Resin ES9, National Parks. 45, W1LDL1 Face A, include, 3 IISM, My sacred grow.s, XII cons, AU on Melia Zaklud product E of I tier, L species M rub 1 cal PMKS. Melbot 1 cal gardens. NL tro fertilization tissue culture propagation cryopres, R.ation of gametes. 1, Na, RNE the 3 UO port C, NT compone, NTS of B10D1, Ursity 2, how do ecologists system CDE Ilia total number of species present Ilia world. 81,001 VUIDA, OC, Uvatio 3, GT 3 Hippo, S is F4E, XPLO, Uang V Hydropy CS show greatest leols of spec 1S Relu, S. 4, WH, 1 LS though of those slope of Reger on 1 and a relationship 5, what day, Realiama, Jorsi, uses of SP1S losses 1 and octapical RF6. How is B1 odd 1 was, idiom LC, NT for ecosystem F7, what me sacred GDOS what 1S Ilia roll 1 NC, on serve, DIR fate. Among Ilia ecosystem set, I vice, me control of flood soil erosion, how LSTHLS by the B10 corn ponens aldo ecosystem 9. The species D1. Tears, IDI of PL3 and TST22 pay, R cent 1S, much less Olumadev, LS 12 cent. What could be the oxplosions to how, LS Achi Edtail, RD1. Tears, Ification 10, woe think of a situation where we deliberately WT to MEO, TLRCT. How would, you Jew, try it R. Chapter 16 Environmental Issues 16.1 ACR Poshion Andlets Control 16.2 Vaterpuxion Andlet Eskan ETD 16.3 SD Idless 16.4 JL Srkimi CDSNDT Hayar Effect 16.5 R Diok, MTT, Evostes 16.6 GRI, W, Use each Fakton GKHA, IVOD Me 9 16.7 OZ Nedaplatk, N. T and the Street Recortosplutter 16.8 Degram DV, J and Biomprik Perezal QCEU Wasatan Unwa NTN and 16.9 Deforestic. Human population CSRUS grown enormously over the last 100 years. This reans increase in remand roar FCX, D. Water horelect, city roods atarabiles and neurosulier QNO, liddies, to lease rarem xereri, retain trerinctus pressure on our natural resources. And died are also contributing to pollution of air, water and soil. The need of the hour is to check the regaptation and repletion of our precious natural resources. N. Pollution will at halting the process of development. Pollution is my WL desirable CH3NGE in physica L. Chemical or biological characteristics of air. Land, water or soil NTS true teben about such an undesirable change. RC3 lead. As pollutants HL order to contra environmental pollution TLLE Government of India HSPS. TLLE Environment Protection Act 1986 to protect and improve the quality of our environment air, water and soil LF.LMPU, TTN A, ON, ESCO, CHLHAL. We are dependent on air for our respiratory needs. Air pollutants cause injury to all living organisms. They reduce growth and yield of crops and cause premature death of plants. Air pollutants also CL, lateriously affect the respiratory system of humans and of animals, harmful. Environmental issues effects depend on the concentration of pollutants, duration of exposure and the organism. Smokestacks of thermal power plants, smelters and other industries release particulate and gaseous air pollutants together with harmless gases, such as nitrogen, oxygen, etc. These pollutants must be separated, filtered out before releasing the harmless gases into the atmosphere. Clean air 1 discharge corona water line spray, dirty ale, dust particles O. F clean a 1 R matter collection plate grounded tetrastatic precipitator I scrubber I figure 16.1 electrostatic precipitator. There are several ways of removing particulatometer, the most widely used of which is the electrostatic precipitator figure 16.1, which can remove over 99% particulate matter present in the exhaust from a thermal power plant. It has electrode wires that are maintained at several thousand volts, which produce a corona that releases electrons. These electrons attach to dust particles, giving them a net negative charge. The collecting plates are grounded and attract the charged dust particles. The velocity of air between the plates must be low enough to allow the dust to fall. A scrubber figure 16.1 can remove gases like sulfur dioxide. In a scrubber, the exhaust is passed through a spray of water or lime. Recently we have realized the dangers of particulate matter that are very, very small and are not removed by these precipitators. According to Central Pollution Control Board CPCB, particulate size 2.5 micrometers or less in diameter PM 2.5 are responsible for causing the greatest harm to human health. These fine particulates can be inhaled deep into the lungs and can cause breathing and respiratory symptoms, irritation, inflammations and damage to the lungs and premature deaths. IEI of automobiles are a major cause for atmospheric pollution, at least in TLLE retro cities. As clean number of vehicles increase on Lee streets, TLL's problem is now shifting to the other cities too. Proper maintenance of automobiles along the use of lead-free petrol or diesel can reduce TLLE pollutants they emit. GA, IDC Commerce having E, Pens IV RREWs Norway Platinum Palladium .3 NDRHCDIUMS Lee catalysts are fitted into automobiles for reducing emission of poisonous gases, as Lee DLS passes through the catalytic converter. Unbumped HJ, carbons are converted into carton dioxyren water and carton unosite and nit and CO, are changed to carbon dioxyren gas. Respectively, motor vehicles equipped with the catalytic converter should use unleaded petrol because lead in Lee petrol inactivates TLLE catalyst. 161. Controlling vehicular air pollution, a case study of Delhi with its very large population of vehicular traffic. Delhi leads the country in its levels of air pollution. It has Andhra states of Gujarat and West Bengal put together. In the 1990s, Delhi ranked 4th among the 41 RMST polluted cities of the world. Air pollution problems in Delhi IB care so Senastrata public interest litigation FU was filed in the Supreme Court of India, after being censured very strongly by TLLE Supreme 03 Under its directives, Lee government was asked to allay, within a specified time period, appropriate measures, including slowing the entire oit of public transport, that is, buses from diesel to compressed natural gas CNG. All the buses of Delhi reconverted to run on CNG by the end of 2002. You may ask the question as to why CNG is better than diesel. The answer is that CNG bums are MSTFL Shinui unlike petrol or diesel in the automobiles, and very little of it is left unburned. Moreover, CNG is cheaper. TLL and petrol or diesel cannot be siphoned off by thieves and adulterated like petrol or diesel. The main problem with switching to CNG is the difficulty of laying CBWN pipelines to Delhi, or CNG through distribution points pumps, and ensuring uninterrupted supply. Simultaneously, parallel steps taken in Delhi for reducing vehicular pollution include phasing out of old vehicles, use of WL-leaded petrol, use of low-sulfur petrol and diesel, use of catalytic converters in vehicles, application of stringent pollution level norms FBR vehicles, etc. The Government of India through a new auto fuel policy has WD out a roadmap to cut DCNJ and vehicular pollution in Indian cities. More stringent norms for fuels means steadily reducing the sulfur and aromatics content in petrol and diesel fuels. Euro D norms for E.O. ample stipulates that sulfur be controlled at 350 parts per million ppm in diesel or 150 ppm in petrol. 1 Aromatic hydrocarbons are to be contained at 42% of the concerned fuel. The goal, according to the roadmap, is to reduce sulfur to 50 ppm in petrol and diesel and bring CBWN up. FJTLT only UNLI1, UTSLIL to 35%. Cones ring to the fuel reticle will also need to be upgraded. The Bharat DEQI 31 and to UMD nons. WCH is currently all place in Delhi, Mumbai, Colt HN2I, Bangalore, Hyderabad, Ahmedabad, Pune, Surat, Kanpur, and A will be applicable to all automobiles throughout the county. JL from April 1, 2006, all automobiles and fuel petrol and CLE, L were to ha, read the Euro emission specifications in these 11 cities from APR 1, 2005 and have to read the Euro V norms by APR 1, 210. The rest of the county JL will high Euro, emission norm compliant all bios and fuels by 2010. Thanks to the FLBRS made the 3 air quality of Selho has significantly improved, according to an SLEARN 3 tell, a substantial fall in company. And Solio has boomed abundant Delhi between 2007 and 2006. In India, the Air Prevention and Control of Pollution Act car into BE in 1181. But was rendered in 1937 to include no 90 as an air pollutant, noises undesired highly of sound. We have FJ, A used to associating loud sounds with pleasure and entertainment, and see really sing that nice causes PS. Comical and Phis, Caldi in humans, the BLR the city the bigger the funky tutor the noise, a brief CPP short to extremely high soundly 1L. 150 decibels or more generated by okov of a jet plane or rocket may damage or drums, thus permanently impairing hearing ability. Even chronic EP, short to a lower noisely 1L of cities may permanently damaging 3 abilities autumn MS. N was 3 LSO causes sleeplessness, increased heart beating, arid breathing pattern, thus considerable stress in humans. Considering the many dangerous effects of noise pollution, can you identify the unnecessary sources of noise pollution around, which can be reduced immediately without any financial loss to NJT. Reduction of noise in our industries can be affected by use of sound absorbent material or burning noise. Slothal general philological late CHWN in relation to N was like relimitation of HCR entry, Z, ones around hospitals and schools. Permissible, uncle ALS of crackers and of loudspeakers timings after which loudspeakers cannot be played. ELC need to be anthrocyclic to product orzo, as for nasepclitic 16.2 water protlunan and IXSC all human beings have been abu sittled the water bosses around the world by using chemical disposal of all kinds of waste. We tend to believe that water can be away everything not taking seasoning out the tact that the water be, diasaurarli, line as well as that of all other leaving CRGWSMS. Chem 90. Saint WLATAU we tend to tie and well snaw, jate around worse and sarer, due to such activities of humankind upon Kaslao, S. Stream RT1 airs oceans and oceans are becorn TG polluted in several parts of the AOI, realsl the importance of maintaining the cleanliness of the water. WIIC, IL, IIEI Ajibtais TLL Ego Emrantafel DISP Astly Water Prevention and Control of Pollution Act. 197 for to Safegort our water resources 16.2.1 domestic sewage and industrial effluents low, EVA KV, lit W8 in our homes in Lee cities.n. J1J.S, we wash a T10.1, I, Jwaped 4 will do, T.ADDL.2, CWI, M11E. C, Rtcar1 bow, TS9 liters. E, Dmetpltech, JL, RF, Dex FDL1 TT1, TMF4 Tom. Ilarathingam drains have huolari oil reared where Lee sewage twat cores out our houses gel what happens in lolliges is the sewage treated. Before being sported to Lee nearest RTR.MDMIXed with Lide mere 0.1% impurities make domestic soda fit FBR human use figure 16. 2. Uhiiriad. About sewage treatments plants in chapter 10, solids are fig, E16.20, Wampas, Iwan of waste whale, are relatively easy to remove. What is difficult to remove? RC0 NCERT NTR, up. 4, I, 0.1, N0 UDLA, RO, N. To 4 TLE, B1. Hill iron 11 I. Sigillus dissolved salts such as nitrates, phosphates, and other nutrients, and toxic metal ions and organic compounds. Domestic sewage primarily contains biodegradable organic matter, which readily decomposes thanks to bacteria and other microorganisms, which can multiply using these organic substances as substrates and hence utilize some of the components of sewage. It is possible to estimate the amount of organic matter in sewage water by measuring biochemical oxygen demand (BOD). Can you explain how in the chapter on microorganisms you have read about the relation between BOD, microorganisms and the amount of biodegradable matter? Figure 16.3 shows some of the changes that one may notice following discharge of sewage into a river. Microorganisms involved in biodegradation of organic matter in the receiving water body consume a lot of oxygen. And as a result there is a sharp decline in dissolved oxygen downstream from the point of sewage discharge. This causes mortality of fish and other aquatic creatures. Presence of large amounts of nutrients in waters also causes excessive growth of planktonic free-floating algae, called an algal bloom figure 16.4 which imparts a distinct color to the water bodies. Algal blooms cause deterioration of the water quality and fish mortality. Some bloom-forming algae are extremely toxic to human beings and animals. You may have seen the beautiful mauve-colored flowers found on very appealingly shaped floating plants in water bodies. These plants which were introduced into India for their lovely flowers have caused havoc by their excessive growth by causing blocks in our waterways. They grow faster than our ability to remove them. These are plants of water hyacinth hornticraspes. The world's most problematic aquatic weed, also figure 16.4 pictorial view of an algal bloom. TT figure 16.5 biomagnification of DDT in an aquatic food chain biology called Terror of Bengal. They grow abundantly in eutrophic water bodies, and led to an imbalance in the ecosystem dynamics of the water body. Sewage from our homes as well from hospitals are likely to contain many undesirable pathogenic microorganisms. And its disposal into our water without proper treatment may cause outbreak of serious diseases, such as dysentery, typhoid, jaundice, cholera, etc. Unlike domestic sewage, wastewater from industries like petroleum, paper manufacturing, metal extraction and processing, chemical manufacturing, etc., often contain toxic substances, notably, heavy metals defined as elements with density 5 grams cm3 such as mercury, cadmium, copper, lead, etc. and a variety of organic compounds. A few toxic substances, often present in industrial wastewaters, can undergo biological magnification biomagnification in the aquatic food chain. Biomagnification refers to increase in concentration of the toxicant at successive trophic levels. This happens because a toxic substance accumulated by an organism cannot be metabolized or excreted, and is thus passed on to the next higher trophic level. This phenomenon is well known from mercury and DDT. Figure 16.5 shows biomagnification of DDT in an aquatic food chain. In this manner, the concentration of DDT is increased at successive trophic levels. Say if it starts at 0.003 ppb parts per billion in water, it can ultimately reach 25 ppm parts per million in fish-eating birds, through biomagnification. High concentrations of DDT disturb calcium metabolism in birds, which causes thinning of eggshell and their premature breaking, eventually causing decline in bird populations. Eutrophication is the natural aging of a lake by biological enrichment of its water. In a young lake the water is cold and clear, supporting little life. With time, streams draining into the lake introduce nutrients such as nitrogen and phosphorus, which encourage the growth of aquatic organisms. As the lake's fertility increases, plant and animal life burgeons, and organic remains begin to be deposited on the lake bottom. Over the centuries, as silt and organic debris pile up, the lake grows shallower and warmer, with warm water organisms supplanting those that thrive in a cold environment. Marsh plants take root in the shallows and begin to FTLL in the original lake basin. Eventually, the lake gives way to large masses of floating plants fully converting into land. Depending on climate, size of the lake and other factors, the natural L3K may spawn thousands of years. However PLLU from man's activities like effluents from Lee Industries MD homes can rapidly accelerate the process. This phenomenon has been called or accelerated eutrophication. During the past century as in many ports of the earth been eutrophied by sewage and agricultural and industry 1031 wastes. The prune contamin NMTS are nitrates and phosphates which act as plant nutrients. They are stimulate the growth of algae causing unsightly scum and unpleasant odors and robbing the water of dissolved A, Ig and W to other aquatic life. The same time, other pollutants flowing into a lull, may poison whole populations of fish whose decomposing remains further replete the water's dissolved in content. In such fashion a lull can literally choke to wreath, heated thermal wastewaters flowing out of electrically generating units. For example, thermal power plants constitute another important category of L3 and TSI. Heavy W wastewater eliminates or reduces the number of organisms sensibly to high temperature, and may enhance the growth of plants and fish in extremely cold areas but only after using dam. Italy indigenous Ora.info WL, A16, 2 a case study of LNT egg, aided W stair water treatment. The case stair water including sewer can be treated in an integrated manner by utilizing mix of .arly, 6CLMD. Natura, processes. An example of such an initiative is Lee town of Arcata, situated atong Lee northern coast of California. Collaborating with biologists from the Humboldt-State University, Lee townspeople created an integrated water treatment process within a natural system. The clearing occurs in two stages of the conventional sedimentation, filtering on chlorine treatments or GN. After this stage, lots of dangerous pollutants like dissolved heavy metals still remain. To combat all this, innovative approach was taken and the biologists developed a synthesis of 6 connected marshes over 60 hectares of marshland. Appropriate plants algae GMD bacteria L air seeded into TLL's area which neutralize, absorb and assimilate the pollutants. Hence as the water oozes through the marshes it gets purified naturally. The marshes also constitute a sanctuary with a high level of biodiversity in the fauna of fishes, animals and birds that now reside there. A citizens group called Friends of the Arcata Marsh Foam are responsible for the upkeep and safeguarding of this wonderful project. All TLL is time lolly have as one TLL, at run DV3LOFWS requires water i.e., leak creation of sewage. But what if water is not necessary to disp? CO1 human waste, like excreta con UINLO now the amount of water that one can save if one didn't have to wash the toilet well. TLL is this already a reality. Ecological sortation is a sustainable system for handling human of excreta using dry composting toilets. This is a practical hygienic, efficient and cost-effective solution to waste disposal. The key point to note here is that with this composting method, excreta be recycled into resource as natural fertilizer, which reduces tile need for chemical fertilizers. Their .rv, Orking Ecosan, toilets in NWLF.R. as of Kerala and Sri L3 and KA. 163 Saj MW, 1.1, 5 solid wastes refer to everything that PS out in trash, local pal solid, asts.r wastes from horse offices, stores, schools, hospitals, etc. that are collected and disposed by the municipality. The renewable solid wastes generally comprise paper B, D wastes, plastics, glass, metals, rubber, liatler, tile, etc. Burnal reduces Lealler of Lee WS, although it is generally not bumped to completion, and open dumps often serve as the breeding ground for rats and flies. Sanitary Land 11 LLSE acted as the subsit FBR open burning dumps, in a sanitary 13 fill. WS.R dumped in a depersion or trench. Intercomp action MD covered with dirt YF Golio Town or DF9, Tugoma where the nearest NCERT site. Landfills are also not really much of a solution since the done of garbage generation, especially in the retrosage, is incorrect. So much TLL at these sites are getting filled too, also Lear is CT, jar of CP of chemicals, etc. from these landfills polluting the underground water resources. A solution to all this can only be in beings becoming more sensitive to TLL's environment issues. All WSTH we narrate and be categorized into 3 types of biodegradable, B recyclable and C the non-biodegradable. It is important that all garbage generated is sorted, what can be reused or recycled separated out, RL. Abadi Wall AHS and rag pickers do a great job of separation of materials for recycling. The biodegradable materials can be put into deep pits in the ground and be left for natural own. That leaves only the non-biodegradable to be disposed of. The need to reduce our garog generation should be a prune guel. Instead, we are encouraging TLL use of non-biodegradable products. Just pick an irritate packet of any good quality eatable, say a biscuit packet, on study the pack and you see the number of protective layers used. Note that at least 1 layer is of plastic. We have started packaging even our daily use products like milk and water in polybags in cities. Fruits and vegetables can be bought packed in beautiful polystyrene and plastic packaging. We pay so much, and what do we contribute heavily to environmental pollution. Street 8 Cloverman SAC 10 SS the C01MLI are trying to page 11 SH for Rect 11 Cheyenne in 11 set of plastics and UCT eco-friendly packaging. W can do Irby by carrying cloth or other natural fiber carried bags when we go shopping and by refusing polythene bags. L6.3 LASE study of remedy for plastic lobster. A plastic sack manufacturer in Bangalore has managed to find the ideal solution to the ever-increasing problem of accumulating plastic waste. Ahmed Khan, aged 57 years old, has been producing plastic sacks for 20 years. About 8 years ago he realized that plastic waste was a real problem. Fbliblend, a fine powder C recycled defeat plastic, was developed by his company. This mixture is mixed with the bitumen that is used to lay roads. In collaboration with RV Ojlaj of Engming and the Bangalore City Corporation, Ahmed Khan Pro. That blends of fbliblend and bitumen when used to lay roads enhance the bitumen's water repellent properties and help to increase road life by a factor of 3. The raw matter E3.1 for creating polyblend is any plastic film waste. So, go into the price of our 0.40 per kg that rag pickers had been getting for plastic waste. Khan now offers 6 rupees. Using Khan's technique, by the year 2002 more than 40 krns of road in Bangalore has already been 13i'd. At this rate, Khan will soon be running short of plastic waste in Bangalore to produce polyblend. Thanks to Innovo Lions like fbliblend we might still avoid being slaughtered by plastic waste. Hospitals generate H3's artist waste still. It contain disinfectants and other harmful chemicals and also pathogenic microorganisms. Such wastes also require treatment and disposal. The use of incinerators is crucial. 31 to disposal of hospital waste. Irreparable computers and all your electronic ODS are nil. As electronic ASTs, EWSs are buried in LMD6 LLS or incinerated. Over half of the wastes generated in the developed world are exported to developing countries. M3 in Lido, China, India and Pakistan, where metals like copper, iron, silicon, nickel, 3JLD gold are recovered during recycling process. Unlike developed countries which have specific ILLY built facilities, FBR recycling of e-wastes. Recycling in developing countries often unknows NW1UL participation SE, passing workers to Y substreams as present in e-wastes. Elent to all Y recycling is TLL the only solution for treatment of EWSs prolyrix. It is carried out in an environment-friendly manner. HLEW3K of green rheoluli use of inorg 3JLIC fertilizers and pesticides. H3S increase ed manifold for ENHMCMG crop production. Pesticides, herbicides, fungicides, etc. are being increasingly used. These incidentally are also two non-target SRNS that are important components of LLESC. Ecosystem D. Lyall inclusion in the terrestrial ecosystems we know what the addition C increasing around soft artificial fertilizers CM due to aquatic ecosystems R1SI vis eutrophica Cheyenne. The current problems in agriculture are therefore extrarily GF.E. IIEI Yogvel 6.4L case study of organic farming integrated all MIC F3 rimming is a cyclical, zero waste procedure, where waste products from One process. Are cycled in as nutrients TBR earlier processes. This allows maximum utilization of resource. N. Increase as GLLE efficiency of production. Rarish Chandra Dr. Farrer and Sonipat, Hatayana. Ispingjastel is. He includes beekeeping, dairy, banana, grant, water harvesting, composting and culture in a chain of processes which support the HLEA and allow m extremely economic md sust, a and 3 venture. There is no need to use chemical fertilizers for crops. As cottal excreta DTMGL are used. As mure, crop WS is used to air. A com P. Street which CM be used. As an adderal fertilizer or can be used to generate an adderal gs for satisfying TLLE energy needs of TLLE farm. Enthusiastic about spreading information and help on the practice of integrated organic farming. 03 Gar has created the Hatayana Kisan Welfare Club, with a current membership of 5,000 farmers. 16.5 Raw or Okal W.SMS Lanashiali, nuclear energy was IWLED, as a non-polluting way for generating electricity. Later on at W is realized TLL, at the use of nuclear energy as TVO varies and OS inherent problems. The first is accidentality, as occurred in the 3 Mile Island and Chernobyl incidents, and the second is soldi spesalivar at Io Octave Ws. Radiation, fetus given off by nuclear waste is extraelectrona and to biocl organisms speaka. Use it causes mutations to occur. At a very high rate, at high doses nuclear radiation is lethal 31 but at low purposes. It creates VNS disorders NDST frequent of alt having cancer. Therefore nuclear WS does an extremely P, 10 polyp.md. H is to be de alt vili atmospherian, it H has been recombinant TLL at storage C1 nuclear RWS. After sufficient pre-treatment should be done in suitably shielded containers buried woodland rocks. At T500 MD telole earth, S surface, however this method of disposal is reading stiff opposition from the public. In 1J Dugochlin, TLL's method of disposal, S not dos Rio LK to Mung P up 16.6 Green Jal S, I, Industrial Global. WRMD3 The term greenhouse effect has been dent from a phenomenon that occurs in a greenhouse. Have you ever seen a greenhouse? It looks like a sternal glass house, and is used for sectional wing plants especially in winter. In a greenhouse the glass panel lets the light in but does not now heat escape, therein the greenhouse warms up. One area much like insectocar TLL at H16 part in Lee Sun Tower a few hours, the greenhouse effect is an naturally occurring phenomenon that is responsible for heating of Earth's surface and atmosphere. ULB 01.C.TLZIHT9WFFTM 2M1 111U IIK Key W8LL ITTD. Surprised to know that without greenhouse eject tray RGL N1 is greater than 0 R. Tour at surface or E3 RTH the old M, even a Chile 18 rather than the present average RT, C. In 00, layer to understand the GN, and house effect it is necessary to know that they or the energy or sunlight that reaches the outerest ATM, sphere figure 16. 6. Clouds on gases react about 1/4 of the incoi forang solar oscillation and absorb some or it but street H3LF or incoming soim radiation cause on equal surface heating it. While a small proportion is reflected back, Earth's surface, MNCEMT's heat and the DRAMAF infrared radiation 110 but partorthis does not escape into space as it, atmospheric gases for example. Carbon dioxide etc. absorb a major fraction EIL1B molecules of these gases ratio it beat energy. 1 ago, N, 16.7 I watt if conch 1 buban of vanus gases total glob 1 and a major part C which again cares to Earth's surface the shatinga poncia, in. This cycle is repeated in Maya till, res, the above, emption gases carton dicrode and N, ethane are unita TLY known UGN, and house gases figure 16. 7 Because they are responsible LBR the greenhouse effect, increase in the LLF gene hotse gases H3/1C considerable heating of Earth leading to global warming. During the past century the temperature of Earth has increased by 6, see most after in the last. Figure 16.8 III Biology 3 decades. Scientists believe that this rise in temperature is leading to deleterious changes in the environment and resulting in odd climatic changes e.g. El Niño effect, thus leading to increased melting of polar ice caps as well as of other places like the Himalayan snow caps. Over many years, this will result in a rise in sea level that can submerge many coastal areas. The total spectrum of changes that global warming can bring about is a subject that is still under active research. How can we control global warming? The measures include cutting down use of fossil fuel, improving efficiency of energy usage, reducing deforestation, planting trees and slowing down the growth of human population. International initiatives are also being taken to reduce the emission of greenhouse gases into the atmosphere. Ozone hole is the area above Antarctica, shown in purple color, where the ozone layer is the thinnest. Ozone thickness is given in Dobson units. See carefully the scale shown in color violet to red. The ozone hole over Antarctica develops each year between late August and early October, courtesy, NASA 1.7 ozone and the you have earlier studied in the chemistry textbook of Class Sci about bad ozone, formed in the lower atmosphere troposphere that harms plants and animals. There is good ozone also. This ozone is found in the upper part of the atmosphere called the stratosphere, and it acts as a shield absorbing ultraviolet radiation from the sun. UV rays are highly injurious to living organisms since DNA and proteins of living organisms preferentially absorb UV rays, and its high energy breaks the chemical bonds within these molecules. The thickness of the ozone in a column of air from the ground to the top of the atmosphere is measured in terms of Dobson units. Ozone gas is continuously formed by the action of UV rays on molecular oxygen, and also degraded into molecular oxygen in the stratosphere. There should be a balance between production and degradation of ozone in the stratosphere. Of late, the balance has been disrupted due to enhancement of ozone degradation by chlorofluorocarbons CFCs. CFCs find wide use as refrigerants. CFCs discharged in the lower part of the atmosphere move upward and reach stratosphere. In stratosphere, UV rays act on them releasing Cl atoms. Cl degrades ozone releasing molecular oxygen, with these atoms acting merely as catalysts. Cl atoms are not consumed in the reaction. Hence, whatever CFCs are added to the stratosphere, they have permanent and continuing effects on ozone. Ozone depletion is OCCU, TLG widely in the stratosphere. The depletion is peculiarly marked, over the Antarctic region. This has resulted, 1 LM1 of a large area within 010 lb layer. Commonly called as the ozone hole FQ, E16.8, UV radiation of a wavelength that's shorter than UVB. Are alrestromplily observed by Earth settings, sporgy that the ozone layer is intact, but UV damages DNA and TTAU may occur. It causes aggressive skin damage to skin C, eggs and 1 cortaust 1 live. Skin cancers. In human eye, cornea absorbs UVB radiation and a high dose of UVB causes malformation of cornea, called UVL1 nea cataract etc. Such exposure may permanently the cornea, rethicular, as effects of depletion. An international treaty known as the Montreal was S. Nectet Montreal Canada in 1987 etfically 1 to eon the etcene of ozone depleting substances. Subsequently many reports have been made and protocols have logged OWN and later roadmaps. Separately for Cl, Veloped and Cl, Veloping counter SI, are, reducing the atoms, on of, CFCs and other ozone separating chemicals. 16.8 Dro, Vidishian LIYIMPJ, OptRF, S01LLTCFLLM, I, Alane, TID Mal1 it be the Degrota Iono, Natural Resources Canoer. No just by the action of pollutants but also by ilk, Mopa resource illicit, ion practices, sod erosion and desert duca yuan, the development of the fertile top NLIL, centuries, who LLT can be T, DLL easily due to human activities like overcultivation, unrestricted GRA, Z, TNG deforestation and poor Gatjon practices resulting in patches of LND. 11 Han L31 Jeb E3 when patches extend and Metaverge Moa Clusterless created internationally. It has been recognized that desertification is a major problem nowadays, particularly due to increased urbanization, waterlogging and salinity Gatjon without proper trapnag of water leads to waterlogging in the soil. Besides, fleeting the crop's waterlogging draws salt to the surface of the oil. When he salt then is deposited as a thin crust on the land surface, CR starts collecting at the roots of the plants. This increased salt content is inimical to the TCWTH of crops and is extremely damaging to agriculture. Water jogging and sail salinity, of the problems that occur in the well, of the green Marilulian. 16.9 Lil4s are all on of Cresaceonless the Eon Resian CTI, Dressled areas to non-forested ooze, JL, Rordington estimate. All's rest 40% I, rest shape and lost in the L Rofts, XLLQ, are to only I% in the Lelthoral Regton. 1 BP1 as an scenario of REI, gestation is killer, J.I. Grimm in India, at the beginning of IEI of TLLE 20th century forests CO air deba 30% of TLLEL3ND of India. By thinned off the Sentusi it shrunk to 19.4% whereas the Naleol forest F. Lissi 1988 of India has Rexi LLLJLL did 33% crest cover F for the plums and 67% for the hills. Hauxesk Teforesta Tionicare a number of Huawei CTI videos contribute to it. One of TLLE majoria sunsis TLLE conversion of forest to agricultural land so as to feed the grow human population. Trees are oxed for timber firewood, cattle ranching and for several other purposes, /unburn agriculture. Umrally called as jump cultivation in TLLE North AOS stem strategies of HLDA, has also contributed juptive prestation. In /unbum old the T-rers cut down the trees of the forest MD bum TLLE PL3 and T remains. The ash is used as a fertilizer. In TLLE L3ND is then used for junior farming or cattle grazing. After cultivation, the area is left for several years so as to low its recovery. The farmers TLLN vary on to other areas and repeat this process. In earlier days when jump cultivation was in prevalence, enough time gap was found such that the land recovered from the effect of cultivation. With increasing population and repeated cultivation TLL's recovery process is done AWIV, live, resulting in deforestation. What TLLE consequences of deforestation? One of TLLE major effects is L3 said carbon dioxide concentration in TLLE ATM sphere because used trees TLL it could hold a lot of carbon in their B01 and 3 SSA we lost with the deforestation. Deforestation also causes loss of biodiversity due to habitat destruction. Disturbs hydrologic cycle. Uses soil erosion, and nuilia detect certification in E. says, reforestation is the process of restoring a forest thought once existed but W is rendered a sorp ointment of time in the pasankri one breastation new occur natura lly in a deforested area. However we speeded up IPL3 ting trees v. little consider a scientific diversity TLL adhere her existed. In TLL TRA, L6.9L, study of people, P.N. Participation in Seonson, at 0.1 of forest people's participation has a long history in India. In 1731 the king of Jaipur and Raja STLL3NA scared one of his ministers to Aranjero, DFBR constructing a new palace. The minister and workers went to a forest near a village inhabited by Bishnois to cut down trees. The Bishnoi community is known for its peaceful coexistence v. nature. The effort to cut down trees by the kings was thwarted by the Bishnois. A Bishnoi woman Amrita DBSH01, DE, employs courage by hugging a tree and drinking Srintoku Thur 6 RST before cutting the tree. The tree mattered more to her than her own life. Sadly the king's wren did not heed to her pleas and cut down the tree along with Amrita D.V., her 3 daughters and hundreds of other Bishnois that followed her, and thus lost their lives. And trees, nowhere in history do find a commitment of Uri MMIN1AIF, IJKLHLS2WL1 and human beings actilist use lore the cause of the NIR realm. In L the GC Environment of India Basrawi I instituted the AMRUA Dimtlamal Wildlife Protection of DI, are individuals RCCRRRNLM. Ties from rural east that have shown the Roordlanal TX, Jandek, Ukraine in protecting wildlife. Umayahi 3 Co movement of Gamwa, Iherno ISLN 110,174 local Warren showed animus Brailer in protecting trees FRRR the of contract talks by hugging them people all are the work have acclaimed the Chipko RRD errant. Realizing the significance of participation by LOC, LTX, communities, the government of India in 1980s has introduced the TX, concept of joint forest management JFJ1 so as to work closely with the local communities, LR protecting and manji, Eng 6 drests and redum, LR their circuito the 6 drest karn millis t tanifi teeked vattenus MST pro, luxig. Exclamation point roots gum rupture medicine etc. and thus the ores can be conserved in a sustainable manner. Lodger, relating to Wipo Utlan 1 Clo Pu1 of, 4 Lu ABM Teral resources, ARETN dimension from ILFA section on Altigloblo, TSW pollution primarily results from bundling C4 load fuelogy. Ellen petroleum and Dusler EIOMN, Utamol, Dleo, they, harmful to L.Anzan P1TS. Antheric, almost Borimo ed 1 oak tepo air clean, domestic so ET1 emosta warn NCQ source of P.2 and of water bodies. Reduces dissolved oxygen but increases blue 40 mil oxygen demand of resolve ETNG water, dorm, itlo NWTSRTCH1NN.TLRHTNTS. Spectali nitrogen and phosphor, S, which cause watrofluo-TBN.ONYANUSAN, blooms, indus TRL1 what it waters me of NR10HTN tox 1C chemicals. SPULLY heavy metals and organ 1C compounds LR, lustral waste RS home HVU, G or J, Munlpalul 4 Wuda soaknate problems and must be spa, ICL1 NBRDFLLS. 01 SPC, Aleph hazardous wastes like defunct ships are 110. I live wastes and L. W. ULRES 81 Sonal Ali, RTS. Soli Palutlan Marli results Laurel CHSN, Cal ZGPs 11C Deslani L Atra IU Solid Depis 1 TED. Mi R1 Liter 2 Majori NL 4 on Medalsus of Global Nat We, Increase, F, QFL, DOI, LCH is Warmal 1 LF Earth 1, LOPLU on 0030 in the STRAL sphere. Enhanced greenhouse Sophia LS mainly DT to UA colon 1 list 10 N of carbon D10 methane N1 tro, J, oxidon, CFLS, and also duodeferanid may drastically R. on blapatum global temperature. Besides delerdonously effecting organisms o, no in U, slura, sphere which prolix us from hotfit effects of 101 ATL radiatlin. Is depletengel, SLU emion C4CFCs thus UU, SLN8 the SKS of SLD anti mutation and osterclsorders. Exercises 1. WH31 me iliavimtus constituents of domestic so, discuss your lead, texts of sodiish mge on a read.10, R. 2. List in U, EW ulmit UF, narrated H, LMESCH, Lollerdermgear trips to oilier playos could ho 10, YE3 silly reduce which would be diff culture R iluron pos 1 bletcher reduce 3. DSCSU, E causes. Male effects of wa, wrong, what JWNSWS nay, L to BT all, and to control global W4. Match U, E items G1 vet, N1 N collar and A and B, Ka anakohun B ecololytic C, R1P matter B iliatristolic PCE, PT. RU carbon monix 1 Di nilrog oxy, la CER mufs U1 lig 8 H nolds TED landfills HV solid W, stes 5. WRT, C. Tickle notes on the following, a eutrophication B biologication C groundwater depletion malways that for its replenishment. 6. Y ozone H, LLEFLSO.U and P. How W1L ultraviolet R3D 11 on ELA 7, discuss the role of women and communities in protection conserve, DIN of F forests 8, what measures as an Indi? I Julia would TE to reduce MU all mental pollution ARF 9, DLSCS protefly the F all AJ radioactive 1 ver waste speedy, funct ships. NDW ost CMWL 101 pal solid wastes 10. What ULLIU, S were taken F were red CUG uba Mr. Pollution in Delhi has quit 3 Lydium Pro D1 and Delhi 11. DLSCS protefly the following, a greenhouse gases B catalytic converter C ultraviolet B,